कोई ऐसा गीत गाओ कि आरज़ू जगाओ अगर तुम कहो। Hi students, are we live? Live. Hello people. Hi everyone. So can you see me, hear me? Hello, happy morning guys, happy morning. Hello everyone. So I hope I'm perfectly audible and visible. Yes, I hope I'm perfectly audible and visible. Hi. Good morning everyone. Good morning. Welcome to the session. Welcome to the class 11 complete zoology marathon. I hope you people are equally excited and you are ready with your NCERT, with your markers and everything. Good morning everyone. So I'm so sorry for cancelling your session yesterday because my health was not well, and if I would have started that session, I don't think I was able to complete the syllabus. That's why I thought that I should cancel that session so that I can take the session with full energy, with high josh, so that we can complete the class 11th zoology. And don't worry people, nothing to worry about. There is nothing to worry about people that ma'am, what about the botany part? Let me finish the zoology portion, then we will of course talk. We are going to talk about the zoology, botany, है ना? And you know what are we planning? Because you know that now biology, right, it is going to give us 360 marks. So first of all, in the chat section, people, tell me everyone, tell me how many of you are targeting 360 out of 360. Do let me know in the chat section, people, how many of you are targeting 360 out of 360. I'll be starting human physiology, nothing to worry about, right? And the session is strictly on the basis of NCERT. Okay, I really enjoy writing on the slides. I really enjoy, explaining the topics in detail. But yes, we have to be very specific. Well, the preparation should be directional at this point of time. So the session is strictly on the basis of NCERT. Okay, it is strictly on the basis of NCERT. But before that, everyone in the class, do let me know that how many of you are targeting 360 out of 360 marks in biology, and quickly hit the like button, people. And I want your response here. I want your response here, guys. 360 out of 360, yes. Do you want that? Of course we want that. So after these zoology marathons, of course there will be botany marathons, right? And there will be separate sessions where we are going to discuss all the diagrams in one shot. So there will be a one shot of diagrams, okay? There will be a one shot of most important topics, right? I hope you all have seen the video where I have posted the top 60 topics, right? Top 60 topics. So I am going to provide you a separate session for that too. Okay, so diagram based session will be there. And yes, I will discuss last 5 years, right? Last 5 years NEET questions. There will be a session of 2 to 3 hours where we are going to complete last 5 years NEET PYQ separately. Okay. And if I will get good response from you people, might be you will get last 10 years PYQs. So you want such sessions? Yes, ma'am, can you add the scientist and the dates and time? Of course, I will. We are here for your service, definitely. It will be done. But yes, I want response from you people. So no one is going to leave the class today, and be energetic. You will get the breaks, and in today's session, I'll give you the small breaks also in between. 10 minutes ka break where we will chit chat, 5 minutes ka break where we will chit chat. So don't worry. Be happy, be energetic, and please watch the session till the end. And you know what? There is something very exciting for you, and I think you all are aware of it already. Aram, right? Why we are emphasizing on this? Why we are saying that it is something very important? Because bache, there was the time when Unacademy subscriptions for plus, right, they were more than 30,000, more than 40,000 rupees, right? So the motto of Unacademy is to provide the quality education to each and every individual. That is why they have lowered the prices, and now it is at 4,999, and that too till 14th of April, right? So plus subscription means that 1 year ka subscription, now you just have to pay 4,999 for that students, and that is something very less, because that much amount sometimes we spend such amount on our clothes, right, on you know, parties and all. But now you can in that amount you will get the quality education, right, proper lectures, revision, study material, MCQs, everything will be there. And it is not the one batch that we are going to provide you. Last year, you know, when we started our journey, exactly a year back, right, I still remember, on 14th of April I joined the Unacademy. So a year back we started our journey, and then we launched NEET Avengers batch. But this time we have planned that we have batches for the students that are moving from 10th to 11th, for the students that are moving from 11th to 12th, and even for the droppers, for the dreamer as well. And from 17th of April your classes are going to start. So just imagine if in our YouTube sessions we can provide you that quality content, if in YouTube sessions we can teach you like this, we can teach you with you know, the enthusiasm, we can teach you with all our dedication, then imagine in our batches how active we are going to be, and how well we are going to train you people here. So quality education will be provided to you. So you know that batches are there, so you know our team very well, people, right? So you can be the part of that. So Nimbus NEET English batch is for class 11 student, Nexus NEET English batch is for class 12 student, and Phoenix NEET English batch is for droppers. And we are going to teach you people, we are going to teach you people, we are not going to leave you in between. Okay, so in the description box there is a link, you can click on that and you can be the part of that batch. So now let's start. So can we start first with cell cycle and cell division? I know you want me to teach the human physiology first, but from cell cycle and cell division 3 to 4 questions will be there, and I need 1.5 hours to complete that. Take care. So 3 to 4 questions means near about 12 marks, and let's say 2 hours for that. Okay, 2 hours for that. And guys, I know you all are registered in AIMT, but please every, right from March 31st to April 28th you have to give the test as well. So be active in that test. Okay, so it is also free of cost, so do not leave it. So can we start cell cycle and cell division? Can we start cell cycle and cell division? I will hardly take your 2 hours for this. So are you people ready? Are you people ready? I know you want me to start with the human physiology. I can start with that too, but the point is these chapters are very short, right? Let's complete it. Let's finish it, then structural organization, then human physiology. Right, I thought that I will start with cell cycle, then biomolecules, then we will move to the human physiology. But if you want me to teach human physiology first, don't worry, let me finish cell cycle and cell division, then structural organization in animals without cockroach and frog, because cockroach and frog I will take in a separate session. Then let's start the human physiology. Ready people? No, today I am not going to include that. From cockroach 1 question, from frog 1 question, and I believe that will be in the option. Okay, so I am not going to spend my 4 to 5 hours for that topic, right? I will provide you the lecture, right, near about, I think on 21st or 22nd you will get the lecture on this frog and cockroach part, because I want you to remember specific points only. Trust me, question will come from that. I know that. It is as per my experience. Animal kingdom is also included in the lecture. So now come back. So the topic is cell cycle and cell division. I will be very specific here in this particular topic. So I want you people to be attentive, because 4 questions can come from this part. 4 questions, right? 16 marks. So we are preparing for 12 to 16 marks in basically in 1 hour 45 minutes. Now, the very first point is cell division. What is the meaning of cell division? A cell is going to divide. Is it important? Of course it is. Of course it is very important. Why it is important, students? Because see, you have a cell, right? Let's say this is a diploid cell. When a cell will divide, it is going to increase its number. Of course, it is going to increase its number, and that is important. That is important for the growth. That is important for the healing, right? That is basically important. Let's relate it with the human reproduction part. So you know that when you talk about the germ cells, even they proliferate. They increase their number. They have to increase their number. Why? Because if they will increase their number, the spermatogonia will form, the oogonia will form, which will further grow, and the gametes will form. So yeah, division is important. But the question is that why cells divide? 2 reasons are there. First is nucleocytoplasmic ratio, right? First is nucleocytoplasmic ratio, and the second point is Second point is surface volume ratio. How many of you are aware of it? These are the two reasons due to which a cell divides. These are the two reasons due to which a cell divides. Why? When there is a disturbance in nuclear to cytoplasmic ratio and when there is a disturbance in surface to volume ratio. Now we have to understand that what is it, right? Pavi bache, cell topic will be completed in the botany part. Okay, so see, when a cell is small in size, okay, because it is small in size, I hope you all know that small size cells, they are the active cells. Okay, small size cells, they are the active cells, isn't it? They are quite active. They are quite active. So when you look at a cell, people, you know that, see, this is the nucleus. It is going to be large. It is going to be conspicuous, right? Visible. It is large. It is conspicuous, right? But batches are there in English language. Niveda, batches are there in English language, but for 1 year you join the subscription, you will also have the access of the Hindi batches as well. Okay, right, bache? So I'll provide you the DPP as well, people, but please focus here now. So we are having the cell. Cell is small in size. When it is small in size, it is having the large nucleus, conspicuous nucleus, and then there is the cytoplasm. So because it is small in size, nucleus size is large. So because of that, what's going to happen, people? This cell is having a good control over this cell. This nucleus is having a good control over this cell. This nucleus can control the activity of the cell in a better way. Now, because cytoplasmic growth is a continuous process, right? So when the cell will grow, right? When the cell will grow, what is going to happen, people? What is going to happen? Now nucleus is of same size, but the cytoplasm will increase. Now the cell is not having a control over this, nucleus is not having a control over the activity of the cell. So now it is a kind of alert that, yeah, I need to divide. So nucleoplasmic ratio, also known as Kern plasmic ratio. So in the case of small cell, this ratio is high, right, bache? Right, bache? So when it increases, the cell will be like, yeah, I need to divide, right? I need to divide. Now come to surface to volume ratio. When a cell is small in size, right, when a cell is small, then you know that its surface area will be more. So if its surface area is more and comparatively its volume is less, so if surface area, more surface area is very important for the exchange of material within the surrounding. Okay, Sudha, right? This is very important for the exchange of material within the with the surrounding. Okay, so large surface area and less volume means quickly the things from the surrounding, they will enter, they will come in. Right, they will enter, they will come in. That's the point. That's the point, people. So when the cell will increase in the size, right, when it will enlarge, its surface area, right, comparatively what's going to happen? Its surface area is less, its volume is increasing, and due to which, right, because less material is getting exchanged, so the demands of cell will not be fulfilled. So it is a signal that now I have to divide. Got it? Got it? Yes, got it. These are the two reasons for the division. Okay, so now let's talk about the NCERT. As I said, this is something we are not going to leave. So guys, even in my regular batches, this is something that I have highlighted, but I think I should tell you that also. See, growth and reproduction, they are the characteristic features of a cell and of all living organism. So let me relate it with the chapter living word, people. So they are saying characteristic feature, not the defining feature. You remember defining features? You remember defining properties of a living cell, where we consider metabolism, right, where we consider consciousness. Can I just increase my speed? Are you people okay with it? I want to maintain a proper speed. It should not be like that ki initially I'm going slow and later on, you know, I just speed up. I want to maintain a speed from the starting itself. So I want responses from your side. I'll be quick. Okay, bache? So what am I saying? That when you people talk about the living word, when you talk about the defining properties which are specific to the living organism, it is something which will definitely be there in the living organism. You cannot say that any living organism is without these properties. Of course not. So metabolism, right, self-consciousness, okay, and then you know that the property of that cellular organization, that all the living organisms, they are made up of cell. Okay, they are made up of cells. The metabolism will be there, the reactions are there, and the consciousness will be there, means that they respond to surrounding. But here what's the point? Growth and reproduction. So growth and reproduction, they are not the defining property, rather they are the characteristic feature, means it is something which we will see in the living organism, but it is not the defining property. So we know that cells, they will divide into two, right? Cells, they will divide into two. Just say this is our parent cell, and these two are going to be the what? They are the daughter cells. These daughter cells will further divide. Okay, these daughter cells will further divide. These daughter cells will further divide. So all these cells, they are the progeny, right? They are the progeny of the single cell. So when I use the word progeny, I'm talking about the generation of this particular cell. Are you getting my point? I'm talking about the generation of this particular cell. So a single cell is dividing. It is increasing its number. So basically it is the next generation of this cell. You will use the word progeny. Sometimes we are not able to answer such simple questions in exam. Sometimes we get confused when they are the statement based questions. So please make sure that you understand that. Okay, you understand that. So this part clear? This part clear? So now when it comes to the cell cycle, one line that I would like to highlight, and you should also highlight it in your NCERT because it is directly a MCQ, that see, it is a sequential event. In a proper sequence, things will go on, right? In a proper sequence, things will go on. So see, what is happening? What is happening when a cell is dividing? Cell division is going on, right? DNA replication is there, cell growth is going on. Okay, bache? So they all are in a coordinated way. In a proper coordination, things are going on. So the sequence of events by which a cell duplicates its genome, it synthesizes the other constituents of the cell and divide into two daughter cells is the cell cycle. It's important. It's important. Right, guys? It's important. So sequence, it's a proper sequence of events. So in which cell will duplicate its genome, means its genetic information, means its genetic constituents, right? It will synthesize the other constituents. Just say, of course, in a cell, cell organelles are there. So when a cell is growing, it will duplicate its DNA. Why? Because in the daughter cells, we need to divide the equal amount of DNA. Plus, the organelles which are present in a cell, they will also divide, they will also double their content so that equal distribution will be there to the daughter cells. That's the point, G. Gopal. Okay, G. Gopal, please start reading properly. Trust me, you will get good score. At least you will get an idea that you are meant for this exam or not. So I will always say, right, Sampak, definitely bache. So I will always suggest you this thing, people, that if you are thinking that what is the advantage of starting now, I will not be able to complete my syllabus. It's not like that. If you will start now, if you will start giving your best, maybe whatever you are reading, you will get questions from that part. And if you will give your time now, right, you will get an idea that whether I'm meant for this exam or not. Am I ready to put that much of am I ready to put that much of efforts? Am I ready to do that much of hard work? So that's the point. So that's the point that you need to consider, bache. And I am pretty sure that if you will work hard, if you will start your preparation from this particular day only, right, bache, you will learn a lot of things. So as of now, if you are thinking that, yeah, ma'am, I'm going to start my preparation, do not focus on the result. Just think about your efforts. That's all. Okay, that's all. So this part clear, this part clear. So one thing that we need to keep in our mind is that when it comes to the growth of a cell, it is a continuous process. Okay, it is a continuous process. What's that? It's a continuous process. It will keep, cell will keep on growing, and cell growth means cytoplasmic growth. So in a cell, always there will be the cytoplasmic growth, people. Why? This is a basic procedure. Cell will keep growing, it will increase its cytoplasm, but when it comes to the DNA replication, it will only occur in, when it comes to the DNA replication, it will only occur in, yes, people, when it will come to the DNA replication, it will occur in, yes, Leharika, exactly, bacha, in the S phase. In the S phase, that's what we need to keep in our mind. Done, bache? So now let's talk about the phases of cell cycle, and I will make one slide and we will just finish it. Okay. Arey yaar. So next is phases of cell cycle. So bache, when you talk about the phases of cell cycle, so here you people will see interphase. What are you going to see, people? Interphase, and then there will be the M phase. Now when we talk about this interphase, it is also known as resting phase, it is also known as preparatory phase, it is also known as invisible phase or I phase, it is also known as energetic phase. right? It is having the different names. It is also known as, interphase is also known as energetic phase. So energetic phase, resting phase, preparatory phase, I phase, you can use any name for your interphase. Now when it comes to the M phase, it is the actual division phase. So what is happening here? Whenever you talk about a cell, right, so it will spend more than 95% of its lifespan in interphase. This is what you need to remember. And I always give one example here, and which one is that example? Which one is that example that I always give you here? And that example is, people, that example is ki you are preparing for your NEET examination. Yes, you all are preparing for your NEET examination, right? So when it comes to the NEET preparation, you know that actual day is that exam day. Just say for you it is the 5th May. 5th May, the time from 2 to 5:20, that is going to be the very important time of your life, isn't it? Isn't it? So same is the case here. When you talk about a cell, it is having two phases. One is interphase and another is M phase. M phase is the actual division phase. M phase is the actual division phase. Let me explain you here. Just say you are preparing for your NEET examination and you are giving your best. You know that preparatory phase is very important. For other people, maybe you are just sitting in front of a laptop, you are just taking a class, yeah, you are just studying something, you're just making notes. So other people, they are not going to judge the seriousness, they are not going to understand the seriousness of your situation, right? They will not be able to realize that how difficult it is to survive in the preparatory phase, because in that preparatory phase you are preparing for this. You are preparing for your actual NEET examination. If you want to give your best in your NEET examination, you have to be very active, you have to be focused, you have to be determined, you should have that perseverance right in that preparatory phase. You know it very well. You know it very well that how important is the preparatory phase, right bache? Right bache? You know that how important is that preparatory phase, isn't it? Isn't it? Isn't it? And in this phase you are under a lot of pressure, but you know that you have to deal with that pressure, you have to manage that pressure just for this particular phase, right? Same is the case here, guys. Same is the case here. Can you people relate with me? Can you people relate with me? If it is a yes, hit the like button and make sure you subscribe to our channel, people. Make sure you subscribe to our channel. So now, guys, I am relating such a wonderful example here. So I am expecting some Josh in the chat section, right? Right, I'm literally expecting some Josh in the chat section, people. I'm giving you such a wonderful example. You will never ever forget this. You will never ever forget this. So same is the case here. So more than 95% of the lifespan of a cell is in the interphase, and then comes the actual division phase. So when it comes to the interphase, obviously it is the phase in which cell is metabolically very fast, very active. So in this interphase, this is what you people are going to get. G1, S and G2, G1, S and G2. And when it comes to the M phase, what will you see here? You will see karyokinesis. Kinesis means division, karyon means nuclear, and then you will see cytokinesis. That's what we are going to see here. Okay, that is what we are going to see here. Are you getting my point? That is what you are going to see here. So karyokinesis and the cytokinesis will be there. Now what is the another point here? See, this G1 phase, it is known as the growth phase. It is known as the pre-DNA synthesis phase. Why? Because it is happening just before the DNA synthesis. Okay. It is also known as post-mitotic phase. I'll tell you what is the meaning of that. Post-mitotic phase. Now S phase, right, where DNA replication occur. What is going to happen in this phase? Guys, 3 questions or 4 questions will come from this chapter, so you make sure you stay tuned to this, right? So DNA replication occurs, okay, but number of chromosome remains same. Number of chromosome remains same. Clear, bache? And you know that what else is going to happen here, right? The centriole duplication will also occur here in S phase. Okay, centriole duplication will also occur here in S phase. That's what you need to keep in your mind. Then comes the G2 phase. So when it comes to the G2 phase, it is the growth 2 phase. It is the gap 2 phase. It is also known as mitotic phase. So this is the phase where cell is preparing itself for the division. So all that important RNA, protein synthesis, DNA replication, everything will happen. So now I will share the story related to this. Okay, I am going to share the story related to this. Okay, so let me tell you what's going to happen. Dekho, in the NCERT there is a line. If you all can find out that line, please highlight that line and tell me, yes ma'am, it is highlighted, right? In NCERT there is a line that your interphase is the phase in between two successive M phase. Can you see that line? Can you see that line? Can you people see that line? That interphase is the phase which is in between two successive M phase. Quick. Interphase is the phase which is in between, right? Which is in between two successive M phase. Which is in between two successive M phase. Can you see that? So if two successive M phase means M phase means one after another. Like M phase is the actual division phase and M means division. M means mitosis. M means mitosis. So interphase is the phase which is in between two successive M phase. It is a phase which is in between two successive M phase, means two M phases, one after another. In between you will have interphase. So of course, interphase is your G1, interphase is your S, interphase is your G2. That is why I am saying, dekho, this is the line of NCERT. I am explaining you the lines of NCERT. Guys, question will come from this part, and trust me, after your NEET 2024 session, you can check these marathons and you will be amazed to see ki Hanji, madam was saying ki this is important, this is something we need to highlight, hai na, this is something we need to revise. So people, please pay attention. Okay, please pay attention. So NCERT is saying that I phase is a phase which is in between two successive M phase. I phase is a phase which is in between two successive M phase. Okay. So after M phase I am having G1. That is why I am calling it as post-mitotic phase. That's why I am using the word post-mitotic phase. Got it? That is why I am using the word post-mitotic phase, right? Or can I say that each and every line is in NCERT? Can I say that it is the phase in between, right, in between cell division and DNA synthesis? Can I say that it is a phase in between cell division and DNA synthesis? Can I say so, people? Can I say so? It's a phase in between cell division and DNA synthesis. Cell division and DNA synthesis. Can I say so? Can I say so? Of course I can. Of course I can. Just look at this line. It is explaining everything. So what is going to happen in this G1 phase? RNA protein synthesis will occur. RNA protein synthesis occurs. Right. And you know that all the organelles, they'll duplicate their content. All the organelles, they will duplicate their content, right? So enzyme synthesis, ATP synthesis, amino acid synthesis, everything will be there. Your enzymes, your amino acids, your nucleotides, everything will be there. Okay? Everything will be there. Now I'll tell you one thing, because students, in G1 phase, right, cell, no doubt, RNA and protein synthesis is going on. Actually in this phase, sweetie, Sudha, actually in this phase, cell will synthesize everything which is required for the next phase. Okay? This is a kind of rule also. Whatever is required for the S phase, it will be synthesized in G1 phase. Whatever is required for the G2, somewhere the things will be synthesized in S phase. I'll relate, I'll give you one example. So up to this part all clear? So just here in S phase, we have to replicate the DNA. So the enzymes for the DNA replication will be synthesized in the G1 phase. It will be synthesized in the G1 phase. And G1 phase, when it comes to the timing, it is the most variable phase. What is the meaning of most variable phase? Most variable phase means, bache, its time is not fixed. Maybe a cell which is actively dividing, in that cell it will be of 10 minutes. Just giving you one example, let's say in another cell it is of 20 minutes, it is of 30 minutes. So its duration is very variable. Its duration is variable. Yashika, what do you want me to explain, bacha? its duration is variable, right? Call me mango G, in all the phases metabolism is going on. In all the phases metabolism is going on. It's just that here actively RNA protein synthesis is there. All the organelles, they will duplicate their content, and it is the most variable phase. What's the meaning of most variable phase? Means its timing is not fixed. Then comes the S phase, right, where DNA duplication, centriole duplication occur. Now let me explain you this. DNA duplication and centriole duplication occur. Are you getting my point? Now let me explain you this, and I will increase the font size also. Maybe you will find it difficult to understand what I'm writing today. Okay, I'll give you one example. Dekho, let's say this is a cell. Let's say it is 2n is equals to 4. What is the meaning of 2n is equals to 4, people? What is the meaning of 2n is equals to 4? Hanji, tell me, what is the meaning of 2n is equals to 4? Yes? Yes, yes, yes. Exactly, means in diploid state the cell is having 4 chromosomes. I know when it is the interphase, and even you know, you should know, in the interphase DNA, the chromosomes, they are not in the compact state, they are just thread-like, they are just tangent, right? They are not the individual units, they are not the discrete units, they are just like, you can just imagine a woolen ball. Okay, it is like that. Okay, but I need to explain you something here. I need to explain you something here. That is why I am taking this example. Kamil Bache, let me explain, all the doubts will be clear. I am saying, imagine in this particular cell, the number of chromosomes, they are 4. Means this will be the condition. This will be the condition. Let me tell you, people, if you think, right, that always this is the chromosome. No. No, it is also your chromosome. It is also your chromosome. It is monad. Monad means it is having single arm. It is having single arm. It is also your chromosome. So I am saying that after DNA replication, after S phase, what is going to happen? In the NCERT it is written now that if initial amount of DNA, right, it is written now that if initial amount of DNA is 2C, it will become 4C. Now ma'am, what's the meaning of this 2C and 4C? है ना? What is the meaning of this 2C and 4C? बच्चे, it is very simple. It is very simple. This 2C, 4C thing is very simple. 2C, it is used to denote the DNA amount. जैसे let's say I am saying that initially DNA amount is 100 gram. I am giving a very layman example to you so that you will be able to retain it for a longer period. So let's say before the S phase, the DNA amount is 100 gram. After S phase, it will be doubled, means it will be 200 grams. It will be 200 grams. But what is the key word here? That number of chromosome will remain same. How is it possible? Let me explain you. But number of chromosome remains same. They will remain same. So what is the point here? It's very simple, people. It's very simple. Just see, how many chromosomes are there? 4. After S phase, DNA duplication. This chromosome, it is also basically what? The chromatin, the condensed DNA and protein. Basically, it is also the DNA and protein. So after S phase, these chromosomes, they will be like this. They will be like this. So look at it. The DNA amount is doubled. DNA amount is doubled, but the number of chromosome is same. So before S phase, it is in this monad form. Single arm is there in this chromosome, right? Single arm is there in this chromosome. But after S phase, its DNA content will get doubled. So these arms are nothing but the DNA. So they will get doubled. So this is what you people are going to get. Okay, this is what you people are going to get. So these are also known as sister chromatids. So both are chromosomes. Both are chromosomes. This is the chromosome before this S phase, right? Having single arm, having 2C amount of DNA. This is the chromosome after S phase, having 4C amount of DNA. But if you look at the number of chromosome, you can see it's same 1, 2, 3 and 4. Got it? 1, 2, 3 and 4. Got it? And yes, it's also a NEET 2021 PYQ that in the S phase, centriole duplication will also be there. So what is centriole, people? What is centriole? What is centriole? It's a non-membrane bound organelle present in the animal cell, absent in the plant cell. I will explain it also. I will explain it also. So this part clear, guys? This part clear? This part clear? S phase clear? S phase clear. Now come to the G, and yeah, in the S phase, histones you know na, and non-histone proteins, histones and non-histone protein synthesis also occur. So if someone asks you where that histone and non-histone will synthesize, in the S phase. So guys, doubts clear? Doubts clear? Got it or not? Got it or not? If it is a yes, I want to see some energy in the chat section, people, quick. I want to see some energy in the chat section, people, quick. And thank you so much for watching my lectures. Thank you so much for being so consistent, people. And yeah, I'm little disappointed. Yesterday I was not well, and in the chat section people were just, you know, why do they cancel the class? What about the other channels? What about this? What about that? People, other channels, right, they will just give you the feel that they are teaching, but we are actually teaching you, right? They will just start one series, they will leave it. Even we have done that, but in all that series we have provided you the quality content without compromising it, right? This is something that you people can observe, right? We are not just, today we are starting one lecture, tomorrow we are doing something else, day after tomorrow we are doing something else. No, if we have decided we are going to teach you in game of NEET, we provided you that because you were not responding well, so we thought that now it's the time for the marathons, and be it your physics class, chemistry class, biology class, you are getting your marathons. Till 20th we will finish your syllabus, we will move to diagram based sessions, we will give you the NEET predictor papers, we will give you the NEET replicas, we will give you all that content, scientist name and data and everything. The question practice sessions will be there, the top 1000 question practice sessions will be there. Every day we are going to solve the paper just like the NEET examination. We will start from biology, chemistry and the physics like this, right? So this is what we are doing, and really you need to understand that thing, bache. So we cannot just come without energy, and we can just come in front of you and we'll be like, theek hai yaar, just, yeah, I just need to do the formality. No, we really want good selections from this channel, right? So if someone spam here, then you know what you have to do, right? Throw them away. The spammers should not be there on this channel. So this is what you have to realize, people. Okay, chalo, now let's focus here. This part clear, this S phase clear. So I don't want any mistake in this portion, that why is it so ki DNA amount is getting doubled but the number of chromosome will not change. So this is what you need to remember. And now when it is the G2 phase, again simple. Here also RNA protein synthesis occur. Okay, bache, right? It is of short duration comparatively, and in G2 phase, write down tubulin protein synthesis. Right, tubulin protein synthesis occur. Can you tell me what is the tubulin protein? Anyone in the class? Can you tell me what is the tubulin protein? Anyone in the class? Can you just tell me what is the tubulin protein? I am mentioning it here for you, tubulin. This tubulin, it will polymerize and it will form microtubules. It will form microtubules. Are you getting my point? Just see alpha tubulin, beta tubulin, alpha tubulin, beta tubulin. They all will polymerize. They will form protofilaments. That protofilaments will join and they will form microtubules. And you know that they are very important for the spindle apparatus. They are very important for the spindle apparatus. So that is how this tubulin is going to help. So it will form the microtubules. Doubt, people? Any doubt? Any doubt? Any doubt? Any doubt? Any doubt? Done. So after this you are having M phase. So M phase may, what I just said, there will be the karyokinesis first, that is nuclear division, and then there is the cytokinesis, that is cytoplasmic division, and that is something we need to understand now. Okay, that is something. And remind me, I forgot to teach it in my class also. So students of Dream Crash Course batch and NEET Endgame, if you are watching this lecture, do remind me. I'll explain it in the class also. So guys, we need to discuss the checkpoints also. After completing this, we have to discuss it. So now let's focus. So this is what we have, M phase and the karyokinesis and cytokinesis, nuclear division. So here the trick is PMAT. What is the trick? The trick is PMAT, because we are starting with the mitosis. I'll explain the mitosis also. So we are starting with the mitosis. So it is PMAT, prophase, then comes metaphase, then comes anaphase, then comes telophase. Prophase, metaphase, anaphase, telophase. This is what karyokinesis is. This is what karyokinesis is. So it is prophase, it is metaphase, it is anaphase, it is telophase. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Repeat it, people. Yeah, we will discuss G0 also. Allow me some time. Even G0 will be discussed. Even G0 will be discussed. Don't worry, I am not going to forget anything. Okay, one more phase is there, na? G0 phase. We will discuss that, but after this. After this. Okay, so this part clear, this part clear. This is what you are going to see in the karyokinesis. And when it comes to the cytokinesis in the animal cell and the plant cell, it is different. Now move to this point, this diagram. Right, let's talk about this diagram. So you can see this G1 phase, S phase and G2 phase. Now you people can see there is something called G0. It is the quiescent stage. It is the quiescent stage. It is also known as cell arrest stage. It is also known as cell arrest stage. What is it? It is the quiescent stage and it is also known as cell arrest stage. Now let me come to this point. It's not, dekho, if you talk about a human cell, you know that in 24 hours it will divide once, right? That cell division ka duration in the human cell is 24 hours, and in the case of yeast, which is a unicellular fungus, it will be 90 minutes. How many of you are aware of it? How many of you are aware of it? It is also a direct P by Q. It is also a direct P by Q. So when you talk about the human cell, as I said, the duration of cell division is 24 hours. The duration of cell division is within 24 hours the cell will divide once. So when it comes to the actual division, it is less than 30 minutes. Okay? It is less than 30 minutes. Right, when it comes to the actual division, it is less than 30 minutes. Rest is interphase only. Rest is interphase only. Okay, bache? And in the yeast it is 90 minutes. Now this is about the cells that are dividing, but it is not mandatory that all the cells will always divide. They will divide when they are required to. Okay, they will divide when they are required to, otherwise there is no need. So when a cell will get a signal that, yeah bro, you have to divide now, right, that yes, you have to divide now, it is important for you to divide now, then only that cell will divide. Is that clear? Then only that particular cell will divide. Is that clear, people? Hanji, is that clear? Then only that particular cell will divide, otherwise there is no need to divide. Okay? So when there is the signal for the division, the cell division will occur. Okay, ji? The cell division will occur. So now some cells, they just divide only at the time of embryonic development. Yeah, they divide occasionally. Some cells they divide occasionally. Some cells they divide only once, just your nerve cell and heart cell. There are other discoveries which explain something else, but I am not going to discuss that. Okay, so what's the point here? The nerve cell and the heart cells, right? They will divide once. They will divide during their embryonic development. So they will leave this G1 phase. They say, let's say a cell is in the G1 phase. A cell is in the G1 phase. It is waiting for the go-ahead signal. It is not getting the go-ahead signal. It will leave the G1 phase and it will enter in this quiescent stage, in the cell arrest stage. Okay? And sometimes it is permanent. A cell is permanently in the G0 phase because they will not get the signal to divide further. But sometimes what happens, right, they are present here occasionally. Just say, let's say these body cells of mine, okay, they are dividing, they have increased their number. I got an injury, now they are not dividing. I got an injury, what will they do? What these cells will do if I will get an injury? Of course, these cells will be like, yaar, chalo, now it's our time to divide. So they will leave G0 phase, they will again enter in G1 phase. Are you getting my point? So it is the quiescent stage. So here in this stage, the cell is metabolically active. Right, the cell is metabolically active, but it will not proliferate. Means it is not going to increase its number, but it will keep, right? It is not going to increase its number. It is not going to increase its number, but it will keep dividing. Right? It will having its metabolism. So it will increase its size. It will do all the necessary activity, the chemical reactions, but it is just that it will not divide. Carpe diem. Ma'am, whether cockroach and frog both are there in the NEET syllabus, and I will teach that also. Okay, I will teach that also. So metabolically they are active, but it will not proliferate. So no doubt we say that G0 is the inactive stage, but being inactive doesn't mean that cell is in inactive state. Always remember, living and metabolism, they are synonyms. Being living and metabolism, these two things are synonym. If a cell is living, okay, if a cell is living, then obviously metabolism will definitely be there. If a cell is living, definitely metabolism will be there in that cell, means that cell will have these chemical reactions. Any doubt, people? Any doubt, people? Any doubt? Okay. Now I told you that I will teach you about the checkpoints, right? It is not given in NCERT, but if you will solve PYQs, you will get the question on the basis of checkpoints. Dekho, in our body is a wonderful machine. I still remember, I think in grade 5th we used to get this question, right? Our body is actually a wonderful machine. In our body there are different mechanisms. Dekho, in our body we have the immune system which will protect us from the intruders. In our body we are also having, what do we call it? Yeah, in our body we have mechanism for everything. Jaise, we have discussed class 12th zoology marathon. Do you remember class 12th zoology marathon? Do you remember our class 12th zoology marathon? Yes, 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 yes. If you have seen that, very good. Otherwise, just subscribe to our channel. You can see that, right? So it is the complete class 12th zoology mega marathon. Okay, so here we have revised everything. Human reproduction was pending a bit because that I have explained so many times. That is why. Okay, so you can check out these sessions also, right? And I will schedule the botany lecture soon. Okay, so what am I trying to say? I am just trying to say that yeah, our body is having everything. In the immune system, this is what we discuss. In human health and disease, this is what we have revised. So I am saying that in our body, if you talk about the cancer, it is the uncontrolled cell division. When you know all the control system of our body is not working, then there is the cancer. So I am saying in our body, there are genes which will suppress that tumor. Okay, which will make sure that even DNA replication is proper, which will make sure that cells, they are not dividing, you know, too frequently. Just you know that if I talk about the chromosome, ends of chromosome, they are having the telomeres, right? So there is one enzyme, telomerase, that makes it. So when a cell keeps on dividing, these telomeres, these ends, they, you know, decrease. Okay, they decrease. That's why the cell, there comes a time when the division will stop. Okay, I'm just trying to relate two different things. So I'm saying in our body there are checkpoints also, and that checkpoints will make sure that division and everything is going properly. Okay, division and everything is going properly. So it's a NEET PYQ. We have to talk about the checkpoints, and the main checkpoints is here. The main checkpoint is here, right? This is here. It is also known as start point. It is also known as restriction point, right? It is also known as start point. It is also known as restriction point. You can also call it as start point. You can also call it as restriction point. Actually, it is the point. If a cell will cross this point, for sure it will enter in S phase. If a cell will cross this point, a cell will for sure enter in the S phase. That's what you need to remember. That is what you need to remember. Just if a cell will cross this point, definitely no one can stop that cell to divide. No one can stop that cell to divide. That cell will definitely enter further in the S phase. So this checkpoint, this first checkpoint is G1S checkpoint. It is G1S checkpoint. It is G1S transition. Okay, it is G1S transition. Is that clear? Is that clear? Yes or no? Is that clear? Yes? Yes, people, is that clear or not? Yes. So this checkpoint will make sure, right? So if there are sufficient growth factors, right? If there are sufficient growth factors, if there are sufficient nutrients, right? If the DNA damage is not there, so it will take care of all that things, and if everything is fine, it will give a go-ahead signal, ki bro, now let's enter in S phase. So once a cell has crossed this checkpoint, for sure it is going to divide, and it is a NEET PYQ. Priya, it is a NEET PYQ. Okay? Then G2 to M. Here also you will get a checkpoint. Here also you will get a checkpoint. That will be G2 to M transition. Let me tell you, students, let me tell you, when it comes to the checkpoints, you talk about certain proteins. They are known as cyclins and CDKs. They are the enzymes. We will talk about the cyclins and we will talk about CDKs. CDKs means cyclin dependent kinase. CDKs means cyclin dependent kinases. Okay, these are the enzymes. So there is periodically there is the making and breaking of these cyclin CDKs. Jैसे we have different CDKs, cyclin A, B, all that things are there. That is not the part of your syllabus, but if by chance you will get these two words, they are related to your checkpoints. Don't get confused. Okay? So when there is a G2 to M transition, right, it will check that DNA replication is proper or not, cell size is proper or not, DNA is damaged or not, right? So it will take care of cell size, it will take care of DNA replication, it will take care of DNA damage. So if anything is there, it will not let that cell to divide to proliferate further. It will not let that cell to divide and to proliferate further. This is what you need to remember. Okay, this is what you need to remember. Right, bache? Right, bache? Are you sure? Are you sure? Yes. And when a cell, right, you know about PMAT, right? You know about PMAT. So when there is the metaphase to anaphase transition, and in that case we need APCs, that is your anaphase promoting complex. This is your anaphase promoting complex. Okay, what's that? It is your anaphase promoting complex. That is all about the checkpoints. Understood? That is all about the checkpoints. Understood? Now come to this. Dekho, it's PMAT, prophase, metaphase, anaphase, telophase. If you will understand the basics, you can answer all the questions. You can answer all the questions, people, all the questions. Let me tell you, in prophase, in general, the condensation of the chromatin material will start. Chromosomes, they will become compact. In the metaphase, the chromosome, they will arrange itself at the equatorial plate. In the anaphase, that sister chromatids will break, they will separate and the centromere split. And telophase, it is just the reverse of prophase. This is something which is common in all. Right? This is something which is common in all. 1 second, Rajanya. Please repeat APC. APC is nothing but the anaphase promoting complex. So for transition from metaphase to anaphase, right, bache, anaphase promoting complex is required. Again, they are the proteins. Okay, again they are the proteins. Okay, bache. So prophase, metaphase, anaphase and telophase. I told you these are the basics. Prophase mein condensation of genetic material will start, nuclear membrane will disintegrate, centrioles will move to the opposite pole. In the case of metaphase, everything will arrange, chromosomes will arrange itself at the equatorial plate. Anaphase mein that separation, telophase is the reverse of prophase. This is something very important. This is something very important. This is the basic. This is the basic. Guys, are you facing the issue of lagging? Is it so? Is it so? But checkpoints, I won't say that it is the least important topic, but if you will see previous year questions, you will get that checkpoints ka part. You will get that checkpoints ka part. So this part clear? Is the class lagging? I don't think so. I don't think so. Lagging? I think few students are facing the lag, otherwise there is no lag. Dekho, today I really don't want to end this class. Right? I don't want to end this class. Yesterday I ended it because of I just deferred it because of my bad health, but today I don't want to. Today I don't want to. And I won't say that today I'm perfectly fine. Yeah, some issue is there, but theek hai, we'll manage. Okay? So it is a two-way process. Whenever I'll feel down, you are going to give me the energy. Whenever you people will feel down, I'll give you the energy. Okay? Done? It should not happen because the internet speed from my side is quite good. Okay, so this part clear. So this is the basics in the prophase, metaphase, anaphase, telophase. So if you will understand that, okay, and then the prophase of the meiosis one, trust me, after that this topic is very, very, very easy. Let me tell you one thing. There you go. Before starting this topic, let me tell you something, and that something is related to the centriole. That something is related to the centriole. Okay? So what is going to happen first? Centriole is what? It's a non-membrane bound organelle. Means it is not covered with any membrane. What's that? It's a non-membrane bound organelle. It's a non-membrane bound organelle. It is not covered with any membrane. And it is present in animal cell. Please listen to me very carefully. I am answering the most asked question. It is present in animal cell. Now in the animal cell, two centrioles. You remember centrioles? You remember centrioles, they are having 9 plus 0 arrangement. What is the meaning of 9 plus 0 arrangement? Just read the chapter cell, you will get the answer of it. Just ciliate flagella 9 plus 2 arrangement, basal body centriole, it is having the 9 plus 0 arrangement. It is having the 9 plus 0 arrangement. Is it so? It is having the 9 plus 0 arrangement. So it is the arrangement of microtubules. The centriole is also made up of microtubules. So two centrioles, they are perpendicular to each other. Right? Two centrioles, they are what? They are perpendicular to each other. Plus, they are covered with the pericentriolar material. Means they are surrounded with this amorphous pericentriolar material. So what do we have here, people? We are having two centrioles. That two centrioles are perpendicular to each other. They are covered with what? They are covered with the pericentriolar material. Clear, bache? They are covered with the pericentriolar material. Okay? So when this is the structure, we call it as, this structure is known as centrosome or cell center or cell sphere. Okay? Or cell sphere. Right? So it is the centrosome, cell center, cell sphere. It is the centrosome, cell center, cell sphere. Repeat it with me. Centrosome, cell center, cell sphere. Centrosome, cell center, cell sphere. ठीक है बच्चे? ठीक है बच्चे? This structure. So now this structure is absent in the plant cell. We know that this structure, it helps in this thing. How many of you remember this? Just say this is the centriole. From this, this is the centrosome. Some rays will radiate like this. Clear, bache? Then they will form some fiber. Right? They will form some fiber, and that's the thing. I think you all are aware of it. This is how the spindle apparatus will form. So it is something which will be there in the animal cell, because animal cell is having the centriole, right? So from the centriole, these aster rays they radiate. Okay, bache, these are the astral rays. Okay, you know that these are the spindle fibers. So here you will see some continuous, some discontinuous fibers. Let's not enter in that depth. Okay, this is what we have. Now because in animal cell you will see the aster formation. So in the animal cell, mitosis is amphiastral. I'll tell you why I started this topic first. Let me explain it for 5 minutes. You will get the answer of your question. So animal cell mein mitosis is amphiastral. Amphiastral because of this aster formation. Aster mane star. That's the word. Now many students, they ask me that if centrioles are absent in the plant cell, then how in the plant cell spindle apparatus will form? How many of you want me to answer this question? Let me ask my team if it is actually lagging or not. Okay? Let me ask my team. I don't think that it's lagging. No lag na? Let me ask them. Okay. So now how many of you want me to answer this question? That whether in a plant cell, then how the spindle apparatus formation is there? Hana, it's very simple, people. It's very simple. Dekho, always remember, when you talk about the centrosome, it is actually the pericentriolar material. Which helps in nucleation. Means it is the pericentriolar material which helps in polymerization of microtubules. I have seen, right, many students, they don't know about it. It is the pericentriolar material actually, right? It is the pericentriolar material actually that will help in the polymerization. Okay? That will help in the polymerization of the microtubules. Okay? That will help in the polymerization of microtubules. Okay? Pericentriolar material means centrioles are covered with a kind of material. ठीक है, pericentriolar, the amorphous one. So it is helping in the polymerization of microtubules, ठीक है, means it will form these spindle apparatus. And what's the role of these centriole? They will, right, they will provide it a proper orientation, right? Basically these centrioles will make sure that it is properly oriented, right? They will just position it properly. They will just position it properly. Now in the case of plant cell, they don't have centriole, but they have this pericentriolar material. They have this pericentriolar material. So due to which even the spindle fibers they will form, but they will be disoriented. They will not be proper, they will just be disoriented like this. Like this, but yeah, they will form. That's why in the plant cell you see polyploidy is very common. That's why in the plant cell you see polyploidy is very common, and that is why in the plant cell we say mitosis is. That is why in plant cell we say mitosis is ancestral. Plant cell mein mitosis is ancestral. Any doubt here? Any doubt here? Yes, people. Any doubt here? Okay, so now explain it to me in the chat section that what is the meaning of equational division. Mitosis is equational division. Mitosis is equational division. Right? Why do we say that it is the equational division? Why do we say it is the equational division? So I hope this part is clear to you all, right? We are taking the NCERT also, so this 2C, 4C point should be clear to you people. Okay, and now answer this question, then we will go ahead. How do plants and animals continue to grow all their lives? Right, you know that in the case of animals, growth is up to a certain point only. When there is the cell division, obviously they will increase number, and in the case of plant cell there is the meristematic tissue which will keep on dividing throughout the life. That's why they will keep growing. Okay, you know that. Okay, so do you think all cells continue to divide in all plant and animals? Of course not. Of course not. When in the plant cell they will acquire a permanent function. When they are differentiated, when they are mature, they will acquire a permanent function, then they will not divide. Okay, and in animal cells also story is same, but in plant cell, meristematic cells, they will keep on dividing. Then can you tell the name and the location of tissue having cells that divide all their life in higher plants? Meristematic tissue. So apical meristematic tissue, shoot apical meristematic tissue, root apical meristematic tissue. Am I right? Am I right? And then do animals have similar meristematic tissue? No, they don't have, but they have the stem cells. They have the stem cells. Understood? Chaliye. So quiescent stage done. Okay, so now we will be discussing the mitosis. I told you that mitosis is the equational division. I have answered all the questions that students generally ask. So if you still have doubt, you are free to ask. Equational division. What is the meaning of equational division? I have seen students explaining it like this, that equational division means the parent cell is diploid and the daughter cell is also diploid. Right, you are right. You are right. If it is the equational division, if parent cell is diploid, the daughter cells will also be diploid, but that is not the actual meaning. Very good, sweetie. Very good, sweetie. So sweetie is giving the answer, so everyone clap for her. Quick. I want to see that. Yes, I want to see that emoticons in the chat section here. Quick. Definitely. So you all need to answer the questions. So no doubt, when it is the equational division, if parent cell is having the diploid number of chromosomes, the daughter cells are also going to have the same. But the meaning of equational division is कि if parent cell are having whatever number of chromosomes parent cells have, daughter cells are also going to have same number of chromosomes. Just if parent cell is having 10 chromosomes, daughter cells will have 10. If parent cell is having 5 chromosomes, daughter cell will have 5 chromosomes also. This is the meaning of equational division, that daughter cells will have same number of chromosomes like the parent cell. This is the one thing. Secondly, bache, mitosis. Where this mitosis is going to occur? Is it only be there in the diploid cell? The answer is no. The answer is no. In our body, we have the somatic cell, our normal body cell. We have the germ cells also. You know na, germ cells, which are the gamete forming cell. They are the cells that will form gamete. So they are what? They are the gamete forming cells. But that doesn't mean they will always undergo meiosis, that is a reductional division. In germ cells, when they need to proliferate, in germ cells, when they need to proliferate, right, what will happen? Mitosis will occur. So mitosis is a division which is very important for increasing the cell number, and it will also take place in the haploid cells. Let me tell you how. You know the plant cells? You know the plants. In the case of plants, we talk about the algae, we talk about the bryophytes, pteridophytes, gymnosperms and angiosperm. So algae is having haplontic life cycle. You remember? Haplontic life cycle is there. Bryophytes or pteridophytes mein haplodiplontic life cycle is there. Guys, are you enjoying the class? We are relating everything. Now you are not preparing for, now you are not studying class 11 zoology, you are not studying class 12 zoology or the botany. You are reading the entire, you are learning the entire biology. Right, you are learning the entire biology. So when you are learning the entire biology, it means we should know how to relate all the topics together. And when you understand this thing, trust me, then you know the concept. Okay, then you know the concept. So what am I saying? I am saying that bache, in the algae, haplontic life cycle, in the bryo and pterido, haplodiplontic life cycle is there. So I will take the example of algae. Haplontic life cycle means main plant body, that thallus, that thalloid, right, that thalloid, it is haploid. Right, main plant body is haploid. So main plant body, because it is already haploid, so when they need to form the gametes, they will go for mitosis. So mitosis will form the gamete here in such case. Mitosis is going to form the gametes here in such case, because plant body is already haploid. Because it is haploid, it will proliferate, right, no doubt, by mitosis only. Plus when it has to form the gametes, that gametes will also form due to mitosis. So mitosis will also occur in the haploid cell. Another example is drone. You know na, male honeybee, okay, which grows parthenogenetically. So this drone also, right, for gamete formation will go for the mitosis. So that is what you need to remember. So the same examples are given here in the NCERT also. Jaise dekho, male honeybee, mitosis is there. Plants mein mitotic division is there in haploid and diploid cells. Okay. So answer this question quickly. Right, dekho, onion root tip cells are the best to study the mitosis. Right, they are the best to study the mitosis. So you have to tell me that if onion root tip cell, if they have 16 chromosomes, this question can be asked in paper, so please mark it. Right, if they have 16 chromosomes, can you tell how many chromosomes will be there in G1 phase, after S phase, after M phase? Can you just tell me the DNA? Can you just tell me the DNA composition in all the phases? I'm asking about all the phases, not just the one phase. I'm asking about all the phases. I'm asking about all the phases. Number of chromosomes. We are talking about number of chromosomes. Hanji, 16, obviously some people are saying 32. Nahi bhai, number of chromosome will always remain same. So it will be 16. Now tell me about the DNA amount. If here, right, DNA is 2C, what will be the amount of DNA here? Answer it to me. What will be the amount of DNA, people? Guys, what will be the amount of DNA? So G1 mein it will be 2C, S phase mein it will become 4C, G2 mein, okay, sorry, here it should be G2, just a minute. Here also it will be 4C, okay? And after M phase, M is the mitosis here. After M phase, in the daughter cells also you will see 16 chromosome and DNA amount will be 2C. DNA amount will be 2C. So that type of questions should be clear in your mind. Okay, now come to prophase. Okay, now come to prophase. Okay, prophase of mitosis. As I said, nuclear membrane will start disintegrating. These centrioles, they will start moving to the opposite pole. These centrioles, these centrosome, they will form some fibers. That fibers will push each other. Right now they will start moving to the opposite poles because they have to form the spindle apparatus. All that organelles which are present, even they will dissolve. Like they will not be clearly visible there. Plus bache, slowly because compaction is taking place, chromosomes, they are getting condensed. Right, they are releasing water, they are getting condensed. They will acquire that shape now because it is taking place after S phase. So now these chromosomes are having two arms. You will see the sister chromatids there. Are you getting it? So prophase, the very first stage of karyokinesis, right, after S and G2 phase. Right. So here dekho, in the S and G2 phase, they are saying new DNA molecules formed are not distinct, means they are not separate but intertwined, means they are all like this, means this is not separate, it is something like that, something like that. Clear bache? So prophase is marked by initiation of condensation of chromatin material. So be it the mitosis or the meiosis, this is something which is going to happen. The only difference is in the prophase of meiosis 1 you will see pairing and all. Okay, but here it is not like that. So chromosomal material, it will be untangled during the process of chromatin condensation. Means now, because everything was so intertwined, now in prophase condensation has started, all the chromosomes, they will become distinct. They will acquire a discrete entity. They will acquire a separate entity. Are you getting my point? So now here they have used the word centrosome. So centrosome, which had undergone duplication during S phase, now begins to move towards the opposite poles. Right, towards the opposite poles. That's what you need to keep in your mind. So this is what you can focus here, bache, that chromosomal material, they condense, they will form compact mitotic chromosomes. Right. So chromosomes are seen to be composed of two chromatids attached to one centromere. Okay, so centrosome, so do not confuse the word centromere and centrosome. Are you clear with the difference of these two words? What is centromere, what is centrosome? If it is a yes, do let me know in the chat section, people. If it is a yes, do let me know in the chat section. Do you know the difference in these two words? And guys, hit the like button and make sure you subscribe to our channel. Do you know? Yes, it should be this. Yes ma'am, we know that. Okay, so this is going to happen when it is the prophase. Done bache? Done. So this is early prophase, where nuclear membrane had started disintegrating and chromosomes has condensation. So at this point of time, the diagrams of NCERT, they are important. So you should know which one is early, which one is late. So in late prophase, all these nuclear membrane is disintegrated and you can see the chromosomes, they are so condensed. Okay, and these centrioles, they will move to these centrosomes, they will move to the opposite poles, bache, and they will start forming the mitotic apparatus. So this diagram is something very important. Your MCQ can come from this part. So it is showing transition to metaphase. It is not metaphase, it is showing transition to metaphase. So you can see this is your centrosome. And it is animal cell. Why is it animal cell? Because we can see the ester rays here. So it is amphiestral. So see, chromosomes are not aligned at the equatorial plate. So it is the transition to metaphase. It is not the metaphase. Okay, it is not the metaphase. Okay, so prophase clear. Now when you talk about the metaphase, what is going to happen in the metaphase? It is the best phase to study the morphology. It is the best phase to study the morphology of these chromosomes. Remember bache, this is your chromosome, the two sister chromatids. Now the centromere, right, this center part is centromere. It is the primary transition. The centromere, from both the sides, it is covered with the disc shaped protein. And we call that proteins as kinetochore. ठीक है, from both the sides it is covered with a disc shaped protein and we call this disc shaped protein as kinetochore. बच्चे centromere is this part, it is the primary construction, ठीक है, the two sister chromatids are attached to it, so from both the sides it is covered with the disc shaped proteins, we are calling it as kinetochore. So to this kinetochore the spindle fibers they join, ठीक है, so spindle fibers they are made up of microtubules, this can also be asked as a question, they are made up of microtubules, so they are not going to directly attach to the chromosome, they will attach to the kinetochore, ठीक है, they will attach to what, they will attach to the kinetochore. So when you talk about the metaphase, all the chromosome they will arrange itself at the equatorial plate, this plate is also known as metaphasic plate. Understood? Understood. So it is the phase where you can even calculate the chromosome number, you can check their size, shape, so it is the best phase to study the morphology. It is the best phase to study the morphology. Okay. Now let's talk about the anaphase from this metaphase. What do you think what should happen? Anaphase mein they will start moving to the opposite pole. So don't you think the don't you think that centromere will split? Jaise we are talking about the anaphase of mitosis. So what is going to happen here? Imagine now the centromere will split. Centromere will split. So if centromere split, sister chromatid separates, centromere split or sister chromatids they separate. This is what anaphase is. Now there is a line in NCERT where they talk about, right, there is a line in NCERT where they talk about the arms are trailing behind, ठीक है, जैसे imagine you are a baby, ठीक है, you are 5 years old or 4 years old baby, wow, at that time life was beautiful, है ना, it was wonderful. So you are crying outside a toy shop, mama, I want the toy, right, I need that, and your mama is just like me, no, you don't need that. So your mama will hold you from this waist and will be like, chal, let's go, है ना, your mama is going to take you like this and you are like, no, I want toy, it's something like that, okay? It is something like that. So imagine your mama is just pulling you and you are just looking at the toy shop and you're like, mama. So same is the case here, now because centromere will split, right, because this depolymerization of these fibers will occur and it will be pulled towards the poles and arms will trail behind like this, something like that. Okay, so arms will trail behind like this. So this is the meaning of arms are trailing behind. Okay, arms are trailing behind. Clear? Right. So very simple example. If it is a metacentric chromosome, metacentric you know na, both the arms are equal, then obviously when it is trailing behind you will see a V-shaped structure. If it is submetacentric you will see L-shaped structure. So this is important question, ठीक है जी, this is an important question. Clear बच्चे, ठीक है, so this is an important question. Clear. So chromosomes will appear either V-shaped, L-shaped, J-shaped or I-shaped. ठीक है, so I gave you the example of V-shaped, it can be L-shaped, it can be J-shaped, it can be I-shaped. So you people need to tell me, okay, that which chromosome will have L-shape, J-shape or I-shape. That will be your homework, ठीक है, in the comment section. This is what I want, and you people are very bad, you don't put comments. Like let's say if 600 plus students are watching me, I'll get comments from hardly 50 students. Yeah, it will take your 10 seconds, ठीक है, you can type something good if you actually like this class, okay, ठीक है. So that's what you need to understand. This part clear? So a metaphase, it's the best to study the morphology, and you know the karyotypes are formed in metaphase stage. Ideograms are formed in metaphase stage. Karyotypes are formed in metaphase stage. Ideograms are formed in metaphase stage. So anaphase clear. So telophase is just, so ye dekho, look at the diagram. So ye dekho, in the metaphase that is going to happen, spindle fiber attached to kinetochore, chromosomes are moved to spindle equator, they will arrange like this. So anaphase mein, you know that what is going to happen, metaphase plate is split simultaneously, two daughter chromatids, right, now referred to as the daughter chromosomes. So when they will get separated from each other, now you will not use the word chromatid, these are the chromosomes, these are the chromosomes. So obviously right bache, at the end, see, this is the anaphase, this is the telophase. So telophase is the final stage of karyokinesis, right, and the chromosomes that have reached their respective poles, they will decondense, because in interphase they again will be in the thread form, untangled one, intertwined, okay, intertwined like this, okay, like this. So it is the final stage of karyokinesis. The chromosome that have reached their respective poles, they will decondense, they will lose their individuality. It is just opposite of the prophase. Your telophase is opposite of prophase, right? So for your quick revision, you should not miss these points, okay? You should not miss these points. So nucleolus, Golgi complex, endoplasmic complex reform, nuclear envelope will develop around chromosome. It will cluster at each pole forming two daughter nuclei. So chromosome cluster at opposite spindle poles, they will lose their identity. Now they are again not the compact one, right? They are not again the compact one, they will again be loose. ठीक है बच्चे? ठीक है बच्चे? Understood? Understood? And in the prophase, you know it very well. Right, at the end of prophase, you can see the microscope under the microscope, you can see the chromosomes under the microscope, but you will not see Golgi complex, ER, nucleolus. Understood? Doubt? Doubt. Now, after that, cytokinesis is there, means cytoplasmic division. Now, I'll tell you one story. Dekho bachche, in animal cell, do we have the cell wall? In animal cell, do we have the cell wall? In animal cell, do we have the cell wall? Do we have the cell wall? Of course not. So, bache, when there is the cytokinesis in the animal cell, it will be because of cleavage furrow. Ye dekho. Right, you will see a cleavage furrow will form. What will form? A cleavage furrow will form. What will form? A cleavage furrow will form. And this is formed due to actin filament. So if you will see, when there is the animal cell, in animal cell, your spindle apparatus will disappear, will disassemble by end of anaphase. Yeah, you can say that in anaphase itself, you will see ki spindle fiber, it is, getting disappeared. Okay, so after telophase, when there is the cytokinesis, what's going to happen? The cleavage furrow will form, and it is due to the actin filament, and you will see it is from periphery towards the center, so it will form centripetally. Okay, centripetally, from periphery towards the center, it is important point. Now in the plant cell, do you know that spindle apparatus stay even in telophase? Even in telophase you will see spindle apparatus. Do you know that? Now you know that in the plant cell there is a cell wall. So what is going to happen? Just say, let's say this is the telophase. Okay, let's say this is the telophase. Okay, so telophase में भी you will see that spindle fibers are there, right? Spindle fibers are there. But बच्चे, Golgi complex will also form here. So they will form certain vesicles which will come in center. These vesicles, they will fuse. So they form a structure known as cell plate, right? So in the case of plant cell plate will form in the center. And who is forming that cell plate? Golgi apparatus. Golgi apparatus is the one that forms the cell wall. So cell plate will form, Golgi apparatus is going to form that, because Golgi apparatus will form some vesicles which will come here in center, they will fuse. Now from the center, they will move towards the periphery. Right, from the center it will grow towards the periphery. This is how the cell plate will further grow, and it is growing centrifugally, right? Okay, centrifugally. So this cell plate will later form middle lamella of cell. Do you remember middle lamella? Right, it will later form middle lamella of the plant cell. right? So all statements are important. Now let's come to the meiosis directly without wasting time. And meiosis mein you need to understand the prophase one, and then everything is very easy. It is smooth. It is smooth. So in next 15 minutes, are you ready to finish the chapter? In next 15 minutes, are you ready to finish the chapter? Hanji, energy people, energy, 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 energy. And today I want you people to write the comment as well if you enjoyed the session. Ki Hanji madam, we enjoyed the session and this is our comment. So are you ready to finish the chapter in next 15 minutes? And make sure guys, make sure you can inform this thing to, you can tell this thing to your juniors as well, right? So this time there will be 3 batches in the English language, Nimbus batches for class 11, right? Nexus batches for class 12 and Phoenix batches for class 12, that too in English for all the NEET aspirants, bache. And this team is going to teach you, you know that we know how to provide the quality content. So in the description box there is a link and this is the coupon code that you can use and you will get the lightning deal, bache. Okay, you will get the lightning deal. So batch is going to start on 17th of April. So make sure you are the part of that batch. And now let's come to the meiosis one. Okay, let's come to the meiosis. So what is it? It is the reductional division. What is it? It is the reductional division because it is the one that will reduce the chromosome number to half. Now it occurs in 2 rounds. We all know it, right? It occurs in 2 rounds. We all know it. So meiosis, reductional division. So let's say you are having a diploid cell. It will divide. You will have 2 haploid cells. That haploid cells will further divide, right? You will get another 4 haploid cells. So in mitosis you were getting 2 daughter cells. In meiosis you are getting 4 daughter cells. So it is the diploid cell. It is the diploid cell. Okay? So firstly there will be the meiosis 1, then there is the meiosis 2. So you must know that meiosis 2 is compared with the mitosis. Meiosis 2 is compared with the mitosis. So meiosis 2 is compared with mitosis. Why? Because dekho, in meiosis 2 this is the parent cell having n number of chromosome, right? Even the daughter cells are also having n number of chromosome. That is why meiosis 2 is compared with the mitosis. So when it comes to the meiosis, it is the one that will reduce the chromosome number to half, and we need it for the gamete formation, right? It is required for the gamete formation. So in the meiosis 2 rounds of, okay, 2 rounds of nuclear division. This is a PYQ. 2 rounds of nuclear division and cytoplasmic division will be there. Okay, 2 rounds of nuclear division and cytoplasmic division occur, but there is only one round of DNA replication. There is only one round of DNA replication. There is only one round of DNA replication. Is that clear? Is that clear? So interphase, then, right, sorry, let me write it like this. Interphase, please pay attention, then meiosis 1, then meiosis 2. In between there is a small gap that is known as interkinesis. Understood? Understood? That's something important. So interphase having G1, having S, having G2. Then there is the meiosis 1. There is the meiosis 1 having prophase 1, having metaphase 1, having anaphase 1, having telophase 1. You know na, trick is PMAT. Then interkinesis, after that prophase 2, metaphase 2, anaphase 2, telophase 2. Okay? So in between this M1 and M2 there is a small gap, that is interkinesis. So no DNA replication will occur here. DNA replication will only be there in the interphase. That's why we say 2 rounds of nuclear and cytoplasmic division, but only 1 round of DNA replication, that too in the S phase. So any doubt people? Any doubt people? Hanji, any doubt? Chalo. Acha, if I'll ask you something, will you do it? Are you going to do that? If I'll ask you something, are you going to do that? Tell me, are you going to do that if I'll ask you something? सच्ची बताओ, be honest. Be honest. Be honest, be honest. No lag, बच्चे, no lag. Be honest. Pakka, sure. If you are ready, then send me the thumbs up. Send me the thumbs up. कि ठीक है मैम, we'll do it. We'll do it. Pakka. अच्छा. What if I'll say even now I forgot that what I was about to ask. Now I just forgot what I was about to ask. I am sorry. Now I just forgot here. I was about to ask something. I got it. See, now I'll mention you the amount of DNA. That's what you need to keep in your mind. Later on I'll explain it. As of now, you just note down. Here, this is the diploid cell. This diploid cell is right after G1S and G2. So here you will see, because during division the DNA content get doubled. Here it will be 4C. This cell will be having 2C and 2C, and this cell will be having C, C, C and C. This is the amount of DNA. This is the amount of DNA. This is what I was about to tell you. And now after explaining this meiosis topic to you, then I'll tell you what you have to do. ठीक है जी? ठीक है जी, then I'll tell you. Okay, so we have prophase 1, metaphase 1, anaphase 1, telophase 1. This is what we have. So now when you talk about the prophase 1, it is having 5 sub-stages. It is having 5 sub-stages. You can check any previous year paper. You will definitely get question from this part. Right? It is NTA's favorite part. It is NTA's favorite part. So everyone, please repeat LZPDD. LZPDD. LZPDD, or little Zara, please do not dance. Right? Little Zara, please do not dance. It's LZPDD or little Zara, please do not dance. LZPDD or little Zara, please do not dance. So it means L stands for leptotene. It is also known as leptonema. Nema mane that thread. Zygotine, also known as zygonema. Pachytene, also known as pachinema. Diplotene, diplonema. And then finally the diakinesis. These are the 5 substages. These are the 5 substages. So it's LZPDD. LZPDD. Little Zara, please do not dance. Little Zara, please do not dance. It is LZPDD. Everyone repeat it. LZPDD. Little Zara, please do not dance. This is the 5 substages. And if I talk about the prophase 1, it is the longest prophase of the it is the longest phase of this particular division. Overall, prophase 1 is the longest phase. Okay? And here, this pachytene, if you compare it with zygotene and leptotene, this one will be the longest substage. Okay? So now let's understand that. One thing is for sure, because it is a prophase, then condensation of chromatin material will definitely be there. So let's start, people, right? Let's start with the word leptotene, also known as leptonema. Let's start with the word leptotene, also known as leptonema, right? What you need to start? You need to start with the word leptotene, also known as leptonema. Obviously, the condensation of the genetic material will be there and the nuclear membrane will start disintegrating. That's all. Okay? Come to the NCRT. Come to the NCRT. Okay? So see this. Meiosis, 2 sequential cycles of nuclear and cytoplasmic division. 2 times nuclear division, cytoplasmic division will be there, but it is a single cycle of DNA replication. Anyone having any doubt? I think there is no doubt. Meiosis 1 is initiated after the parental chromosomes, they have replicated and they have produced sister chromatid, means when DNA is duplicated. Meiosis may, there will be the pairing, there will be the recombination. That's what we have to discuss. So see this, prophase 1, right? The first meiotic division, it is typically longer and more complex. Okay, so it is having 5 substages. So it is clearly mentioned, that during leptotene stage, the chromosomes, they become gradually, means slowly visible under the light microscope. So mark this thing. Leptothene में condensation of the chromatin material will be there, condensation of the chromatin material will be there, and chromosomes, they will become gradually visible under the light microscope. Please highlight this part. Please highlight this part. Chromosomes will become gradually visible under the light microscope because compaction has started, condensation has started. Okay? So this is what leptothene is. This is what leptothene is. Do I need to write it? Do I need to write it? I don't think so. Next is leptothene, then zygotine. Zygotine. So zygotine may one word answer. What will be there? Synapses. What will be there? Synapses. This is also known as syndesis. It is also known as zipping. It is also known as zipping. So this is going to happen in the zygotine. We know that we have pair of chromosomes. One chromosome is contributed by mother, another chromosome is contributed by father. So if we have 46 chromosomes, so 23 chromosomes are from mother, 23 chromosomes are from father. This is what we have. We all are aware of it. So now, because in this division, we have to reduce the chromosome number to half. Okay? We have to reduce the chromosome number to half, isn't it? We have to reduce the chromosome number to half when it is what? When it is the zygotine, right? You have to reduce the chromosome number to half. If we have 46 chromosomes, we have to divide it into 23. So that pairing should be there. So because if they will be paired, then we can separate them, right? If they will be paired, we can do what? We can separate them. That's what you need to keep in your mind. Okay? So in the zygotine, right, what is the meaning of synapses? Means there is the pairing. Okay, there is the pairing of homologous chromosome. There is the pairing of homologous chromosomes. Clear, bache? Pairing of homologous chromosome. How that pairing will occur? Very simple. That maternal homologous pairing, pairing of homologous chromosome. So maternal chromosome and paternal chromosome. Maternal chromosome and paternal chromosome. They'll be paired. They'll be paired. Okay, their pairing will be there. So the question here is ki what is the meaning of homologous chromosome? The chromosomes having same shape, size and centromere position. They are all same. Same shape, same size, same centromere position. Even they will have same genes. Do you know that? Even they will have same genes. Do you know that? Do you know that? They are having same genes. How can you say that? Dekho, it is the homologous pair of chromosome. This is the gene locus, mane position of gene. So here if you have the gene A, of course it is just the duplicated arm. Here it is also having A, and now here, you will also get the A gene. Allele can vary, but the gene will be same. Allele can vary, but the gene will be same. Remember, allele can vary, but the gene will be same. So the pairing will be occurring in the zygotine. So how that pairing will occur? Just imagine these are two homologous chromosomes. These are two homologous chromosomes. They are coming closer, right? We have to pair them. So in between them, a complex will form. We will call it as synaptonemal complex. It's a kind of glue. Just imagine, right? This is one chromosome. Okay, this is another chromosome. So they are paired. How are they paired? How are they paired? We are having a synaptonemal complex in between. Okay, a glue will be there, the synaptonemal complex will be there in between. Is that clear? Is that clear? Sure. Synaptonemal complex. So question can also come, right, that when will the synaptonemal complex form? So you should know this. So this is your synaptonemal complex which will form in between this homologous pair. What will be there? Synaptonemal complex. What will be there? Synaptonemal complex. Clear, bache? Arey. So it is nothing, it's a ribonucleoprotein complex. What's that? It's a ribonucleoprotein complex. Ribonucleoprotein complex. What's that, people? Ribonucleoprotein complex. So RNA is there, literal element is having the DNA, other proteins are there, so it will help in the zipping. So this is what zygotine is. So now let's move to the next sub stage, which is longer than the other two substages, that is packetine, in which what is going to happen? Crossing over will be there, right? Crossing over will be there. That crossing over is also known as recombination. It is also known as recombination. So it is an enzyme mediated process, and the enzyme recombinase will achieve it. What's that? It is an enzyme mediated process. It is an enzyme mediated process. So which enzyme is going to do that? Which enzyme is going to do that? It will be your recombinase. Now see people, देखो यहाँ पे it's very simple. It's very simple. These two are sister chromatids. These two are also the sister chromatids. And these two, they are what? They are non-sister chromatids because they are attached to different centromere. Okay? So this is the homologous pair of chromosome. These two are sister chromatids, these two are sister chromatids, and these two are non-sister chromatids. These two are non-sister chromatids, right? So whenever you talk about the crossing over, so the exchange of segment, okay? So the exchange of segment is in between non-sister. Language should be very clear here. It should be in between non-sister chromatids of homologous pair of chromosome. It will always be in non-sister chromatids of homologous pair of chromosomes. ठीक है? Non-sister chromatids of homologous pair of chromosomes. ठीक है? Clear? So that's what you need to remember. So the exchange of segment, you know that it's very important for the variation. So this one will exchange the segment with this one, right? So a part of this chromosome will get a part of this chromosome. So basically segments will be exchanged. So recombinase enzyme is going to do that. Which enzyme is going to do that? Which enzyme is going to do that? Yes, people. Just let's say this is one. This is another one. So that is how the exchange of segment will be there. That is how the exchange of segment will be there. So after it, what will you get? Like this, right? Like this. So a part, so their X segments will be exchanged. Okay? So this is what crossing over is. So before crossing over, let's say this is the site. This is the site at which crossing over will occur. Let's say people that this is the site. Let's say, just a minute, let's say this is the site at which crossing over will occur. Yeah, this is the site at which crossing over will occur. So these sites are known as you will see a recombination nodule here. What will you see? You will see a recombination nodule here. What will you see? A recombination nodule. So recombination nodule is a place at which the crossing over will occur. What is recombination nodule? A place at which the crossing over will occur. Everyone just repeat it. Recombination nodule is a place at which the crossing over will occur. Understood? 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 Recombination nodule. So from that recombination nodule, the crossing over will occur. Now after crossing over, what is going to happen? First of all, in packetine, crossing over will occur. But yeah, before that, see, when there is the pairing of homologous chromosome, understand this thing please, everyone. When there is the pairing of homologous chromosome, please look at it very carefully. When there is the pairing of homologous chromosome, right? They are paired. Okay, what are they? They are paired. Look at this, bache. They are paired. So obviously, can I call them as bivalent? Because they are a pair. Can I call them as bivalent? And they are having 4 arms in totality. So can I also use the word tetrad here? Of course I can. This is the bivalent and tetrad here. So this bivalent and tetrad, no doubt, in the zygotine when there is the pairing, it will form, but it will be very clearly visible in packetine. Okay? So in packetine, this bivalent, right? In packetine, this bivalent and tetrad, it will be clearly visible. it will be clearly visible. So bivalent is known as tetrad. Why? Because of these four arms. Because of these four arms. That's what you need to keep in your mind. Okay, that is what you need to keep in your mind. Clear bache? So recombination nodule will be clear. Okay, synaptonemal complex clear. Now let's move to the next substage, that is your diplotin. Right, that is your diplotin. So do revise this point again and again, that recombination between homologous chromosome is completed by the end of packet T. By the end of packet T. Okay? So leaving the chromosomes linked at the sites of crossing over. So when there is the crossing over, so at the point where crossing over will be at the point where crossing over will be there, no, so these sites will remain attached to each other. Okay, it will remain attached to each other. Okay, so the next stage is your diplotin. So diplotin mein what's going to happen? The dissolution of the synaptonemal complex. Right, so in the packetine, see, in the zygotine we were bringing them, we were pairing the homologous chromosome. Why? For the crossing over. Now after packetine, right, in packetine when crossing over is done, obviously these chromosomes will be like, okay, this is the site at which the exchange of segment is done, so it will stay joined to each other. Now you know that in the chromosomes, okay, now you know that in between what do we have? In between what do we have? We have the synaptonemal complex. We have the synaptonemal complex. So in diplotin, the synaptonemal complex will start dissolving, right, and the chromosomes, they will start moving away from each other. They will start getting separated from each other, but still they will not leave this point. They will stay connected to each other at the site of crossing over, right. So when synaptonemal complex will dissolve in the diplotin, and when synaptonemal complex will dissolve, right, the chromosomes, they will start moving away from each other. They will start getting separated, but still they will remain attached at the site of crossing over. So this is basically the X-shaped structure. We call it as chiasmata. It is the site of crossover, right. It is the X-shaped structure, chiasmata. It is the site of crossover, means it is the site where crossing over has been done. Okay, bache, it has been done. Right. Clear? So after this diplotin, next is the diakinesis. So now what's going to happen in diakinesis? Let's say this is one chromosome, this is another. Okay, this is one chromosome, let's say this is another. Okay, another chromosome. So this is the site at which crossing over has been done. Now in the diplotin, I told you dissolution of synaptonemal complex is there, and chromosomes will remain attached to each other at the site of crossover only. Now next is the diakinesis. So when it is the diakinesis, so in diakinesis there will be the terminalization of chiasmata. Terminalization of chiasmata means this chiasmata is at this point, it will move to the ends. Let me show you. This chiasmata will move to the ends like this. So this chiasmata at this point, so when it is the diakinesis, its terminalization will occur. Any doubt? So you know that in the diplotin, many eggs, they can stay for a longer period. Just even in humans, you know that, right? During embryonic development, the meiosis starts and it will arrest at diplotin. We call it as dictate arrest. Okay, so not all the oocytes will get a chance. To, you know, continue that division. So you will stay in that diplotin for many years. Just oocyte of some vertebrates, they can last for months or years in diplotin stage. Okay, so final is diakinesis. So when it is diakinesis, it also indicates transition to metaphase. Okay, so terminalization of chiasmata is there, chromosomes are fully condensed, meiotic spindle is also assembled in diakinesis, right? So by the end of diakinesis, your nucleolus disappear, your nuclear envelope breaks down, and it represents transition to metaphase. So this question can be asked in meiosis 1, which state represent transition to metaphase? I want the answer in the chat section, people. I want the answer in the chat section, quick. Let's finish it in next 5 minutes. Then let's move to the structural organization and then to the human physiology. Yes, exactly, diakinesis is there. Chalo, let me ask you one question. I don't know whether you have noticed it or not, and if you will understand it, the topic, the entire topic is over. ठीक है? Okay, let's start with a very simple story. We are taking the example of a cell. Let's take the example of a cell. This cell is having 2n is equals to 4. It is having how many chromosomes? 4 chromosomes are there. So after S phase we know that that will be the thing. After S phase we know that that is going to be the thing. Yes or no? After S phase, this is the thing that we will be having. Now, when it is the mitosis, so in the mitosis, when there is the metaphase, you know that what is going to happen. There is the center equatorial plate where all the chromosomes will align. Yes, where all the chromosomes will align. है ना? So we took the example of a cell which is having 4 chromosomes. So when it is the metaphase of mitosis, that is how it will be. Now bache, when it is the metaphase 1 of meiosis 1, so in the metaphase 1 of meiosis 1, you know what will be the story? In the metaphase 1 of meiosis 1, bache, there will be 2 metaphasic plates, right? There will be 2 metaphasic plates. Yes, people, what will be there? There will be 2 metaphasic plates. Okay, 2 metaphasic plates will be there. Okay, right? So 2 metaphasic plates, they will be there. Why? Because we know that in the prophase 1 the pairing of homologous chromosome was there. So now you have to separate this pair, right? You have to separate this pair. So after metaphase of meiosis 1, when there will be the anaphase 1 of meiosis 1, what do you think what's going to happen? In the anaphase, there is the separation of bivalent. This bivalent will get separated. Right, there is a separation of bivalent. There is no chance that centromere will split. There is no chance that sister chromatid will split. Right, bache? Okay. Then when it is the metaphase 1 of meiosis 2, what's going to happen in the metaphase 1 of meiosis 2? What is going to happen in the metaphase 1 of meiosis 2? See this, in the metaphase 1 of meiosis 2, because we are taking the example of the same cell having 4 chromosomes. Okay, this will be the scenario. Okay, this will be the scenario. Are you getting my point? So when it is the mitosis, all the 4 chromosomes will be arranged like this. When it is the meiosis 1, metaphase 1, the bivalents will form, and in anaphase 1 bivalent will get separated. And now when it is the meiosis 2 metaphase, now that haploid cell, its sister chromatids will separate. So when it is the anaphase 2, right, when it is the anaphase 2, then in the anaphase 2 there is the splitting like this. There is the splitting of centromere and sister chromatids separate. That's it, that's all. This is all about this topic. I'll again explain it. Dekho. This is after interphase. This cell is after interphase, right? Because it is diade. 2 arms are there, na? It is a diade. So after meiosis 1, what is going to happen? You can see this. After meiosis 1, what is going to happen? You can see this. After meiosis 1, this will be the case. This will be the case. So we started the story. Now DNA amount is doubled here. It will be 4C. It will be 4C. DNA amount is doubled here. It will be 4C. So after meiosis 1, the chromosome, they separate, right? Chromosome number, it become half now. So see, 4 chromosomes here, 2 chromosomes here. So even the DNA content will also be half, 2C and 2C. Now why the meiosis 2 is required? Why that meiosis 2 is required? Obviously because we have to, in the gametes we want the genetic material, the DNA content to be C. Okay? So what will be the thing here? See, now the sister chromatids will split. So when these sister chromatids, they will split, that will be the condition. So guys, in the marathon, right, itself, I'm explaining the things in that detail. If still you cannot get it, I cannot help you guys. I cannot help you. That's the point. Done, bache? Done, bache? Done, bache? Okay, so meiosis 1, then comes the meiosis 2. So that's the point. So 4C, 2C, 2C, C, 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 C. Now you got it. Now you got it. So obviously, let's say there is a sperm, C, ovum, C. So when they will fuse in that cell, the diploid content will be 2C in the zygote. That's the point. So this is all about the, prophase, metaphase 1. And see the prophase 1, the synapses, metaphase mein 2 equatorial plate, anaphase mein bivalent is getting separated, telophase mein number of chromosome is half, right bache, right bache. And then after that, see, this is going to happen in prophase 2, metaphase 2, anaphase 2, and finally the telophase 2. So just read it once from NCERT. So with it, we are done with the chapter. So can we start the animal tissue now? Can we start the animal tissue now? Actually, my original plan was कि after cell cycle and cell division, let's discuss biomolecules, then structural organization, then human physiology, because you people are demanding human physiology. So let's start with that. Okay, let's start with that. So are you people ready? Hanji. Are you people ready? Sure? Are you sure? Okay, so animal tissue, that's what we need to start. So from this chapter, mainly questions, right? They go. I will provide you the PDF, of course, right? And mainly you know that questions are there from the examples. So because it is a, they go, if you want to discuss this chapter in detail, okay, you can check my detailed lecture of this chapter on this channel, right? Otherwise, let's stick to NCERT. What say people? Let's stick to NCERT. So here you are going to talk about the tissue basically. Okay? So let's have a break of 2 to 3 minutes. Let's have a break of 2 to 3 minutes. Okay, so 2 to 3 minutes will be fine according to me. So you can ask your doubts, bache, and then let's start this chapter. Okay, then let's start this chapter. Quick. Doubt? Do you have any doubt? We have to be specific here in this chapter, but if you want something extra, you can watch my detailed lecture as well. Plant physiology will be there in the botany class. From this chapter, Rohit, according to me, 2 questions can come. It can, because now 6 chapters are deleted, it can be 3, but I believe 2 questions will be there. And this chapter is related, na? It is related to human physiology. ठीक है. This chapter is also related to human physiology, so obviously we have to relate to, and after this we will be starting breathing and exchange of gases. So do you want any break, or can we just start the chapter directly? If you want break, I will give you a break of 10 minutes, right? 10 minutes means 10 minutes, and then after finishing this chapter there will be a proper lunch break. Okay, so do you want break or not? Tell me honestly, because I want you people to concentrate. देखो, today's lecture is for, I think रुक जाओ, 13 or 13 to 15 questions from this part. Means somewhere we are, right? Somewhere we are targeting 30 questions or above 30 questions. So even if I will take 30 questions ka number, so 120 marks. So today you are studying for 120 marks, and trust me, we will cover everything here. Right? So after the class, if you will just revise your short notes, right, just revise NCERT, even the summary, that will be more than sufficient for you. Break needed for 10 minutes, for 10 minutes. Wow, no breaks, break, no break. ठीक है, it's 12:10 to 12:20 p.m. This time I will be punctual, even I will be punctual. ठीक है, so it's washroom break. That will be your washroom break, and that is actually for 10 minutes. So take a stroll, relax your eyes, then let's discuss it. I'm here only, I'm not going anywhere, don't worry. Hello people. Oh my God, you have captured my heart in ways I never thought possible. Oh my God, seriously, Deva, seriously, I believe that this line is for me, not for someone else, because it is, if it is for me, then it is very good. If it is not for me, then it is very bad, right? It is very bad. Trust me. You don't have time to waste, people. You don't have time to waste. You don't have time for in Hindi we use one word, ashiki. You don't have time for ashiki. Okay, so you better study properly. So, ma'am, in the cell shape, bache, if it is a metacentric chromosome having equal arms, so when arms will trail behind, so it will acquire a V shape. So when it is the submetacentric, it will acquire the L shape. Rest you need to answer. And yeah, I was just checking out few questions. Some students were saying, is the centriole, centrosome same? People, I already explained everything in detail. What were you doing at that time? So if it is, देखो, centriole 9+2 arrangement. When two centrioles, they are perpendicular to each other plus covered with pericentriolar material, then you will call it a centrosome. Any other doubt? Ma'am, acha, Prashanti, ma'am, punctuality. It's 12:22. Hello, I started the class at 12:20. Chill. Prashanti ji, chill. Oh my God, Diya, you better start teaching properly without taking breaks. But I will take breaks like this. If you don't like the session, you are allowed to leave the session. Ah, okay, okay. Be little humble, be little gentle. So Divya, bye bye, bye bye. You can leave the class. Tada. But chiasmata will form after the crossing over, right? The X-shaped structure. Right after the crossing over. Yes, yes, recombination nodule will form before the crossing over, where recombination will be done. Done, bache? Done. Chalo, let's start. Obviously, yeah, one should have the swag. You better start teaching without taking breaks. Seriously? Oh my God. No chance. I am your teacher, mentor. I always say that I'm just like your elder sister. That doesn't mean you will type anything. And I'll be like, oh, I'm so sorry, I took the break. Oh, why? It was the washroom break. Even I have the excretory system. Even I need to urinate. Simple. Hai na? Simple. So biology students, yeah, at least you should understand it. You should understand it. Don't take break. Even if you are feeling like, even if you have the urge to urinate, just hold it. You are not allowed to take the break, because this is your duty to teach us. And when we started our lectures from the starting, when we teach in details, like in the different series, games of NEET, conquer back, then you people are not going to attend the lectures. But at the end, in the marathon, teachers need to deliver everything. They need to provide you everything. Wow. And you will be like, we are not grateful at all. Why should we? Hai na? Chalo, leave it. So structural organization in animals. So here we will be talking about the tissue first. Dekho, it's very simple. When you talk about the living organism, we all are made up of cell. Cell is the basic unit of life. Okay? Without the level of cell, there is no independent existence of life. It is the minimum, that minimum thing when it comes to life is cell. What's that? It is the cell. What's that? What's that? It is the cell. Now cells together, they will form tissue. Right? Tissues together will form organ. Then there will be the organ system. And then there comes the organism. That's what we know. Then there comes the organism. That's what we know. That's what we know. So cell, tissue, organ system, organism. This is what we have. This is what we have. Thank you, Bachaniki. Now the point is, even if you can relate it with the animal kingdom, we see cellular level of organization. So many cells together, they are forming a multicellular body. We see that cells together, they form tissue, and in organism, tissue level of organization is there. Then organ level of organization, organ systems are there. That's what we have. That's what we have discussed already. Isn't it, Bache? Isn't it? Now here in this chapter, we are mainly going to talk about the tissue. Right? So study of tissue. First of all, let me tell you, Bache, the study of tissue, it is known as histology. What is the study of tissue? Study of tissue is known as histology. Now what is a tissue? Tissue is, of course, it is a group of cells. It is what? It's a group of cells, right? So many cells together having similar or dissimilar function, having similar structure. Sorry, not the function. Let me tell you, when you talk about the tissue, tissue is a group of cells which are having similar structure, which are having right bache, which are having similar structure, which are having kind of similar function, and they are having the same origin. So what is the most important thing? That their origin should be same. Even their structure can be dissimilar, but when it comes to their origin, the origin should be same. So it's a group of cell having similar structure, function and origin. It's a group of cell having similar structure, function, having similar structure, function and origin. This is what a tissue is. This is what a tissue is. But what is the another most important point here? That they may have, right, these cells, they may have dissimilar structure, right? It doesn't matter, even their structure can be dissimilar, right? They may have dissimilar structure, but origin, okay, origin should be same, right? They should always be same. So their origin should always be same. This is what a tissue is. Is that clear? This is what a tissue is. Is that clear? Yes, bache. So on the basis of function and structure, you can see it here also. Let's give it a reading so that we can finish it very quickly. You make sure you mark the NCERT, right, bache? So see, in the last chapter, yes, we have seen unicellular, multicellular organisms. We know it very well, right, bache? We also know that unicellular organism, they can perform all the functions by their own. It's just that they do not have division of labor. Even a single cell can eat the food, can digest the food, can excrete it out, can reproduce. We know it very well. But when there is a complex body of multicellular organisms, right, so these functions are carried out by different cells, right? There is a division of labor. One part will do something, another part will do something. So here they have given the example of Hydra. So can you tell me about the Hydra? It belongs to which phylum? Yes, bache, this is a question for all of you. This is a question for all of you, right? This Hydra, it belongs to which phylum? I want to see the answer, and guys, your energy should be fine. I'm really amazed to see that we have just started the session 2 hours back, and near about 100 plus students, they are done with the session. Like seriously, this is your dedication for NEET. It should not be there, guys. You need to be energetic, people. You need to be energetic. Okay, so obviously Hydra belongs to phylum Ctenophonidaria, also known as Coelenterata. We know it very well. It is having what? Tissue level organization. So always remember, bache, whenever you see a question which is common, which is even there in another chapter, you should always note it down. Okay? Okay? So Hydra may be, there are different types of cells. Human body may billion of cells are there. So see, group of similar cell with intercellular substances, they perform a specific function. That organization is tissue. So as such, NCERT definition should be there in your fingertips, that cells in the body, they work together. Yes. So group of similar cells along with the intercellular substances, they perform a specific function, and such organization is known as tissue. Just for an example, this is a single cell that we have. So group of cell, this is going to form the tissue. And in the surrounding, what will you see in the surrounding? Right, in the surrounding we will see intercellular substances. What are you going to see, people? Yes, tell me, what are you going to see, people? Intercellular substances, right? Intercellular substances, they are holding them all together. Yeah, you can also say that, say that this is ECM. It is all same. ECM means extracellular matrix. Extra means outside the cell, right? And you know that it is the ground substance. What is the meaning of ground substance? It will help in the linking. It will help in the linking. That is what you need to retain. Is that clear? So this is what tissue is. So on the basis of, as I said, on the basis of what? On the basis of structure and function, tissue is of 4 types. On the basis of structure and function, tissue is of 4 types. Can you name that? Can you name that? Yes, bache. Can you name that? So see, tissues are organized in specific portion and pattern, and they form organs, right? How tissues will form organ? How tissues are going to form organ? Tissues, they'll be organized in a particular proportion and pattern. Focus on these words, please. Focus on these words. In a specific proportion and pattern, they will form organs like stomach, the lung, the heart, the kidney. Okay, so two or more organs which perform the common function, they perform it by their physical or chemical interaction, they together form organ system. ठीक है बच्चे? Right. So they are saying when two or more organs, they perform a kind of common function by their interaction. It can be physical or chemical, right? Bache, in NTA, in NEET examination, they pick up the lines from NCERT, so make sure you don't miss anything. Okay, so they will form the organ system. So this is the part. So as I said, on the basis of right structure and function, on the basis of structure and function, we are having four types of tissue: your epithelial tissue, your connective, muscular, and neural tissue. Is that clear? Is that clear? Your epithelial tissue, your connective tissue, your muscular tissue, your neural tissue. Because we have to save our time for the human physiology's topic, that's why bache, I'm not writing everything by my own. Don't worry, I'll write all the important points here. Okay, bache? So epithelial tissue, obviously it is meant for covering and protective. It is the, what kind of tissue it is? Covering and protective tissue. It is going to cover the body organs and it will protect them. Connective, obviously connective, the word is telling us connective mane, it will connect, nahi, it will connect the structure, it will provide the support to the body, isn't it? It will connect structures, it will provide support, it will connect structure, provide support to the body, right? It is going to connect structures, it is going to connect structure, it will provide support to the body and help in transport of things. Okay, then comes the muscular tissue. What is the role of muscular tissue? Hanji bache, sweetie, what is the role of muscular tissue? Obviously contraction and locomotion, and here to generate and to conduct the nerve impulse. So it will help in contraction, it will help in locomotion, and this one, this neural control and coordinate, this neural tissue, it will help in, it will generate and conduct nerve impulse. It will generate and conduct nerve impulse. This part clear? Yes, everyone. This part clear. Now let's move to the epithelial tissue. So epi means above, thelial means growth. So epi means upon, thelium means growth. So obviously it is the tissue which will grow over the another tissue so that it can act like a protection and the covering. Are you getting my point? So here you guys can see epithelial tissue, epithelium, right? So epi means upon, thelium means growth. So it will grow over the another tissue. Now there is one line which is written. It is also a previous year question. This tissue has a free surface which faces either a body fluid or the outside environment. That is why it is acting like a covering or it is lining some parts of body. Very simple. Just say, let's say this skin, this outer covering, epithelium, it is facing the outside environment. Another side will face the internal environment, your body fluids. Just say, let's say there is a let's say this tissue is covering a body organ, right? So that outer covering, so obviously it will face what, right? It will face either the outside or the body fluids, here also and here also. That is the meaning, right? That is the meaning. Clear bache? Clear bache? So this is what epithelial tissue is. Now it's the time for the flow chart. So when you people talk about the epithelial tissue, commonly described as epithelium, we divide it, you know na, we divide it. It is simple epithelium, yeah, simple epithelial tissue. It is simple epithelium and simple epithelial tissue. Then comes the compound epithelium. So only this part will take time. So please pay attention. Okay. So simple is uniserrated. What is the meaning of uniserrated? It is single layered. It is one cell layer thick. It is one cell layer thick. Okay. What is it? It is one cell layer thick. Right. So on the basis of different cells, it is of different types. Just a cuboidal simple epithelium. Okay, one second. So firstly squamous. Let's go in a sequence. Cuboidal, columnar, You will also talk about the pseudo stratified epithelial tissue. Okay, pseudo stratified epithelial tissue. So you can make the flow chart for your quick revision. Okay, you can make the flow chart for your quick revision. Is that clear? Is that clear? Yes. Is that clear? And when it is the compound epithelium, so it is multi-serrated. Multi means many, serrated means layer. So it is many cell layers thick. So this is the major and the first difference here. So it is many cell layers thick. It is many cell layers thick. So here you will be discussing stretchable compound epithelium. Stretchable compound epithelium, and we'll be discussing non-stretchable. So when it is the stretchable one, then it is also known as urothelium. It is also known as transition epithelium. It is also known as urothelium. It is also known as transition epithelium. And of course, when it is the non-stretchable, you will be having compound, squamous compound epithelium. Okay, you will be having cuboidal compound epithelium, columnar compound epithelium, brush bordered, ciliated. This is what we have to discuss. So up to this part all clear? This flow chart clear? This flow chart clear? So simple mane one cell layer thick, mane uniserrated. So before discussing it, let me tell you how the arrangement will be there. Dekho, when you talk about the epithelial tissue, you will see these cells are very compactly packed. Can you see that? Can you see that? Please understand this part, everyone. Please understand this part. So so basically epithelium, right, cells are compactly packed. What are they? They are compactly packed with little intercellular matrix. It is in NCERT also. Cells are compactly packed with little intercellular matrix. Okay. See, epithelium grow over another tissues, then also it can act like a layer. So here you will see in the epithelial tissue, you will see that they grow over the connective tissue. Just here you will see connective tissue, right? So connective tissue means that blood supply, that cells, they will be present. So it will grow over the connective tissue. So because cells are compactly packed here, so you won't see any blood supply. Okay, you won't see any blood supply here within the compactly packed cell. And actually these epithelial cells, they lie over, they grow over, just a minute. They lie over, grow over a basement membrane. It will always grow over a basement membrane. So below that basement membrane, you will see your connective tissue. So they will grow over what? They will grow over the basement membrane. Now when you look at this basement membrane is having two components. Wait, let me show you. So first of all, tell me, this basement membrane part clear? This basement membrane part clear? So epithelial tissues, they will always lie on the basement membrane, they grow over the connective tissue. So when you look at this, this basement membrane is having two parts. Can you name that parts? Yes, people, can you name that part? Yes, can you name that part? Two parts are there, this is important. One is basal lamina, another is fibrous lamina. One is basal lamina, another is fibrous lamina. These are the two words. Basal lamina and fibrous lamina. So basal lamina. So basal lamina, it is secreted by epithelial cell itself. It is secreted by, this is a dead layer, this is acellular. It is secreted by epithelial cells. They go basement membrane, it is a dead layer. It is not living. It is not made up of cells. Okay, it is not made up of cells. That's something very important, guys. That is something very important. So basement membrane, it's a non-living acellular layer. It's a non-living acellular layer. What's that? It's a non-living acellular layer. Right बच्चे, right बच्चे. Yes, so it's a dead layer, non-living, acellular and highly permeable, right? So permeability is required, of course. It is highly permeable, and it should be, because then only we will get the essential things from the connective tissue. So this basal lamina secreted by epithelial cells, it is made up of glycoproteins. It is made up of glycoproteins. Clear बच्चे? It is made up of glycoproteins. Understood? Understood? ठीक है? And now when you talk about the fibrous lamina, it is secreted by connective tissue. For connective tissue, let's use the short form CT, and contains fibers, reticular fibers. It will contain mucopolysaccharides. Okay, it will contain reticular fibers, it will contain mucopolysaccharides, etc. So can I say that basement membrane is formed by both epithelial tissue and the connective tissue? Can I say that it is formed by both epithelial tissue and the connective tissue? Yes, bache. Can I say so? Yes. Can I say so? Of course we can. Of course we can. है ना? Of course we can. ठीक है? Now obviously these cells are compactly packed, so obviously they need to be fixed with each other ना. They need to be attached to each other, they need to be fixed with each other. So बच्चे, here you talk about certain cell junctions. It is given in NCERT as well, but let me explain it first, then we will directly discuss it from NCERT. So we discuss some things called cell junctions. What do we discuss here? We discuss some things called cell junctions. So do you remember cell junctions? जैसे we talk about the gap junction, we talk about the adhering junction, and we even discuss the interdigitation. I'll tell you what are interdigitations. Hanji, so how many of you know about it? How many of you know about it? If it is a yes, I want the answer in the chat section, guys. If you know this, then tell me. If you know this, then tell me. Hanji, quick, quick. I think I missed one. I missed the tight junction. Okay, I missed the tight junction. So all that junctions are going to hold them together. They're going to hold them together. Wait, let me show you the images as well. That will be better for you then. Right, let me. See this. We are having wonderful diagrams. Look at this. So types of cell junctions. So as I said, we will be having tight junctions, we will be having adhesion junctions, we will be having these gap junctions, right bache? So let's discuss that. Okay, let's discuss that junctions first. So you can see this picture. You can see this picture. It is showing you the junctions properly. So okay, fine. Let's start with the tight junctions first. So tight junction. The word is making it clear, sweetie. The word is making it clear. Right bache, right bache. So if it is a tight junction, what are you going to see? That it will not allow the leakage of substance from one cell to another, right? It will prevent the leakage. So tight junctions, we will see them in the columnar cells mainly. You will see them in the columnar cells mainly. Columnar cells means the tall cells, right? Tall and cylinder cells having nucleus at its base. Columnar cells means pillar-like cells, tall cells having nucleus at their base. So in columnar cells you will see this. So they prevent leakage of substance. They prevent leakage of substance. They will not allow the substance to move from one cell to another. This is the meaning of tight junction, right? This is the meaning of tight junction. So what that tight junction will do, right? So two plasma membranes, they are fused, so substance will not leak across a tissue. Okay, substance will not leak across a tissue. Is that clear? Is that clear? Now in the cells you also see that, see, one cell, it is continuous with another cell. They are having a gap in between. Can you see that? They are having a gap in between, right? This is the continuous cytoplasmic gap. So this is a gap. This is the gap junction. So you can see two cells, they can exchange, right? Two cells, these two cells, right? From this gap junction, the cytoplasm will cross here and there. Okay, the cytoplasm will cross here and there. Can you see that? Can you see that gap junction? So these are the continuous cytoplasmic connection in between two cells. Can I say so? These are the continuous cytoplasmic connections in between two cells. Can I say so? These are the continuous cytoplasmic connections in between two cells. Can I say so? Yes, of course we can. Of course we can. So these are what? These are the gap junctions. Continuous cytoplasmic connection. Now, just for your quick revision, right? Dekho, these gap junctions, they are like this. So here they are made up of a protein. You know, this gap is basically made up of a protein and you call it as connection protein. ठीक है, जैसे here you can see this picture, this diagram, they are having a gap. This gap here is made up of connexin protein. This gap here is made up of connexin protein. And if you people remember, right, in the plant cell also we see something like this. In the plant cell also we see something like this. Plant cell में you only talk about this continuous connection and we call it as plasmodesmata. We call it as plasmodesmata. We call it as plasmodesmata. Done bache? Done bache? So gap junction part clear, continuous cytoplasmic junction part clear. Now bache, desmosomes या adherent junction. Adherent, the word is adherent, means to join, right? Means to join, isn't it? Isn't it? Just these are the adherent junction, right? You can also use the word macula adherence, adhere, to join, right? To adhere, to join. So these are, right, what are they? These are the adhering junctions. Desmosomes are the example here. Desmosomes are the example here. Right, bache? Right, bache? Yes, yes, yes. Yes, so you are going to see such junctions जैसे desmosomes भी हैं in the stratified epithelium. What is the meaning of stratified epithelium? What is the meaning of stratified epithelium? Stratified माने layered, multi-layered. Means you are talking about the compound epithelium here. So it is multi-layered, Sudha, multi-layered, stratified, multi-layered it is. Just for an example, let's say this is one cell, this is another cell. Right, now in a stratified epithelium, you need to link these two cells together. So you will see some structure, some disc-shaped proteins, and from that proteins, you will see some fibers will arise. Right, some fibers will arise like this, and there are something like this. So basically these are for the, like let's say you are using the word adherence, means to join two cells together, means to fix them all together. So in the cytoplasm you will see these proteins and the fibers are present. Can you see that? Can you see that some disc-like proteins are there? They are having some fibers with them, right? But they are having some fibers with them. They are in the cytoplasm of the respective cell. So they will provide the mechanical support. They are acting like a cementing material because it will link them all together. It will link them all together. So these are, when desmosomes, these are the disc-shaped proteins in the cytoplasm. So these filaments that are associated with it is intermediate filament. These are intermediate filaments, not the actin filaments. Question can come from this part. Just say 3 type of cytoskeletal elements are there that we discuss. We talk about the microtubules, we talk about the actin filaments, also known as microfilaments. We talk about the intermediate filaments, जैसे tonofibrils, keratin filaments, they are intermediate filament. Okay, so this is nothing but the cell skeletal element. So this is how you discuss the desmosome. Even now, when we have to link a cell with the extracellular matrix, you know na, extracellular matrix माने surrounding. So when we have to link a cell with the extracellular matrix, then we talk about hemidesmosomes. ठीक है, half. जैसे here तो you have 2 disc-shaped protein, then they are having protein fibrils. They are like this, so it will be hemidesmosome. Hemidesmosome help a cell to attach to the extracellular matrix. Is that clear? Help the cell to get attached to the extracellular matrix. Is that clear? Hemidesmosomes clear? Hemidesmosomes clear? Okay. So now I also use the word interdigitation, right? You will, when you will read the NCERT properly, right? When you will read the NCERT properly, this is the word that you will see, interdigitation. देखो, जैसे let's say you have one cell, this is your another cell. So basically what is happening? the plasma membrane of one cell will form finger-like processes that will enter in the cytoplasm of another cell. Plasma membrane of one cell will form finger-like processes that will enter in the cytoplasm of another cell. So that's how two cells are linked. We call it as interdigitation, and you will see it in transitional epithelium. Okay, you will see it in transitional epithelium. Okay, this interdigitation. Where are you going to see this? You will see this in transitional epithelium. But it is as per NCERT. Trust me, when you will read the text, you are going to find such words. Okay, you are literally going to find such words. Okay, bache. Okay. Now, when you talk about the cells, I told you different shapes are there. Okay, just let's say if the shape is irregular, right, some wavy. Let's say if the shape is what? Irregular, wavy. So that will be squamous epithelium. These are the flat cells. Okay, what type of cells they are? These are the flat cells. And the epithelium formed by them will be the pavement epithelium. Because wavy kind of shape is there, flat surface is there, you will call it as pavement epithelium or tessellated epithelium. Okay? So when you have simple squamous epithelium, flat cells are there, irregular boundaries they have, right, wavy appearance they have. So you will use the word pavement epithelium or the tessellated epithelium. Now, if the cells are having cube-like shape, cuboidal epithelium, right? And if I have used the word simple, means single layered it is. If it is columnar, it will be like this, tall and cylinder, and pillar-like cells are there. What kind of cells are there? Pillar-like cells are there. Nucleus is present at their base. Nucleus is present at their base. Understood? Understood? This is the columnar cell. This is the columnar cell. This is the columnar epithelium. Now, what I want to teach you, just say, let's say, sometimes these plasma membrane, they are having some processes. Just say, let's say, if it is a cube-like cell, and if it is having finger-like projections like this, if it is a cube-like cell, and if it is a finger-like projections like this, what will you call it? These finger-like projections are microvilli. Obviously, they will increase the area for absorption. They increase surface area for absorption. They increase surface area for absorption. Right, this is the role of microvilli. What that microvilli will do? People, quick. I want to see the responses here in the chat section. Quick. I'm telling you about the processes that can arise on the one side of the free side of the cell. So tell me quickly, guys. Tell me quickly. Yes, what will be there? What will be there? Right, if finger-like projections are there, we will call it as microvilli. We will call it as microvilli. And such type of epithelium, such type of cells which are having microvilli, that epithelium will be brush bordered. What will be the word? Brush bordered. Just let's say if cuboidal cells are having microvilli, brush bordered cuboidal epithelium. If columnar cells are having microvilli, brush bordered columnar epithelium will be the thing. This is the first point. Secondly, sometimes on their surface you will see small hair-like structure, bristle-like structures. Can you see that? This is cilia. What's that? This is cilia. Cilia is some cylindrical process. They are some cylindrical process. Yes or no? It is the cylindrical processes, and they are motile and contractile. This is important. This is not motile. This is not contractile. It is motile, means it will show movement, and it is contractile. It is motile and it is contractile. It is motile and it is contractile. Can you see that? What is it? It is motile as well as contractile. That's what you need to remember. Okay, that is what you need to remember. Ciliated epithelium, it helps in the movement. Just in that trachea, we will see ciliated epithelium is there. So if there is any dust or sputum is there, so cilia will help in movement. Fallopian tubes are having ciliated epithelium, that will help in the movement of egg. That's what you need to remember. That's what you need to remember. So it is motile as well as contractile. It is motile as well as contractile. Now, sometimes some cells, they are having the processes like this. They are having the processes like this. Conical processes are there. some cells are having conical processes. ठीक है, they are conical in shape. Right, this word is also in NCERT, please. They are conical in shape. They are also non-motile. They are also non-contractile. ठीक है. So what's the role of this? It increases surface area. It increases, yes बच्चे, it increases surface area. What will it do? It increases surface area. ठीक है. So this is the stereocilia. What's that? It is the stereocilia. So it's conical in shape. It will increase the surface area, right? So where are you going to find it? It will be in epididymis and vas deferens. It is important. That's why I am telling you this. That's why I am telling you this. So epididymis and vas deferens. ठीक है बच्चे, epididymis and vas deferens. Clear बच्चे? So it is stereocilia. So cells can have these processes. They can have cilia. They can have microvilli. They can have the stereocilia. Clear बच्चे? Clear बच्चे? Done? Understood? Sure. The process is clear. I'll come to the NCERT. The process is clear. The process is clear. So now when you people talk about the epithelial tissues, right? So this epithelial tissue, it is made up of all the 3 germ layers, right? Can you name that 3 germ layers in the chat section, people? Can you name that 3 germ layers in the chat section? I think you all can do that. Yes. Can you name that 3 germ layers in the chat section? Can you name that germ layers? Very good. It will be your ectoderm. It will be your endoderm. It will be your mesoderm. Right? So epithelial tissue is made up of all the 3 layers, but when you talk about connective tissue, mesoderm, skeletal, muscular tissue, mesoderm, exceptions are there. Your nervous tissue, ectoderm. Right? Your nervous tissue, ectoderm. It is also important to know. Okay? It is also important to know. Right, bache? Right. Sure. So I'll give you one example here: ectoderm, endoderm, mesoderm. So when it is the ectoderm, epidermis. When it is the mesoderm, when it is the ectoderm, it will form epidermis, the outer layer. Of course, it will be stratified. It will be multi-layered. When you talk about the endoderm, you know your inner lining of gut. That is basically the epithelial tissue which is made up of endoderm. ठीक है, epithelial tissue which is made up of endoderm, inner lining of gut. This is important. So here also the kind of cell is simple columnar epithelium. What kind of cell will be there? It will be simple columnar epithelium. Then mesoderm, बच्चे, mesoderm जैसे you have the blood vessels, right? Their outer covering, you know na, simple squamous epithelium. When you talk about simple squamous epithelium, so that is basically made up of this mesoderm. Okay, so epidermis, epithelial tissue is made up of all the 3 germ layers. Epithelial tissue is made up of all the 3 germ layers. Understood? Any doubt? The cell, the structure, the shape. So this is the part where students, they get confused. So that's why I'm explaining it. Okay, now let's go to the NCERT. Let's master the NCERT. So are you people ready to master the NCERT? I have given you the overview. So are you people ready to master the NCERT? Hanji, done? So obviously there are many extra examples also that I am not going to mention. Let's keep it strictly as per NCERT. So are you guys ready? Yeah, number of likes are quite less. I think it should be 1000 plus by now. It should be 1000 plus by now. है ना? Quick. It's still 981. Why? Let's make it 1000 plus quickly. Let's make it 1000 plus. Share the video link with your friends, yaar. Quick. Let's make it 1000 plus. it's now 1k plus. नहीं यार, on my screen it's still 981. अरे वाह यार, quite obedient you are, quite obedient you are. Very good, very good, very good. So let's get started now. Let's focus here. So do you want me to tell you the extra examples, or you strictly want NCERT based examples? You want me to tell you the extra examples, or NCERT based example will work for you? Tell me. Or NCERT based examples will work for you? Guys, see, quickly. Quick. Guys, quick. Madam, we want NCERT only because we know that we are at that point of time where we cannot afford to read anything extra. है ना? NCERT is too much, ma'am. What to do? चलो ठीक है, let's come back to the NCERT. So basics clear. ठीक है. Now see, the cells are compactly packed with little intercellular matrix, done and dusted. So we have two type of epithelial tissue. So if I will say simple epithelium, what should be there in your mind? What should be there in your mind? If I'm using the word simple epithelium, what should be there in your mind? What should be there in your mind? Simple epithelium, single layer, uniserrated, right? Uniserrated. What is the word that I'm using? Uni mane single, serrated mane layered, means it is single layered. It is one cell layer thick. Okay? It is one cell layer thick. Right, it is one cell layer thick, right? So it lines the body cavities, means the empty spaces. Just your gut, I told you, inner lining is made up of columnar epithelium. Remember that. Your ducts are lined with it, tubes are lined with it. Okay, when it is compound, it is multiserrated, two or more cell layers are there. Okay, two or more cell layers are there. So this is the flat cells, this is the simple epithelium. Cube like cells, it is the columnar epithelium. So it will be this D is what? It is the ciliated columnar epithelium, right? Because it is having the cilia on its surface. It is the ciliated columnar epithelium. Diagram clear? So when you see these diagrams, you need to be very clear, people, ki hanji, okay, this means cuboidal, this means columnar, this means ciliated columnar. That should be there in your mind. Okay? So squamous epithelium mein, I told you, wherever there is the filtration, wherever there is the diffusion, you will get simple squamous epithelium. This point clear? Wherever there is the filtration, wherever there is the diffusion, which epithelium you people are going to get, Monisha? Simple epithelium. So it is made up of single thin layer of flat cells. Single thin layer of a flat cell. What kind of boundaries? Irregular. And that is why we call it as pavement epithelium or tessellated epithelium. Pavement epithelium or tessellated epithelium. Everybody right now just take out a pen and paper. Right, this is something we are doing for our NEET. You have to write it properly and send the pictures to me on the Telegram. On my personal Telegram, you can send it to me. Okay? Quickly everyone, quickly. Just take out a pen and paper. When it's done, send the thumbs up. I'll tell you what you people have to do. Quick. Take out a pen and paper, quick. Whatever I'm going to tell you, you have to do it. Right? Done hai? ठीक है. So now what's the point? What you have to do? Just take out a pen and paper. Just write it down. ठीक है? NCERT. NCERT. That's all. Simple squamous. Even if you have made the notes, still you have to repeat it. Write down the examples. Then simple columnar. Write down the examples. Same we will do for dense, regular and irregular part. This is what you have to do right now with the lecture itself. Are you ready? Right now with the lecture itself. I have many students here, but I am not getting the sufficient responses. Guys, quick, do it, quickly. So come to this part. So these are the examples that they have given us. There are many extra examples because and I'm just holding myself back to tell you that examples. Why? Because ultimately it is the NCERT that we have to master. Because if we will master NCERT na, then okay, I did something. Okay. Okay, then that will be more than sufficient for us. So what's the point? Where are you going to get it? Blood vessels, air sacs, mane alveoli, right? So wherever there is the diffusion, you will get it. Wherever there is the diffusion, you will get it. Now think about diffusion. Nephron, glomerulus and woman's capsule, diffusion is there. There also you will get this. That's how we have to remember. That's how we have to remember. Now when you talk about the blood vessel, you remember in the case of blood vessels, we say they are lined with the endothelium, right? They are lined with the endothelium. So that endothelium is what? It is the simple squamous epithelium. It is a simple squamous epithelium. That's what you need to write down. Okay? Right? In the alveoli also, right? In the alveoli also, you know that simple squamous epithelium is there. Surrounding me, remember, this is the respiratory membrane. This is the blood vessel. So blood vessel is having endothelium. This is having simple squamous epithelium. Both of them are having basement membrane in between. Right? That's how the exchange of gases will be done. So that is what you need to keep in your mind. So please mention it in your notes as well. Next, cuboidal epithelium. You know the cell shape. So where are you going to find it? In the ducts of the glands and the tubular part of the nephron in kidney, right? Ducts or tubular part, proximal convoluted tubule. You know nephron, right? When you hear these words now, the diagram should be there in your mind. That's how we draw it. The proximal convoluted tubule, PCT, proximal convoluted tubule. So in PCT, bacha log, you will get cuboidal epithelium. But you know what type of cuboidal? It is brush bordered cuboidal epithelium. It is brush bordered cuboidal epithelium because here the absorption is required. Write down. Because here the absorption is required. Write down. Okay? So PCT of nephron, this is something very important, bache. This is something very important. Mention it in your notes. PCT mane proximal convoluted tubule of the nephron. It will be having brush bordered epithelium. Brush bordered cuboidal epithelium means that cube kind of cells, they will be having the microvilli. Any doubt? They will be having the microvilli. Any doubt? Any doubt? Sure? Sure? So there is one very lovely example which is again not given here. I told you I will give you the examples which are in NCERT, which are the PYQs. I'm not providing you anything extra. All that things are PYQs. You just solve the questions once, you will get it. You will get it. One more example that you should definitely write down, that when you talk about this simple cuboidal epithelium, this is important, this is given in our book itself, right? But I'm just mentioning everything here only. So simple cuboidal epithelium, you remember germinal epithelium lining your testes, ovary. That is also the example of cuboidal epithelium. It's a PYQ. It's a PYQ. It's a PYQ, people. What's that? It's a PYQ. Understood? Understood? Sure? Sure, sure, sure. Move to the next part. Move to the next part. So columnar epithelium, they are the single layer of tall and cylinder cell. They are nuclei at the base. Free surface may have microvilli. So if free surface is having microvilli, you will call it as brush bordered epithelium. Okay, you will call it as brush bordered epithelium. So they line stomach and intestine and help in secretion and absorption. So they are helping in lining the stomach and intestine, okay, and they are helping in secretion. So this is important. Clear, bache? Clear, 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 clear. So jaise when this is also given in NCERT. So when you talk about the intestine, okay, your intestine is having simple glandular. You understand glandular means they are secretory. Simple glandular, brush bordered. This is a question. Brush bordered columnar epithelium. Brush bordered columnar epithelium. So basically your gut lining is made up of columnar cells, and in intestine, right? In the intestine, especially your duodenum and ileum. Especially your duodenum and ileum, they are having this type of cell. So they are glandular also. Glandular you mean secretory. Glandular you mean secretory. Clear, bache? Clear, clear, clear. This part clear. This example is very important. It's a PYQ. Moreover, ciliated columnar Ciliated columnar means when columnar cells are having cilia. So ciliated columnar epithelium, you will see it in fallopian tube. Again a previous year question. Please write it down in NCERT. You can solve the questions. You will get all the questions there. You will get all the questions there. Done bache? Done bache? Yes, yes, yes. Yes, so ciliated columnar epithelium, it is lining the fallopian tube, and even ependymal cells of brain ventricle. जैसे brain के ventricles, the cavities in brain are lined with these cells. They are also ciliated. That's what you need to mark. That's important, right, people? That's important, and that is a PYQ. That's why I'm mentioning it here. Understood? That's why I'm mentioning it here. Done, bache? Done, done, done. Sure. Sure. So these examples are in NCERT, so write down. So see, if cells, they will bear cilia, they are the ciliated epithelium. Done and dusted. Okay, so this line, where are you going to find the ciliated epithelium? Hollow organs mein, where there is a requirement of the movement. Where there is the requirement of the movement. So see this, ciliated epithelium, they are mainly present in the inner surface of hollow organs. Fallopian tube hollow. Okay, that ventricles, this is a cavity, hollow. So hollow organs like bronchioles and fallopian tubes, like bronchioles and fallopian tubes. Okay, like bronchioles and fallopian tubes. Done, bache? Done, bache? So see, this is important. Understood? This is something very important. Before going ahead, one more thing. It is not written here, but again they ask the question from it. Pseudo stratified epithelium. What is the meaning of pseudo stratified epithelium? All of them are PYQs. I am not teaching you anything extra. They all are PYQs. Okay. Pseudo stratified epithelium. What is the meaning of pseudo? Pseudo mane false. False. Stratified means multi-layered. So it is not actually multi-layered. It looks like multi-layered. Okay. It is not actually multi-layered. It looks like multi-layered. Clear, bache? Clear, 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 clear. Clear, bache? So basically, just for an example, this is the modification. This type of epithelium is the modification of columnar epithelium. It is the modification of columnar epithelium. And what is happening here, bache? Again I'm repeating, it is the modification of columnar epithelium. And what is happening? There will be, you know, cell layer is one only, but there are two types of cell, long and short, right? Two type of cells are there, long and short. They are lying on the same basement membrane, people. They are lying on the same basement membrane. These are the two type of cells. So because these are the two type of cells, it looks like, okay, it looks like multi-layered, but actually it is not. Okay, it looks like multi-layered, but actually it is not. Just for an example, some cells are small, some cells are large, and even their nucleus, they lie at different position. Okay, even their nucleus, they lie at different position. So that's why, what do we say? Just a minute. Done. So what's the point here? You can see this. So here nucleus are present at the different base. Okay, nucleus are present at the different base. Okay, bache. So that's why it looks like multi-layered, but actually it is single layered. So example is important. So you will see it in the case of trachea. So this is a very famous question. Trachea is having pseudo stratified ciliated columnar, pseudo stratified ciliated glandular columnar epithelium. Pseudo stratified ciliated glandular columnar epithelium trachea mein. Even in the bronchioles you are going to get it, right? Even in the nasal chambers you are going to get it. Okay, even in the nasal chamber, respiratory epithelium of nasal chamber you will find it. This is important. This is important. Okay, this is all about the simple. This is all about the simple. This is all about the simple columnar epithelium. Yes, it is deleted in new NCERT, but it is still the part of your syllabus. Okay. Okay, chalo. So come to this part. So they are saying that some of the columnar or cuboidal cells, they get specialized for secretion. We call them as glandular. Glandular means secretory, right? Glandular means secretory. Look at this. ये देखो, look at this, right? So when, right, a single cell, unicellular gland means single cell is secretory. Of course, multicellular means when so many cells together, right, together they are forming one gland. Multicellular gland it is. ठीक है. So unicellular में again they can ask you the examples, जैसे goblet cells, you know ना goblet cells माने mucus cells, they are the mucus secreting cells of the alimentary canal. They are the mucus secreting cells of the alimentary canal. And multiple में salivary gland is example, and there are many other example. So ultimately question is going to come from NCERT. No doubt it is this thing is not given in NCERT. जैसे what is apocrine, what is holocrine, what is merocrine, but I'll suggest you to read it once. I'll suggest you to read it once. I am sticking to NCERT, but I'll suggest you. ठीक है. बाकी you know that exocrine and endocrine, right? Do I need to tell you exocrine or endocrine? देखो, it's simple. Exocrine glands, they are having duct. They are having duct. Means let's say this is a gland you have. This gland is having a tube. ठीक है. So through this tube they are pouring their secretions to the destined place. Right, they are pouring their secretion to the destined place. That type of glands are exocrine. जैसे for an example, your salivary glands, right? Your salivary glands are the example of exocrine gland. I'll repeat this part. Salivary glands are the examples of exocrine gland. I'll give you one example. Let's say this is your buccal cavity. Your salivary glands, right? Your salivary glands are located outside the buccal cavity. They are having the ducts. ठीक है. Their salivary glands are located outside the buccal cavity. They are having the ducts through which the secretion will be poured here in the buccal cavity. So salivary glands, what type of glands are they? They are the exocrine glands. What type of glands are they? Quick people. What type of glands are they? They are the examples of exocrine gland. ठीक है बच्चे. They are the examples of exocrine glands. ठीक है. Understood? So even your milk glands, even your oil glands, even that ceruminous gland, means your ear is having that gland na, which secretes cerumen, ear wax. So these are the example. So when it is the endocrine, this is a ductless gland. They do not have that tube. So it is what kind of gland it is? It is ductless gland. What type of gland it is? It is ductless gland. ठीक है. What type of gland it is? It is ductless gland. So this gland will directly pour its secretion in the blood. ठीक है, into the blood. So through that blood, that will go to the target organ, endocrine. ठीक है, endocrine. Then heterocrine, obviously in chemical control and coordination we will discuss this. Heterocrine, also known as composite, also known as mixed gland. So it is having heterocrine, it is having endocrine part as well as exocrine part. Endocrine part as well as exocrine part. So you have the example of pancreas, you have the example of gonads. Gonads means primary sex organs. That is your ovary, that is your testis. Understood? Understood? This is about the glands. Understood? This is about the glands. ठीक है. So now compound epithelium. I already told you about the compound epithelium. Because they are more than one cell layer thick, so they are meant for protection. Right? They have limited role in secretion and absorption, but mainly their function is to protect against chemical and mechanical stresses. Right? To protect against chemical and mechanical stresses. So they cover the dry surface of skin, moist surface of buccal cavity. Examples again noted down in NCERT. ठीक है. Moist surface of buccal cavity, pharynx, lining of ducts of salivary gland, they are of, right? So this is what we have. Still, I'll give you one very quick recap, okay, because somewhere it is in NCERT. So compound epithelium means multi-layered. ठीक है. So what do we say? Let me tell you. जैसे we say stratified squamous epithelium. Stratified माने multi-layered. Okay. Now stratified cuboidal epithelium, okay, and stratified columnar epithelium. You just need to remember one thing here, okay, then I will again come back to NCERT. You just need to remember one thing here, people, right? What you need to remember here? You just need to remember one thing here. And what is that? What is that? Dekho, when it is stratified squamous epithelium, when it is stratified squamous epithelium, it means the topmost layer, right, the upper layer is having squamous epithelium. Dekho, it is stratified means multi-layered. So at the base you will see some cuboidal cells. At the base you will see some cuboidal cells. Of course, that cells are laying over the basement membrane. Somewhere in between you will also see some columnar cells. But when it is the stratified squamous epithelium, always remember topmost layer will be of squamous epithelium. Any doubt? Topmost layer will be of squamous epithelium. Let me finish this chapter, then we will go for the lunch break. Okay, then we will be having the lunch break. Let me finish this chapter first. Okay, let me finish this chapter because already I have given you 10 minutes ka break. So let me finish this chapter, then there will be the lunch break. Done. Done. So now when it is stratified cuboidal epithelium, again stratified cuboidal, so the topmost layer is of cuboidal cells. So at the base you will see some cuboidal cells, you will see columnar cells, but the topmost layer is of cuboidal cell, and stratified columnar means topmost layer is of columnar epithelium and remaining will be cuboidal and all. Okay? So any doubt? Any doubt here? Are you sure? Any doubt here? Sure. So one thing that I want you people to remember is, dekho, I'll give you the example of skin. We have two words. One is keratinized, stratified. This is important, that's why I'm telling you, because it is also a previous year question. Keratinized stratified squamous epithelium, or non-keratinized stratified squamous epithelium. Okay, non-keratinized stratified squamous epithelium. These are the two words that we have. So skin is basically the stratified epithelium. So the innermost layer, which is having high mitotic index, just say here, let's say this is the basement membrane. So you will see the innermost layer, that is of cuboidal cells. It is bache known as stratum germinativum. Solve last 10 or 15 years ka MCQ, you will get this word. Stratum germinativum. Why? Because it is having high mitotic index. What is the meaning of having high mitotic index? Means it will divide, it will proliferate, and it will add cell. It will add more and more cell to this layer. So innermost layer in that skin is stratum germinativum, which will divide and add more and more cells. Any doubt, people? Any doubt here? Then in between polygonal cells are there. And that's how the skin will form. Okay, so skin will come under that stratified squamous epithelium part. Understood? Now, when it is keratinized, what is keratin? Let's say when topmost layer, this is the thing which is there in our skin. The topmost layer, when the cytoplasm, it is replaced by, okay, cytoplasm is replaced by keratin. Okay, that will be the keratinized stratified squamous epithelium. Just in our skin, through our skin, water absorption will not be there, right? Water will not evaporate. Why? Because our topmost layer of the skin is dead. It is horny layer. Okay, you might know this word. It is the dead layer. It is the horny layer. It is the dead layer. It is the horny layer, topmost layer, topmost layer. So what is happening in this topmost layer? Cytoplasm is replaced by the keratin. It is replaced by the keratin. So that topmost layer will be of dead cells and without a nucleus. No nucleus is there. So you know that even in our skin there is the shedding of skin. In winters also we see that dead skin will remove. So that is upper dead layer, keratinized. So water wastage is also not there. Okay, water wastage is also not there. So that's the point. Keratinized stratified squamous epithelium, best example your skin, of course. Okay, so skin, right? Best example your skin, of course. So this is something that you need to know. Skin is there, nails are there, right? You know na, wherever keratin is there, when we cut our hair we don't feel pain. Why? Because they are dead. Nails, when you cut it, no pain. Why? Dead. So skin may, nails may, feathers may, hoofs may, etc. they are keratinized. Now when it is non-keratinized, simple, topmost layer is living, it is not having the keratin. So there are many examples. One example that you can write down is cornea of eye. Okay, though there are many examples, जैसे buccal cavity of mammal is also having the same. Okay, buccal cavity of mammal, it is also having the same. So now I will help you out how, जैसे ये देखो, they have given moist surface of buccal cavity. So you can write down in NCERT कि it is basically stratified cuboidal epithelium. ठीक है, what's that? It is, sorry, not cuboidal, stratified squamous epithelium, है ना? Stratified squamous epithelium. Done बच्चे? Done. Sure. When it is the pharynx or inner lining. चलो, what about pharynx? Pharynx will be somewhere stratified columnar epithelium. ठीक है. Next is your lining of ducts and all. When you talk about the linings of duct na, so this is your stratified cuboidal epithelium. Clear बच्चे? It is stratified cuboidal epithelium. That's what you have to remember. Let's go. ठीक है. So okay, so one more thing which is not here in this chapter but it is in your excretory product, transitional epithelium, right? I'll take another 1 minute and I'll finish the epithelial tissue. We'll move to the connective tissue. We are about to finish this chapter because muscles and neural tissue will be discussed in their respective chapters. So let's finish this and the next chapter, then let's move to let's go for the lunch break. Okay, then let's go for the lunch break. Ready? Yes people, are you ready? Arey yaar, now to you are getting the lunch break. Show some energy guys, show some energy. Yes people. Frog and cockroach will be done later. Done. After lunch break, let's start the human physiology. So transitional epithelium, which is also known as urothelium. It is the compound epithelium and we also call it as stretchable. Why? Dekho, sometimes it happens na when we are having urge to urinate. Right, sometimes it happens now when we are having urge to urinate, we want to pee but we can control it for some time. You all know about it, isn't it? We want to pee but we control it for some time. है ना? We want to pee but we control it for some time. कि ठीक है यार, we are not going to pee, let's hold it for some time. But then there comes a time कि we are not able to hold it. Even if we will try to hold it, the urine will come out. Why? Transitional epithelium. How? It is a stretchable epithelium. When you talk about the topmost layer, it is of kind of globular cells. Okay, when you talk about its topmost layer, na, it is a kind of globular cells, right? And these are the cells which can get stretched, right? These are the cells which will get stretched. They say you will use the word distensible for it, right? Which word are you going to use for it? Distensible is the word. ठीक है बच्चे? Distensible is the word. So obviously it is also multi-layered epithelium. So it is globular. It is basically a kind of cuboidal cell. So here also other cells are also there. other cells are also there. So below that you will see 2 to 3 layers of polyhedral cells, right? Polyhedral cells will be there, and ultimately it is lying over the basement membrane. We know it very well. Ultimately it is over the basement membrane. We know it very well. Okay, so outermost layer is kind of oval cells, then some polyhedral cells are there. So that's how this multi-layered epithelium will form. Now when see, it is lining your renal pelvis. Okay, you know the kidneys, this is your pelvis, the ureter, this is the urinary bladder. So renal pelvis, ureter, urinary bladder is lined by this. Let me relate that how it will help in controlling the urge to urinate. Very simple. When urine will fill here, when urine will come here, so obviously when the volume of urine will increase, it is going to exert a kind of pressure on this topmost layer. So these cells, they will distend, they will become flat. These cuboidal cell, they will show that distension, they will become flat, right? They will become flat. Yes or no? They will become flat because volume is increasing. So now initially they were globular, now they are just getting flat. So obviously they are increasing the capacity of that urinary bladder there. That's why when we have urge to urinate, we can control it for some time, right? We can control it for some time, right? But later on we cannot. So when this urinary bladder is, you know, a little above half filled, then we start getting that urge to urinate. But still urinary bladder can store the urine because these cells, they will become flat, so the accommodation capacity can increase. Right, so this is about the transitional epithelium. Now let's move to the connective tissue. So junctions done and dusted, let's move to the connective tissue. So connective tissue mein very first instruction that I would like to give you is this diagram. Most important. It is the most important diagram. It is the most important diagram. Yes or no? Yes or no? Some students are saying, can you make a video to get, can you just make a video to get what they are saying? To get 270 marks. Bache, you can do that top 60 questions ka video. You can do the, you can check the top 60 important topics ka video, you will get that marks. And one more thing, I think you people are even going to expect that how to get 650 plus marks in last 10 days. How to get AIMS Delhi in last 1 day. What to revise in last 1 day so that you can get admission in the top medical colleges. Really? Do you think it is possible at all? It's not. It's not. Those who are studying, we can motivate them again and again. We can tell them that this is what you have to revise and you will get a proper college. Just if you will see my last strategy video, I clearly mentioned that at this point of time, if you are just starting studying, you should not focus on your result. You should just put the efforts there. Right? But not like this. Not like this ki you are expecting ki hanji, I will watch the strategy video, definitely now I'm going to crack it. No, you have to study. You have to study. That's why we keep shouting that join the batches from starting. Do not wait for the last. Dekho bachche, the Aramb is something really amazing for all of you, right? You can just, you have to pay 4,999 rupees and you can access that all that courses in for 1 year, right? This is what you people can do. You will have the access of that batches, and obviously if you are clear that you have to go for the NEET, then why do you start late? What are the reasons? If I'll say financial reasons are there, now your fees is very less, right? If you will compare the fees with the offline courses may 2 lakhs, 1 lakh, 2 lakhs, then all that, you have to go there, you have to bear that expenses, right? The traveling, the food and everything. But here you just need to pay for your internet and then electricity. This is what we have to do. So my point is that why do you waste your time? Why do you wait? Ki hanji, I'll start in the month of November, then I will watch the strategy video and I will increase their views. In my case, even if you will check my strategy videos, right? If you check my strategy videos, no one can tell me that it is not realistic. You are just over motivating them. Never ever. Because I know the reality. I'm not doing it just to, get some good views. Ki bache will be very happy. Nahin yaar. Nahin, I cannot. I still remember a student commented on my video ki whenever I see her face, I get depressed. Arey yaar, I'm just telling you the real things. If you will over expect na, then when you will get your result, you will cry a lot and maybe you will not be able to handle that emotion. So you have to be realistic. You should know where are you standing in this race, right? What's your position, what you have studied. If you will be clear about these things, like, you'll be able to handle that things in a proper way. Okay, that's why I always give you the advice which I even follow. So be realistic, people. Don't expect how to score, how to get AIIMS Delhi in last 5 days. And yeah, this is what you demand, this is what you want to see. Even sometimes we post it, right? Sometimes I post it. But yes, still always in a realistic way. So guys, this is my advice. Please realize it. Students are not fool, those who are giving their 2 years and 3 years for these exams. Okay, chalo, connective tissue, first line, direct MCQ, most abundant and widely distributed in our body for sure, right? What's that? It is the most abundant and widely distributed, most abundant and widely distributed tissue in our body, right? Connective because it is going to link and support other tissues, right? So they range from soft to specialized types, right? So what I was saying, that this is the most important diagram. You should know the function of these cells. Like they can just give you the diagram and they can ask you name them. Okay, they can just ask you the function of these cells. Jaise ye dekho, collagen fibers. You should know what are collagen fibers, what are fibroblasts, what are macrophages, and even they are what? They are your WBCs, your leukocytes. You know that they are your WBCs, they are leukocyte. This one is the mast cell. So be very clear about it. Okay? So let's start the connective tissue. Let's revise the connective tissue. Let's categorize it first. I'll give you a flow chart, right, that will help you to understand this in a better way. So when it comes to the connective tissue, you will be talking about the fibers. You will be talking about the cells. Basically you will be talking about the matrix. Okay, so matrix and fibers. Okay, matrix and fibers. Jaise, let's say connective tissue, loose connective tissue, dense connective tissue, specialized connective tissue. Loose में what do you have? Adipose and areolar. What do you have? Adipose and areolar. Dense connective tissue में dense, regular, dense, irregular. Specialized में you have vascular connective tissue, you have skeletal connective tissue. So vascular में you know that you have blood, you have lymph. Skeleton में we have bones, we have cartilages. Okay, we have cartilages. That's what we need to know. Okay, that is what we need to know. And examples should be clear. So connective tissue mein, when it is the loose connective tissue, it will be having more matrix. Matrix means ground substance. It is basically for linking. It is the cementing material. It is having high hyaluronic acid. It is having other mucopolysaccharide. So high hyaluronic acid. What is high hyaluronic acid? Do you remember? High hyaluronic acid. It is the cementing material also. Right, it is also the cementing material. So you will see it in matrix. This is important to know. That's why I am specifying here. So you will see that this is going to link two cells. That's why in human reproduction, you talk about the enzyme hyaluronidase that will penetrate, that will help in penetrating the corona radiata, because the cells of that corona radiata, they are connected with hyaluronic acid, and hyaluronidase enzyme will help in dissolving that. ठीक है बच्चे? So here it will be more matrix, less fiber. So basically in connective tissue the story will revolve around matrix and fiber. Here you will be having less matrix, more fibers, and of course regular and irregular is as per what? as per what? It is as per the arrangement of these fibers and the cells there. ठीक है. Now when it is the specialized connective tissue, obviously specialized में if it is skeletal, so matrix will be solid and mineralized, right? Here it will be matrix is solid and mineralized. ठीक है. blood में या vascular में of course it will be the fluid matrix. What will be there? It will be the fluid matrix, right? So liquid and fiber free matrix will be there. Liquid and fiber free matrix will be there. So this is the gist of the topic that you should know. Okay, this is the gist of the topic that you should know. Okay, so see, they have given the examples also. So this line is also PYQ, but I'll explain it in a little different way, that in all the connective tissue except blood, this is a PYQ, right? This is a PYQ, except blood, in all the connective tissue cells, there are cells which will secrete fibers of proteins, right? They can secrete collagen or elastin, and fibers will provide strength, elasticity and flexibility. So basically leaving blood, all are having the fibers. Okay, leaving blood, all are having the fibers. So before teaching you that particular diagram, the cells there, their parts, let's discuss about some general things, and then we are about to finish it in next half an hour. Okay, we are about to finish it in next half an hour. So be quick, bache, be quick. Okay, done? Sure? ठीक है. So when you people talk about the connective tissue, so connective tissue is having something known as cells and fibers. It will be having some cells, it will be having the fibers. ठीक है. I am explaining the diagram now. So very important thing about the cells is fibroblast cells. You need to talk about the fibroblast cell. So can you just tell me their function? You can see this diagram also. I am talking about this. These cells, fibroblast cells, right? These cells, macrophages, collagen fibers are there, mast cells are there, WBCs are there, mesenchymal cells are also there. So this is what I am explaining you, okay? So what is the first thing? Fibroblast cells. Can you tell me what is the role of these fibroblast cells? Anyone in the class? Can you just tell me? Yes, so these are first of all largest cell of connective tissue. This is a very important point. It is the largest cell of connective tissue. It is maximum in number. It is maximum in number, and it is a chief matrix producing cell. Do you know that? It is irregular, right? It is the chief matrix producing cell. Okay, chief matrix producing cell, and it is so because it is fibroblast, so according to you, what will be the role? They will produce fiber. They will produce fiber. So this line, largest in number, maximum in number, they have irregular cytoplasmic processes. You have seen their diagram. The diagram is something like this now. So irregular cytoplasmic processes. So these two lines are given in NCERT, and I think you all should know that they are chief matrix producing cells. So matrix of the connective tissue is produced by them. They are chief matrix producing cell. And what's their role? They secrete fibers. So that's why we say it is fibroblast, right? They secrete fibers. That's why we say it is fibroblast. Any doubt? Any doubt? You know the function, right? You know the function. So when it comes to the fibroblast, so they are having irregular shape, right? This is their main function. So they are chief matrix producing cell. They are chief matrix producing cell. Understood? Sure. They are chief matrix producing cell. Clear, bache? Clear, clear, clear? Sure. Sure, sure, sure. Okay. Now, do you know bones and cartilages? Bones and cartilages. Sometimes these fibroblasts, they form some undifferentiated cells also that can produce your bones and cartilages. Do you know that? They can also form, yes bache, right? They can also form, जैसे fibroblasts ना, they are undifferentiated. The undifferentiated forms osteoblasts and chondroblasts. Osteoblast and chondroblast. So osteoblast is bone forming, you can write it down in your notes. Chondroblast is cartilage forming. Chondroblast is cartilage forming, understood? Chondroblast is cartilage forming. So fibroblast can also form these type of cells. Okay, they can also form these type of cells, and these two are their main function, right? These two are their main function. Is that clear? Is that clear? Now macrophages, I think you all know about the macrophages. You all know about the macrophages. What are macrophages? You know that they are the squangers. They are the squangers, है ना, squanger cells. They are phagocytotic in nature. They are phagocytotic in nature, right? But if there will be any foreign particle in the body, these cells are going to destroy it, and macrophages of connective tissue are called as histiocytes. Maybe this word is new for you, you want to write it down, you can, you don't want to write it down, it's your choice. जैसे brain में you have microglial cells as macrophages, liver में you have Kupffer cells as macrophages. So in the case of connective tissue, histiocytes, in the case of connective tissue, histiocytes are your macrophages, understood? Histiocytes are your macrophages, understood? Understood? Next is the mast cell. Even in the chapter Human Health and Disease, we have discussed about the mast cell. Remember, they secrete, what do they secrete? They secrete histamines, they secrete serotonin, ठीक है, histamines, serotonin, right? Even heparin, है ना? Even heparin, and moreover mast cells, they also produce matrix. Means after your fibroblast, mast cells are going to produce matrix, right? After your fibroblast, your mast cells are going to produce matrix. Is that clear? Is that clear? So just like the WBCs, the basophil, it also secrete the histamine, serotonin, heparin, so it also produce mast cell. And if you remember Human Health and Disease, they also release interferons. They also release interferons. Can you relate that? Can you relate that? Macrophages of different part? नहीं, Rajshri, I am telling you ना, connective tissue में histiocytes, brain में microglia, liver will be having Kupffer cells. ठीक है, liver will be having Kupffer cells. So you remember mast cells? You remember mast cells? They release interferons. They'll protect other cells to from the virus infection. You remember? So histamine. What is histamine? Histamine is a vasodilator. It's a vasodilator. It dilates the blood vessel. It dilates the blood vessel. Remember, when there is any allergy or inflammatory reaction is there, how these cells, they protect our system. So you can relate it with Human Health and Disease. I'm not going to explain it in detail. Serotonin is vasoconstrictor. It constricts the blood. It constricts the blood vessel, and it is going to increase the blood pressure, of course. Heparin, you know that it is natural anti-blood coagulant. It is natural anti-blood coagulant. Remember, it is natural anti-blood coagulant. Do you remember that? Yes, these are the mast cells. Then there are fat cells also. Fat cells also. Okay, we'll come to that also. So this part clear? जैसे ये देखो, this is your fat cell. It is not marked here, but can you see these fat cells? Can you see these fat cells? So fat cells are also known as adipose cells. Adipose cells, or you can also call them as adipocytes. You can also call them as adipocytes. ठीक है? You can also call them as adipocytes. Done? Just a minute, people. ठीक है, this part clear. So role of mast cells clear. So WBCs में you will see the lymphocytes mainly, and how the lymphocytes are going to help. How the lymphocytes are going to help? Yes, बच्चे, how the lymphocytes are going to help? They will protect from the pathogen. But before that, let's talk about the adipose cells. What we need to discuss? The adipose cells. So देखो, here also they have given you this. Here also they have given you this. And do you know what type of fat cell it is? It is monolocular adipocytes. They say, can you see that this is the fat storage area, this is the nucleus. So they are having just single fat globule. They are just having single fat globule, so it is monolocular. Clear बच्चे? It is monolocular. Clear बच्चे? Yes or no? It is monolocular. I'll give you one example. ठीक है? I'll give you one example. When you talk about the adipocytes, yes, when you talk about adipocytes, what are they? They are the fat cells. So you have two things. One is monolocular adipocytes. Another is multilocular adipocytes. So when it is the monolocular, single locule is there. Just let's say this is your fat cell. This is the large fat globule. So because of that, your cytoplasm is at periphery. Where are you going to get the cytoplasm? It is at periphery. There will be the flat nucleus here. Okay, so this is the large fat globule. Okay, so such type of things you are going to see in the white fat. Where will we see this? White fat having less mitochondria, produces less energy comparison to this one, because this thing you will see in brown fat. If you don't know about it, you really need to answer it. You really need to note it down in your notes. Multilocular में जैसे let's say you will have many fat globules. Of course, you will have many fat globules. Okay, so it is in the brown fat. So they produces more energy, right? It produces more energy. Why? Because they have more mitochondria. What do they have? They have more mitochondria. They'll produce more energy. So where are you going to get the brown fat? Of course you are right, in the case of infants, because later on it will be replaced by the white fat. And when the animals, they go for hibernation, for winter sleep, they will use this brown fat. Okay, they will use this brown fat. Understood? So brown color is because of the pigment here. So that is something you need to know. Okay, that is something you need to know. So any doubt? Okay, so this is about the cells. So this diagram clear? Question will come from this diagram. Clear? The role of cells clear? Now let's talk about the fibers. So basically 3 type of fibers are there. Can you name that fibers? Yes, bache, can you name that fibers? 3 type of fibers are there. Anyone in the class who can tell me the name of that fibers? Collagen, you know na? Collagen. Do you remember collagen? It is the most abundant protein of extracellular matrix. Okay? Then it is the elastin fiber. Then it is the reticular fiber. So collagen is known as white fiber. Okay? This one is known as yellow. So yellow fiber is having elastin protein. That's what you need to remember. Okay, that is what you need to remember. So collagen, white, elastin, yellow, reticular fiber is also there. Okay, reticular fiber is also there. So collagen, you know that bright and white fibers. Okay, yes, bache. So it is present in maximum quantity, right? So they are present in maximum quantity. They are wavy and tough, right? They are not providing the flexibility, right? One question will come from this part. So they will not provide the flexibility. So they are wavy and tough बच्चे, right? They are wavy and tough fibers, ठीक है? So bright and white colored. So these white fibers are having collagen protein, ठीक है? They are having collagen protein. So collagen protein is the most abundant protein of extracellular matrix, or you know that most abundant protein of animal world. That is also PYQ. It was in NEET 2020. Okay, it is of PYQ. It is of NEET 2020. It is of NEET 2020, collagen fibers. Then comes the elastic fibers, yellow because of elastin protein. Okay, so they are having maximum elasticity, right? They are having maximum elasticity. So remember, even your collagen fiber can boil. Collagen fiber can boil, it can form gelatin, but this one will not boil. Okay, it is having maximum elasticity. It is resistant to chemicals, bache. It is resistant to chemical, right? This is what you need to remember. Okay, this is what you need to remember. So maximum elasticity, resistant to chemicals. And reticular is having no, they are very delicate. Okay, they are having reticulin protein. Okay, or they have no elasticity. So mainly you will see them in the lymphoid tissues. Okay, okay, this is about the fibers. And when it comes to the matrix may, you know that, right, many polysaccharide, mucopolysaccharides will be there. So you will see hyaluronic acid, which is considered as the cementing material. So this is the key word here. Okay, this is the key word here. Okay, what's that? This is the key word here. Now come to the NCERT, because now we have to remember the examples only. Okay, so when it comes to the loose, how many examples do you have? When it comes to the loose connective tissue, how many examples do you have? How many examples do you have? You have to respond in the chat section, people. Be quick. Be quick. Write down the examples. Let's finish it quickly, then let's move to the human physiology. Let's finish it quickly and then let's move to the human physiology. Adipose and areolar. Very good. Dekho, first thing is, so they are having cells and fibers which are loosely arranged. Obviously, you know that here what are you going to see? The matrix will be, that ground substance will be less, fibers are there, and fibers and cells, they will be loosely arranged. So matrix is basically kind of semi-solid ground. Dekho, ground substance means matrix. Ground substance means matrix. Okay, so what's the point that you need to consider here? That when it is the areolar connective tissue, it is present beneath the skin, right? Areolar, it is having air spaces. It is having air spaces. We should know about it. Okay, it is having air spaces. What are you going to see here in the areolar connective tissue? Air spaces. So it is present beneath the skin. It is present beneath the skin. Okay, so it serves as a support framework for epithelium. You should know it. In the NCERT, start mentioning it in your notes, right? In your notes, start mentioning it. So it is having fibroblast, you know that, cells that secrete matrix and produce and form fibers. It is having macrophages and mast cell. So basically this number is more in areolar tissue. This number is more in areolar tissue. Now adipose, fat cell, you know that. It is also loose connective tissue. It is also beneath the skin. Okay? And these are specialized. Why? They will store fat. Just let's say areolar tissues are making the adipose tissue because it is specialized for storing the fat, bache. Okay, so excess of nutrient which are not used immediately, they will be converted into fats and they will be stored. So remember, in our body, whenever we take, just a minute, whenever we take So ठीक है, so what's the point here? It is specialized to store fats. So whenever we eat anything extra, our body is like, I will use the carbohydrates I need, I will use the proteins I need, rest everything will be converted into the fats. Rest everything will be converted into the fats, and that's how they are going to store it. Now let's talk about the dense. देखो, when it is loose, fibers and fibroblasts. देखो, when it is loose, here you can use this flowchart for the revision. देखो, when it is loose, more matrix, less fiber. Now less matrix, more fiber, plus it is dense. So we will see how that matrix and cells they are arranged. They'll be compactly arranged for sure, because it is dense. You can just remember it from this word. It is dense. They'll be compactly arranged. But what is the point? If that fibers and cells they are properly arranged, right, parallelly arranged, the orientation is proper, dense, regular. If it is not like that, dense, irregular. So examples will come for sure. So they talk about the ligaments and tendons. So ligaments and tendons. Ligaments and tendons, they are the examples of the dense regular connective tissue. So this is another important topic. Okay, this is another important topic. Okay, so dense regular and dense irregular. Okay, dense regular and dense irregular. Okay, dense regular and dense irregular. So dense regular and irregular. So when it is the dense regular, you also use the word cord, right? The way they are arranged, we also use the word cord for it. When it is dense irregular, you also use the word sheath for it. I know you are never going to notice such things, right? I know it very well. You people are never going to notice such things. When you read here the chapters in human physiology, you will see they have used the word cords, they have used the word sheath. So cord means when it is a regular connective tissue, and sheath means when it is irregular. So two examples are there. The example is of tendon, example is of ligament. So you should know that they are the examples of dense regular connective tissue. So when it is the tendon will help in linking. Tendon will help in linking. Yes, people, tendon will help in linking. This is something which should be on your fingertips. It connects what? It connects what? Muscle to bones. Muscle to bones, right? And it is having white fibrous tissue. This is the difference that you can note down. It is having white fibrous tissue, means it is having that collagen fiber. Collagen is in abundance. Okay? It is having collagen fiber in abundance. Okay, so tendon is going to connect muscle and bone. Tendon is going to connect muscle and bone, and ligament, they will connect bones only. Okay, it connects bones. It connects bone. Okay, and one more thing here, this is going to have yellow fibers, means elastin proteins, with the collagen fibers. But of course, the number of yellow fiber will be more. Here you will only see collagen fibers. That is also the difference. That is also the difference, and this is important. This is important. Okay, this is important. So let's go. This is literally important. So see, this is given here. So orientation of fibers will show a regular and irregular pattern, right? So when it is the dense regular, which word are we going to use for it? Which word are we going to use for it? We'll be using the word cord. We will be using the word cord. When it is the irregular, we'll be using the word sheath. We will be using the word sheath. So in the dense regular connective tissue, collagen fibers are present. Okay? In between many parallel bundle of fibers. So tendons they are attached skeletal muscle to bones, and ligaments, they will attach one bone to another. Very important examples they are. So dense irregular fibroblast and fibers, right, they are oriented differently. So this is the diagram that we need to know. This is the diagram. So they are oriented properly. This is the example of dense regular, and this is the example of dense irregular. So diagrams should be clear, people. Diagrams should be clear. Any doubt? Any doubt? So dense irregular I can just give you some examples. Jaise I can give you the example of your vocal cords. You know, true vocal cords are the example of dense irregular tissue. Your pericardium, the covering of heart, is the example of this dense irregular tissue. So there are other examples also. Doubt? Doubt? Yes? Any doubt? Any doubt? So next is what? Specialized connective tissue where matrix is mineralized. ठीक है, where matrix is mineralized. So the diagram, बच्चा लोग diagram. So collagen fiber, then cartilage cell, chondrocytes. Bone forming cells are osteocytes. So see, these are the lacunae, these spaces are the lacunae, right? And when you look at this diagram, if you will see this diagram, can you see some concentric layers, right? Concentric means having same center. Can you see some concentric layer having same center? So we use the word concentric lamellae for it. Okay, so this is the structure of the bone, right? Here this is the space where all the blood vessels will be there, and see here these structures, just let's say it is something like this. It is something like this. So surrounding me you will say canaliculi. Just canal is the word na, so here you will say canaliculi. Okay, canal is the word na, but here which word are you going to use? You will be using the word canaliculi. Is that clear? Is that clear? Is that clear? Okay, okay. So this large canal is nothing but it is the Hebrasian canal. Hebrasian canal, sorry. It is the Hebrasian canal. It is the Hebrasian canal. You should know about it. You should know about it. Okay, done? You should know about it. So understand the diagrams from this part first, right? Understand the diagrams from this part first. ठीक है? Sure? Okay. देखो, when you talk about the cartilages, okay, so again I'll keep it to NCERT only. I'll not explain you anything extra. Outer covering is perichondrium. That's what you should know. Outer covering is what? It is perichondrium. What is the name of outer covering? What is the name of outer covering? It is perichondrium. It is perichondrium. Now you will talk about two types of cells. This is a direct question. Chondroblast cells. Actually they are the, देखो, you know that fibroblasts, they will form chondroblasts. So chondroblast cells are the cartilage forming cells. Of course I told you they are cartilage forming cells. So now these chondroblast cells, when they are mature, then which word are you going to use for it? You will use the word chondrocyte. You will use the word chondrocyte. So when they are mature, which word are you going to use for it? Which word are you going to use for it? You will use the word chondrocyte. Chondrocyte. So when it is mature, then it is chondrocyte. When it is small, when it is forming that cartilage, you are going to call it as chondroblast. And there is one more word that is known as chondroclasts. Hanji, tell me, what is the role of this? This is a PYQ. Chondroclast. Chondroclast, anyone? It is a kind of phagocytotic cell of cartilages. It is a cartilage destroying cell. It is a cartilage destroying cell. What type of cell it is? It is a cartilage destroying cell. Okay, it is a cartilage destroying cell. Are you getting it? Are you getting it? It's a cartilage destroying cell. Direct question, direct answer. Direct question, direct answer. Another important question from this part can be, let me tell you, another important question from this part can be, there are different types of cartilages. Okay? This is a PYQ again, that's why I'm mentioning it. Hyaline cartilage, right? Just say if you will solve the PYQ, you will find that which cartilage is glassy or bluish. Hyaline cartilage, right? That is the hyaline cartilage, right? Then fibrous cartilage is there. We have hyaline and fibrous cartilage. These two are the one that we mainly need to remember. Okay? Hyaline and the fibrous. So obviously it is hyaline. It is maximum in our body. Okay, so it is glass-like clear. This is the PYQ. It is glass-like. Okay, it is glass-like. Okay, okay. So fibers are completely absent here. Fibers are completely absent. Fibers are completely absent in this particular cartilage. Clear, bache? So it is glass-like clear. So you can say that here matrix color is bluish. Solve the question, you will get this PYQ. Solve the question, you are going to get this PYQ for sure. So you know your nasal septum, it is the example of hyaline cartilage. Your C-shaped cartilaginous rings, your C-shaped cartilaginous rings, they are also the example of this hyaline cartilage. Any doubt? Any doubt, bache? Any doubt? You know, sternum में costal cartilages are also there. Sternum में costal cartilages are also there. जैसे sternum में 8th, 9th and 10th pair of rib, it is attached to the 7th one, है ना? That is also the hyaline cartilage. That is also the hyaline cartilage. This is important. This is important. Your larynx is having the hyaline cartilage. This is important. That's why I am telling you. ठीक है? That's why I am telling you. ठीक है बच्चे? ठीक है? Yes बच्चे? Okay. Okay, so hyaline cartilage clear? Hyaline cartilage clear? Sure? Sure. So fibers are absent here. So fibrous cartilage में there are different types. जैसे one is your elastic cartilage. So if word is elastic, obviously it is a kind of flexible cartilage, right? It is having yellow fiber. Which fiber is present here? It is having yellow fiber. Which fiber is present here? In the elastic cartilage, which fiber is present here? Yellow fiber is present here. So that's why this is highly flexible. Highly flexible. जैसे ये देखो, this is also a PYQ. Tip of the nose. This tip of the nose, you can move it. Your ear pinna, you can move it. So tip of the nose is the example. Ear pinna, it is made up of cartilages. ठीक है? Your epiglottis, it is also basically the cartilaginous flap. So this is the yellow या fibrous elastic cartilage. ठीक है बच्चे? It is the elastic cartilage. It is the elastic fibrous cartilage. ठीक है बच्चे? ठीक है? So fibrous cartilage में there is another type, that is your white fibrous cartilage. So if it is white fibrous cartilage, this is important. It is written in other chapters. So white fibrous cartilage. So white word is there, means collagen. So you remember intervertebral discs. In our vertebral column, we are having some discs in between, right? That intervertebral disc is the example here, Leharika. Intervertebral disc is the example here. Okay? So here, what's the point, right? So basically, your white fibrous connective tissue is converted to this. Okay? It is converted to cartilage. Okay, bache? So it is the strongest cartilage. It is the strongest cartilage. Okay, this white fibrous cartilage is the strongest cartilage. Okay, so pubic symphysis is also the example here, and perichondrium, that is the outer covering, it is absent here. Yes, examples are important. So all the questions are PYQ. You can check it. You can check it even after the class. Okay, you can check it even after the class. Understood? Sure? Now let's move to the bone part. So when you talk about the bones, direct question, outer covering is periosteum. Okay? And you know that in the bone there is inner covering also, जैसे endosteum, and that endosteum is lining bone marrow cavity. That endosteum is lining bone marrow cavity. Okay? So बाकी you know that matrix is present, right? They are having salts. जैसे cartilage में chondroitin sulfate is there. Which salt is there? Chondroitin sulfate. Here calcium salts are there. Okay? In the bones, which salts are you going to get? You are going to get the calcium salts. Okay? Inorganic, organic compounds are there. So calcium salts will be there. Isn't it? Isn't it? Isn't it? Right? Or in the cartilage, chondroitin sulfate is there. If I talk about the bones, the protein is osine. So that's your wish to remember. Right? So bone marrow cavity, endosteum, matrix is present. This is the periosteum. So when you take the section of a bone, internal section of a bone, जैसे cartilages में if there is injury, it takes a lot of time to heal because they don't have the blood supply. But here you will see. So what's the point? When you take the section of the bones, you will see some longitudinal canals. जैसे the diagram which is in NCERT, na, you see the top view there. You see the top view there, that canal, that layers. So when you take the longitudinal section, you take this longitudinal canal. This is actually the Hebercian canal. This is actually the Hebercian canal in bone. So it is having all that blood supply and all. So let's say this is one Hebercian canal, this is another. So in between, you will see that there is a H-shaped canal. There is a H-shaped canal. This canal is linking one Ebersian canal to another. So this is transverse canal, also known as Walkman's canal. Transverse canal, also known as Walkman's canal. So it is just having that blood vessel, nerve vessels like this. Okay, blood vessels and all. Okay, so it is going to have what? It is going to have all that blood capillaries, nerve fibers and all. Okay, understood? Understood? Sure. And moreover, one more thing, osteoblasts, they are the bone forming cells. Again the story is same here. They are the bone forming cells. Osteoclast, bone destroying cell. Osteocytes mature when these are mature osteoblasts. This is the story that you need to remember, and your homework is to check the difference in red bone marrow and the yellow bone marrow. Yeah, you will get a break. You will get a break. Let me come to the NCERT. You will get a break. So the intercellular material of cartilage is solid and pliable. Pliable means flexible. So it is pliable. You need to focus on this point. It is pliable. It resists compression. Cells of this tissue, chondrocytes. I told you chondrocytes are present here. You should not mention chondroblasts. Chondrocytes, they are the mature cells. This is what you need to note down. Okay, so most of the cartilage in vertebrate embryos are replaced by bones. We know it. So tip of the nose में yellow cartilage. Outer ear joints में that yellow fibrous cartilage. Adjacent bones of vertebral column में intervertebral disc is your white fibrous cartilage, and then ear joints and adjacent limbs and hands are there. So bones में they are hard, they are non-pliable, right? They are not flexible. Calcium salts are there. Collagen fibers are present there. ठीक है बच्चे? So again cells are there. So I told you already. Baki blood is a fluid connective tissue having formed elements and all. This will be covered in body fluid and circulation. Okay, so next is the muscles part, and you know that we have to discuss the same in the body fluid and circulation. So now I'll be providing you a break. So after that break, let's come back and let's continue. Right, after that break, let's come back and continue. ठीक है जी? Okay, so it's 2:18. Done? Chalo, let's go. What's up? Ma'am, I love you. Thank you, Ji. Same to you. And I just got a message on my Telegram that thank you so much for ma'am for these notes. Thank you so much, Bache. Thank you so much. So keep studying, keep revising, and trust me, we are going to complete the human physiology. My main target today is human physiology. Remember, I took a poll on the channel itself and in our Telegram group also, where I was asking that what do you want me to complete. So why I was asking human physiology, because if you will give 12 hours to human physiology means you are going to cover each and every minor detail. Okay, so then no one can stop you to score full house in your human physiology portion. Okay, chalo, so let's start, people. What are we going to start now? What are we going to start? I hope you guys have seen the Telegram group. So we will be starting the locomotion and movement. I'll tell you why. Right, dekho. Is it mandatory to follow that sequence? Is it mandatory to follow that sequence? Ki chalo ji, firstly let's do breathing and exchange, then go for this, go for that. Nahi na. Nahi na. Sri Hasini, yes, it is as per the guess. I will not say exactly they will be repeated. No, not at all. But it is as per the analysis of the previous year paper. Okay? So what am I saying? Yes. So I am saying that there is no need to go with the sequence, right? So let's, because in structural organization we are having the muscle tissue part. Okay? We are having the muscle tissue part here. So what will we do? Let's start the muscle tissue part from this side and then let's relate it with the locomotion and movement. Okay? So in one go we will finish it. What say? So that in one go we will be able to finish it. Is that fine? Tell me, is that okay? Is that, yes, is that okay for all of you? है ना? I think that's fine, right? Chalo, let me see the points from NCERT that we need to pick. Okay, let me check the points. Okay, let's get started, people. So let's target the limb part first. Muscles. Are they important? Are they important? Muscles. Are they important? Please explain. Please explain. Dr. Secret will finish human physiology, then biomolecules, then animal kingdom. So let's speed up. Okay? So guys, before starting this portion, let me tell you one thing. When it comes to locomotion and movement, now I am relating structural organization with locomotion and movement. So when you talk about the locomotion and movement, I hope you guys have seen the top 60 topics of biology video that I have posted on Unacademy NEET English. I think you people should definitely check it. So locomotion and movement may say the very important is your sliding filament theory, right? This is something that you should never ever leave, and for this particular year, this is my hunch that question will come from the examples of the joints. So I believe these are the two most important topics for this particular year. Okay, these two are the most important topics that you should not at all miss. Okay, though we are going to complete the entire portion, but still. Now we are talking about the muscles. Okay, let's discuss the muscles here, then let's move to the locomotion and the movement part. So what are we discussing? We are discussing about the, yes people, what are we discussing? We are talking about the muscles here. So muscles, as I said, mesodermal in origin. What are they? It is mesodermal in origin. Exceptions are there. You can note down that exception. There are, right. So exception may iris muscles. Iris muscles. Muscles of iris. Iris, you know na, this colored portion of our eye. So iris muscles, they are ectodermal in origin. Iris muscles, they are ectodermal in origin. Okay, so this is an exception. Otherwise your muscles, they are mesodermal in origin. This point clear? So each muscle is made up of long of many long cylindrical fibers which are arranged in a parallel array. So muscles are having what? They are having the fibers, they are having the fine fibrils. So now let's discuss the structure of skeletal muscle. Okay? Let's talk about the structure of skeletal muscle, then we will talk about the difference. Is that okay? Or you want me to start with the difference directly? Tell me. Or you want me to start with the difference directly? Chalo, I'll tell you how are we going to discuss this. Chalo, firstly let's discuss this part from the NCERT only. So they are saying when it comes to the muscles are having fine fibrils. The word is not fiber, it is the fine fibril. Actually muscles will have muscle bundles, they will have muscle fibers. Fibers are going to have fibrils, they are obviously made up of proteins. The word used is myofibrils. Okay? So muscle fiber, they contract. Of course this is the property of the muscle. They will contract, they will shorten when they will get the stimulus, and then they will relax, they will come back to, right, they will come back to their uncontracted state, they will lengthen. Understood, bache? So this thing will be done in a coordinated manner. The contraction, relaxation, right bache? Right? So they are saying, so their action moves the body to adjust to the change in environment and to maintain the position of different bodies. So we are having three types of muscle. The first one is skeletal. Direct MCQs, I'll tell you. They can ask you which muscles are having. Listen to the questions. Which muscles are having intercalated discs? Which muscles will never undergo fatigue? Which muscles are multinucleated? Which muscles are fusiform? Which muscles line the hollow organs of your body? Which muscles will undergo rhythmic contraction and relaxation? Which muscles will help in the childbirth? Which muscles will help in peristalsis? These kind of questions will come in your paper. Okay, these kind of questions. Okay? Understood? So that type of question will repeatedly come. They can ask you which one is cylindrical, which one is fusiform, which one is branched, which one is unbranched. That type of questions will come. So now when you talk about the skeletal muscle, obviously why are we calling them as skeletal muscle? Because they are associated with the skeletal system. Okay, bache? Right? So they are, right? So these are the typical muscles of biceps. They are striated. Striated means striped, means they will have these lines. I'll explain why these lines are there, but this is what they are going to have. And muscle bundles, they will be parallel together. I will explain it, just allow me some time. Okay, now this is the diagram. One question will for sure come from diagram. So this is of course the skeletal muscle. So skeletal muscle mein you can see these striations, you can see these bands, you can see these lines. Understood? Even that type of striations are also there in these muscles. They are the cardiac muscles. What are they? They are the cardiac muscles. But such type of striations are not there here in the smooth muscles. You know that? So they are fusiform. Fusiform mane they are spindle shaped. Okay, fusiform mane they are spindle shaped. What type of shape do they have? They are spindle shaped. Understood? So that's what you need to observe. Now here, if you will look at this diagram, I'm teaching you very specific points. So you pay attention. So junction between adjacent cells. Jaise dekho, cardiac muscle. Obviously it is associated with heart. It will help in the heart contraction and relaxation. These muscles, they work together as a unit. Work together as a unit means, yes people, these muscles, they work together as a unit means, like they are like, whenever a stimulus will come, they all need to contract together as a one unit. So at points they are fused. Okay, at points they are fused. They are having the junction. So that all the muscles will receive that stimulus and they will contract together as a unit. So when you talk about the cardiac muscles, this is what you are going to see. These junctions are basically the intercalated discs. Okay, these junctions are basically the intercalated discs. That's what you need to keep in your mind. Clear, bache? So come to this part. So smooth muscles, they taper at both the ends. Means like this. They are fusiform. They are spindle shaped. Right? They are fusiform. They are spindle shaped. Okay, so one question was there in this part from NEET 2021. Yeah, that which muscles they line the hollow organ. So in NEET 2021, one question was there from this part. So these are the muscles which do not have striation. Cell junctions will hold them together. They are bundled together in a connective tissue sheath. Now, please pay attention. Pinky, can you see this? The word is connective tissue. Sheath is going to cover the bundles of muscle fibers. So sheath mane, it is a dense, irregular tissue. sheath mane it is a dense irregular tissue. If it will be the cord, it means it will be dense regular tissue. It will be dense regular tissue. So wall of internal organ, you can still check the paper of NEET 2021. This question has been asked there. The wall of internal organs such as blood vessels, stomach, intestine, they all are having this muscle, right? They are. So you know that they are involuntary, means you cannot control them with your own will. It's not like that you are having pizza and then suddenly you are realizing, no, I want to throw it out here, I want to throw it out. Okay, so it is not going to happen here, right? That once you will eat it, once peristalsis will start, it will reach in the stomach. So if there will be voluntary, you can say muscle, stop, bring it back, bring it back. Of course, so it will not be controlled with your own will. Take care, bache. Okay, so smooth muscles, remember they undergo fatigue but at a very slow rate. Jaise your cardiac muscles, they will never undergo fatigue. Your smooth muscles, they will undergo fatigue. But at a slow rate. It takes time. Skeletal muscles, they immediately undergo fatigue, right? We will discuss about the fatigue as well. So Jay, if you will not stop spamming, I'll ask my team to block you. Done? I'll ask my team to block you, so you better stop spamming. This point clear? This point clear? Okay, this point clear? So smooth muscles, they will undergo fatigue at a slower rate, right bache? Right bache? So they are right, so you know their function, isn't it? You know their function. Done bache? Done. So they are also what? They are fusiform, so of course they are unbranched, and even your skeletal muscle, they are also unbranched, right? They are also unbranched. They are cylindrical, but they are what? They are unbranched. Okay, okay. And this one, your cardiac muscle is branched. Cardiac muscle is branched. This is what you need to remember. Okay. ठीक है बच्चे? So cardiac muscle, it is the contractile tissue present only in the heart. Okay, so cell junctions, they fuse the plasma membrane of cardiac muscles and make them stick together. And you know that these communicating junctions are nothing but the intercalated discs. So it is also a previous year question. Okay, so they will not undergo fatigue at all, and they will always be functional. You know that if cardiac muscles will stop functioning, our heart will not contract or relax. And due to which what's going to happen? Right, obviously the pumping of the blood will be affected, and of course our organs will not get the oxygen. The problem will be there. So this intercalated disc point is very important. Now why I just said that I will start this chapter, देखो neural tissue में there is nothing much, you know it already. So why I was saying that I will start this chapter with locomotion and movement, I'll tell you the reason. Now because you are reading about the muscle, okay, of course you are reading about the muscle, so you can, in that sync itself we can complete this portion, है ना? In that sync itself we can complete this portion. So let me open the NCERT Didi. Where is our NCERT Didi? Okay, अरे यार, I'll share the PDF with you all so that at the end you will, you know, get some time for the quick note revision. But yeah, you should make your notes. Okay, you should make your notes. आ जाओ दीदी, आ जाओ. Okay. Done. There are two words. So guys, first of all, tell me, are you excited or excited? Anurag, I too did same, but tell me in English. Acha, chit chatting is going on. कोई बात नहीं. In the paper you will not be allowed to chit chat. Then you will cry. Guys, are you ready? Excited? If you are not, then tell me here. I'll end the session. Even I am not interested then. In teaching you. More fire emojis here. I deserve that. Yeah, NCERT Didi is here. Ma'am, we are and we should be interested. Very good, Leharika. Guys, some more, and hit the like button. Yeah, it doesn't mean that if I said 1k, then you will keep it at 1k only. It should be 1.5k at least. It should be 1.5k at least. Okay, so let's start the locomotion and movement. This is the first chapter of human physiology that we are going to start now. After it, let's go for the excretory products, chemical coordination, neural, and then let's start the breathing and body fluid, because that are the chapters that even we have discussed before. So let's start it. Let's make locomotion and movement easy for you. So sliding filament theory, joints, very important topics. When it comes to muscles, we will remember them in a fun way and you are going to love it, people. So let's talk about the locomotion and movement. So when it comes to the movement, yeah, it is the significant feature of the living beings. And I'm not going to just give you the feel that I'm teaching you, I'm actually teaching you, people. So make sure you are making the notes, make sure you are revising everything. I'm not just going to target the most important topics. Instead of that, I will even tell you that yeah, from this line that type of question can come. So please pay attention, share the session link with your friends, invite more people, yeah. Invite more people, we will have better competition. So when it comes to the movement is what? Movement is basically, right, when it comes to the movement is basically a very significant feature of living beings. You will see that living beings, they are moving, even if they are sessile, even if they are attached to substratum, the things will move in their body. A little movement will be there even if they are attached to that place. Okay, so it is something very significant. Okay, so animals and plants, they are having different range of movements. Obviously, just say if I talk about a eukaryotic cell, in the case of a eukaryotic cell, right, the cytoplasm here, it will also stream. Cytoplasmic streaming is there, yes or no? You are having cytoplasmic streaming, yes or no? And cytoplasmic streaming is known as cyclosis. So that is also a type of movement. ठीक है बच्चे? So streaming of protoplasm in the unicellular organism like amoeba, it is also a movement. जैसे I told you, eukaryotic cell, you can pick up the example of amoeba. So cytoplasmic streaming, cyclosis. Cytoplasmic streaming, cyclosis. ठीक है? So cilia, flagella, tentacles, they also show the movement. जैसे hydra, let's take the example of hydra. So hydra, in the case of hydra, this is what we see. We see that yeah, there are the tentacles, so that tentacles are also going to show the movement. Let's take your own body. You know that trachea is lined with pseudo-stratified ciliated glandular columnar epithelium, so that cilia will show movement. Fallopian tubes mein movement is there, yes or no? Yes or no? Right bache, right. So we are moving our limbs. Jaise I am doing this, okay, I am doing this, it is what? Movement. And which muscles are helping here? Skeletal muscles, right? Because it is working with my own will. Okay, it is working with my own will. So movement, okay, movement. What is this? Skeletal muscle. Skeletal muscles are associated. This is voluntary movement. We can move our eyelids, the tongue, etc. ठीक है बच्चे? ठीक है? So when any movement by which we are changing our location. So these are the movement, right? Things, food is moving within our body, right? The cilia movement, this movement, that movement. Movement. In general, it is movement. Now, because of some movement, if I'm changing my position, like I was here, now I am here. ठीक है? This is locomotion. This is locomotion. So any movement which is changing my location, that is known as locomotion. ठीक है? That is known as locomotion. जैसे walking, running, climbing, flying, swimming even, that is known as. ठीक है बच्चे? So other than that, they have given some examples. जैसे paramecium में cilia will help in the movement of food through the cytopharynx and in locomotion as well. Remember your paramecium, right, which is the slipper's organism, it is having the cilia. So cilia will help in its locomotion, and moreover it will slide the food over this cytopharynx. You remember paramecium? So paramecium is what? Is it a prokaryotic cell or eukaryotic cell? Tell me. Paramecium, what's that? Is it a prokaryotic cell or is it a eukaryotic cell? Guys, it's a very simple question. I'm expecting an answer. Is it a prokaryotic cell or is it a eukaryotic cell? Thank God, it's a eukaryotic cell. It's not a prokaryotic cell, people. It's a eukaryotic cell. It belongs to kingdom Protista. ठीक है? So, right, so cilia, so hydra के case में tentacles are there. So this is about the movement. So yes, we can say that all movements all locomotion are movement, but all movements are not locomotion. Listen to me very carefully. All movements, all locomotions are movement, but all movements are not locomotions, isn't it? Isn't it? All locomotions are movement. It is the type of movement only, but all the movements are not locomotion, because locomotion is something by which we will change our position. Okay, okay. So these locomotion and the ways of that, it depends upon our habitat, our level of complexity in our body, and locomotion is very important to find out the food, shelter, protection, reproduction. There are various things. Okay, so they are talking about the types of movement. So we are going to pick up the gist. So are you ready? Are you ready? You have your pen paper? You have your pen paper? Are you ready to make the questions? Okay, so the very first thing that we have discussed here is, right, amoeboid movement. The very first thing that we are discussing here is amoeboid movement, right, bache? It is amoeboid movement, right? Amoeboid. So amoeba, it is irregular in shape. So if you talk about your WBCs, if you talk about macrophages, that is how they move, right, with the help of pseudopodia, right, with the help of pseudopodia. So when pseudopodia is there, so that's why we are saying amoeboid movement. Now I'll highlight one more thing here, bache. Just when you talk about the amoeba, right, in general, when you talk about the amoeba, so you know that pseudopodia is what? Pseudo mane false, pseudopodia. So false feet it is. How the pseudopodia will form? Bache, below this plasma membrane here now, you will see actin filament. So this is the cell skeletal element. This is the cytoskeletal element which helps in that. This is the cytoskeletal element which helps in that. So pseudopodia, actin filament. Pseudopodia is formed by actin filament. So actin filament is also known as microfilament. So this question can come in your paper. So your macrophages, your leukocytes, right, they will show amoeboid movement. So amoeboid movement means pseudopodia is there. Okay, bache? And how that pseudopodia will form? With the help of microfilament. Microfilament mane actin filament. Microfilament mane actin filament. Understood? Understood? Ciliary movement, when cilia is moving. So you know that what is cilia? Cilia is that cylindrical process that is arising at the free end. So it is motile, it is contractile. It is motile, it is contractile. So it lines the inner tubular organs, right? Right, so coordinated movement of cilia in the trachea help in removing dust particles. Passage of ova of the female reproductive tract is also facilitated by this. So in NCERT this thing is hidden. No doubt ciliated epithelium is lining your fallopian tube, ciliated epithelium is lining your trachea, but you should know which type of cells are doing it. Is that clear? Which type of cells are doing this? Is that clear? Is that clear? So when we move our limbs, jaw, tongue, muscular movement. So muscles are involved there. So when you talk about the muscles, they are having the property of contractility, means they can contract, right, they can reduce their size, plus they will again come back to their size. We will discuss about the muscles property. ठीक है बच्चे? So locomotion require a perfect coordinated activity of muscular, skeletal and neural system, right? So this is what we have to discuss here. So let's start the muscles. Okay, let's start the muscles in detail. So they are also talking about the flagellar movement. You know na flagella? So in eukaryotes, right students, in eukaryotes, you know that flagella is having 9+2 arrangement. And if you talk about prokaryotes are having very simple type of flagella, and that is made up of flagellin protein. Okay, right? So flagella is very simple in prokaryotes, that is made up of flagellin protein, but in eukaryotes it is 9+2 arrangement of microtubules. So microtubules, which itself they are made up of tubulin protein. Okay, so that is also an important difference. Fine, bache. So you know that flagella movement we will see in the sperm. Okay, even in the sponges, canal system, you remember? Canal system of sponges. Anyone in the class? Bache, I am decoding NCERT here just because of you, otherwise I'll write it in a free flow. I know you people don't pay attention on the words, on the lines written there. You don't link it with another chapter. So that's something very important. Kindly pay attention. So we are saying that flagellar movement, yeah, we know in the case of sperm. And even in the case of canal system of sponges. So question can come from this part for sure. So when it is a canal system, you remember choanocytes, Do you remember choanocytes, collar cells? Choanocytes, collar cells, they are the flagellar cells. They are the flagellar cells. So it is the flagella that will maintain the water current there. So choanocytes, they will line the choanoderm. Right, bache? Right, bache? So now when you people talk about the euglena, euglena is what? Spindle shaped organism. Spindle shaped organism. It is also having the flagella, but it is having two flagella. One is reduced, very small in size, one is longer. So spindle shaped euglena is there. Take a spindle shaped euglena is there. So now when you talk about the muscles, so study of muscle is known as, write down all these points. Study of muscles is known as myology. Study of muscle is known as myology. So write down myology word in the chat section, everyone. It's not mycology. Mycology is something else. Right? Mycology is something else. Mycology is study of fungi. I am saying muscle. Myology. M-Y-O. Okay. Myology. Okay. So 40 to 50% of the body weight, 40 to 50% of the body weight is due to the muscles, bache. So these are the properties that you all need to underline. Right? Excitability, contractility, extensibility, and elasticity. So explain the meaning of these properties to me. Excitability, contractility, extensibility, and elasticity. Yep. Explain the meaning to me. These are the properties of the muscle. These are the properties of the muscle. Excitability, obviously it can receive stimulus, but after receiving that stimulus it will be excited, right? It will contract, it can reduce its size, right, bache? Then moreover it can come back to its original size as well. It can come back to its original size as well, isn't it? Isn't it? So explain the meaning to me. Explain the meaning to me. I'm telling you the things I'm telling you these things in journal. So you have to explain the meaning of extensibility. Yes, you have to explain me the meaning of extensibility. Quick. Quick, quick, quick. Quick. Exactly, देखो, excitability, you know that. Contractility means it will reduce its size, it can get it will be contracted. Extensibility means its ability to stretch. Muscles can stretch themselves, plus they are elastic. You cannot say that they are fully elastic like a spring, but still they have the property of elasticity. So you need to remember this. Okay, you need to remember this. So it is E3C, right? When it comes to the property of muscles, it's E3C, right? Or you can call it a CE3, CE3, whatever you want to say, right? Whatever you want to say. So C means contractility, E means excitability, extensibility, and yes, excitability, extensibility, and elasticity. Okay, so C3, EC3, whatever you want to remember, it's your wish. It's your wish. So you know that we have skeletal muscles, visceral, and the cardiac muscle. So visceral muscle mane your smooth muscles, right? Smooth muscles. So see, this is something common. It is also there in structural organization. So it will line the hollow visceral organs. It is going to line the hollow visceral organs, right? Hollow visceral organ mane alimentary canal, your gut, it is hollow within. Reproductive tract, you know na reproductive tract mein which part? Do you remember reproductive tract, right? Reproductive tract mein uterus, uterus mein myometrium mein we see this muscle, right? In uterus and myometrium we will see this muscle, right, bache? So they do not have striation, they are smooth, they are non-striated, right? They are involuntary. Clear, bache? They are involuntary. So transport of food, right? Jaise, you know na peristalsis, rhythmic contraction, relaxation, right? That will help in transport of food. That is also because of smooth muscles. And even the gamete movement is also through these smooth muscles. That's what you need to remember. Next is your cardiac muscle, muscle of heart. I told you, you will see the branching pattern here, plus they are also striated, and very important is intercalated disc. So cardiac muscles are having intercalated discs and they are also involuntary. जैसे sometimes we feel like ना, oh my god, I don't want to live, I wish to die. होता है? Yes, it's obvious ना. Sometimes we feel like, अरे यार, I don't want to live, I wish to die. Right? So imagine if our cardiac muscles will be voluntary, then what is going to happen? One day you will wish, heart, stop working, no need to beat. But that is your low movement. Maybe after that you want to live. So if you will be like, "Yare yaar, heart, stop beating, I want to die," heart will actually stop beating and you will be tata bye bye, because brain will not get the oxygenated blood. No, okay? So it's good it is involuntary. It is good it is involuntary. Otherwise we will die, I think 10 to 20 times every day. है ना? We will die 10 to 20 times every day. कि चलो यार मर जाते हैं, let's die. है ना? Let's die. So that's what you need to remember. Okay? So now we will be discussing about the skeletal muscle. So understand the structure properly, rest everything will be clear. Skeletal muscles. First of all, what are we talking? Ultrastructure. What are we going to talk about? Ultrastructure. Right? What are we going to talk about? Ultrastructure. And I am not going to use any word other than NCERT here, but there are other words also, but here I will not be using it. Okay? So first of all, when you talk about a muscle, in that muscle you will see many muscle bundles. What you people are going to see? You will see many muscle bundles. What you people are going to see? Yes, people? You will see many muscle bundles. जैसे let's say it is the side view, you are looking at it like this. जैसे sometimes we have that pen na, so many pens are there in a box. So if you will look at it like this, so see, these are the muscle bundles, right? These are the muscle bundles that are making a muscle. Are you able to understand that? Right? So this is a kind of side view that I'm showing you, and this is like this. Yacha, it is okay. So many muscle bundles are there. So when you talk about these muscle bundles, for these muscle bundles we use a word. Okay? We use a word for these muscle bundles, and that is fascicle. Again I'm mentioning, I'm not going beyond NCERT. So it is fascicles. What's that? It is fascicle. So everyone write down this word in the chat section. Everyone just write down this word in the chat section. Right? We are discussing the ultrastructure of skeletal muscles. Right? We are discussing the ultrastructure of skeletal muscle. So they are having so many muscle bundles. Muscle bundles are known as fascicle. Now I want to join these muscle bundles. Right? I want to link these muscle bundles. Here you all should join. So when it comes to the linking and joining, what do we have? When it comes to the linking and joining, what do we have, people? Hanji, tell me, what do we have? What do we have? Obviously the connective tissue. Just let's say this is a muscle, this is one muscle bundle, it is attached to another, right? So obviously they will be having a connective tissue sheath. Again, the word that I'm using is, right, they will be having the connective tissue sheath, and that is known as fascia. So fascia is going to link them. Right? Just say you have so many pens, you are holding them all together. So connective tissue sheath is fascia. Now when you look at these muscle bundles, they are further made up of, each muscle bundle is further made up of muscle fibers. Okay? Each muscle bundle is further made up of muscle fiber. So anatomical unit of muscles is this muscle fiber. Do you know that? Do you know that? Yes, people? Yes, yes, yes. Are you getting my point? So this is the kind of anatomical unit of muscle, right? It is the anatomical unit of muscle. Okay? Muscle fiber. Understood? So let me pick this. Just say, let's say this is your one muscle bundle. So this muscle bundle is having many muscle fibers. Okay? This muscle bundle is having many muscle fibers. So these pinky structures, they are what? They are the muscle fibers. So you can mention it in your notes that these muscle fibers are nothing but the anatomical unit of muscle. What are they? They are anatomical unit of muscle. What are they, people? They are anatomical unit of muscle. Right? The structure of muscle is made up of this. This is the anatomical unit of muscle. Now when you look at this muscle fiber, you look within it, so you will see some parallel. You will see some parallel fibrils. These are actually the myofibrils. These are actually the myofibrils. right? These are actually that proteinaceous myofibrils. Are you getting my point? Are you getting my point? So let's say if this is the anatomical unit of muscle, okay, we are calling it as muscle fiber. This is the anatomical unit of muscle, we are calling it as muscle fiber. So its outer covering is sarcolemma, right? Its cytoplasm is sarcoplasm. Okay, so endoplasmic reticulum present here is, right, sarcoplasmic reticulum. It is sarcoplasmic reticulum. Okay, and you know that the sarcoplasmic reticulum is a storehouse of calcium. Okay, it is a storehouse of calcium. Understood? It's a storehouse of calcium. Understood? Understood? So muscle fiber, sarcolemma, sarcoplasmic reticulum, sarcoplasm is there. Sarcoplasm is there. This part clear? This part clear? Are you sure about it? Now I'll mention one, as I mentioned, that here in these muscle fiber, you will see some parallel, parallelly arranged fibrils. They are the myofibrils. So myo word is associated with muscle. Now at places you will see, that the sarcolemma, it shows some invaginations. At places you will see that the sarcolemma is having some invaginations. Right. We will discuss about this. Okay? So at places it shows the invagination, and you call these invaginations as T-tubules. At places sarcoplasmic reticulum, just imagine this is the sarcolemma. At places you will see the infoldings are there, right? And you are calling it as T-tubule. So up to this part all clear? Up to this part all clear? Are you sure about it? Up to this part all clear? Are you sure? Are you sure? So somewhere here you are going to find the sarcoplasmic reticulum. That's what I can say. Now let's focus on this myofibril, right? So when you look at this myofibril, okay, when you people look at this myofibril, so can you tell me what is this myofibril? Anyone in the class? Can you tell me what is this myofibril? Myofibril is having some alternate dark and the light bands. Myofibril is having some alternate dark and light bands. Please pay attention. Myofibril is having some alternate dark and light bands. Dark and light bands. Trust me, if you will understand this part once now, that's why I'm going slow, because question will come from this part. The structure of sarcoplasmic reticulum clear? Sorry, the structure of sarcolemma clear? Sliding filament theory is clear. That's what you have to remember. Sliding filament theory is clear. So can you see this? Can you see this? What is it? It is the muscle. It is the myofibril. Myofibril is having alternate dark and the light bands, right? It is having alternate dark band and light band. So students, today promise me, if this concept is clear now, then yes, right? If you are reading this concept for the first time or even for the 10th time, but today you believe ki yeah, she made it clear, right? She made it clear. We are able to understand everything. Then you are going to write a comment in the comment section. What say? You are going to mention it in a comment section. What say? Commitment, pakka, promise, promise. ठीक है, चलो आ जाओ. So myofibril, what I just said, it is having alternate dark and light band. Dark A, light, light. Can you understand that? Dark, arc A, light, I. So one is the dark band. ठीक है, dark band. Another is the light band. One is the dark band, another is the light band. So it is also known as A band and I band. A band and I band. So A here means anisotropic. You know it better. Anisotropic. And when you people talk about the I band, it is known as isotropic. Okay, it is known as isotropic. So light, I. Okay, light. So here you will see actin filament. Which filament will be there? Actin filament. It is the thin filament, right? Actin filament mainly, and it is forming the thin filament, right? Actin, thin, I, all are associated. Then dark band, A band. So usually in our dark phases, our friends, they stay with us, yes or no? Right? If you have good friends, they will stay with you in your dark phases. Am I right? If you have good friends, they will stay with you in your dark phases. So when it is the dark band, you will see myosin filament here, and you will also see actin. Majority of part is of myosin, M for mota, right? So thick filament, it is thick myofilament, okay? And actin is also there in A band. I know some students will be like, oh, what are you saying, ma'am? In the dark band, how can you say that actin is there? But it is there. It is there. Let me explain that. It is there. Okay? So look at this. This is your I band, right? Alternate dark and light band is there. So I told you that actin is thin. I told you that actin is what? Actin is thin. Actin is thin. So when you look at this I band, right? So it is bisected by a Z line, a Z line. So when I'm using the word bisected, so it means it will divide it into two equal halves, yes or no? Z line bisect I band. Okay, you can even write down. Now this is the I band, so it is bisected by this Z line. So alternate I band, A band, I band. Now this complete portion is of A band. So when you look at this A band, in the center you will see one M line. And here in the A band, you will see there is a place where actin and myosin, they are what? Actin and myosin, they are overlapped, and in between only myosin is there and it is not. Overlapped by actin. So it is overall the A band where actin and myosin is also overlapped. That's why I mentioned that when it is the A band, no doubt majorly it is having myosin, but part of actin is also there in that A band. And the part of the A band, right, which is not overlapped by myosin, it is known as H zone. Henson zone. So what is H zone? What is this H zone? Part of A band where myosin is not overlapped by actin. Are you getting my point? Myosin is not overlapped by actin. Is that clear? Sure. So H zone clear, A band clear. Now I'll tell you Z line. So two Z lines are there. So Z line, do not ask me what is the meaning of Z, right? It will be confusing for you. So it is the elastic fiber. It is what? It is basically the elastic fiber that bisects the I band. And when it comes to the M line, it is a thin fibrous, right? So the thick filament in the A band, they are held together by this thin fibrous membrane, right? This is the thin fibrous membrane, right? So all this A band, these myosin of A band are held together by this thin fibrous membrane. This part clear? So here, if you look at this diagram, let's recall myofibril having alternate A band, I band, alternate A band, I band, alternate A band, I band. Am I right? They are having what? They are having alternate A band, I band, alternate A band, I band is there. Am I right? So now when you look at the I band, it is bisected by Z line. This is what I just said. Now I'm going to take the portion of these two things. I'm going to take the portion in between two successive Z line. So right, so the portion in between two successive Z line, it is the sarcomere. ठीक है बच्चे? ठीक है. So portion of myofibril, between two successive Z line is sarcomere, and sarcomere is the functional unit of contraction. It is the sarcomere, and this sarcomere is the functional unit of contraction, because during contraction it is the one that will undergo, it will contract. Okay, so what is this sarcomere? It is the functional unit of contraction. It is the functional unit of contraction. Understood? Understood? This is the sarcomere. So portion of myofibrils between two successive Z lines. So what was there in two successive Z lines? What was there in two successive Z lines? Can you tell me? What was there in that two successive? Successive means one after another. What was there? If you will look at it, you will say this portion of I band, this portion of I band, and this A band. So can I say that two half I band and one A band makes a sarcomere? Can I say that two half I band and one A band makes a sarcomere? Can I say so? Can I say so? This is the overlapping zone. Two half I band and one A band. One A band. And see, this myosin, which is thick, it is also attached to this Z line. It is also attached to the Z line with the help of a protein, not given in NCERT, but I think you should know that. So this is the titin protein. So with the help of this titin protein, these myosin, they are attached to this Z line, right? This is just extra information. So this part clear, this part clear, this portion clear, the structure of sarcomere clear, the structure of sarcomere clear, muscle bundles clear. ठीक है. So now let's discuss. ठीक है. Now let's discuss what? Let's discuss the thick and thin myofilament. After that, sliding filament theory topic will be over. You are going to love this. Trust me, you are going to love this. So after completing this chapter, let's start chemical control and coordination. Then let's start excretory products, neural control. Neural mein there is nothing much. I need to explain you the ionic theory. Brain is rest, everything from the NCERT. Then let's go for the breathing and exchange body fluid wala part. ठीक है? ठीक है, फिर we will start animal kingdom and biomolecules. Ready? So up to this part all clear? Up to this part all clear? If there is any doubt, do let me know. Up to this part all clear? If there is any doubt, do let me know. We need to speed up. We need to speed up, guys. Bones तो भाई अभी we'll discuss ना, है ना? So muscle fiber is a syncytium. This is another important MCQ. What is the meaning of syncytium? It simply means that your muscle fiber is multinucleated. What's that? It is multinucleated. What's that? It is multinucleated. Done, bache? Done, bache? Okay. So it is the storehouse of calcium ion. That's what we have discussed. So now what we need to discuss, the thick and the thin myofilaments. ठीक है, जैसे here it is written कि muscle fibers, they are having the myofibrils. So myofibrils are also known as myofilaments, and I told you they have alternate dark and light band. So actin and myosin is present there. So anisotropic band clear, Z line clear. So after the class, just read it from NCERT. There is nothing that you will not be able to explain. So look at the structure here, right? So this is your actin filament. These are your myosin. So where myosin is not overlapped by actin, this is the H zone, Henson zone, right? So this is the half I band, this is the half I band. So two half I band and one A band will form your sarcomere, the anatomical unit. Now the next thing is structure of contractile protein. So the very first thing written here is contractile protein. The very first thing written here is contractile protein, the protein that can undergo contraction, right? The protein that can undergo contraction, yes or no? The protein that can undergo contraction. So we will be discussing thin and the thick filament. So let's start with the actin. ठीक है, let's start with the actin. So when it is actin माने thin actin filament, right? It is making thin myofilament. So बच्चे, when you talk about the actin filament ना, the thin filament, so first of all let me tell you, it is not just the actin protein which is making it. Troponin and tropomyosin is also there. Let me tell you, first of all you will have G actin. You will have which actin? You will have G actin. G माने globular. G माने globular. So of course it's a monomer. What's that? It's a monomer. हाँ जी बच्चे, what is it? It is a monomer. Am I right? Am I right? It's a monomer. है ना? G actin, G actin. So many G actins, they will polymerize. Many G actins. G माने globular actin and it's a monomer. Many G actin, they polymerize, okay, they polymerize and form F actin So what's that F? What's that F? You can also call it as filamentous, or you can also call it as fibrous actin. So G actin polymerize, and magnesium ion is required for it. Right, magnesium ion is required for it. So many G actin, they polymerize and they form F actin, that is your filamentous actin. Right, they form F actin, that is your filamentous actin. So you can also call it as a fibrous structure. Take a fibrous structure. So what's going to happen? Just imagine this is your one fibrous filament, this is your another. That's what you are going to have. Okay, that is what you are going to have. Just let's say this and this. Right, one F actin and another F actin. Right, this is what you have. Right, so they will be helically bound to each other. Can I say so? Two F actin, F mane polymerize, this is filamentous. F actin, they are helically bound to each other. Is that clear? They are helically bound to each other. Is that clear? Anyone in the class? Is that clear? Is that clear? Now here you will talk about another protein, that is tropomyosin. So thin filament is made up of this tropomyosin. What's that? Tropomyosin. So जैसे let's say this is your helical. हाँ, when you talk about these globular actin na people, when you talk about these globular actin na, G actin, so they are having the sites. They are having the sites to bind to myosin. That's what you need to keep in your mind. They have sites to bind to myosin head. Okay, they have sites to bind to myosin head. Is that clear? Is that clear? They have sites to bind to myosin head. Clear bache? Clear bache? Now what's the next point? That's the condition. Okay? Just a minute. Done. So now what's next? So जैसे let's say these are the sites which are present. These are the sites which are present on this F actin that can bind to your what? That can bind to your actin head. So tropomyosin, they'll cover this, right? Tropomyosin also, they will also be helically bound to this thin filament, and that is how they are going to cover it. Is that clear? So two filaments of tropomyosin, they will run close to this F actin bache, right out of its length. So in resting state, tropomyosin molecule, they will be on the active sites. They will be on that sites, right? They will be on these active sites. Just let's say these are the active sites that can bind with the myosin head. So they will be covered with your tropomyosin. Is that clear? So it is also running parallel to this. That's how it works. Okay. So we have another protein, that is your troponin. That is your troponin. So it is a trimeric protein. It's a trimeric polypeptide. What type of protein it is? It's a trimeric protein. So 1, 2 and 3. 1, 2 and 3. So it is thick. What's the trick here? It is thick. This is extra, but it will help you to understand this topic in a better way. So another protein, troponin, is there. So it is a trimeric protein, like this, like this. It will be present at some intervals. It will be present on this tropomyosin like this. Can you see that? So this is your thin filament. This is your thin filament. Now this troponin is basically thick. This is basically thick. Troponin T, troponin I, troponin C. So it is basically thick. It is basically thick. Troponin T, troponin I, troponin C. So troponin T. So T is indicating that it is attached to tropomyosin. Okay, it is attached to tropomyosin. This part of this protein is attached to tropomyosin. I means inhibitory. So it will inhibit this actin to get attached to the myosin. Are you getting it? This troponin I, it inhibits F-actin myosin interaction. Simple. Right? It is also, so one component of this I is inhibiting this actin myosin interaction, and another one is attached to this other components of troponin. So troponin C, it will bind with calcium. It is going to bind with calcium. This point clear? It is going to bind with calcium. So this is all about the thin filament. So any doubt? Any doubt? It is all about the thin filament. Any doubt? Now let's talk about the thick myofilament, that is your myosin. Let's talk about the thick myofilament, that is your myosin. So myosin is also made up of monomers, and here the monomers are meromyosin. Okay? Here the monomers are meromyosin. Here the monomers are meromyosin. Done, bache? So each myosin filament, it is a polymerized protein. Okay? Each myosin filament is a polymerized protein. Clear, bache? Each myosin filament is a polymerized protein, right? And how will it polymerize? Obviously with the help of meromyosin, right? With the help of meromyosin. Clear, bache? Clear. Yes, right? Just let's say if you have meromyosin. So meromyosin is having two components. It is having a globular head and short arm, and it is having a tail. So this globular head and short arm, it forms HMM. What is this HMM? Heavy meromyosin. Heavy meromyosin. And when it comes to the tail, it is LMM, that is light meromyosin. Can you see that? Light meromyosin. So it is something like this. It is something like this. Can you see that? It is something like this. Can you see that? Yes? Can you see that? It is something like this, right? So what am I saying? That it is its globular head, right? Its globular head and short arm, it is making heavy meromyosin. And when you talk about its tail, so its tails form light meromyosin. Its tail form light meromyosin. So when you look at this head now, so here in this head, see, this is the place, this is the place. So this is the actin binding site. Just say actin is having a place to get attached to myosin, so myosin is also having actin binding site. Okay, it is also having actin binding site, and these two are what? These are the ATP binding sites. These are the ATP binding site. So now let me ask you what type of, let me tell you what type of question can come. So they can ask you that when you talk about the myosin, right, which part of meromyosin is having the ATPase activity, right, which is having the ATPase activity. So see, heavy meromyosin, because this is the ATP binding site. So this head is basically an active ATPase. It can hydrolyze that ATP. Okay, so it is an active ATPase, it can hydrolyze ATP. So this is the globular head part, this is a kind of short arm, and then it is a kind of tail. Okay, it is a kind of tail, right bache? Right bache, it is a kind of tail, right bache? Clear? Clear, 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 clear. So globular head and short arm, heavy meromyosin, tail, light meromyosin, right bache? Tail is the light meromyosin, so that is what we have. Okay, that is what we have. So it is an active ATPase because it can hydrolyze the ATP. It can hydrolyze the ATP. Understood? Understood? So many. So imagine so many meromyosins, they will polymerize and they are going to form the thick myofilament. Right? Just try to imagine it like this. So many meromyosin, they are going to polymerize and they are going to polymerize and they are going to form the thick myosin. Can you see that? So if you will imagine it like this, ki haan, theek hai, okay, fine, that small meromyosin, they are getting polymerized. So don't you think at regular intervals their heads will project out like this? Don't you think at regular intervals their head will project out like this? Yes? Don't you think so? Their head will project out like this. Of course they will. Right, of course they will. Right, they will. And now imagine this is the myosin and here actin is coming like this. Actin is coming like this. Hai na? Actin is coming like this. So now what's going to happen? Let me tell you what is going to happen. It's simple. You have a neuron. You know that neuron? Anji, what is a neuron? Don't tell me that you don't know what is a neuron. I hope you know that what is a neuron. Yes, do you know what is a neuron? Do you know what is a neuron? So these are the nerve endings. So let's say this is a muscle. So the junction of this neuron and muscle, it is the neuromuscular junction. What is it? It is the neuromuscular junction. So when the stimulus will come here, but it is a neuromuscular junction, we also call it as motor end plate. Okay, motor junction or motor end plate. So you know that what's going to happen. Here you have the synaptic vesicles, they will release the neurotransmitter. Let's say they are releasing acetylcholine. So you know that this is your muscle fiber, this is your muscle bundle, this is your muscle fiber. I told you at places invaginations are there in muscle sarcolemma. I told you at places, yes bache, Sanjay, at places invaginations are there in muscle sarcolemma. So in its surrounding here, you know what do you see? This is the sarcoplasmic reticulum. This is the sarcoplasmic reticulum. What is that? This is the sarcoplasmic reticulum. So imagine it like this. Let's say this is the T tubule, right? This is the T tubule, and in its surrounding here you will get the sarcoplasmic reticulum. Okay. So these three things are making a triad. These three things are making a triad. These three things are making a triad. Clear bache? Clear bache? Clear? So when the signal will come from this, okay, when the stimulus will come, these synaptic vesicle, they will fuse, right, they will release the neurotransmitter. So that neurotransmitter through these sarcolemma, through these invagination will tell sarcoplasmic reticulum ki bro, please release the calcium ion. So once that calcium ion will release, the sliding filament theory now we have to discuss. Right? But your T tubule is nothing, your muscle fiber. I told you sarcolemma at places is forming some invagination. So this is something known as T tubule. If you don't want to remember it like this, that's totally fine. That's totally fine. Okay. So motor end plate pe neurotransmitters will be released, right? So they will tell this. Through this T tubule, they will tell the sarcoplasmic reticulum ki bro, I need to release the calcium ions. So once calcium ions are there in the sarcoplasm, now the contraction will start. Now the contraction will start. So first of all, tell me, up to this part all clear? So question can come from the diagram also. Question can come from the diagram also. So this is the actin binding side, this is the ATP binding side. So this globular head and short arm, they will form the cross arm. Globular head and short arm, they will form the cross arm. So the diagrams should be very clear to you people, okay? Okay, so mechanism of muscle contraction, it is best explained by sliding filament theory, right? So before explaining sliding filament theory, I will mention few points. You can check PYQs, they are from that points only. And if you will master these points, there is no need, right? There is no need to get worried. You will be able to answer all the questions. So are you people ready? Are you people ready? Yes? Sure? Sure बच्चे? So write down these points. So you will be able to answer all the questions from these points only. Okay? You will be able to answer all the questions from these points only. Yes, write down all the questions will be from these points. Write down. So this is going to happen during muscle contraction. This is going to happen during muscle contraction. Let me explain that to you. But write down these points, then I'll explain it. You will understand everything then. This is going to happen during muscle contraction. Okay? This is going to happen during muscle contraction. Now let's move to the next part. Dekho, what is happening here? What is happening here? Basically from the a signal will be sent by central nervous system by a motor neuron. Why do we say motor neuron? Because we know that we have sensory neurons. You know na, the trick here is ASME. The trick here is ASME. So afferent sensory motor efferent. Remember this? Afferent sensory motor efferent. This is ASME. This is ASME. Afferent sensory motor efferent. So sensory neurons will carry the information for processing to the brain, to the central nervous system, and motor neurons will bring back the processed information to the target organ, to the target tissue. That's the point. Okay? That's the point that we have. So when you people talk about the muscle contraction, as I said, it will be started by the signal sent by the CNS via a motor neuron. Okay, by a motor neuron, a signal will be sent by CNS. Okay, bache? So, motor neuron along with the muscle fiber, you know that they will form a motor unit. I told you motor end plate, neuromuscular junction, yeah, motor unit. Okay? So, you can read it again, people. You can read it again. The junction between a motor neuron and sarcolemma. It should be the sarcolemma of muscle fiber. Okay, it is your neuromuscular junction or motor end plate. So neurotransmitter will be released here and action potential will start, means depolarization will start. Okay, depolarization will start. So it will spread through the muscle fiber and the calcium ions will be released. So once calcium ions will be released, do you remember troponin? Do you remember troponin? I told you in troponin, tick is the point. Troponin में tick is the point. Tick, C. So this part of troponin will bind to the calcium. So once it will bind to the calcium, it will slide. So when it will slide tropomyosin also. So the sites on actin which are going to join with the myosin, they will get exposed. Are you getting my point? Are you getting my point? This is the whole game. What is happening? Calcium ions are in sarcoplasm. Troponin will be like, wow, calcium ions. So troponin will join with that calcium ion. Troponin will be like, wow, I got the calcium ion. So troponin in the excitement will slide a bit. So when troponin, because it is also attached to tropomyosin, so tropomyosin will also slide. So tropomyosin will also slide. So on that globular actin, on that filamentous actin, the sites which can bind with the myosin, they will get exposed. So myosin head and actin will join. Okay? So isn't it amazing love story? People will trying to keep actin and myosin away, but they are like, no, we are meant for each other. We will join. And for that joining, the ATP is also required. Right, the hydrolysis of ATP is also required. Now come to this diagram. So when globular head will hydrolyze ATP into ADP plus PI, means when, this is a previous year question, when to this actin filament, when to this myosin filament, ADP and P is attached. जैसे globular head will hydrolyze ATP, will break it into ADP and PI. This is providing the energy. So when it is hydrolyzed here, your myosin head will be like, oh my god, my actin, please join with me. They will join, and now the structure is the cross arm. Okay, this is the cross bridge. So when the myosin will join with actin, myosin will be like, you please come closer, we'll slide it. Are you getting my point? So myosin will be like, actin, now we are attached. So actin, myosin will slide actin over myosin. So if it will, just imagine, here you are having actin, here myosin. Now myosin is sliding the actin over it. So actin is moving like this. It is coming towards the M line. So in that process, H zone will disappear because we are trying to contract sarcomere. In return, that will contract myofibril. So obviously their length will reduce. So actin. So this question can also come in paper, that which filament will slide over what? Which? So it is the actin which will slide over myosin. It is the actin which will slide over myosin. So it will move it towards the center, then obviously H zone disappear. H zone, what was H zone? Where myosin is not overlapped by actin, because now actin is coming closer. Okay? So H zone will disappear. Understood? So remember, when ADP and PI is attached, then only the cross bridge will form. So once they will be released from this head, this connection, this cross bridge will break, and again new ATP will come. So when ATP join to this myosin head, means this cross bridge formation will stop, because firstly it has to hydrolyze it, then energy will come, then they can join. So that's what you need to remember. This is the sliding filament theory. So after this, see, you have seen the diagram. Now come to this part. Right, so increase in calcium level leads to the binding of calcium with troponin on actin filament. So it will remove the masking of active sites for myosin. So utilizing the energy from ATP hydrolysis, myosin head binds to the exposed active sites on actin to form a cross bridge. Got it? So it will pull the attached actin filament towards the center. So Z line attached to these actin are also pulled inwards. That's why we are saying length of sarcomere decreases, length of your, yes, myofibril decreases, length of I band will also decrease. Right? Just say I band was here. Now you are, right, just say, let's say I band was here. This is the Z line. So when you are contracting it, it will be like this. So initially its length was this. Now it will reduce. That's the point here. Take care. So it is clear that during shortening of muscle contraction, the I band will get reduced, A band will retain the length. Means it will stay like this. So myosin, when it is releasing ADP and PI, it will go back to its relaxed state. So when new ATP binds, the cross bridge is broken. So that's a PYQ. Many students, they marked it incorrectly, right? They were not able to answer it. So when new ATP binds to the myosin head, the cross bridge is broken. Write it down. The cross bridge is broken. Write it down. Quick. The cross bridge is broken. Write it down. That's important, people. That's important. Okay? So when ATP is again hydrolyzed by the myosin, the cycle of cross bridge formation will start again. That's the point. Okay? And breakage is repeated, causing further sliding. So process continues till the calcium ions are there. Right, so this caused the return of Z line to their original position. So you know these calcium ions, they can be resequestered by sarcoplasmic reticulum. Sarcoplasmic reticulum can take it back when muscle contraction is not required. So once it is not there, again muscles will come back to their original position. So this is the point. Now let me teach you it from this part. So what you have to remember, that during muscle contraction, I band ka length, it will reduce. It will reduce. Length of A band remains same. So sarcomere ka length will decrease, myofibril ka length will decrease, and H zone disappear. Mark my words, check the PYQs, you will get a question from this part. You are going to get a question from this part. Now students, they get confused with this. Ki length of actin and myosin, you are saying it's same, and now you are saying ki length of I band is getting reduced. है ना, it can cause the confusion na कि madam you are saying that length of I band it is getting reduced, but here you are saying कि length of actin and myosin filaments it remains same. So what's the point? It's simple, students. It's very simple. जैसे let's say actin filaments are 4 centimeter. ठीक है? Let's say here also it is 4 centimeter. Now when you are pulling them like this, so they will stay 4 centimeter. Initially your I band was capturing this much of length. Just let's say you have in a room, you have 3 chairs. You are putting one chair to one side, another chair, one chair is in center, another chair is on another corner, right? So you are putting them like this, but now you are keeping them together, first, second and third chair. So you can imagine their chair, their weight and everything will remain same. It's just that earlier they were, you know, they were taking, they were in the entire room, but now they are at one place. It's like that. So I band was like this, like this. Now because we are pulling it towards the center, so we will keep it like this. So that's why length of I band will decrease, but overall the length of actin and myosin will stay. Just let's say if they are of 2 centimeter, they will stay like this. It's just that earlier it was placed here, now it is placed here. Simple, right? Simple. So that's the point. Understood? That is the point. Take care. Pakka? Sure? Done? So now let me tell you about the fatigue. Just you know that skeletal muscle, they immediately undergo fatigue. In the case of skeletal muscle, you know that myoglobin is present na? It is not the hemoglobin, they are taking that oxygen from the myoglobin, which is a tertiary protein. So when you are using your muscle excessively, let's say today I have decided that after your class, even if I am going to finish your marathon at 10 PM, I'll go out for a walk. This is what I have decided. Okay, this is what I have decided today, कि that's what I'm going to do. Okay, that's what I'm going to do. So obviously because I'm not doing it daily, so my muscles, I will get the muscle cramps. Why? Because of the accumulation of lactic acid. So when you don't have muscle tone, when you do something, when you use your muscles excessively, you start feeling a pain, that cramps. What are that cramps? That is the lactic acid. How is it possible? Your muscle cells, firstly they will, right, देखो, first step of respiration is glycolysis. You know that from glycolysis 2 pyruvate will form. Now if oxygen is available, that pyruvate will enter in the Krebs cycle, right, and we will be getting carbon dioxide and water. But if let's say when muscles they start working excessively, so instead of entering here, because oxygen availability will be decreased, this pyruvate will start forming the lactic acid. Okay, so when this lactic acid will be accumulated in our muscles, we call it as muscle fatigue. We call it as muscle fatigue. And you know what? The Cory's cycle, right? The Cory's cycle will basically help in that. Now what is going to happen? This lactate या lactic acid from blood, it will go to liver. ठीक है? In the liver, here it will again be converted to pyruvate, and from pyruvate again we will form glucose here in liver, and then obviously the glycogen. Then via blood again this will be carried to the muscles. So this is the Cori cycle where liver and blood, liver, muscles and blood, they are involved. ठीक है? Liver, muscle and blood, they are involved. ठीक है? Okay. So in liver and muscles we will see Cori cycle. So what's going to happen? Muscles में lactate will form या lactic acid. Via blood it will be carried to liver. Liver will again convert it into the pyruvate, and then from this lactate, pyruvate, and finally glucose and glycogen will be formed. So this will release again in blood, and then it will again pass to muscle. So this is the Cori cycle which will operate in your liver and muscles. ठीक है? This is the Cori cycle. It will operate in your liver and muscles. ठीक है? It will operate in your liver and muscles. Understood? So that is how, you know, again our muscles, they'll be like, ठीक है, now I'm normal. Okay? So focus on the NCERT. So repeated activation of the muscle can lead to the accumulation of lactic acid. Why? Because there is anaerobic breakdown of glycogen in them. Right? So basically again and again that muscle is getting activated, means again and again you are doing something, the lactic acid will be accumulated. So muscle me, you know that myoglobin is there. Myoglobin is quite high there. If myoglobin is very high, that will be red muscle fiber. If myoglobin is less, that will be white muscle fiber. So usually in okay, so in these muscles, in these red muscle fibers, right, mitochondria are also plenty. Okay. So mitochondria, which can utilize large amount of oxygen stored in them for ATP production, right? So these are the aerobic muscles. So red muscle fibers, they are the aerobic muscles. They are having mitochondria, they are having myoglobin. So mitochondria can use oxygen, right? So oxygen stored in them for ATP production. ठीक है बच्चे? So the one having less quantity of myoglobin, they'll be pale or whitish. That's all. So remember, in the red muscle fibers, mitochondria are plenty, but they have less sarcoplasmic reticulum, and in white muscle fibers mitochondria are less but sarcoplasmic reticulums are more. ठीक है बच्चे? Right. So the muscles that move, eyeballs, they are white muscle fibers. So they will contract immediately, but yeah, they have less mitochondria. And flight muscles in birds, okay, flight muscles, right, they are the red muscle fibers. So up to this part all clear or not? Because after this we will be starting the bones wala part. Tell me, all clear? Guys, I don't think that you have the energy. What do you want? Do you want me to end the class? Yes, do you want me to end the class? Sandra, for getting 500, yes, you have to listen to these marathons, plus you need to practice some questions, you have to give the mock test. So yes, people, understood? So if you have any doubt, tell me. If you have any doubt, just let me know. If you have any doubt, let me know. Tell me. Eye muscles, yes, the muscles that move the eyeball, right, they are white muscle fibers. Yes बच्चे Sandra, you can. ठीक है, doubt clear? So can we start the skeletal system? Can we? Oh my God, Shaila is saying waste of time to watch it. नहीं यार. Just check these marathon and check other marathons. I can bet you are going to get the quality content here. But still, doesn't matter. छोड़ो. ठीक है? Okay. Now let's move to the next part, that is the skeletal system. Let's talk about the muscles. So it will take your another half an hour. Then let's move to the next chapter. है ना? Let's move to the next chapter. So guys, feeling good? Feeling good? बच्चे रेड मसल फाइबर और वाइट मसल फाइबर, रेड मसल फाइबर आर हैविंग मोर मायोग्लोबिन, मोर माइटोकॉन्ड्रिया, लेस सार्कोप्लाज्मिक रेटिकुलम. वाइट मसल फाइबर्स आर हैविंग लेस मायोग्लोबिन, लेस माइटोकॉन्ड्रिया, मोर सार्कोप्लाज्मिक रेटिकुलम. ठीक है, चलो लेट्स स्टार्ट. लेट्स टॉक अबाउट द स्केलेटल सिस्टम. सो हाउ मेनी बोन्स डू वी हैव? हाँ जी, हाउ मेनी बोन्स डू वी हैव? आवर स्केलेटल सिस्टम इज बोनी ना. So how many bones do we have? Acha, in the embryonic stages. And what about the embryonic stages? What about the embryonic stages? Yes, in the embryonic stages. They are near about yes 300 something. You can say that's near about 270 or 270 plus. ठीक है, so at the time of birth also near about 270 or 270 plus bones are there in our case, because that baby is still developing. So in adulthood basically that bones will fuse and they will grow. ठीक है, so it is near about 270 or 270 plus. ठीक है, so now when you talk about the skeletal system, we are obviously talking about an adult. So total number of bones will be 206. How many bones will be there? 206. But we divide the skeletal system into two parts. One is your axial skeleton. One is your axial skeleton. Please listen to me carefully. Another is your appendicular skeleton. Axial skeleton and another is your appendicular skeleton. ठीक है? Another is your appendicular skeleton. ठीक है बच्चे? Okay. Okay, so one is what? It is your axial skeleton, and trust me, it is also easy. So here I'll give you one option that if you want to remember it, you can. If you want to leave it, it is again your choice. I'm not going to force you here. Okay, so axial skeleton is having, can you tell me the number of bones in axial skeleton? Can you tell me the number of bones in axial skeleton? It is 80 bones, right? It is 80 bones. It is having 80 bones, and here you are having 126 bones. Now, what is the meaning of axial and appendicular skeleton? Axial, this axis, right? Your brain, the spinal cord, the sternum, ribs, vertebral column. Brain, spinal cord, sternum, ribs, vertebral column. This is making the axial skeleton. Let me write it here. Right. So here you will be having skull. Okay. You will be having vertebral column, sternum, and ribs are there. And ribs are there. Okay. Now when it is the appendicular skeleton, you will talk about the girdles and the limbs. What will be there? Girdles and the limbs. What will be there? Girdles and the limb. जैसे pectoral girdle, that is your shoulder girdle. That is your shoulder girdle. Hip girdle is pelvic girdle. Okay, you can simply write, रुको, हाँ, let's make it more easy. Girdles and limbs. Trust me, it is very easy, right? I started remembering it as a, like I remember it, but in a fun way. I'll tell you how. So limbs में total 120 bones are there, and here in the girdle 6 bones are there. When you talk about the girdles, you have pectoral girdle. Pectoral girdle. Pectoral girdle is the shoulder girdle. But if you don't remember the name of the bones, at least you should, the number of the bones, at least you should know the name. Okay, you should know the name. It is very important for your MBBS also. So pectoral girdle में 2 bones each. Pelvic girdle में overall 2 bones, ठीक है, overall 2 bones. जैसे pectoral में you have 2 coxal bones in each girdle, ठीक है, so 2 coxal bones in each girdle, right? 2 coxal, pelvic girdle is having 2 coxal bones, and when it is the pectoral girdle, you will be having clavicle, collarbone, you will be having scapula, shoulder blade, ठीक है, so 2 in number, 2 in number. Now when you talk about the skull में cranium, how many bones are there in cranium? How many bones are there in cranium? Cranium is formed by the fusion of, how many bones are there in cranium? Cranium is formed by the fusion of 8 bones. Then your facial bones, how many bones are there in your face? 14. Then ear ossicles, right? In our ear, we are having ear ossicles, no, 6 bones are there, okay? Ear ossicles 6, and one is your hyoid bone. One is your hyoid bone, that is also 1 in number. That is also 1 in number. So let me write it down, then I will explain. So skull में total 29, vertebral column, okay, vertebral column, before fusion 33, after fusion 26, after fusion I'll explain everything, 26. Sternum 1, ribs में total 12 pairs, means total 24 bones are there. 24 bones are there. This is the flow chart. Okay, this is the flow chart. Let's revise it with me. Alright, let's revise it with me. Let's talk about axial skeleton. So tell me, what do we have in our axial skeleton? What do we have in our axial skeleton? Right, you can take the help of a book. Okay, you can also take the help of a book. How many bones are there in axial skeleton? How many bones are there in axial skeleton? How many bones are there in axial skeleton? People, be quick. Total, how many bones are there in axial skeleton? 80. So what do we have in our axial skeleton? We have skull, right? We have vertebral column, right? We have sternum, and we have ribs. Now I am going to write something, please pay attention. I am saying skull is having 29 bones. How come 29? Because the, right, how come 29? Because when you talk about the cranial bones, you will be talking about the facial bones here, right? You will be talking about the cranial bones, you will be talking about the facial bones, you will be talking about the ear ossicles, okay, and you will be talking about the hyoid bone. So please pay attention, pay attention. So cranial bones 8, 14, 6 and 1, okay. Vertebral column, again I am repeating, I am writing the same line again. So it is before fusion 33, after fusion 26, okay, after fusion 26. So it is CTLSC, right? Come to lunch, stay calm, okay, come to lunch, stay calm, come to lunch, stay calm. So CTLSC, CTLSC, CTLSC. Question can come from this part. It is CTLSC. Question can come from this part. So cervical, it is thoracic, it is lumbar, it is sacral, and it is coccygis. It is coccygis. So cervical, thoracic, lumbar, sacral, coccygis. Okay. So it's 7, 12, 5, 5, 4. So if you will look at this bracket, right? So bracket is saying that these are fused. Bracket is saying these are fused. Bracket is saying these are fused. So 5 means 1, 4 means 1. Means sacral is formed by the fusion of 5 bones. Coccygis is formed by the fusion of 4 bones. So at least this question will come. So that's why I'm saying that before fusion it will be 33, but after fusion it will be 26. Okay? Right, so this can come vertebral formula. Sternum, that is the breastbone, that is also cause 1 in number. Ribs are total 12 pairs. So when I'm saying 12 pairs means total 24. Now I know you must be thinking कि in the NCERT total 22 bones are given in skull, है ना? Total 22 bones are given in skull. Total 22 bones are given in skull. Then why are you mentioning 29? Nahin? Why are you mentioning 29? देखो बच्चे, in the NCERT initially they mentioned this only. right? Then it is 22. But we have ear ossicles also. When you talk about the complete skull, skull will include these facial bones, skull will include these cranial bones, skull will include the bones in the ear, that are ear ossicle, and we have our tongue bone also, hyoid. It is the U-shaped bone, it is the tongue bone. Okay, it is the U-shaped bone, it is the tongue bone. It is the U-shaped bone, it is the tongue bone. Now, I'll mention few important points from skull part, then one by one we will discuss everything important. Okay, then let's move to the next chapter. Okay, then let's move to the next chapter. So, the point is, hyoid bone. So, as I said, hyoid bone, this is important, please pay attention. It is U-shaped bone. What type of bone it is? It is the U-shaped bone. It is the U-shaped bone. And what is the beauty of this bone? Obviously, it is supporting the tongue, right? It is supporting the tongue without any doubt. It is doing what? It is supporting tongue, right? So, this is the only bone which is not articulating with any other bone. So, in this chapter, this is the word that you are going to hear again and again. It is not articulating with any other bone. So, it is a direct PYQ. It's a direct PYQ, right? So, basically this bone is going to support our tongue. So, with the help of ligaments it is fixed. So, it is not articulating, not attached with any other bone. This is the hyoid bone. So, it is the U-shaped bone. It is the support. It is going to support the tongue. It is not articulating with any other bone. This part clear? This part clear? Now, ear ossicle. Because the topic of ear is deleted, maybe question will come from ear ossicles. So, in the middle ear, okay? In the middle ear, we have 3 ear ossicles. Where do we have these 3 ear ossicles? In the middle ear. Where do we have these 3 ear ossicles? In the middle ear. So, what is the name of these 3 ear ossicles? Now, I am not drawing their proper shape. So, it is trick. That is Miss has. Hai na? So, in our batch also, this is what we have discussed. So, it is Miss has. So, you always need to write it like this. So, it is MISHAS. Miss has. Miss has. So, M stands for malus, I stands for incus, S stands for stapes. So, malus is your hammer shaped. Okay? It is your hammer shaped. Okay? Incus is your anvil and stapes is your stirrup. Stapes is your stirrup. Clear, bache? So, Miss has malus, incus, stapes, hammer, anvil, stirrup. So, this is also a PYQ. So, each ear is having 3 ear ossicles, 3. So, one other thing that you need to remember is stapes is the smallest bone of our body. It is the smallest bone of our body. Even it is the smallest ear ossicle. Are you getting my point? It is even the smallest bone of our body. It is even the smallest ear ossicle. Got it? Got it? That's what you have to remember. Now, let's come to the NCERT. Right? Dekho. First of all, let's discuss this cranium part then. Whatever is given in NCERT, I'll suggest you to focus on that only. Dekho. Otherwise, there is a lot that we can share here. Let's come to the cranium. What is this? Frontal part, frontal, parietal, temporal, occipital, ethmoid, sphenoid. Try to remember the name. If you cannot, you can leave it. But it is my duty. I cannot leave this topic. I am teaching you. Maybe some students, they know this. So, frontal, parietal, temporal, near to the ear, temporal, then occipital, sphenoid, ethmoid. Look at this. Front one, frontal, sides mein parietal, then near to the ear, temporal. One last is occipital, and what else you have? You have sphenoid, you have ethmoid, right? You have sphenoid, you have ethmoid, isn't it? Isn't it? What do you have here? You have sphenoid, you have ethmoid. So I will mention one very simple thing here. Your sphenoid is a butterfly shaped bone, right? It's a butterfly shaped bone. And why am I mentioning it? Because this is the bone. This bone is having a depression, sella turcica. You remember this word, sella turcica? You remember this word, sella turcica? We read it in chemical control and coordination. So this bone is having a depression, sella turcica, and that is the home of pituitary gland. That is the home of pituitary gland. Okay? That is the home of pituitary gland. That's why I am telling you. Right? That's why I am telling you. ठीक है बच्चे ठीक है, so this is something that you should not miss. Now when you talk about these bones, I'm just mentioning the highlighted and the important points there. Only your, see, only the parietal and temporal, they are 2 in number. Only parietal and temporal, they are 2 in number. 2 to 4, 5, 6, 7 or 8. It's like that. 2 plus 2, 4, 5, 6, 7 and 8. It's like that. So related to sphenoid, this point should be very clear in your mind. Now this is your skull. This is your skull. It is going to protect your delicate brain, and here we have the vertebral column which will protect our spinal cord. So vertebral column and skull, they articulate with each other. So whenever we will use the word articulate, means one bone is getting fixed with another. One bone is getting fixed with another. One bone is getting fixed with another. That's the point. Now when you talk about this occipital, I'm just picking up the PYQs. Occipital bone. In this bone, through this bone is having a kind of opening. This bone is having a kind of opening that is known as foramen. You know na, foramen is for opening. Foramen magnum. Foramen magnum. Right? So through this foramen magnum, that medulla oblongata, right, that medulla oblongata will come and it is going to join with your spinal cord. That's how it is. Okay? That is how it is. It is the foramen magnum that you will see in the occipital bone. Occipital bone. So adjacent to this foramen magnum, you are having two condyles. These are the places which are going to articulate. Imagine, this is your skull. Here you are having your occipital bone. Just let's say you are having an adapter, you are having a wire. If they are compatible, you are going to fix them. If they are compatible, you are going to fix them. It's like that. Same is the case here, people. Same is the case here, people. So you are having this occipital bone. It is having foramen magnum, from where your medulla oblongata is coming with, joining with spinal cord. So adjacent to it, these two are the occipital condyles. These two are the occipital condyles, and they will articulate, means they will get fixed with, articulate with first vertebra. Articulate with first vertebra of vertebral column, right? So when you talk about the vertebral column, I told you it is CTLSC. It's 7, it's 12, it's 5 is fused, 4 is fused. ठीक है, so it's cervical. So cervical में first vertebra is atlas. This is again a PYQ, right? First vertebra, the name of the first vertebra, it is atlas. It is atlas. Second vertebra, it is axis. Second vertebra, it is axis. So atlas axis, atlas axis, atlas axis. So these two occipital condyles, they will articulate with this atlas. Okay, this movement, atlas, and then atlas is attached with the axis. So that's why we are able to move like this. So but occipitals will articulate with the atlas. That's what you have to remember, because we have two condyles. That's why we have dicondylic skull. We have dicondylic skull. Understood? That's why we have dicondylic skull. So whatever I have mentioned here, that's important for your exam. So this is about the bones. So facial bones total 12, cheekbone, zygomatic, right near to eye part. This is the neck, rhinal, nasal bones are there. So basically you need to focus here. Dekho, upper jaw, maxilla, formed by fusion of two bones. Okay? Maxilla is your upper jaw, formed by fusion of two bones. And when you talk about mandible, it is the lower jaw. Only one bone is making your lower jaw. So one point that you need to write here, everyone, please pay attention. It is the only movable bone of skull. It is the only movable bone of skull. What's that? It is the only movable bone of skull, your mandible. Okay? So two bones are nasal, two bones are maxilla, two bones are zygomatics, one is mandible. Okay? One is mandible. Okay? Clear? Clear? Right? So they are total 14 na. Lacrimal 2, nasal, okay, zygomatic 2, maxilla 2, mandible 1, so 2 to 4, 5, 6, 7, 7, 7. Nasal bones are also 2, 7, 8, 9. Right? So we are also having one vomer and one is the palatine. Right? One is the palatine. Okay? These are also the name of the bones. Okay, nasal is also, I think, nasal is 2, vomer is 1, and palatine is also 2. ठीक है, vomer is 1, palatine is 2 and 1 is inferior nasal concave. So I am not saying that you have to remember this, but yeah, if there is a doubt in your mind that ma'am, what about the other bones of face, I told you, but you have to remember maxilla formed by the fusion of 2 bones, mandible 1 bone, it is the only movable bone of skull, it is the only movable bone of skull. ठीक है, so देखो, come to this part. So skeletal system, it is having framework of bones with few cartilages. You cannot say that ki cartilages are not there, है ना? Right? So chewing food without jaw bones, walking around without limb bones, obviously not possible. So you know that bones and cartilages, they are the specialized connective tissue. We have already discussed that. So bones are having hard matrix because it is having calcium salts, and your cartilages are having chondroitin salt. So yes, MCQ can come from this part, but we have already discussed. Right? So now when you talk about this, so axial mein total 20 bones are there. And I just said that skull is having 29 bones, but in NCERT 22 is given. So sometimes students are like, why? I'll tell you. The skull is composed of 2 sets of bones. Skull is composed of 2 sets of bones. Skull is composed of 2 sets of bone, cranial and facial. So that's why they are just including 22. But if you will read the next line, ki cranial are 8 in number, right? They will form this outer covering. The facial is 14, which form the front part. Now see, a single U-shaped bone called hyoid is present at the base of buccal cavity. It is also included in skull. And each middle ear, which is having 3 tiny bones, malleus, incus and steps, which are ear ossicle, right? The skull region articulates with the superior region. So basically they all are included in skull. They are the part of skull. They are the part of skull. So in NCERT here they have mentioned 22 bones. So if they are asking which 2 sets of bones skull is having, cranial and facial, then 22. But if overall total 29 are there, why? Because we have to consider these 6 ear ossicle, 3 here, and we have to consider 1 hyoid bone as well. Take it, 1 hyoid bone as well. So skull region articulates with the superior region of vertebral column with the help of 2 occipital condyles. I told you already. Take it. Now come to the vertebral column. So as I said, before fusion 33, after fusion 26. So you know that we are having CTLSC 7, 12, 5, 5, 4. Understood? Right? So you know that there is a curve here in the vertebral column, especially in the lumbar region, and that is why we are having bipedal locomotion. जैसे before that we were having quadrupedal locomotion, ठीक है? But because of the curve in this lumbar vertebrae, we are having the straight posture and the bipedal locomotion. Clear, bache? And the bipedal locomotion. Now see, in between these vertebrae you will see intervertebral disc. This is important. So you know that it is cartilaginous, and this is the yellow, right? This is the yellow fibrous cartilage. Clear, bache? It is only in mammal and it is fibrocartilaginous disc. Okay, you can also write it like this. It is the fibrocartilaginous disc. Okay? It is the fibrocartilaginous disc. Okay, so it is only in mammal. This is important. It is also a PYQ. Okay, this is important. It is also a PYQ. Take it, bache? So towards the dorsal side we are having this. So it will extend from the base of skull. Okay, so each vertebrae, let's say they will be having central hollow portion. So that hollow portion is neural canal, and your spinal cord will move from this. Okay, right, bache? So first vertebra is atlas, will join with occipital condyle. Okay, baki cervical, thoracic, lumbar, I told you already. So number of cervical vertebrae are 7 in almost all mammals including human beings. Even in the camel it is 7. Sorry, not in the camel. In the camel also it is 7. Okay? Done. Okay, so vertebral column will protect the spinal cord, support the head, this and that. Now let's talk about the sternum. So sternum is the breastbone, bache. It is at the center, ventral, right? This is dorsal side, this is ventral. So ventral midline of thorax is having this. See, look at this, right? Look at this. Clear? This is your sternum. First of all, look at this. This is your sternum. Any doubt? Right? It looks like a tie, na? It looks like a tie, yes or no? Don't you think so? It looks like a tie. Sternum. हाँ जी बच्चे, sternum. Okay, चलो let's start. ठीक है? Done. So this is the sternum. Sternum में basically 3 parts are there. That we are not going to discuss. Let's stick to the NCERT. But it is a flat bone. That's what you can remember. It's a flat bone. So when you look at this part now, which is tie like, जैसे we have the note of the tie, no? So it's like that. So just remember, it is manubrium. Okay, it is manubrium. So the clavicle muscle of your shoulder girdle and first pair of ribs will attach here. Okay, I'll discuss it later, but this is what you need to remember. Now one question can come from the ribs part. One question can come from the ribs part. So how many ribs do we have? Before that, how many? Okay, so let's discuss about the ribs first. What we need to discuss, bache? Let's discuss about the ribs first. So they are thin, flat bones. Okay, they are thin, flat bones, right? They are what? They are thin, flat bones. So you know that dorsally means this side. Dorsally attached to vertebral column, it is important, please pay attention, and dorsally attached to vertebral column and ventrally to sternum. Okay, ventrally to sternum. So sternum is also considered as dagger shaped. This type of shape is actually known as dagger shaped. Okay, so sternum. Okay, bache. Clear? So when you talk about the ribs, they are, you know, bicephalic. I'll tell you what is the meaning of bicephalic. Bicephalic. See, bache, cephal word means head. Okay, cephal word means head. Bicephalic, cephalization, formation of head. Bicephal means 2 heads. Just let's say if this is the rib, it's a flat bone, right? It's a flat bone. So when it attaches to the vertebral column, it is having 2 sites for articulation. Just let's say it is having 2 heads to get attached. Okay, it is having 2 heads to get attached. So that is why we are saying that ribs are bicephalic. Okay, that is why we are saying they are bicephalic. So all the ribs are bicephalic. Okay, all the ribs are bicephalic. Done. So we will divide these 12 pairs of ribs into 3 parts. So true ribs, which are also known as vertebro-sternal ribs. Vertebro-sternal ribs means, just let's say here you are having the manubrium. Okay, so one to shoulder muscle. So first pair of rib is attached to this part, remaining are attached to this part. Okay, remaining are attached to this part. This is what you need to keep in your mind. So first 7 pairs, right? First 7 pairs, they are considered as, bache, they are considered as what? They are considered as true pair of ribs. Let me tell you why. Because ventrally, right? Ventrally they are attached to this sternum, and dorsally they are attached to vertebral column. Okay, ventrally they are attached to sternum, dorsally they are attached to vertebral column. That is why. Okay, that is why. So first 7 pairs, that's why they are vertebro-sternal. So they are attached to the vertebral column as well as to the yes bache, as well as to the sternum. Okay, next is false ribs. It is vertebrochondral. It is vertebrochondral. See, it's simple. Just imagine. This is the sternum. 7th pair is attached to this. So now 8th pair, okay? 8th, 9th and 10th pair. It will not directly attach to the sternum. Rather, it is attached to the 7th pair, and through the cartilages, it will be attached to the sternum. So basically, it is indirectly attached to the sternum. It is indirectly attached to the sternum. Okay? So dorsally to, for sure, dorsally to it is attached to vertebral column, right? But ventrally, it will be attached, right, bache? It doesn't articulate directly with the sternum. Okay? So which pair? 8th, 9th, 10th pair, right? It doesn't articulate. It doesn't articulate directly with the sternum. It doesn't articulate directly with the sternum, right, bache? So your costal cartilage, you remember? So they will attach, right? 8th, 9th, 10th. They will attach 7th pair with the help of 7th pair with the help of costal cartilage. So you remember na, costal cartilage, basically you are talking about hyaline cartilage, right? You are talking about hyaline cartilage here. Clear? You are talking about hyaline cartilage here. Clear, bache? Clear? So last two pairs, okay, this is important, this is very important. Last two pairs. So last two pairs means 11th or 12th. It is considered as floating ribs. Why? Because it is only attached to the right, only attached dorsally to vertebral column. Okay? So it is only attached dorsally to vertebral column. So these are the vertebral ribs. Okay? These are the vertebral ribs. So they are only attached to the vertebral column. And these ribs, they even protect the kidneys. Okay? They even protect the kidneys. Done, bache? Done. Okay? Okay? So when you talk about the rib cage, that we have to discuss in breathing and exchange. So when you people talk about the rib cage, okay, ya thoracic basket, you can call it as thoracic basket or rib cage. So it is made up of, can you tell me? It is made up of course, laterally ribs, right? Then sternum, then vertebral column. Am I right? Then vertebral, right? But when it is the thoracic basket, you have to be very specific, ribs, sternum, and it will be thoracic vertebrae. Okay, when it is thoracic basket, okay, then you have to be very specific, it will be thoracic vertebrae. And overall, when you talk about the rib cage, you know that dorsally vertebral column, specifically the thoracic vertebrae, ventrally sternum, laterally ribs, and posteriorly it will be, yes bache, posteriorly it will be, quick, it will be diaphragm, hai na? Posteriorly it will be diaphragm. Remember that? Right? Posteriorly it will be diaphragm. ठीक है? Right? Posteriorly diaphragm, the one that we discuss in the breathing and exchange. And anteriorly, bache, neck can be there. Right? Neck and your clavicle. Neck and your clavicle will be there. Understood? Understood? Take care. So this part clear? Yes. This part clear. So this is all about your axial skeleton. Now let's talk about the appendicular skeleton. Right? Let's talk about the appendicular skeleton. So then we have to move to the joints part, and then we'll go for the another chapter. So which chapter you want to start next? Hanji, which chapter you want to start next? Tell me which chapter you want to start next. Ma'am, nothing, hai na? Nothing. heart, circulation, biomolecules, neural control, biomolecules, chemical coordination, circulation, excretory, chemical coordination. So let me see. So majority will win. Yes, neural will see. Chalo. So should we start? Breathing, normal, chemical, neural. चलो ठीक है ठीक है ठीक है. Equal votes are there for all the chapters. ठीक है, equal votes are there for all the chapters. Okay, so let's start. Biomolecule, neural. अरे chill, we'll do it. Okay, we'll cover everything. Chill, people, chill. So appendicular skeleton, that's what we need to discuss. Okay, what we need to discuss? We need to discuss appendicular skeleton. So as I said, in appendicular skeleton we will be having girdles and we will be having limbs. 120 bones here. Oh, okay, sorry, 126 bones here and oh my bad, sorry, total 6 bones here and 120 bones here. Okay, so when you people talk about the girdles, so as I said, shoulder girdle, which is also known as pectoral girdle, it is the shoulder girdle, pectoral girdle. Hip girdle is your pelvic girdle. So pectoral girdle is total having 4 bones, it is having 2 bones. How come 4? Dekho, basically what is the role of these girdles? They will join these limbs with the axial skeleton. Here you are having shoulder girdle, here you are having shoulder girdle, 2 shoulder girdle. So each shoulder girdle is made up of clavicle. That clavicle is also known as collarbone. It is also known as beauty bone. Let me come to this. Then scapula. Scapula is known as shoulder blade. Scapula is known as shoulder blade. Okay, so 2. You have seen models. I don't have this clavicle, right? Because I'm not that lean. Okay, I'm not that lean. I don't have this clavicle. So you have seen the models. Okay, that they are having this bone which is highly prominent. So this is the beauty bone. This is the beauty bone. And secondly, just in our village, if someone is having the beauty bone, then people say, "Arey yaar, that person is having malnutrition. That person should eat properly." But yeah, people, spend a lot of energy, a lot of money to get this, right? But in our village, the story is different. So this is clavicle. So it will attach to the sternum, manubrium part. This side, you will be having scapula, that is shoulder blade. It will not articulate dorsally with any other bone, ligaments are there, but yeah, it is attached to the clavicle, right? So these girdles will help these limbs to get attached to the axial skeleton. Now when it comes to the pelvic girdle, it is having 2 coxal bones. We have only one pelvic girdle, it is having 2 coxal bones. Actually, this pelvic girdle is something like this, if you know. So each coxal bone here, okay, each coxal bone is made up by the fusion of three bones. So three bones will fuse and you will get this. So that three bones are ilium, ILI. So what it should be? Is it ILEUM or ILIUM? Yes, this is the biggest question. Jैसे we have duodenum, jejunum or ileum. So which ileum it is? Is it ILIUM or ILEUM? It is IL. Nikita, very good. It is ILIUM. Ileum, then comes the ischium, then comes the pubis. ठीक है, ileum, ischium, pubis. These three bones, they will fuse, they will form coxal bone. So coxal bone is also known as innominates. They are also known as innominates, right? Innominates. Clear, bache? Clear. So next is limbs. Okay, next is limbs. So total 120 bones are there. So you know that we have forelimbs and hind limbs. जैसे these are our forelimbs, legs, hind limbs. ठीक है, legs, they are what? They are hind limbs. Okay, so forelimbs are 60, hind limbs are also 60. So when it comes to forelimbs, you know that you have humerus, the largest bone of this forelimb, radius ulna या radio ulna. Okay, you have carpals there, metacarpals and phalanges. And when you talk about the hind limb, we have femur, tibia fibula, patella, that is kneecap, tarsals, metatarsals and phalanges. Right? So 1 into 2. Why? Because we have two forelimbs, radius and ulna. Radius 1, ulna 1. So 2. That's why 2 into 2. Carpals here. They are 8 into 2. Okay, metacarpals, they are 5 into 2. Phalanges, they are 14 into 2. Right? 14 into 2. So do you know about it? Yes, people, do you know about it? Tell me, do you know about it? Right? So see, very simple. Humerus, radius, ulna. Radius and ulna. One humerus, total 2. Radius, ulna. One radius, total 2. One ulna, total 2. 4. Carpals, mane wrist bones. Metacarpals, palm bones. So you have 5 fingers, so you can relate it like this. Metacarpals are 5 in number. Then these are the phalanges. So it is having a formula. 3 bones here, 3 here, 3 here, 3 here, 2 here. So when you talk about the phalanges, phalangeal formula is 2, 3, 3, 3 and 3. So 3 plus 3, 9. Then, right? Then, okay. Yes, tell me, phalangeal formula 2, 3, 3, 3, 3, 3. Hanji, so what should be there? 2, 3, 3, 3 plus 3. अच्छा 6 ना, मैंने multiply किया. 3 plus 3, 6, 7, 8, 9, 10, 11, 12, 13, 14. So 14 into 2 it will be 28. ठीक है? So 2, 4, 16, 10, 28. So this is the phalangeal formula. So this side 2, 3, 3, 3, 3. Okay, so radio ulna, you ulna, ungli means it is towards this. So radius is towards your thumb, ulna is towards your finger. That's what you have to remember. Okay, that is what you have to remember. Now femur is 1, tibia 1, fibula 2, patella, that is kneecap. Okay, kneecap and knee joint is different. Question can come from this part. And this patella is a sesamoid bone. So you will tell me what is the meaning of sesamoid bone. Right? This is what you have to tell me, that what is the meaning of sesamoid bone. Right? What is the meaning of sesamoid bone? Right? So this is 1. Tarsals are 7 here, metatarsals are 5 here, phalanges are again 14 here. ठीक है. So बच्चे see, if you will compare, you have to see that in the knee we are having a kneecap that is known as patella. We are having a kneecap that is known as patella. So it is sesamoid. So it is 1 into 2. So tarsal. So we have to reduce one bone of tarsal. So tarsals are total 7. So means ankle bones are 7. Ankle bones are 7. ठीक है. And wrist bones are 8. So that's what you have to remember. ठीक है. So that's how it is total. It is total. Yes. What is it? Total 60. It is total 60. Clear बच्चे? That is why it is total 60. So you can see this, right? So this is your clavicle shoulder bone articulated with sternum. So your clavicle and it will attach to scapula. You know how are they going to attach? How will they get fixed? Yes. Do you know how will they get fixed? Quick bache, then after that we have to start the joints. Anyone? Clavicle and scapula. Clavicle and scapula. Yes, देखो, they are having a process in between. जैसे ये देखो, this is a clavicle, then shoulder blade. Okay, shoulder blade. So see, scapula is a large triangular flat bone situated in the dorsal part of the thorax, and it is between 2nd and 7th rib. So this can be asked as a question. So it is triangular body of scapula, has a elevated ridge called the acromion process. This is important, please pay attention. When you talk about that scapula, it is shoulder blade, it is something triangular. Okay, so it is having some elevated ridge, and that ridge, ridge called spine, which projects out as a flat expanded process called acromion. So clavicle and scapula, you can simply remember that they will articulate with the help of acromion. So this acromion process is there in scapula, where your clavicle will get fixed. So this can be asked, where is acromion present, in clavicle or in clavicle or in scapula? So you have to remember it is in scapula. Now to this scapula, if you can see, humerus head is also articulating. To this scapula, if you can see, humerus head is also articulating. So it is the glenoid cavity, right? It is the glenoid cavity. It is also in scapula. It is a previous year question. Okay, this glenoid cavity is also in scapula. Okay, so it is scapula. So it is also in scapula, just below the acromion process. So yes, this is something that you need to remember. Okay, right? So glenoid cavity question can come. So this is the shoulder joint. Okay, so clavicle is a long slender bone with 2 curvature. So this is the collarbone. Understood? Now this is the pelvic girdle. So ilium, ischium and pubis, they will fuse. So here this is acetabulum. So this is the acetabulum where your femur will articulate. Simple. This is the acetabulum where your femur will articulate. Again I am repeating this, bache. Patella is a sesamoid bone. You will tell me what is the meaning of sesamoid bone. Right, that's your homework. What is the meaning of sesamoid bone? Okay, so ilium, ischium, pubis is there. So at the point of fusion of above bones, there is a cavity, acetabulum, where your thigh bone, that is your femur, it articulates. Okay, and you know that this or this, and even in pelvic girdle, if you will notice, see this, a part of vertebral column is there in the pelvic girdle. So basically pelvic girdle, it articulates with the vertebral column. Okay, it articulates with the vertebral column here. Okay, and these two coxal bones or innominates, right? These two coxal bones or innominates. Just a minute, bache. Done? So let's say, so this is the vertebral column, and here these coxal bones, they will articulate, and here you are again having that pubic symphysis, which is flexible. You remember pubic symphysis? Just in the case of female, pelvic girdle is broader because they have to carry a baby, and this pubic symphysis, it is basically what? It is also flexible. At the time of childbirth, it will open, if you remember. Yes, bache, pubic symphysis is there. So pubic symphysis is basically the fibrous cartilage. Okay, it is basically the fibrous cartilage. What's that? It is basically the fibrous cartilage. Understood? Understood? So that's what you have to remember. Okay, 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 okay. Okay, so now let's talk about the joints, but before that, if there is any doubt, do let me know, and please revise it. Please revise it. It is something that, you know, I'm telling you honestly, when for the first time when I saw this chapter, when I was in school, I was like, yeah, this chapter is going to be a very difficult chapter, right? It will be the worst chapter. It will be the worst chapter. But actually, it is not like that. Okay, actually, it is not like that. Why? Why? Yes, bache? Right? Why is it so? Because this chapter, like, if you will read it in a fun manner, na, you are going to enjoy this chapter. You will be like, yeah, there is nothing tough in this chapter. Okay, right? There is nothing tough in this chapter. Okay, it is a very easygoing chapter. Okay, you are sitting, you're like, acha, this humerus, radius ulna, carpal, metacarpal, phalanges, femur, tibiofibula, patella, tarsals, metatarsals, phalanges, like this. And let me tell you, there is something called knee joint. Okay, there is something called knee joint. So when you talk about the knee joint, it is having your femur, it is having your tibia, and it is having your fibula. So no one is going to ask you which one is stronger, which one is thinner, so that's why I'm not touching it, but if you can read it, that will be good for you. So knee joint is formed with the femur, tibia and fibula. No, not the, my bad, femur, tibia and patella. These three will form your knee joint. These three will form your knee joint. It is again a PYQ. Okay, it is again a PYQ. Got it? Yup. Sure. So can we talk about joints? Can we talk about joints? We'll be very specific. We will be discussing the NCRT only. Can we talk about joints? So the study of joint is arthrology. Okay, study of joints is arthrology. What's that? It's arthrology. Yes, everyone, please mention it. It is arthrology. Study of joints, what is it? It is arthrology. Hai na? It is arthrology. Clear? Clear, clear, clear. So how many types of joints are there, if you can answer? There are 3 types, but if you can name them, I'll be very grateful. Ki Hanji, my students, they know something. I'll be proud of you. Yes, I'll be proud of you. Ki Hanji, you know something. I'll be very, 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 very happy. I'll be very, very, very happy. Quick, we are talking about the joints. There are 3 types. This is what I'm saying. Very good, very good, very good. So joints are very important for the movement, right, bache, involving the bony part. So locomotion movements are no exception to this. So joints are point of contact between bones or between bones and cartilage. So it is not a point of contact between bones only, but yeah, between bones and cartilages also. So force generated by the muscle is used to carry out the movement through joints, and joints, they will act like a fulcrum. I hope you know that fulcrum wala thing, something like this. So joints, they will act like a fulcrum. This is what you need to know out. So mobility of these joints, it varies. So here you can see the fibrous joint. So they are immovable joint. So examples can come in paper. Immovable joint, they are fixed joints. They will not move at all, and you will call them as synarthrosis. You will also use the word synarthrosis for it. Synarthrosis for it. They are not at all movable. They are totally fixed. Jaise joints of your skull bone, sutures, you can see this, right? Jaise flat skull bones, they fuse end to end. They are having dense fibrous connective tissue which will fuse them, right, to form sutures. So this is the example. So immovable, no movement at all. Understood? No movement at all. Understood? Now the cartilaginous joints, they are slightly movable. right? They are slightly movable. You will not say they are freely movable, but rather they are slightly movable. So it is also known as synchondrosis because it is cartilaginous. Synchondrosis because it is cartilaginous. So very simple example, right? This is joint between the adjacent vertebrae. One vertebra, second adjacent vertebrae, the joint between them. Okay, it permits a limited movement. So this is it. So bones are joined together with the help of cartilages, right? So cartilages are playing the role here. Then bache, synovial, you know that they are freely movable. I'm not making the flow chart here, you please note it down from NCERT. It is freely movable, right bache? So it is known as diarthrosis. It is known as diarthrosis. Okay, so why is it so? Just let's say you are taking two bones, 1 and 2. So in between there will be a synovial membrane, there will be a synovial cavity, and there will be a synovial fluid. So this synovial fluid will provide the movement here, bache, right? You know that when there is the inflammation in the synovial fluid, it can result in rheumatoid arthritis, which is autoimmune disorder. It is also a PYQ. Okay, it is also a BYQ. So here in between these joints we are having a fluid, right? Fluid filled synovial cavities there, right? That is how the two surfaces of bones they are articulating. So considerable movement is there. So locomotion will be there. So dekho examples, ball and socket. This is humerus and pectoral gland. Ball and socket type of joint. Then see, this is hinge. You have the laptop, no? Laptop is having the hinge, na? Hinge, this part, your legs. This is the hinge joint. So your hinge joint, knee joint. Pivot, it is in between atlas and axis. Okay, gliding movement between the carpals, and saddle joint, it is in between the carpals and metacarpals of thumb. These are the example. Directly remember the example. I'm telling you nothing will come beyond NCERT, so I really don't think that you should miss this part. Okay, right? So if they have given the example of humerus and pectoral, you should understand the joint in between femur and acetabulum, because same type of movement is there. So joint in between femur and acetabulum, that is also ball and socket. So if the knee joint is hinge joint, so this elbow joint is also hinge joint. It is also the hinge joint, right? So next is pivot joint. It is also atlas and axis plus radius and ulna. Your radius and ulna is also having same type of joint. Gliding ke liye this part is sufficient ki carpals, right? Your carpals are having this. So obviously if carpals are having this, you can say that this joint is also in between tarsals. Okay, it is also in between tarsals. So saddle joint ke liye this example is sufficient. Baki, that's what you have to remember. That's it, that's all. Right? So next is disorder. Okay. Arthritis is inflammation of joint, but you know that arthritis is of different types, isn't it? Arthritis is of different types. How many of you remember this? Arthritis, it is of different types. Yes, it is of different types. You remember that? Do you remember that? Just say you have rheumatoid arthritis, which is autoimmune. That's a PYQ. In the chapter Human Health and Disease, it is given. You have gouty arthritis also, which is due to the increase in uric acid. So rheumatoid arthritis, which is autoimmune. Myasthenia gravis, autoimmune. You should know about this. Right, people? So gouty arthritis, it is when excess of uric acid is there. Okay, rheumatoid arthritis when there is excess of RA factor. So rheumatoid arthritis mein bache, basically there is the inflammation of that synovial membrane. You can write down. Okay, so rheumatoid factor is there, right, which is a kind of IgM, which is a kind of immunoglobulin M. Okay, so inflammation of synovial membrane is there. So if it is left untreated, synovial fluid increasing, pressure will increase, pain will be there. Fine, pain will be there. So joints with the time, it can even become immovable because pressure is there. Further, it will become immovable. So one word that you have to remember, that there is the inflammation of synovial membrane. Okay, so again I am repeating this, people. It is an autoimmune disorder. Okay, it is an autoimmune disorder. So gouty arthritis mein uric acid will shoot. Fine. So when it is osteoporosis, porosis means pores in the bones. So age related disorder hai, bone mass will decrease, and you know that in the females it is very common because after menopause the estrogen level will decrease. So bones, they will become porous, so they will become brittle, osteoporosis. Right. So gout, inflammation of joints due to accumulation. So gouty arthritis and gout, it's same. So simple arthritis and rheumatoid arthritis, it is different. You have to mark this. So myasthenia gravis is also autoimmune. Right, it affects the neuromuscular junction. So you can write down, bache, this is important. It is also autoimmune disorder. Fine. So muscular dystrophy, you know that dystrophin, it is related to that dystrophin protein. So it is a degeneration of skeletal muscles. It is due to genetic disorder. Okay, bache? Okay, so tetany and tetanus, these are two different things, right? Tetany means when there is a wild contraction because calcium level is low. Okay, we will talk about it in hormonal control chapter. Tetanus is because of that bacterial toxin. So these are two different things that you have to remember. This is your another homework to check the difference in tetanus and tetany. To check the difference in tetanus and tetanus. Now tell me, which chapter do you want to start next? Which chapter do you want me to start next? Yes. I'll go as per majority, guys, be quick. So it is not like it was a kind of one shot where, you know, we missed many things. No. As of now, we have completed 3 chapters, right? We need to increase our speed, of course, right? So 3 chapters, may you can say 2 hours we have given to each chapter, and that is something that we need to devote. That is something we need to devote. So I will go as per the majority. So chemical ke liye 3 votes are there, chemical 4 votes, biomolecule circulation, then breathing, chemical, neural, chemical, breathing, biomolecules, yaar. Acha. But mainly it is chemical. Let's pick up the chemical. Let's pick up the chemical. Okay? Let's pick up the chemical. So I think we should take the break later, hai na? Let's finish it and then we should take the break. Dekho, chemical is purely theoretical, so I'll tell you what you need to focus and what you need to avoid. Okay? Chemical is purely theoretical. Okay? So I'll tell you what you need to focus and what you need to avoid. So it is something which I cannot, it is not like that ki hanji ma'am, tell us something, explain it to us. No, it's not something that I can explain you. Okay, it is something that that need to revise again and again. I can explain you mechanism of hormone action because one question will come from that part. I'll explain you why the hormone action is different. Then I'll tell you these are the hormones and gland that you should not leave, that you should not miss. But it is not something that, you know, where I can tell you, arey bachche, you know, that is the mechanism. Mechanism is of hormone action. But this is something theoretical that yes, you have to revise. So I think we should go for the conceptual chapters first. जैसे ionic theory में people are having problem. Okay, urine formation, counter current mechanism, difficult topics for students. Oxygen hemoglobin dissociation curve, mechanism of breathing, capacities, difficult for students, right? So blood clotting mechanism, blood groups. So let's pick it up like this. So don't worry, let's pick up chemical control and coordination. I'll explain you mechanism of hormone action because that is very important. Okay, that is very important. Then I'll give you 10 minutes ka break. It's 5:15. Chalo, I'll give you 15 minutes ka break. You should be happy. Okay, you should be happy. So let's pick up topics like this. Okay, so जैसे neural control में I know students they find it difficult to understand ionic theory. I'll pick up ionic theory. I'll pick up that type of important topics. ठीक है? So that is a better approach, right? That is a better approach. Dekho, I don't have this thing na. I cannot take your session for 20 hours, right? If I will get 20 hours for your sessions, I'm telling you this thing honestly, I will not skip anything. I will teach you everything. I'll teach you everything. But the problem is time constraint. I cannot go above 12 hours. That's the problem. Okay, so 15 minutes, then let's start again. Hi everyone, what's up? I made myself read and Yuga, even I made tea for myself. Chalo, invite others also. Yes, Monish, let's take it. So no, देखो, already breathing and exchange of gases, right? And your animal kingdom is already there on this channel, even the biomolecules in detail. Okay, so the chapters that I left, right, that we were not able to cover in the Conquer batch, let's discuss that first. Okay, rest everything is on this channel, and moreover this is not the only session that we are going to have. We will be having diagram based sessions where there will be the discussions. We will be having the PYQ session. One more session is there, I just forgot that title of that session, right? We have to practice some more things also, so we will cover everything. So chill bache, sorry, I don't remember, yaar. I don't remember I was planning one more session, I just forgot. Disorders, we will keep a separate session for disorders, because from disorders questions will come. That will be for 1 or 2 hours. Okay, so can we start? Can we start? So the topic is neural control and coordination. What's the topic? It's neural control and coordination. Okay, what's the topic? What's the next topic? It's chemical control and chemical coordination and integration. So tell me, what do you know about this chapter? Yeah, first of all, you all tell me that what do you know about this particular chapter? Quick, whatever you know about this particular chapter, just let me know about that. Yes, just let me know about that. Chemical coordination, yeah, chemical coordination and integration, whatever you know about this chapter, right? Even if you don't know something, still admit that in front of me. Just let me know. Let me know, bache, because I will take a guess na ki haan, theek hai, my students, they are not aware of anything. Then accordingly I have to teach. So I'll decide accordingly. So you tell me, please. Yes, you tell me, please. Acha, you don't know much about this chapter. Arey yaar, we cannot leave this chapter like this. Okay, we cannot just leave this chapter like this. Okay, so the chapter name is chemical coordination and integration. In our body, you know that we have different systems which are going to do different jobs. And today, you know what, I'm not feeling tired. Okay, I'm literally not feeling tired. I don't know about you, but I am enjoying your lecture. Sita, you're not going to suffer from this moment. From this moment, you're not going to suffer. I'm not feeling tired today at all. I don't know. Why is it so? I should feel tired, yaar. Right? It's been 7 hours. It's been 7 hours, but still I'm feeling ki theek hai yaar. Matlab today I'm enjoying teaching you. I don't know why. Last time also it was good, it was fun, but there was a point when I was like, theek hai yaar, let's finish it quickly. But now I'm like, okay, the session should be for 14 hours, 15 hours, 20 hours, so that we can cover everything. Because today I'm not teaching you, today it is not my motivation that yes, I have to finish a task because this is what we have committed. No, today's lecture is for 120 or 130 marks. Okay, today's lecture is actually for 120 and 130 marks, and maybe more than that. Okay, so that is why you know I want to deliver the content, because if you will revise this part only again and again, you will solve majority of questions. In the human physiology, the distribution is not uniform. You cannot say that, bache, ki ma'am, this chapter, what is the duration of this chapter? No, you cannot specify it like this, right? You cannot specify it like this. Okay, chalo, let's start. So the chapter name is chemical coordination and integration. Dekho bache, it's a very simple story. Okay, it is a very simple story. In our body, we have the systems that are going to manage everything in a coordinated way. We have the digestive system for digestion, we have the excretory system for excretion, reproductive system for reproduction, and we have the neural system which is going to coordinate everything, which will generate the nerve impulse, which will conduct the nerve impulse. This is what we know. So when it comes to regulate the body, we have the neural control and the chemical coordination as well. Okay, so here you guys can see that the neural system provides point to point rapid coordination, right? Point to point rapid, the fast coordination will be provided by your neural system, right? It will be provided by your neural system, right? Why? Because that nerve, see, and neural coordination is fast but it is short-lived. So these type of details are important. Okay, so neural system is going to provide a point-to-point content. You know that we have the nerve fibers in our body, all the neurons, they are so connected, right, bache? But this transmission is, this coordination is fast, but it is short-lived. It is for some time. Why? Because the nerve fibers, they do not innervate all the cells, and the cellular functions need to be continuously regulated. So one question that can directly come, that is neural system, right? Is neural system is going to innervate each and every cell? Of course not. But when it comes to, right, bache? No, it is not. But when it comes to the coordination, right, even the cells, they need to be regulated. Okay, so special kind of coordination and integration has to be provided. So hormones are going to take care of that. So even your neural system, even your endocrine system, they will together regulate everything. They will regulate the physiology of our body together. Okay, they will regulate the physiology of our body together. So endocrine glands and hormones, let's talk about it. Okay, so you already know which glands are known as endocrine gland. They are the ductless gland. Okay, which glands are known as endocrine gland? They are the ductless glands. What are they? They are the ductless glands, means they are without ducts. Let's say this is a gland. It will pour its secretion into the blood. It is going to pour its secretion into the blood, and from the blood it will go to the target organ, and accordingly the effect will be there. So here one thing we need to understand, the mechanism of hormone action. So whenever I teach this chapter, I always start with the mechanism of hormone action. Okay, so we will come to this, but before that, understand that. So this part clear, that endocrine glands, they are the ductless glands, and they will pour their secretion directly into the blood, and from that it will affect the target organ. So you should have a question mark here, that ma'am, how is it possible? How that hormones will get to know that, yeah, we have to go to the target organ? So this is something that we understand in the mechanism of hormone action. This point clear? This point clear? So the hormones, they are the chemicals which are produced by endocrine glands. They'll be released into blood, transmitted to a distantly located target organ. This is the definition. But if you talk about the scientific definition, they are non-nutrient chemicals. Please focus on the details. Detailing is very important, bache. Non-nutrient chemicals, they are not nutrient. This is not something essential that should be in our diet. They are the non-nutrient chemicals, bache, and they act as the intercellular messenger. Inter means in between two cells. They are acting like a messenger, and they are produced in very trace amounts. So secretin is the first hormone discovered. You know that? Secretin, it is released by small intestine. It is the first hormone discovered. Do you know about it? It is the first hormone discovered, secretin. It is released by your small intestine. What is it? It is the secretin, which is the first hormone discovered, and it is released by the intestine. Is that clear? Is that clear? Right. So term hormone was given by the starling. Who gave the term hormone? The term hormone was given by, yes bache, the term hormone was given by starling. Okay, it was given by starling. This part clear? This part clear? So even in the invertebrates means which are not having the vertebral column, they are also having simple endocrine system. They have few hormones, right, but obviously they are more in the vertebrates. So when you talk about the endocrine gland, that's what we have to discuss. You know how can you remember this? Start from the brain. Brain is having hypothalamus, supramaster gland, pituitary, pineal gland. It is in the brain. Then come to this region. You will be having thyroid, parathyroid. Go a little down, thymus will be there. Right. Then you have cap of kidneys, adrenal gland, your pancreas, which is the heterocrine gland. right? In the structural organization, I told you what is the meaning of heterocrine gland or composite gland. So this is the gland which is having the endocrine and exocrine part as well, then adrenal gland, and when it comes to gonads, this is what we have. So other than these endocrine gland, we will also discuss the hormones of GI tract here. We will also discuss the heart, the kidney here. Okay, we will also discuss the heart and kidney here. Clear bache? Clear bache? Yes. Clear? Sure? Sure? Are you sure? Okay. So we have the hypothalamus, pituitary, pineal, thyroid, parathyroid, thymus, pancreas, adrenal gland, then we have the gonads, in the males testes, in the females ovary, right bache? And other than that, we will also discuss the hormones of GI tract. We will also talk about the hormone released by the kidney, hormone released by the heart, right? That's what we have. This first part clear of hormone action, but before that we need to know about the nature of the hormones. Okay? What we should know? We need to know about the nature of the hormones. So if you understand the nature of the hormones, then this mechanism of hormone action will be comparatively easy for you, right? It will be comparatively easy for you. Dekho, obviously we are saying, right, students, we are saying that a gland, an endocrine gland, okay? What is it? It is an endocrine gland. This endocrine gland is going to secrete something. It is going to secrete something. That non-nutrient chemicals which are formed in very minute amount, which are formed in trace amounts, we call it as hormones. What do we call it? We call it as hormones. What do we call it? We call it as hormones. First part clear? So now they will be released in bloodstream. Then how will they get to know that, yeah, this is my target organ? Let's say the target organ of this hormone is thyroid. Then how this hormone will get to know that, okay, fine, I need to act on the thyroid gland. How? Of course, it's very simple. Here comes the role of the receptors. You know that when you are talking about these glands, ultimately they are also having the cell membrane, right? They are also having the cell membrane, right? Right. They are also having the cell membrane, right? So when you talk about these cell membranes, right, so they are going to have receptors. Actually, we are going to talk about two types of receptors. What are receptors? They are, of course, the proteins. They are the proteins to which, right, they are the proteins to which a particular, right, or you can say that a specific chemical will join, isn't it? They are the kind of proteins to which a specific chemical will join. What will join? A specific chemical will join to these proteins, to these receptors. Is that clear? So these receptors are very specific. A specific protein, specific hormone will join here. This is the first point. Now I told you receptors, they are of two types. One is cell surface receptor. So what is the meaning of cell surface receptor? Means these receptors are present on the surface of cell, that is plasma membrane, right? And another are intracellular receptor. The word is intracellular, means within the cell, students, and here we are going to talk about the nuclear receptors, the receptors which are present within the nucleus. Okay, so these are the receptors which are present within the nucleus. Understood? These are the receptors which are present within the nucleus. Understood? So cell surface receptor is there, intracellular receptor is there, so they are also known as the nuclear receptor. Now, when you talk about these hormones, right, so they need to bind to their specific receptor. Just say, let's say if the hormone should act on the thyroid gland, then its receptor is on the thyroid gland only. So it will not join here and there and it will cause the problem. Not at all. Now, how can we say that on what basis we decide that some hormones, their receptors are cell surface receptor and some of them are having intracellular receptor? This is another question that we need to answer. Simple it is. Now hormones are having different nature. Just they are made up of proteins, peptides. They are also made up of amino acids. They are also steroidal in nature. But these things are going to decide about their receptors. I'll give you one example. Steroidal hormones, the hormones which are made up of steroids. What are steroids? They are the derived lipids. What are steroids? They are the derived lipids. What is the meaning of derived lipids? The meaning of derived lipid is ki basically they are fats, they are the lipids, in simple words. Now your sex hormones like progesterone, okay, progesterone, estrogen, testosterone, right? They are what? They are the sex hormones. Right now I'm picking up one example: progesterone, estrogen, your testosterone. They are the sex hormones. But for sex hormones we also use the term androgens, right? We also use the term androgens. So these sex hormones, because they are made up of steroids, even the hormones of your adrenal medulla, some hormones are also steroids, right? They are also steroid. So these hormones which are steroidal in nature, which are made up of steroids, don't you think that they can cross the cell membrane? Yes, you can answer it by your own people. Don't you think that they can cross the cell membrane, because it is also a lipid? It is also a lipid. Don't you think that it can cross the cell membrane? Yes ma'am, it can cross the cell membrane. So within a cell you are having a nucleus, and here you are having the DNA. Here you are having a DNA. Here you are having a DNA. So obviously what is going to happen? These hormones, they can control the cell membrane. They will enter within the nucleus, and within the nucleus obviously it can, right, it can play with that genome part, that's all. So that is why we will say that when you are talking about sex hormones which are steroidal in nature, they can cross the cell membrane and their receptors will be intracellular. Now when you talk about certain protein hormones, they cannot cross the cell membrane, students. They cannot cross the cell membrane. They will just bind to the receptor on the cell surface, and that is how within the cell they will generate the response. Are you getting it? So it is the nature of that particular hormone that will decide that whether it will be having cell surface receptors or the intracellular receptor. This point clear? This point clear? Chalo, theek hai. Now come to this part. Okay, so understand this thing. Let's say you are not allowed, let's say there is a government hospital, okay? Sorry, not a government hospital, let's say there is some very, some offices there, right? Just CBI ka office, CID, like this. You got an information. You are not allowed to enter there, right? Because of the protocol, you are not allowed to enter there. But you will share your information, right? Let's say you will tell it to the security guard, you know what, I have the exclusive information. So security guard will be like, you just wait, I'll talk to my seniors, like this. A chain will start, because you are not allowed inside. You are not allowed inside. So that person will pass your information, right, to the seniors, and accordingly actions will be there. Same is the case here. So when you people talk about the hormone, what's going to happen? This hormone is going to bind with this receptor, right? This hormone is going to bind with this receptor. So can I call this hormone as first messenger? Can I call this hormone as first messenger? Because it is not allowed within the cell. So when it will join with receptor, inside it the cascade, right? A cascade of information will start. A cascade means where steps are linked. One step is linked with another. One step is linked with another. So when in the case of the hormones where their receptors are present on cell surface, so we can also call that hormone as first messenger. So once it will join with this receptor within the cell, the cascade of information will start, and in response to that it will produce a second messenger. So in simple words, when you talk about the hormones which are having cell surface receptors, okay, which are having cell surface receptor, so in such hormones you will talk about the second messengers. So what are second messengers? Second messengers are the chemical compounds that will form when the hormone join with the receptor. So in response to that joining, in response to that attachment, whatever will form within the cell, that will firstly be the second messenger. This point clear? Any doubt? That will be the second messenger here. Any doubt? Hanji, here do you have any doubt? That will be the second messenger here. Sure? Sure. So this is something very important that you need to know. Okay. right? So can I say that the hormones which are having water, which are having cell surface receptor, they are water soluble, they are not lipid soluble, that's why they are not able to cross it. That's why they are not able to cross it. So they will be having the second messenger. So in the paper they can ask you the examples of second messenger, and that includes your calcium ions, that includes your IP3. IP3 means inositol triphosphate. Okay, so CAMP will also be there. CAMP will also be there, like your cyclic adenosine monophosphate. That is cyclic adenosine monophosphate, right? That will be cyclic adenosine monophosphate. Other than that, there is CGMP also. So cyclic guanosine monophosphate. So question can come from the examples of this second messenger. So second messengers will form in response to the hormones which are water soluble or which are having receptors on the cell surface. Other than that, DAG is there, DAG, diacylglycerol. Okay, so these are the examples of the second messenger. So calcium ions, IP3, inositol triphosphate, cyclic adenosine monophosphate, guanosine monophosphate, diacylglycerol. This is what we have. Understood? This is what we have. Are you able to understand? So they will form in response to the hormones which cannot control enter the cell. They are having their receptors on the cell surface. So accordingly, a cascade of information will start. Okay? So now come to the diagram, then I will explain. So see, this is the ovarian. Okay, this is the ovarian cell membrane, matlab cell membrane of ovary. This is the hormone. This hormone name is FSH. FSH means follicle stimulating hormone, and it's a protein hormone. Okay, it is a protein hormone, right? Just because it is acting on gonads doesn't think that, oh my god, it is also steroidal in nature. No, it's a protein hormone. So it cannot cross the cell, so it will bind to this receptor and it will generate the response, and due to that response there is the generation of second messenger. So they have given two examples, I have given all the examples to you. Then after that, biochemical responses will be there, physiological response, that is ovarian growth, will be there. So in the paper directly diagram can come, they can just put the blanks here, they can just write down A, B, C, they can ask you what is A, what is B, what is C. So you have to remember this diagram as such. You have to remember this diagram as such. Right, now move to the next image. Now this is the estrogen. Estrogen means steroidal hormone. Steroidal hormone means it can cross the cell membrane. So because it can cross the cell membrane, so its receptors are within nucleus. So it is having nuclear receptor. So in that case there will be no second messenger. It doesn't need any second messenger because it will directly enter within the cell. So see, it will join here. Hormone and receptor complex will form. This is the genome, mRNA and protein. So physiological response, tissue growth and differentiation will be there. So that's how it works. So tell me if you have any doubt related to this. If you guys are having any doubt related to this, you are free to ask. So let's revise it from NCERT. So the hormones produce their effects on target tissues by binding to specific proteins called hormone receptors. Okay? So they are present on the cell membrane of target cell. So then they will be membrane bound receptor, या cell surface receptor. So if they are within the target cell, they are intracellular receptor, nuclear receptors they are. Okay? So each receptor is specific to one particular hormone only, right. So then after that there will be the biochemical changes in the target tissue. So see this, that's what you have to remember. So your peptide, polypeptide and protein hormone. Insulin is a protein hormone. Remember, A chain. How many amino acids are there in A chain? Anyone in the class? How many amino acids are there in A chain? 21 amino acids. How many amino acids are there in B chain? 30 amino acids. Remember, glucagon, protein hormone, pituitary hormone, all are protein. Hypothalamic hormones, protein hormone. Steroidal में cortisol, released by adrenal cortex, testosterone, estradiol, progesterone. Idothyrionins, the hormones of thyroid. Right, then amino acid derivatives, the hormones that are made up of amino acid, they are epinephrine and norepinephrine. So this is the nature of the hormone. So as I said, they are of different types. So the examples which are given in NCERT, this is what you need to revise. Okay? This is what you need to revise. Clear, bache? Clear? Sure? This is what you all need to revise. Okay? So your steroidal hormones, even your triido, T3, T4, their receptors are also steroidal hormones, and even स्टीरॉइडल हॉर्मोन्स जैसे कि हॉर्मोन्स ऑफ योर एड्रीनल कॉर्टेक्स एंड आइडोथायरोनिन्स। आइडोथायरोनिन्स में बच्चे द हॉर्मोन्स ऑफ थायराइड ग्लैंड लाइक T3 T4 देयर रिसेप्टर्स आर आल्सो इंट्रासेलुलर, ठीक है, देयर रिसेप्टर्स आर आल्सो इंट्रासेलुलर, क्लियर बच्चे, क्लियर क्लियर क्लियर, देयर रिसेप्टर्स आर आल्सो इंट्रासेलुलर, क्लियर बच्चे, श्योर श्योर। सो मैकेनिज्म ऑफ हार्मोन एक्शन क्लियर, examples clear. So see, the examples of second messenger should also be clear, so that is what you need to remember. So see, hormones which interact with intracellular receptor, they are steroidal hormones and idothyoronins, okay, okay. So they regulate gene expression or chromosome function by interaction of hormone receptor complex within the genome. Done, done. Doubt. Some people say na that, oh my god, this is a very difficult topic, I really want to know what's the difficulty. I really want to know what's the difficulty. देखो, for this particular chapter and for locomotion and movement, if you are interested in knowing about your body in this way, then only you can remember this. Otherwise it is a difficult chapter, ठीक है, otherwise it is a difficult chapter. If you you know love to know about your bodies कि हाँ जी ठीक है, we have this hormone, that hormone, like this, then it is fine. Otherwise it's a problem, it's a problem. So up to this part all clear, up to this part all clear. If it is a yes, do let me know in the chat section, people. If it is a yes, do let me know in the chat section. Yes, quick. Quick. Done. So endocrine glands and hormone producing diffused tissue. Endocrine glands and hormone producing diffused tissue or cells, they are located in different parts of our body, so they are forming the endocrine system. Not just the endocrine gland, but there are some hormones producing diffused tissues, right? There are some hormones producing diffused tissues, okay? So you know the sequence here, bache, right. So now let's start with the hypothalamus, but before that let's discuss few things, okay? Let's discuss few things. I think you all have seen the diagram like this, some diagram you know, like this. I'm not saying it is totally accurate, but like this. Just say if you have seen this part is known as hypothalamus, okay, this part is basically the infundibulum, okay, it is the infundibulum, also known as stalk, right, also known as stalk-like structure, stalk-like infundibulum is there, okay, and this part here is known as your pituitary. This part here is known as your pituitary. Are you getting my point? This part here is known as your pituitary. Clear, bache? So when you talk about the hypothalamus, hypothalamus is the super master gland, okay, it is the super master gland, and when you talk about this pituitary, it is the master gland. When you talk about pituitary, it is the master gland, okay. So why this hypothalamus is super master gland? Because it is controlling the master gland, right? Why this hypothalamus is the super master gland? Because it is controlling the master gland. That's the point, okay, that's the point. Chalo, now let's discuss something else. First of all, let's focus on the pituitary, the flow chart. I think I have discussed it so many times. So pituitary is also known as hypophysis. Pituitary is also known as hypophysis. Pituitary, it is also known as hypophysis, okay. So bache, we divide pituitary into two parts. One is adenohypophysis. Trust me, if you will remember this now, you will, right, that will be helpful for you. Adenohypophysis, and this one is neurohypophysis. This one is neurohypophysis. One is adenohypophysis, one is neurohypophysis. One is adenohypophysis, one is neurohypophysis. So adeno, anterior part, actually it is further having two things. It is having the anterior lobe of pituitary, it is having the anterior lobe of pituitary, and it is also having the middle or the intermediate lobe of pituitary, okay. It is having the middle and the intermediate lobe of pituitary. Are you getting my point? middle and the intermediate lobe of pituitary. So even if you will read NCERT, you are going to find a word here that this anterior lobe is known as pars distalis. It is also known as pars distalis, and this is also known as pars intermedia. This is also known as pars intermedia. Clear bache? It is pars distalis, it is also known as pars intermedia, and here it is posterior lobe, right bache? It is posterior lobe, it is known as pars nervosa, right? It is known as pars nervosa. Are you getting my point? It is known as pars nervosa. So they are having, right, they are having the direct neural regulation. This is under the direct neural regulation. Let me explain what's that. First of all, focus on this flow chart. Pituitary, adenohypophysis and neurohypophysis. So adenohypophysis is further having two things: anterior lobe of pituitary, middle lobe of pituitary, or intermediate lobe of pituitary. It is not properly separated, that's why we take it together. Okay, anterior lobe of pituitary, intermediate lobe of pituitary, that's the point. Okay, that is the point that we have. Now here you are having the neurohypophysis, which is the posterior lobe of pituitary. Now students, now what you need to remember, right? See, say there is a trick. Go to AC parlor, let's have facial. Everyone, go to AC parlor, let's have facial. That's how you have to write it. Go to AC parlor, let's have facial. Go to AC parlor, let's have facial. Quick, quick. Go to AC parlor, let's have facial. Okay, this is our trick. So go to AC parlor. G stands for GH, that is growth hormone. T stands for TSH, that is thyroid stimulating hormone. AC, adrenocorticotropin hormone. Parlor, that is known as prolactin. That is known as prolactin. Let's have LH, luteinizing hormone. Facial, FSH, follicle stimulating hormone. Right, it is the follicle stimulating hormone. Is that clear? So go to AC parlor, let's have facial. So you have growth hormone, thyroid stimulating hormone, adrenocorticotropin hormone, prolactin is there, luteinizing hormone is there, and then comes the follicle stimulating hormone. Okay? So these two are gonadotropins. Why? These two are gonadotropins. Why? Because they act on the gonads. They act on the gonads, and gonads are the primary sex organ. So tell me, any difficulty here? Can't you just read it and say go to growth hormone is responsible for growth. TSH will act on thyroid gland, will stimulate thyroid gland. Adrenocorticotropin hormone will stimulate adrenal gland. Prolactin, you remember human reproduction, prolactin, it is important for the milk production. It will act on the alveoli of the milk. It helps in milk secretion, and these two will act on the gonads. That we have completed in the, right? We have completed in the human reproduction. So tell me, what is the difficulty here in this chapter? So repeat the trick. Go to AC parlor, let's have facial, growth hormone, thyroid stimulating hormone, adrenocorticotropin hormone, prolactin, luteinizing hormone, and FSH, gonadotropin. Now when you talk about the middle lobe, it will release only one hormone, that is MSH, melanocyte stimulating hormone. I really don't think that it is a difficulty. Melanocyte stimulating hormone. So melanocyte matlab, right, melanin producing cell, of course, so it will act on it. Now when it comes to the posterior lobe, they will release, the word is release here. The word is release here. They will release two hormones, vasopressin. They'll release two hormones, vasopressin, and which is also known as ADH, antidiuretic hormone, and they will release oxytocin. They will release oxytocin, which is the bonding hormone, love hormone, milk ejection hormone. Take a milk let down hormone. Tell me, first of all, tell me this part clear. First of all, tell me this part clear. If this part is clear, now let me tell you one more thing. Now your hypothalamus is the super master gland. Okay? Your hypothalamus is the super master gland. What is the role of this hypothalamus? This hypothalamus is going to guide the pituitary that now you have to release the hormone, you have to inhibit the hormone. Right? Understand it? Understand it, then everything will be easy. So hypothalamus releases two types of hormone. Two types of hormone, releasing hormone and inhibitory hormone. Releasing means it will tell pituitary to release something. Inhibitory hormone will tell pituitary to inhibit the secretion of something, isn't it? So it is the name that is making it clear. Its name is making it clear, isn't it? So releasing hormone or inhibiting hormone, this is what we know. This is what we know. Chalo, can I ask you one question? Can I ask you one question? Can I ask you one question? I'll relate everything, don't you people worry. You just need to allow me 1 more hour to finish this. Okay? So can be related? Okay, देखो, growth hormone is not acting on any other gland. Growth hormone, it is having another name. It is also known as somatotropin. Do you know that? It is also known as somatotropin. It doesn't act on any other gland, right? Pituitary is releasing it. Okay, it is not acting on any other gland. Pituitary is releasing it and it will help in the growth. Thyroid stimulating hormone, it will act on another gland. Yes. ACTH, it will act on another gland. Yes. Prolactin will directly release, right? It is not acting on any other gland, it is directly acting on the alveoli, right? And will tell them to secrete the milk. These two are also will act on gonadotropins. Okay? MSH, melanocyte stimulating hormone, again pituitary is directly controlling it. Pituitary is directly telling melanocytes to do this thing and that thing. This part clear? Now, why am I telling you this? Let me tell you. You all know that even in the chapter human reproduction we have studied that these hypothalamus will release GnRH, growth hormone releasing hormone. It will release thyroid stimulating hormone releasing hormone. Okay? I told you na that 2 types of hormones are released by the hypothalamus, releasing and the inhibitory. So देखो, releasing में जैसे gonadotropin releasing hormone, they will stimulate the pituitary to release the FSH and LSH. ठीक है? Like this. Right? वैसे ही ACTH, RH, adrenocorticotropin hormone releasing hormone, thyroid stimulating hormone releasing hormone, right? Prolactin releasing hormone. Can you relate it? Yes? Can you relate with it? Yes, people, tell me. Can you just relate with it? I think it's easy, right? It's easy, right? So it will also tell them to release growth hormone releasing hormone. Right? Growth hormone releasing hormone. Am I right? Yes, bache, am I right? Yes. Okay, one is pending melanocyte stimulating hormone releasing hormone. So that will be released, right? That will be released. Now when it comes to the inhibitory hormone, it will release only 3 inhibitory hormone. Let me tell you which one. Prolactin inhibitory hormone, growth hormone inhibitory hormone. It is also known as somatostatin. This is important. Okay, it is also known as somatostatin. So it will also release melanocyte stimulating hormone, inhibitory hormone. These 3 inhibitory hormones are released by your hypothalamus. Right? This can be asked as a question. These 3 inhibitory hormones are released by your hypothalamus. Now, I will relate it with something else. Dekho, I told you clearly that prolactin released by pituitary, right? Directly pituitary is controlling that. Growth is also directly controlled by pituitary. This melanocyte wala part is directly controlled by pituitary. So hypothalamus has formed only that inhibitory hormones, right, which are, you know, directly the hormones which are directly released by pituitary, right? Only their inhibitory hormones are produced by hypothalamus. Hypothalamus is not producing thyroid stimulating hormone, inhibitory hormone. No, because it will work on feedback mechanism. When pituitary gland will act on thyroid, in response to that, thyroid will release something. Then when, you know, there is a feedback mechanism, feedback positive, feedback negative. Just say, let's say if something is increasing, sometimes it sends a signal to its to the cell that is secreting it, ki bhai, you have to stop the secretion. You can relate it with spermatogenesis. So that's what you need to focus. That is what you need to focus. Is that clear? That is what you need to focus. Is that clear? Is that clear? Now look at this part. See, there is a portal system in between this. There is a hypophyseal portal system. What's there? There is a hypophyseal portal system. जैसे there is a blood circulation का system. बच्चे, your hypothalamus is having these hypothalamic neurons. ठीक है? Your hypothalamus is having these hypothalamic neurons that produces the hormones, right? These are the cells that will produce neurohormones. Hypothalamic neurons. Some student was using the word nuclei. Yeah, we use the word nuclei for it. right, you are right, we use the word nuclei for it. These are the neurosecretory hormones, they will release the neurohormones. ठीक है, जैसे ये when it comes to the hypothalamus and adenohypophysis, when it comes to the hypothalamus and adenohypophysis, okay, जैसे hypothalamus will act on adenohypophysis and it will also and on the neurohypophysis. So बच्चे, the point here is, okay, the point here is, one is adenohypophysis, another is neurohypophysis. So this hypothalamus is right going to so when it comes to the adenohypophysis, so hypothalamus, from its hypothalamic neuron, it will release that hormone in the bloodstream, right? It will release that in that hypophyseal portal system. So hypophyseal portal system is nothing, it is just that blood vessels, right? It is just that blood vessel. So it will carry that hormones to the adenohypophysis. For an example, just say, let's say your boss is not calling you directly, your boss is telling your task to your to his PA, right? And that PA is conveying it to you indirectly, ठीक है, indirectly. So this is the relation कि boss is like Mr. PA, you please convey this to team member A or team member B. Boss is not directly going, this is the first part. Now secondly, when it is the neurohypophysis, so in the NCERT there is a one line that it is under direct neural regulation. It is under direct neural regulation of hypothalamus. What is the meaning of that? That directly that neuron from the hypothalamus, directly that neurosecretory hormone will directly pour its neuron here in neurohypophysis. Just imagine your boss is not telling you anything directly, your boss is conveying it through the PA, but now you have one colleague and your boss is like every time coming to the colleague, are you okay? Are you having something? Okay, are you comfortable here? If you're not able to do your work, that's fine, you can leave that too, right? So you will be curious now, why is it so? Why that biases are there? So obviously later on you will get to know कि your colleague is the relative of your boss, right? That is the relative of your boss. So same is the case here. During embryonic development, this hypothalamus and the posterior lobe of pituitary, this hypothalamus and posterior lobe of pituitary, this hypothalamus and posterior lobe of pituitary, they formed from the same group of cells. So that's why these biases are there. And let me tell you, this neurohypophysis, it is not even a true endocrine gland. It is not even considered as a true endocrine gland. Why? Because it doesn't form hormone by its own. This hypothalamus, from these hypothalamic neurons, hypothalamus will form these two hormones, right? The vasopressin, also known as antidiuretic hormone, and your oxytocin, they both are formed by, right? Vasopressin and your oxytocin, they both are formed by hypothalamus. So hypothalamic neuron will give it to neurohypophysis ki chalo, shh, I am giving these hormones to you. Whenever I will give you the signal, you please release it. So release is the word. So posterior lobe of pituitary is not making any hormone by its own. The hypothalamus will give that hormones to the posterior lobe of pituitary, and posterior lobe of pituitary in return to that will release it. Are you getting it? So that's why it is not even the true endocrine gland, right? So hypothalamus is showing a favoritism for posterior lobe because during embryonic development they formed from the same type of cell. Okay? So hypothalamus is strict with this adenohypophysis, not with the neurohypophysis. It is treating neurohypophysis as favorite. So that's why we say direct neural regulation. Okay, that's why we say direct neural regulation. Here देखो, directly that hormone is coming to posterior lobe, but here it is releasing it in portal circulation and then it is coming to the anterior pituitary. So see how bad it is. So this is something that you need to understand. Secondly, you need to understand the thyroid gland ka structure. Rest everything is just like a story, bacha log, and trust me, you have to revise it. If you are interested in this chapter, then only you can master this chapter. That's the beauty of this chapter. Right? If you think nahi yaar, I am not interested, I don't want to do it, right? So you will find that it is a burden, okay? then you will be like, yeah, it is a burden. Okay. Okay, then it is a burden. ठीक है? Okay, chalo. So see, this is given here. The hormones are also given here. Right. So that's it. That's all. This is what I have already explained. Right. So growth hormone will obviously help in the growth. So I will be telling you the disorders. Then you have to read it by your own. So who's ready to read it and revise it? Who is ready to read it and revise it? Who's ready to read it and revise it? Yes, बच्चे गुना, चीनू, समीक्षा, पूजा, लेहारिका. Yes. Pakka? Sure. So do you want to get 360 out of 360? Do you want to get 360 out of 360? Chalo, very good, very good. So disorders, important. Growth, over secretion of growth hormone will result in gigantism. Okay, and low secretion will result in stunted growth, that is dwarfism. Okay, so growth hormone is more, that is gigantism, right? It is gigantism. If it is less, dwarfism. Okay, and sometimes in middle age, right? Matlab, the childhood is normal. Sometimes in middle age, na, the secretion of pituitary hormone, it increases, but it is not something uniform. It results in disfigurement. Just say, let's say hand, the fingers of hand, they will keep on growing like this. So you should know about it, right? So when there is the severe disfigurement, especially of the face, acromegaly, it is acromegaly, right? So it can also result in premature death. So in the paper they can ask you acromegaly is caused by, what will happen in acromegaly. ठीक है बच्चे, ठीक है. Then prolactin, you know that its role, TSH का role done. ठीक है. ACTH is going to act on the thyroid hormone, adrenal gland, it will release glucocorticoid. Done. So do you want me to explain the role of LH and FSH also? Do you want me to explain the role of LH and FSH, luteinizing hormone and FSH? Luteinizing hormone in the case of ovaries, in the case of female, it is responsible for ovulation. It also forms corpus luteum, that is also that will release progesterone, and FSH will act on the follicle cells, will help in their proliferation, will help in their growth, and in response to that FSH will release ovarian cells will release estrogen. Now in the case of males, your FSH will act on Sertoli cells. Sertoli cells will nourish the developing spermatozoa, and your LH, luteinizing hormone in the case of males, right? In the case of males, that luteinizing hormone, it is also known as ICSH. Do you remember that? It is also known as ICSH. Luteinizing hormone in the case of males, it is interstitial cell stimulating hormone. Okay, it is also known as interstitial cell stimulating hormone. Okay, so it will act on that interstitial cells, and in response to that, that will release testosterone. Hope it is clear, right? I hope it is clear. So what is the next part here? So next part may MSH, melanocyte stimulating hormone, which will act on melanocytes. So melanocytes are melanin containing cell, and what is melanin? Our skin pigment. Okay, so they will regulate pigmentation of the skin. So O for out. So oxytocin will act on smooth muscles. It is responsible for rhythmic contraction and relaxation. Oxytocin, that is why it is helping in the parturition, because it will be released, it will act on smooth muscles, then there will be the contraction and relaxation, and childbirth will be there. Oxytocin acts on smooth muscle, it helps in peristalsis, right? So when baby will give the suckling stimulus, when baby will suck the nipples of the mother, the milk will come out. So it is also known as milk ejection hormone or milk let down hormone. Milk ejection hormone or milk let down hormone. So it is also known as bonding hormone and love hormone. ठीक है, bonding hormone and love hormone. ठीक है बच्चे, ठीक है. So when it comes to the vasopressin, it is antidiuretic hormone. So it will mainly act at the kidney and stimulate the reabsorption of water and electrolytes right from the distal tubules, and it reduces the loss of water. So it is antidiuretic. It will prevent diuresis. Diuresis means excess of urination. It will prevent excess of urination. Okay? So we will relate this one with the we will discuss this one in the excretory system. Okay, so let's say when you are having excess of tea ya coffee, ya someone is alcoholic, right? If they are having alcohol, what will they do? They will again and again go for urination, again and again they will go for urination, right? For है ना? Why? Because at that time when these beverages are there in our body, they lower down the secretion of ADH. So if in our body ADH is less, right, then it will result in the excessive urination. Okay? And when will ADH be less? When in our body blood fluid volume is normal. When there is no need for excessive absorption of water, ADH will be low. But if because of any any impairment, right, due to which if there is the release of ADH will be less, if it is diminished, right, if it is less, if it is diminished, so what will happen? There will be the water loss and dehydration. So it is diabetes insipidus. So when it is diabetes mellitus in the urine, these two are different. Diabetes mellitus means in the urine there is the glucose. You remember diabetes mellitus? It is having glycosuria, means glucose will be there in urine, and ketonuria, means ketone bodies are there in urine. Okay, so it is telling us that metabolism is not proper. And when it is the diabetes insipidus, okay, when it is the diabetes insipidus, okay, then in that case what is there? There is the low. It is because of hyposecretion of ADH. It is because of hyposecretion, less secretion of antidiuretic hormone. It is because of the less secretion of antidiuretic hormone. ठीक है बच्चे, diabetes insipidus. It is because of the less secretion of ADH and that will result in polydipsia. ठीक है, means excessive thirst will be there. Okay, excessive thirst will be there. Is that clear? Is that clear? ठीक है, so next is your thyroid gland. Okay, so here you can see that it is on the either side of your trachea, and when you look at it, it is also a bilobed structure. So when you look at this thyroid, it is having two lobes, and in between this part is bache isthmus, right? It is the non-glandular and the connecting part. It is non-glandular. Non-glandular means it is not secretory. ठीक है, non-glandular means it is not secretory. It will not secrete something. These lobes are going to secrete something, right? These lobes are going to secrete something. Clear बच्चे? Clear बच्चे? Right? So let's finish this part and then let's move to next part. So tell me if there is any doubt here. Any doubt here? Sure. Be quick, people, please. Yes, be quick. Done? Sure? Sure. Chalo, let's go. Let's discuss this thyroid part, because thyroid gland is important one. है ना? It is the important one. So let's talk about it, okay? Let's talk about it. So first of all, let's take a section of it. So when we take the section of the thyroid gland, we are going to get something like this. We are going to get something like this. Tanana. This is interesting, okay, and this is important as well, so you please pay attention. Okay, so you are taking a cross section of the thyroid gland. What are we doing, people? We are taking the cross section of the thyroid gland. So when we take the cross section of the thyroid gland, this is what you are going to get. Okay, just a minute. That's what we have. Take it. Right, that is what we have. So you need to understand one thing here. Just when you talk about the thyroid, right, when you talk about a thyroid, so it is having some cells. First of all, thyroid is the largest endocrine gland. But overall, right, overall if they will ask you the largest gland of our body, then it is liver. But if they will ask you largest endocrine gland of our body, largest endocrine gland of our body, then it is, yes bache, then it is thyroid. So you will see two things here, right? We will see follicles here and stromal tissue. Follicles, you understand now? Follicles mean group of tissue. Okay, what is the meaning of follicles here? What is the meaning of follicles here? Follicles mean group of tissue. So it is having the follicles and stromal. It is having the follicles and stromal tissue. Right, what will it contain? It will contain follicles and stromal tissue. Take it, bache? Right, bache? Yes, yes, yes. So now when you look at this diagram, these cells are what? They are the follicular cells. And these follicular cells, these are the follicular cells. And what is it? It is the cavity. It is the cavity where thyroid hormones will be stored. This is something important, please pay attention. Normally, hormones, they form in trace amount, right, when required. But only in the case of thyroid gland, these hormones can form and they can get stored in this cavity as a colloidal solution. They can even get stored here for 90 days, 90 days. That is something very important. So you have the follicular cells here, and this surrounding part is the stromal tissue. These are the parafollicular cells. Okay, these are the parafollicular cells. We also call them as C cells. Okay, these are the parafollicular cells. We also call them as C cells. Can you see that? So these follicular cells, they release T3, T4. What do they release? They release T3, T4. What do they release? These follicular cells, what do they release? They release T3, T4. So it is triiodothyronine and tetraiodothyronine. Tetraiodothyronine. Can you see that? Triiodothyronine and tetraiodothyronine. Can you see that? Triiodothyronine and tetraiodothyronine. So this tetraiodothyronine is also known as thyroxine. Okay? So it is produced in more amount. It is produced in more amount, but it is less potent. Just let's say कि some people they study for 12 hours, 13 hours, but when it comes to their productive hours, they are very less. Okay, they are sitting for 12 hours, 13 hours, but when it comes to the topic that they have covered, like it is very less, like this. Okay, so triiodothyronine, it is more potent. Okay, what's that? It is more potent. Are you getting my point? It is more potent. Is that clear? So T3 and T4, T3 and T4. So these follicular cells, they will release triiodothyronine, tetraiodothyronine, which is also known as thyroxine. Now when you talk about these parafollicular cells, which are also known as C cells, they release calcitonin. What will they release, people? They will release calcitonin. What will they release? They will release calcitonin. It is having a role in the calcium metabolism. But first of all, tell me, is that clear? So in this jelly-like suspension, these thyroid hormones will get stored. Okay? So let's discuss that. Okay, just remind me, I missed the pineal gland. We have to discuss that also. So let's quickly revise everything from the NCERT. So thyroid is having two lobes. Okay, it is butterfly shaped, right? So both the lobes are interconnected with a thin flap of connective tissue, which is isthmus. So isthmus is non-glandular. Okay, isthmus is non-glandular, means it is not secretory. It will not release anything. Any doubt? It is not secretory. It will not release anything. Any doubt? Any doubt? So thyroid is having follicles and stromal tissue. So thyroid follicle is having follicular cells. They are having a cavity. So this is what they are going to release. ठीक है. So we know that for these two hormones iodine is required. If iodine is less, then obviously these hormone production will be affected. So when there is the hypothyroidism, when thyroid is less, you know, some people they say ki we have the thyroid problem and we are gaining the weight, right? That is hypothyroidism, where weight gain is there. Why? Because metabolism will slow down. That person will have very lethargic, that person will be lethargic, they don't feel like doing anything. Even depression can occur, even because the metabolism is very slow when it is hypothyroidism, right. And if there is the enlargement of thyroid gland, you will call it as goiter. So goiter में different ways. This is the simple goiter when there is the enlargement of thyroid gland. So if in our salt iodine deficiency is there, it can also result in goiter. So sometimes बच्चे mostly during pregnancy also hypothyroidism occur. If it occurs, it can retard, right? It can, there will be the defective development and maturation of the growing baby, and due to that cretinism will occur. So cretinism, this is important. Cretinism is a condition when female is having hypothyroidism during her pregnancy. You have to be specific here, right? She is having the hypothyroidism during her pregnancy. Are you getting my point? During her pregnancy. So baby will have stunted growth, mental retardation, low IQ, abnormal skin, deaf, and mutism will be there. Okay, and if there is the hypothyroidism, so there are two types of disorder, hypo or hyper. Hypothyroidism when it is less, hyper when it is high. So hypo में menstrual cycle can be irregular, right? If cancer of thyroid gland is there, means excess of development of thyroid gland is there, so thyroid hormones will be more, so this is hyperthyroidism. So I think this is something that you can answer. So बच्चे, when it is the simple goiter, it is hypothyroidism, but if it is the exophthalmic goiter, can you read that? If it is the exophthalmic goiter, what's that? If it is the exophthalmic goiter, which is also known as Graves' disease, which is also known as Basedow disease, right? It is Graves' disease is also known as Basedow disease. So cretinism and this one is important. So it is hypothyroidism. So बच्चे, enlargement of thyroid gland, eyeballs will be protruded out, metabolism, BMR will be high, weight loss will be there. This is the Graves' disease. Okay, so thyroid hormone is very important for our basal metabolic rate. They support the process of RBC formation. So sometimes students, they miss it. They are not aware that it is also helping in the RBC formation. It is also helping in the RBC formation. ठीक है बच्चे? So metabolism of carbohydrate, protein, fat, the maintenance of water and electrolyte balance. This is also the another point that students miss. ठीक है? And the another hormone I told you ना, TCT, your C cells, also known as stromal cells. Stromal cells, they release calcitonin. It is also known as thyrocalcitrophin. It is also known as calcitonin. It is also known as calcitonin. So बच्चे, it maintains the calcium metabolism. How? When there is, see, when in the blood there is high calcium, so TCT will be released, right? TCT will be released. So this calcium will be absorbed by bones. Excess of secretion of calcium from the food will be there. Stomach will absorb less calcium. So calcitonin will help in maintaining the calcium metabolism. Let me start the PCT, then we will relate these two together. Okay? Then we will relate these two together. So before starting the parathyroid gland, let me tell you about the pineal gland, because it is something very important, because it maintains the circadian rhythms of our body. It is going to maintain the circadian rhythms of our body. It is maintaining the biological clock. How our body get to know that it is the time we should sleep, right? During night, why do we feel sleepy? Yeah, why our body is able to regulate that periods properly? Why? Because of this pineal gland. So it is located on the dorsal side of forebrain. So this question can be asked. So where is it present? In the forebrain. Okay? So it will release melatonin. This is the what we have to discuss. So it will maintain that 24-hour rhythm, bache. Okay, so it will maintain that sleep-wake cycle, body temperature. It also influence metabolism, pigmentation, and the menstrual cycle. Okay, that's why bache, when our sleep cycle is disturbed, even our menstrual cycle can get disturbed. Okay, Then is the parathyroid. Just let's say if this is the thyroid gland, just behind the thyroid gland, in each lobe there will be two structures like this. These are 4 parathyroid glands. Okay, these are 4 parathyroid glands. So parathyroid glands, they are 4. They are on the back side of the thyroid gland, right? So they will release hormone PTH. It's a peptide hormone, parathyroid hormone. But it is also known as Collips hormone. Write down. It is also known as Collips hormone. Write down. It is important. It is also known as Collips hormone. Please write it down. Okay. So it is hypercalcemic hormone. Right, it is hypercalcemic hormone. It increases the calcium level in blood. It is hypercalcemic hormone. Please write down. Done? Sure? It is the hypercalcemic hormone. Quick. Done? Okay. Dekho, it's very simple. It will maintain the calcium level. I'll tell you. Okay, diagram is not there. ठीक है. देखो बच्चे, when in our blood the calcium level is low, you know, in 100 gram of blood, okay, can you tell me this amount? 100, not gram, 100 ml of blood should have 12 milligram of calcium. Do you know that? 100 ml of blood contains 12, it should have 12 milligram of calcium. But if is the calcium level is low, it will send a signal and PTH will be released. Okay, it will send a signal, PTH will be released. This PTH will increase calcium absorption from food. Okay, so this will increase calcium absorption of food, plus it will decrease its excretion. Moreover, it will so it will cause bone reabsorption of calcium from bone reabsorption, means it will start acting on bones. Bones will start releasing the calcium. Are you getting it? Bones will start releasing the calcium. So imagine if the secretion of PTH, if it will be very high, don't you think it will release more and more calcium from bones, and that can result in osteoporosis, porous bones? Don't you think it can result in osteoporosis? Yes, people, tell me. It can do that, right? So that is how it works. So there should be a balance, right? The hormone level should be optimum. It should not be very high, it should not be very low. It should be in that standard range only. Okay? It should be in that standard range only. So when it comes to the calcium metabolism, this Collips hormone, this TCT, and vitamin D3, that is your calciferol. Vitamin D3, that is your calciferol, right? These 3 hormones, they maintain calcium level. They maintain calcium homeostasis in our body. These 3 hormones. So these 2 are antagonists. What is the meaning of antagonist? Their function is opposite. Okay. Okay, so it is hypercalcemic, it is hypocalcemic. Done, bache? Done. So parathormone, PTH, it stimulates. Remember osteoclasts. So if they will stimulate osteoclasts, it will, these cells will start digesting the bones. And that is how they will meet out the need, right? The need of the calcium. Okay, bache? That is how they will meet out the need of the calcium. Clear? Done? Okay. So hypersecretion can cause osteoporosis. And hyposecretion can result in tetany. You remember tetany? So tetany mein there will be wild contractions because calcium level is low. So obviously when vitamin D low, see, even vitamin D is low, low calcium absorption, and even it is also playing the role, it is also include PTH. So PTH increase the calcium level, so it acts on bone, stimulate the process of bone reabsorption, right, means dissolution of bone, yeah, demineralization of bone. Okay, so it also stimulate reabsorption of calcium by renal tubules, right, so it will be absorbed, so it will increase calcium absorption from digested food. So PTH is hypercalcemic, it increases the blood calcium level. Understood? It increases the blood calcium level. Next is thymus. I think you all know that thymus, when we attain puberty, its size is large. With time, its size decreases. That's why immunity decreases, and it will release only one hormone, that is known as thymosin. Do you remember that? Do you remember that? Yes. So can we just move quickly? Right. So it is situated. It's a lobular structure located between lungs, behind sternum on the ventral side of aorta. So if I have to confuse you, I will pick up this statement as a question. Instead of ventral side, I will mention the dorsal side. So if you will not read it carefully, you will make a mistake. Okay, right. So where is it located? The thymus gland is a lobular structure. It's a lobe-like structure which is located between lungs, behind sternum on the ventral side of aorta. Right, on the ventral side of aorta. Understood? Understood? Right, so it will release thymosin, which is a peptide hormone. Okay, so it will play a major role in the differentiation of T lymphocytes, so it will maintain cell mediated immune system. Okay, done. Next, adrenal gland. Okay, it is the cap of kidney. Adrenal, adjacent to renal, means adjacent to kidney. So it is the adrenal gland, cap of kidney. Can we discuss this now? Can we discuss this now? So bache, your kidneys, your adrenal gland, your ovary, they are having two parts, right? Just if I talk about the adrenal gland also, middle one is adrenal medulla, outer one is adrenal cortex. Outer one is adrenal cortex. Okay? So they will release different hormones. When you talk about adrenal medulla, they will release amino acid derived hormones. Means these hormones are made up of amino acids, right? We call it as catecholamines. This is a previous year question. We call it as catecholamines. Okay? They are amino acid derived. So what's the name that we have? This is epinephrine. It is epinephrine, or it is norepinephrine. It is epinephrine or it is norepinephrine, right? So norepinephrine is also known as noradrenaline, remember? And this one is also known as adrenaline. So you know that these are the emergency hormones. They will be released when there is a emotion of fight or flight. Okay, right? And you know na, in the case of smokers, nicotine also increase the release of these hormones. So that's what you need to remember. Now when it comes to adrenal cortex, the story is little different. Okay, when it comes to the adrenal cortex, the story is different. Okay, what? So here you will talk about 3 layers. So for that 3 layers, the formula is GFR, GFR. For that 3 layers, the formula is GFR, GFR. What's the formula, Priya? It is GFR. Dekho, layer means zona. So outermost is zona glomerulosa. It's GFR. Then zona fasciculata. Then zona reticularis. So we have 3 layers. It is GFR. Zona, glomerulosa, fasciculata or reticularis. Okay, or reticularis. These are the 3 layers, and these 3 layers will release different types of hormones. Is that clear? These 3 layers are going to release different types of hormone. Clear, bache? Clear, 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 clear. Sure. So jaise these, they will release mineralocorticoids. Mineralocorticoids, they all are steroidal hormones, right? It will release glucocorticoids, and it will release sex corticoids. Okay, so everyone in the chat section, please repeat GFR. Everyone in the chat section, please repeat GFR. Quick. Please repeat GFR. GFR. It's GFR. Done? So mineralocorticoids, you know that we have the example of aldosterone. Here we have cortisol or cortisone. And this is the sex corticoids. We also call them as androgens. Okay, we call them as androgens. So the name of the hormones should be clear. Okay, the name of the hormones should be clear. Okay, so one is cortisol, another is corticosterone. Done? Chalo. Let's focus. And yes, A for adrenal, A for Addison. Everyone, A for adrenal, A for Addison. Repeat it. A for adrenal, A for Addison. The question can come for this part. A for adrenal, A for Addison. So when there is the underproduction of hormones by adrenal cortex, it is specifically adrenal cortex, it can alter carbohydrate metabolism, weakness and fatigue will be there. This is the Addison disease. I still remember I was solving PYQs and I got this question. So this is how I remember adrenal Addison, adrenal Addison, adrenal Addison. Okay? Okay. Categola means clear, the guffer clear, guffer, guffer, guffer. So this is what we have, or let's go. So see, adrenaline and noradrenaline, they are catechola means they are rapidly secreted in response to stress. That's why they are emergency hormones or hormones of flight. Okay, so obviously when there is emergency, alertness will increase, pupils will be dilated, pupillary dilation will occur, piloerection, raising of hair. You know na goosebumps, right? When we are in a troubled situation, we get that goosebumps. So goosebumps means these hair of our body, they will be erected, piloerection. Take a raising of hair, raising of hair. Right, so sweating will be there. Obviously, when you are in an emergency situation, say sweating, alertness, right, that increase in heartbeat, anxiety, all that things will be there. So see, heart contractions will strengthen, the heartbeat will increase, rate of respiration will increase, and even the breakdown of glycogen will also increase. That will result in increased concentration of glucose in blood. Okay, so breakdown of lipid and protein will also be there. So whatever in NCERT, just revise that. As of now, that will be more than sufficient for you. Okay, as of now, that will be more than sufficient for you. Done, bache? Then zona reticularis, fasciculata and glomerulosa is there. So they will secrete corticoids. They are steroidal. Okay, so they will play a role in carbohydrate metabolism. Glucocorticoids released by zona fasciculata. So cortisol is main glucocorticoids. So they regulate balance of water and electrolyte in our body. They are mineral corticoids. You remember aldosterone? Do you remember aldosterone? So it helps in the conditional reabsorption of sodium. Related with the NCERT, bache, in excretory products. So conditional reabsorption of sodium from the distal part. Remember? Conditional reabsorption of sodium from the distal part. Remember this aldosterone. So they will ask on the they will act on the distal convoluted tubule and collecting duct. So they will activate sodium potassium pump, bache. So reabsorption of sodium will be there and excretion of potassium and phosphate. This is what you should note down because it is a PYQ. Okay, so aldosterone, it will reabsorb sodium and excretion of potassium and phosphate is there. Okay, this is what you need to note down. Excretion of potassium and phosphate is there. Understood? Right? So that's going to happen. Understood? Yes? Okay? So if it is hyposecreted, then obviously loss of sodium, calcium and water will be there. Potassium and phosphate and potassium will increase. Done, bache? Done, done, done. Sure? Sure. So this line is important. Please write down. This line is important. Please write down. Okay, please write down. So glucocorticoid, they will stimulate gluconeogenesis. What is the meaning of neo? Neo means new, right? Genesis means formation. Neo means new, genesis means formation. So it is gluconeogenesis, means basically from non-carbohydrate source, the formation of glucose, right? Formation of glucose from non-carbohydrate source. Okay, so formation of glucose from non-carbohydrate source. right? Then there is the lipolysis, means breakdown of lipid. Okay, there is the breakdown of lipid, because ultimately we have to make glucose. Proteolysis, breakdown of protein, right? So cell uptake of that amino acid and their utilization will be decreased. Okay, so cortisol is also help in maintaining cardiovascular system as well as kidney function. So this is something you need to revise again and again, and do not forget this line of sodium and water, right? So this is your homework bache, right? 3 times we have discussed. Jaise thyroid gland also help in RBC production, cortisol also help in RBC production. So you need to mention that lines. You need to mention that line, that which hormones are playing role in the RBC formation. Done bache? Okay. Okay, so sexocorticoids will also help. They will help in the growth of axial hair, pubic hair, facial hair. So that's what you need to revise. There is nothing that I have to explain. So in next 10 minutes this chapter will also be over. So you cannot say that ki ma'am is just giving us the overview. No, I am discussing things in detail. Okay, so after this, which chapter you want to start? Neural control, because neural control mein mainly it is the ionic theory. Ionic theory is conceptual, brain is again memory based. So brain, if you have studied before, then that's fine. Otherwise, just check out the PYQs and remember that. Neural control, theek hai, we will start neural control. But biomolecules is a time taking chapter. So let me finish neural control, then let's go for excretory products, body fluid and breathing, then biomolecules. Okay? And if you are, see, let me tell you, ye dekho, if you are so interested in biomolecules, I know you don't have time as of now, but you will see that I have taken a lecture on it, see, in detail. Okay, right? I know it's lengthy, you can skip some part, you can increase the speed and you people can revise it. Okay, biomolecules. Even other than biomolecules, देखो frog as per new NCERT syllabus is also discussed here. Structural organization though we have already covered, or breathing and exchange of gases. If you want it in detail, that's why I'm asking. That's why I'm saying. Okay, bache, and do not forget. See, so bache, do not forget about the ARAM 2024, right? So we are launching 3 batches for the students that are moving from 10th to 11, 11 to 12th, and those are droppers. So from 17th of April classes will start. In the description box there is a link, and at this time till 14th of April you will get this batch at a price of only 4,999. That's all, right? That's all. So guys, you can suggest this thing to your friends, to your juniors, those who are looking for better classes, right? You know our content, there is no compromise in the quality. We are not just providing you the quantity, it is the quality content. Trust me, questions are going to come from this part only. And you can even check our last year's marathon, whatever we have taught in that 15 days, that was there in the NEET paper. Okay, that was there in the NEET paper. Even animal kingdom in detail, Amrita, it is also there. I will teach you the basics. I'm not saying that I will leave that, but what am I saying? I am saying that if you want a chapter, just sometimes we have difficulty in a particular chapter, we know the another chapter, so you can check old videos. You can check old videos. I'll give you the break once. Let me finish this. I'll give you the break. Let me finish this. I'll give you the break. Pancreas, okay, it's a heterocrine gland, it's a composite gland, it's a heterocrine gland, it's a composite gland, it's a mixed gland. What type of gland it is? It is a mixed gland. What type of gland it is? It's a mixed gland. Are you getting it? Are you getting it? Heterocrine, composite, or the mixed gland. So when you talk about its endocrine part, it will be having islets of Langerhans. These are the cells that are going to make it, islets of Langerhans. And when you talk about its exocrine part, which is less than 1%, less than 1%, yeah, endocrine part is less than 1%. Tell me please, which part is less than 1%, endocrine or exocrine? Of course, this endocrine gland is representing. So it is having islets of Langerhans, so that are basically 1 to 2 millions, right bache, and it is just making 1 to 2% of the pancreatic tissue. 1 to 2% of the pancreatic pancreatic tissue. Are you getting my point? It is making just 1 to 2% of the pancreatic tissue. So islets of Langerhans. Are you getting my point? So exocrine part, it will release the digestive juices, bache. It will release what? It will release the digestive juices, and you will see the acini here, right? Surrounding में acini are there ना, जैसे see cells like this. So acini will be there. Okay, so exocrine part is there, endocrine part is there. So 1 to 2% of the pancreatic tissue, islets of Langerhans. So you are having alpha cells, beta cells, delta cells, pancreatic polypeptide cells here. So alpha cells, beta cells, delta cells, PP cells means pancreatic polypeptide. Right, they are pancreatic polypeptide cells, and bache, they are also known as F cells. Okay, they are also known as F cells. So alpha cells, they release glucagon, again a protein hormone. Beta cell, it will release insulin. Delta cell, it will release somatostatin. So this thing you should note down, that somatostatin is a growth hormone inhibitory hormone. It is released by hypothalamus, plus it is released by your delta cells of pancreas. What is this somatostatin, Lakshmi? What is this somatostatin? It is the growth hormone inhibitory hormone. It is the growth hormone inhibitory hormone. It is also released by the hypothalamus. Here also it is released, so it will inhibit the pancreatic secretions. So this pancreatic polypeptide cell, it acts on exocrine part and stops their secretion, and stop its secretion. Right, and stop its secretion. Is that clear? So you know that glucagon and insulin, they are also antagonist, है ना? Do you remember that? They are also antagonist. Chalo, come to this part. So there are about 1 to 2 million islets of Langerhans in a normal human pancreas. Okay, bache. So they alpha cells and beta cells are the main cell. Alpha cells will release glucagon, beta cells will secrete insulin. Okay, right? So glucagon is also a peptide hormone. It plays role in maintaining normal blood glucose level. So it will act on liver cells, जैसे your hepatocytes. So when in the blood, bache, blood glucose level is low, then glucagon will be released, it will increase that level. That's why it is hyperglycemic. So MCQ can come, which one is hypercalcemic, which one is hyperglycemic, which one is hypercalcemic, which one is hyperglycemic. This is what you need to know. Okay? So in addition, these hormones, they stimulate the process of gluconeogenesis, right? So they will increase the level of glucose in the blood. Okay? Okay, so glucagon will reduce the cellular uptake of glucose by adipocytes, by hepatocytes. So it is a hyperglycemic hormone because it will increase the level of glucose in the blood. Okay, and insulin is a peptide hormone. It is also help in regulation of glucose homeostasis. So they will act on hepatocyte and adipocyte. So when in the blood glucose level is high, insulin will be released, it will act on liver cells, it will act on fat cells, and in response to that, what's going to happen? The glucose will get converted to glycogen. So can I say that there will be the glycogenesis? Can I say that there will be the glycogenesis? Yes, people. Can I say that there will be the glycogenesis? Okay, so that's how it works. And you know that if prolonged hyperglycemic conditions are there, diabetes mellitus is there. So diabetes mellitus means glycosuria and ketonuria. This is glycosuria and ketonuria. Baki bache dekho, testis and ovary, it is done and dusted in the human reproduction part. You know that seminiferous tubules are there, right? In between you are having Leydig cell. Leydig cell in response to LH will release your testosterone, that is important for spermatogenesis, and here you know that FSH will act on Sertoli cell, that will help in nourishment. That's all. Okay, so androgens will be released, that will help in the maturation and the function of male accessory sex organ, jaise epididymis, vas deferens, right? So growth of facial axillary hair, aggressiveness, all that things will be there. So in the case of males, low pitch is there, females ke case mein high pitch is there. In the case of males, low pitch. In the case of female, high pitch is there. Okay, so androgens, they will also act on the central nervous system and they influence the male sexual behavior, that is libido. Libido माने sex drive. ठीक है, libido माने sex drive. So obviously these hormones, they play the role here. Okay, so they are having anabolic effect माने build up anabolic effects on the protein and carbohydrate metabolism. So ovaries, you know that FSH will act on the ovarian follicles. In response to that, they will release estrogen, which will further act on the breast, which will help in the enlargement of breast. It will also act on the uterus endometrial lining and will help in its proliferation. And you know that when it comes to the LH is responsible for what? Ovulation. When LH surge is there, and remaining Graafian follicle will turn into corpus luteum and will release progesterone, which will maintain pregnancy. So I really don't think that we need to discuss it in detail. Okay, so when it comes to the hormones of heart, बच्चे heart के case में remember ANF, atrial natriuretic factor. So it is like a check on RAS mechanism. So this hormone is a vasodilator. It dilates blood vessel, it decreases blood pressure, right bache, it maintains the heart rate. So see arterial wall of heart, they release peptide hormone atrial natriuretic factor, which decreases BP, right? So when BP is high, it will be released because it will cause dilation of blood. Now your kidneys, they release erythropoietin. Erythro means RBCs, poietin means formation. So this hormone is released by juxtaglomerular cells. This is important. This will help in erythropoiesis, that is formation of RBC. So when it comes to the GI tract, gastrin released by the G cells of stomach. Secretin is the first hormone discovered. It is released by small intestine. Then cholecystokinin, pancreazymin, and gastric inhibitor is there. So gastrin, you know that it will act on the stomach glands, stimulate secretion of HCl and pepsinogen. Though bache, this part is deleted, but you never know, because see, digestion and absorption is deleted, but still it is the part of your syllabus, so you should revise it. So secretin acts on exocrine part of pancreas and stimulates secretion of water and bicarbonate. CCK will act on pancreas and gallbladder. So that's something you need to revise. That's it, that's all. So the next chapter that we are going to start is neural control and coordination. Okay, it is the next chapter that we are going to start is neural control and coordination. And honestly tell me, please tell me, are you enjoying this human physiology sessions? Are you enjoying this human physiology session? Because it is not something that we are skipping, we are discussing everything in detail, right? I want you people to understand the concept. I'm not here for any formality. I literally want you people to understand this concept. So after this, we will start excretory production and its elimination, because there are three main topics there: countercurrent mechanism, regulation of kidney function, and urine formation, of course, right? So we really need to revise that. And after neural control and coordination, excretion, then let's go for the body fluid and breathing. Guys, 20 minutes ka break, yaar nahi yaar, that will be too much. Bache, peptide hormones, peptides, how will you form peptides? Jaise proteins are made up of many polypeptides, many chains will join. So peptide which are made by the formation of amino acids. Chalo theek hai, 7:05 is there, so break is there till 7:15. That's what I can do. So this is the break, washroom break. ठीक है? So break time में it is till 7:15. So it is the washroom break. Next break will be there after this chapter. Okay, that will be for half an hour. हाँ जी, what's up people? We have completed 4 chapters, just 4 chapters. नहीं यार, seriously, shame on me. But बच्चे honestly speaking ना, if we have to provide the quality content, we cannot compromise. मतलब I know there are fast revision sessions also, I can do that, but if I know that my students, they know something, right? But if I have to teach something where I actually want those students to understand the topic, those who are reading it for the second time maybe, so it is a time taking process. It is a time taking process. Okay, literally it is a time taking process. I cannot say that ki haan ji theek hai, okay, this is the chapter, this is the line, these are the lines that you need to remember. But yeah, we are running out of time as well, but I still believe that see, after the zoology marathon still you have time, because we are planning to complete your syllabus before 20 or 21st. Okay, so of course in that time period, what will be the best thing ki whatever content I'm delivering you, if it will be sufficient, then after in that remaining 10 to 15 days you can revise that. Yeah, I'll share my notes on Telegram. So you quickly share the session link with your friends, tell them that I am teaching again and this is the chapter that we are going to start. So we have to be quick. Okay, we have to be quick. So neural control and coordination. So tell me which topic, right? Thank you so much, Yoga. Which topic you find difficult? Guys, Hanya, I can go for 15 hours. And today though I can go for 20 hours as well, because I'm really looking forward to finish it completely. Means there should be something on YouTube where, you know, everyone can like it. If you will attend this lecture, you can complete the entire zoology portion. But the point is, as per my knowledge, if I will teach above 12 hours, by tomorrow you won't get this video. YouTube will remove it. It is as per my knowledge, I'm not sure of it. Okay, so YouTube will remove it because like it allows only a specific duration ka video. That's the problem. That's the problem. See, I'm telling you my plan again. If you are confused, let me tell you, 3 sessions are going to be there. 1, there will be a diagram based session. Okay, and in that diagram based session, of course, because we know that paper will come from the diagrams, and sometimes there are some questions that are from diagrams. So that will be a quick revision session for you. I will take the class where I will be teaching you the questions from 2019 to 2023, right? Last 5 years ke PYQs, they are going to be the short sessions. I'll be taking a session where I will pick up the frog and cockroach, but yeah, again, that session will be for 3 to 4 hours. Either I will take it separately, 2 hours for frog, 2 hours or cockroach, right? So this is something that I have to complete before 25th. Now one more session was there, again and again I'm forgetting that. Okay. So here even I will include NCERT based questions. So diagram based session is a priority. Yeah. So top 10 concepts I will pick up. Right, in a very brief manner we will discuss it. So one more thing is there, I forgot, disorders. So if I will pick up topics like this now, we will complete many things. And if I will pick last 5 years ke question, of course, if let's say if we will leave something today, that we can cover there. ठीक है, so that is my plan. Botany के तो होंगे ही होंगे. Botany is for sure. 2 marathons of botany. Okay, botany class 12th, botany class 11th, and in the class 11th botany I am going to prioritize the chapters anatomy, I am going to prioritize the chapter plant physiology first, then plant kingdom first, right? Because that is something that you can understand by your own, जैसे biological classification, living world, but that is something which I need to discuss. Morpho bhi, it's your revision. So if I will take the botany ka marathon, I'm strictly telling you, I'll be teaching you plant physiology, that too in detail. ठीक है? That too in detail. Thank you, Hanya. Okay, so I'm telling you this thing, if there will be the botany marathon, I will not touch morphology. If I will get time, I will touch. I will quickly revise anatomy because that is very easy to revise quickly and 2 to 3 questions can come. I'll pick up mainly plant physiology and the plant kingdom in detail. Otherwise, I'm not going to touch biological. This is something that you can read by your own. But when it comes to plant physiology, you have problem in many things. Okay, so if there will be the botany ka class 11th marathon, please make up your mind that madam is going to finish plant physiology mainly. So that is a myth. That is a myth. ठीक है कि she will be teaching us complete botany. नहीं भाई, if it is class 11th का botany marathon, I am going to finish plant physiology first, then I will prioritize other things, because directly from plant physiology, if I will get, let's say 15 questions, so 15 question means 60 marks. ठीक है, so 15 question means 60 marks. So this is my priority. This is my priority. Then we will go for plant kingdom, 8 marks. ठीक है, then I will go for anatomy where I can consider, sorry, not 8 marks, 12 marks. Here I can consider anatomy में 8 marks. Then cell chapter, चलो again 16 marks. Here also वैसे plant kingdom में भी we can consider 16 marks. Okay, so that will be my target. That will be my target. ठीक है, so feeling energetic, getting that energy. So can we come to the topic directly? Can we come to the topic directly? चलो ठीक है. So what's the topic? Ambika, we are running out of time. We need to speed up. Neural control and coordination. So obviously you are talking about the nervous system here. ठीक है, neural control and coordination. Definitely you are going to talk about the nervous system here. The main and the conceptual question here is your ionic theory. The main and the conceptual question here is your ionic theory, right? Which one is that question? It is the ionic theory. Acha guys, I'm not saying that I'm going to do that, but I'm just asking one question. If any chance, by any chance, if let's say we will miss few topics today, if I will put the short videos, are you willing to see that? Do you want that? Just let's say I'll be putting the short videos on the short video on your information, your respiration, your photosynthesis. Are you willing to do that? Are you sure? Then only I'll put efforts. Otherwise I don't want to. Otherwise I don't want to. So obviously here we need to something which is known as neuron. Okay, here we need to understand something which is known as neuron. So firstly, let me explain you the neuron structure, then we will focus on NCERT, and my main target here is the ionic theory, because in the ionic theory I have seen many students, they struggle a lot. Okay, many students, they struggle a lot, so that is my main priority. Okay, so what are we discussing? We are going to talk about the neuron. What are we going to discuss, people? We are going to talk about the neuron. So let's do that. Okay, so you know that here this part is your cyton or the cell body. Okay, how are we going to discuss it? You are talking about the neuron. So what are neurons? They are the structural and functional unit of nervous system. What are they? They are structural and functional unit of nervous system, yes or no? What are neurons? They are the structural and functional unit of neural system. Yes or no? Is it so? Is it so? So basically what is there? This part is what? This is the cell body or the cyton. It is the cell body and the cyton, and the processes will arise from it. So when you talk about the neuron, that's what you need to understand, that firstly we will be talking about the cyton. Okay, we will be talking about the cyton. Okay, bache? Right. So cyton, also known as perikaryon, also known as soma, also known as cell body. Isn't it? It is having so many names. Isn't it? It is having so many names. So it will have what? It is having the neuroplasm. It is going to have the neuroplasm, yes or no? What will it contain? Yes, people, the one question can come from this part. It will be having the neuroplasm. It will be having mitochondria. It will be having Golgi bodies. Okay, it will be having mitochondria. It will be having Golgi bodies. But the main thing that we need to remember is Nissls granule. ठीक है. So in the cell body you will see Nissel's granule, and even they are in dendrites also. Even they are in dendrites also. So Nissel granules, they are what? Right. This question can come in the paper. This question can come in the paper. So what are Nissel granules? What are Nissel granules? Anyone in the class? So you will see Nissel granules and neurofibrils here. Just we were talking about microfibrils, you will be talking about neurofibrils. So what are Nissel's granules? Yes, anyone in the class? What are Nissel's granules? Anyone in the class? Yes. They are the masses of ribosome and rough endoplasmic reticulum. They are the masses of ribosome and rough endoplasmic reticulum, right? So obviously they are playing role in the protein synthesis. So let me mention you here, bache, Nissel granules, they are absent in axon. When you look at this neuron, this is the cell body, it is having the processes. Okay, so processes of neuron. Okay, when you talk about the processes of neuron, you call them as neurite. You will use the word neurite for it. Which word are you going to use for it? Which word are you going to use for it? You will be using the word neurite. Which word are you going to use for it? Yes, people. Which word are you going to use for it? You will use the word neurite. So here you are having two processes, that is dendrites and the axon. Okay? So dendrites, when you look at the dendrites, all that branching and all. Okay? All that branching and all. What are they? All that branching and all, they are the dendrites. ठीक है बच्चे, so they are also having the Nissel granules. Now when you look at this cylindrical part, ठीक है, this is your axon. This is your axon. So when you look at this neuron, can you see that it is like this? It is like this. So this portion here, okay, it is the axon hillock. You call it as axon hillock. It is highly sensitive. Why is it highly sensitive? Why is it highly sensitive? Because it is having many ionic channels here. It is having many ionic channels here. Are you getting my point? It is having many ionic channels here. Now here again I am repeating, Nissel granules are absent. This is important. Okay, this is important. Moreover, bache, here in this, see, if it is covered with this covering, it is known as myelin sheath. It is known as myelin sheath. So such axon is known as, right, myelinated axon. Such axon is known as myelinated axon or medullated axon, yeah, medullary sheath. This is the medullary sheath or the myelin sheath. If there is no sheath, it will be non-myelinated axon, right? It will be non-myelinated axon, yeah, non-medullary sheath, non-myelinated axon or non-medullary sheath. Are you getting my point? Non-myelinated axon or non-medullary sheath. Are you getting my point? This is what it contains. So these are the axon terminals, yeah, axon endings or terminal arborization. Here we will see the synaptic vesicle. Okay. Now when you look at this part, we will differentiate also. As of now, I'm just telling you the basics. Let's say this is the axon. Okay, so here I am drawing what? It is the myelin sheath. Here these are the cells. These are the Schwann cells. These are the Schwann cells. Okay, bache. And this sheath is the myelin sheath. Again, I am telling you myelin sheath. So if you will see at places you do not have the myelin sheath. Can you see that at places you do not have the myelin sheath? Can you see that, people? That at places you do not have the myelin sheath. So this region is NOR, means node of Renwier. Are you able to recollect it? Because I believe that in the lower classes also this is what we have discussed. So node of Renwier is the place where there is no myelin sheath. Yeah, but still a neurilemma covering is still covering it. Okay? A neurilemma covering is still covering it. So this is the basics of neuron. This is the basics of neuron. So now we have to understand it in a proper way. But if you have any doubt up to this part, please feel free to ask. Right? Please feel free to ask, because then we will be discussing it from the NCERT in detail. Hanji, we are going to discuss it from NCERT in detail. Done it? Any doubt? Sure? Sure? I am directly starting with the nervous system, that is neuron. Then we will discuss it further, okay? Then we will discuss it further, okay? Sure? right? So the function of organ or organ system in our body is must be coordinated to maintain the homeostasis. So what is homeostasis? Homeostasis means the constant internal condition. Homeostasis means the constant internal condition. जैसे for the homeostasis the thermoregulation and osmoregulation should be there, means the temperature should be maintained properly and the salt and water balance should be there when you are talking about what? When you are talking about the homeostasis. When you are talking about what? When you are talking about the homeostasis. So coordination is a process through which two or more organs they interact and complement the functions of one another. ठीक है बच्चे? ठीक है बच्चे, this is what we know. Right. So let's forget about this introduction, come to the main part. So when you people talk about the neural system, what do you think what will be there? Obviously neurons are there, the structural and functional unit, but it is not just the neurons, you will also talk about the neuroglial cells. ठीक है, we are not just going to say that neurons are going to make the neural tissue, neuroglial cells are also there. ठीक है बच्चे, न्यूरल ग्लियल सेल्स आर ऑल्सो देयर. सो न्यूरल सिस्टम में वी आर हैविंग हाईली स्पेशलाइज्ड न्यूरॉन्स एंड you know that it is the longest cell of human body. What's that? It is the longest cell of human body. Am I right? So it is something which can detect, which can receive, which can transmit different kinds of stimuli. It will detect it, it will receive it, and it can transmit it. ठीक है, it can transmit it. So neural organization is very simple in lower invertebrates. So can you tell me the phylum name where for the first time we will see the neural regulation? If you remember, phylum Cnidaria, also known as Coelenterata. You remember we talk about the hydra. In the hydra there is a diffused network of nerve cells. There is a diffused network of nerve cells, if you remember. What will be there? There will be the diffused network of nerve cells, if you remember. So for the very first time we will see it there. Okay, for the very first time, where are you going to see this? You will see it there. ठीक है? Okay, so hydra में you know that it is composed of network of neurons. So neural system is better organized in insect. They are having a brain, बच्चे, right? They are having the ganglia. So I will explain you the meaning of ganglia. Ganglia in general means it is composed of cell body. So many cell bodies together they form the ganglia and the neural tissue. ठीक है बच्चे? And vertebrate में it is well developed. So how they have divided it? They have divided it in a very simple way. So when you talk about the neural system, you will read CNS, that is central neural system, and you will talk about the PNS. PNS means peripheral nervous system, या peripheral neural system. So in the case of CNS, what will be there? There will be the brain and spinal cord. So it is the brain and the spinal cord. And in the case of PNS, we will be having a somatic neural system, and yes बच्चे, there will be autonomic neural system, your autonomic nervous system. Yes or no? There will be autonomic neural system or there will be autonomic nervous system. Am I right? That is what we are going to have. So autonomic and somatic nervous system will be there. So when you talk about this autonomic nervous system, you will be having the sympathetic nervous system, you will be having the parasympathetic nervous system. Sympathetic and parasympathetic nervous system will be there, right? Sympathetic and parasympathetic nervous system will be there, right बच्चे? Now when you talk about this somatic neural system, ठीक है, जैसे देखो central part, brain and the spinal cord. So obviously we know that some nerve fibers will also arise from it. So this peripheral part will come under PNS. So here you discuss about two things. You talk about the afferent nerve fibers, right? You talk about the afferent nerve fibers and you talk about the efferent nerve fibers. What are we going to discuss here? Afferent nerve fibers and efferent nerve fibers, that is what we are going to discuss. So A for A, you know na, ASMI is the trick. So afferent S mane sensory, they are the sensory neurons. Efferent mane motor neurons, and we also see mixed kind of neurons. So sensory neurons will carry the information to the brain and spinal cords. Motor neurons will bring back the processed information. They will bring back the processed information. That's the point. Clear bache? They will bring back the processed information. That's the point. Understood? Understood? Sure? Sure, sure, sure. Are you sure? Right, right. So when it comes to the somatic neural system, so you will see it is basically related to the skeletal system, right? It is basically related to the skeletal system. So obviously it is voluntary. It is voluntary. है ना? It is voluntary. We can control it with our own will. So it is voluntary, right, बच्चे? It conduct impulse from CNS to skeletal system, right, to skeletal system. Now when it is autonomic, so autonomic hai, so it means it will be involuntary. You cannot control it with your own will. So we have parasympathetic and sympathetic. Sympathetic will deal with the situation when there is a fear, right, and here it will deal with the rest and digest when you are in a relaxed state, right, when you are in a relaxed state. Okay, so it will deal with the rest and digest, it will deal with the fear wala situation. So this part clear, this part clear, right? So brain, spinal cord, somatic neural system, sympathetic, parasympathetic, that is what we have. Understood? That is what we have. So you can come to this part. Okay, so efferent, so see this somatic neural system and autonomic is also there. So even your visceral nervous system, yeah, visceral neural system, viscera, it is also considered as the part of PNS, bache, right? So it is having its nerves, it is having its fiber, ganglia and networks by which impulse will travel from CNS to the viscera. Viscera mane body organs. Okay? So somewhere it is also the part of PNS. So PNS ke case mein you can also draw it like this, right? That one is your visceral nervous system. Is that clear? Visceral nervous system. Okay? Okay? Any doubt? Any doubt? Sure? Sure, sure, sure. Are you sure? So next part is the neuron. So we have already discussed that. So the main point here is of Nissls granule. So they are absent in axon. So in NCERT they didn't mention it clearly. But yes, the question can come from this part. Okay, the question can come from this part. Right, bache. So cell body is having cytoplasm with typical cell organelles, and there are granular bodies which are Nissls granule. So short fibers which branch repeatedly, they are project out of the cell body. So that is what we have already covered, right? That is what we have already covered, right, bache? So you know that when it is the axon terminal, right? So this is, here you will see some synaptic vesicle which are having the neurotransmitters, okay, which are having the neurotransmitters. Up to this part all clear? Yes, bache, which are having the neurotransmitter. Up to this part all clear? Sure? Sure, sure, sure. Are you sure? Are you sure? Okay, so next is the types of neuron. Next is the types of neuron. So when you talk about the types of neuron, can you tell me how many types of neurons do we have? Can you just tell me how many types of neurons do we have? Yes, people, how many types of neurons do we have? So of course we have to start the story with the unipolar neuron. The diagram that I drawn at first time, it is the multipolar, so it is unipolar. So uni means single, so only one single process will arise from it. Okay, only one single process will arise from it. So what type of neuron it is? It is the unipolar. What type of neuron it is? It is the unipolar. Is that clear? So single process will arise from it, and that will be the cyton. Okay, single process will arise from it, that will be the cyton, right? That will be the cyton. Do I need to mention it? So example is important. It is in the nervous system of embryo. It will be in the nervous system of embryo. Can you? Right, it will be in the nervous system of embryo. Is that clear? Is that clear? Then comes the bipolar. So can you give me the example of bipolar? Yes, can you give me the example of bipolar? So the word is bipolar, so the two processes will arise. Just let's say one dendrite, one axon, like this. So now I is deleted from your syllabus. I is deleted from your syllabus. So if you remember, it is in the retina. right? It is in the retina of the eye. You know, na, retina of the eye is having the layers, photoreceptor layers, bipolar cells, the ganglia cells are also there. So right, so when you move from outside to inside, it will be photoreceptor cells, rod and cones, bipolar cells, and then the ganglia cells is there. So retina of eye is the example. And even in the olfactory epithelium, ठीक है बच्चे, even in the right, so even in the, yes bache, even in the olfactory epithelium we are going to get it, okay, even in the olfactory epithelium we are going to get it. And let me tell you that your rod and cone cells, right, they are also the modified bipolar cells. Rod and cone cells, they are modified bipolar cells, okay, they are modified bipolar cells, okay, doubt people? Yes, any doubt? So unipolar, bipolar, then comes the multipolar, so multipolar mein the diagram we have already studied. So you know that from the cell body many processes will arise, ठीक है? So there will be many dendrites, right, there will be many dendrites, and then there will be the cell body. Is that clear? So you will see the most neurons in vertebrates, they are like this, okay, most neurons in vertebrates they are like this, most neurons in vertebrates they are like this. So example mein we are having cerebral cortex, right, so brain mein that cortex part is majorly the example, okay, right. So we have one more category, that is pseudo unipolar. So pseudo mane false, so pseudo unipolar, जैसे let's say this is the cell body, so it will appear that only one process is arising, but actually that one process will further divide and it will form many processes like this, pseudo unipolar. I know the diagram is worst, the diagram is pathetic, but I hope it is clearing your doubt. So one axon but a small process developed from axon, right, that will act as a ganglia. So this is the pseudo unipolar and it will be in the dorsal root ganglia of spinal cord, right, it will be in the dorsal root ganglia of spinal cord. Example is important, dorsal root ganglia of spinal cord, example is important because this is also given in our book NCERT. Clear bache? So non-polar type of neurons where there is no proper dendron and axon, they are also there, like in the hydra, theek hai ji, like in the hydra. Is that clear? Like in the hydra. Is that clear? So let me mention that non-polar neurons don't have proper dendrites or axon, right, they don't have proper dendrites or axon. What is it? Non-polar neurons. So they do not have proper dendrites or axon bache, and they are in the hydra, right, they are in the hydra. Is that clear? They are in the hydra. Is that clear? So look at this, this part clear, the unipolar and all clear. So even I told you about the myelinated and non-myelinated, ठीक है? So let me explain you this myelin or non-myelin wala part. So up to this part, if there is any doubt, do let me know. Because if it is clear to you, then bache, the nerve impulse conduction will be easy, ठीक है? The nerve impulse conduction will be easy, okay. Sure. Are you tired? Tell me honestly, are you people tired? Because I am a bit, okay, I am a bit. So bache, I told you about the neural system or the nervous system, and I mentioned this thing to you that there will be the neurons. So other than that, you are also having the neuroglial cells. What do you have there? You are also having the neuroglial cells. So when you people talk about the neuroglial cells, obviously they are also forming the major part, है ना बच्चे प्रेरणा, they are also forming the major part of your nervous system. Do you know that they are basically the supporting cells which form a, supporting cells which form a major component, supporting cells which form a, basically the protection of neurons, they will help in the growth, right? They will help in the packing substances around the neuron, so they will help in the kind of protection. ठीक है जी, they will help in kind of protection. So supporting cells which form a packaging around neuron. ठीक है, so these are the neuroglial cells. So can you tell me the types of neuroglial cells? Can you tell me the types of neuroglial cells? Yes, be quick. Can you just tell me the type of neuroglial cells? And they comprises about 50% of the brain cell. They forms about 50% of brain cells. Okay, so can you just tell me the types here quick? After this, let's start the generation and conduction of the nerve impulse. Be quick. After that, let's start the generation and conduction of the nerve impulse. Be quick. So tell me the types here. Tell me the types here. Quick. Tell me the types here. Anyone? Yes, anyone? न्यूरोग्लियल में देखो, न्यूरोग्लियल में एनीवन? देखो बच्चे, वन इज एस्ट्रोसाइट्स, अनदर इज ओलिगोडेंड्रोसाइट्स and one is microglial cells. So microglial cells, I think in this lecture itself I have repeated so many times. But other than that, you are also having ependymal cells. You are also having ependymal cells in your nervous system. You are also having your neurosecretory cells, right? You are also having your neurosecretory cells. Clear बच्चे? You are having your neurosecretory cells here. Let me explain this part. So when you talk about the astrocytes, you know, astrocyte, aster word is associated with star. So the main role of this cell is to form blood brain barrier. The main role of this cell is to form blood brain barrier. So this cell, it is just like a star. So if I will relate it with the star, so you know that star is having many processes. So yes, it is also a large cell having numerous processes. Are you getting my point? It is also a large cell having numerous processes. Clear बच्चे? So it is going to form blood brain barrier. This is something important. Can you just explain? Yes बच्चे, Dr. Vignesh, Mango, right, Sweetie, can you just explain that what is the role of blood brain barrier? Yes बच्चे, what is the role of these blood brain barrier? Anyone? Obviously बच्चे, blood brain barrier, these cells, they are arranged in such a way, they are having the junctions, they will not allow any neurotoxin, any harmful compound that can damage our nervous system, it will not allow here in the nervous system. Done बच्चे? So oligodendrocytes, they will form myelin sheath. Okay, they will form myelin sheath in central nervous system. They will form it and preserve it. Microglial cells, I told you, they are the squamous. They are the squamous cells of phagocytotic cell of nervous system, right? They are the squamous, right? Or you can say that phagocytotic cells of nervous system. Clear? Now when you talk about a brain, ठीक है, brain में there will be the cavity. That cavity is known as बच्चे ventricle. Brain is also having the cavity, we will call it as ventricle. So it is having the cells, right? That cells are ependymal. If you remember structural organization, ependymal cells, they are also the ciliated epithelium. It is also the ciliated epithelium. है ना? So this ependymal cells which will line up the ventricles, they are also present and they also secrete cerebrospinal fluid. Do you know that? It also secretes cerebrospinal fluid. And neurosecretory cells, you know that, what are they? They will release the hormones, if you remember. Okay? So these are the nerve cells, right बच्चे? These are the cells of the hypothalamus, right? These are the cells of the hypothalamus and they will release hormone. What will they release? They will release hormone. Is that clear? They will release hormone. Is that clear? Yes people, is that clear? So this is what we have. Now the question is, ma'am, if oligodendrites are going to form the myelin sheath, then what is the role of Schwann cells? We will come to this. First of all, I will tell you what is this Myelinogenesis. Myelinogenesis means formation of myelin, of course. Myelinogenesis means formation of myelin, of course. Yes, it is the formation of myelin, of course. So you know that we are having the peripheral nervous system and we are also having the central nervous system. We are having the peripheral nervous system. We are also having the central nervous system. Yes or no? So first of all, myelin, what is it? Myelin, first of all we should know what is myelin, which is also known as medullary layer. So bache, it is an insulatory layer. It is acting like an insulation. It will not allow the ions to go out. It is made up of fat. It is made up of protein. It is made up of water. What is this myelin, people? What is this myelin? Everyone in the class, tell me. What is this myelin? It is having lipid, proteins and water. ठीक है बच्चे? ठीक है बच्चे? It is having lipid, proteins, it is having lipid, proteins and water. This is what myelin is. This is what myelin is, and it is highly electrical resistant. Okay, it is highly electrical resistant. It will not allow, right people, it will not allow the ions to go here and there. So this part clear, this is the myelin. Okay, this part clear, this is the myelin. Okay. So this myelin, just let's say this axon. Okay, this is the axon. It is a myelinated axon. So this myelin is basically the made up of phospholipid, yes, sphingomyelin. Okay, these are two compounds which will form it. Phospholipid, yes, sphingomyelin. Can you see that? Phospholipid, yes, sphingomyelin. Yes or no? Yes or no? Yes or no? So this myelin, na, bache, this myelin is covered with one more layer. Just let's say this is the myelin. It is covered with the layer. You call it as neurolemma. You call it as neurolemma. Lemma mane covering. What is the meaning of lemma? Lemma mane covering. So neurolemma is nothing but it is the sheath of, it is the covering of Schwann cells. So the covering of Schwann cells is known as neurolemma. So Schwann cells are the cells which are present in PNS. So in peripheral nervous system, Schwann cells will form myelin. In the central nervous system, oligodendrocytes will form myelin. Is that clear? Right? Right? So this part clear? Yes people, this part clear? This part clear? Yes or no? So is it clear? Do I need to explain it? Do I need to explain it? So basically Schwann cells will form myelin sheath here in PNS, and here in CNS it is the oligodendrocytes. It is the oligodendrocyte that will form myelin. That's what you need to keep in your mind. Understood? That is what you need to keep in your mind. Understood? So this is the CNS. Done, bache? Then the CNS, it is the oligodendrocyte, which is a type of neuroglial cells. It will form it. Understood? Understood? Yes. So myelinated nerve fibers are there in the spinal and the cranial nerves, and unmyelinated are there in your autonomic and somatic neural system. So now come to the NCERT, because I need to highlight something. Okay, I need to highlight something. ठीक है? So ये देखो, जैसे the myelinated nerve fibers, they are covered with the Schwann cells which will form myelin sheath. So you can write down, it is in PNS. It is in PNS, and that gaps are nodes of Renvier. So myelinated nerve fibers are found in cranial and spinal nerves. Again, the important MCQ. Please highlight. Right? It is found in, right, bache? This myelinated nerve fibers are found in spinal and cranial nerves. They are found in spinal and cranial nerves. And unmyelinated nerve, acha, theek hai. Now I'll ask you one question. Myelinated, right, in the PNS, ma'am, where is oligodendrocyte form? In CNS, bache. Acha, answer my question. You know about the myelin sheath. You know about myelin sheath is there in the neurons of peripheral nervous system and even in the neurons of central nervous system. So you tell me one question. If a neuron is unmyelinated, if it is not having the myelin sheath, so what do you think? Do they have the Schwann cells? Answer this to me. Do they have? Yes, they do have. Even if a neuron is unmyelinated, that doesn't mean that Schwann cells will not be there. Schwann cells will be there, but they will not form the myelin around it. That's something very important, right? That is something very important. So unmyelinated nerve fibers is enclosed by a Schwann cell that doesn't form a myelin sheath. So I still remember in my class, right, one of the students हाँ, some students they made the mistake there. ठीक है, so that's what you need to highlight. So unmyelinated nerve fiber is enclosed by a Schwann cells that is not going to form a myelin sheath around the axon, and it is in the autonomous and somatic neural system. ठीक है बच्चे, ठीक है, sure. So now let's discuss about the generation and conduction of nerve impulse. ठीक है, generation and conduction of nerve impulse. Let's discuss about this. So how many of you understand this topic? Because it is a conceptual one. This topic is conceptual one, so you tell me. Do you understand that? Do you understand that? How many of you find it difficult? Be quick बच्चे. See, if you understand it this, if you don't understand it this. ठीक है, if you understand this, if you don't understand it this. Accordingly, I'll increase my speed. Quick. Okay, I'll see thumbs up and thumbs down. So many thumbs up are there. So if we have thumbs up, matlab I can go quickly, है ना? I can go quickly. Many thumbs up are there. चलो. So collection of cyton. There are two terms. One is nuclei and another is ganglia. ठीक है? One is nuclei and another is ganglia. जैसे I just told you that ganglia means so many cell bodies together. This is what I just said when I explained ganglia. So it is outside the CNS. So this ganglia is outside the CNS term used for PNS, right? So basically nuclei is also the collection of cyton, but this term is used in CNS. I'll give you one example. Just in hypothalamus, when I was teaching you about the hypothalamus, we use the word nuclei, right? That the neurons together here, they form the nuclei. That's what we have discussed. So nuclei is nothing. Nuclei is the collection of cell body. So in PNS, this is the term we use for it, and so in the CNS, this is the term that we use for it. So देखो ना, nuclei. So NC या CN, cartoon network. Remember it like this, cartoon network. C for CNS, N for nuclei. So ganglia is the term which is used for PNS. Ganglia is the term which is used for PNS, that is peripheral nervous system. Then we have tract and nerve. Then we have tract and nerve. Then we have tract and nerve. Nerve word is used for PNS. Track word is used for CNS. ठीक है? So देखो, cartoon network NT, CNT. That's how you can remember. Cartoon network, CNT. C stands for cyton. N stands for nuclei. C stands for central nervous system. N stands for nuclei. T stands for track. T stands for track. ठीक है? T stands for track. ठीक है बच्चे? So it means in central nervous system, right, the cyton is present, the collection of cyton is nuclei and the collection of nerve fiber is tract. That's what you need to remember, right? जैसे you will at places you will read the word nerve, right? At places you will read the word tract. So you should know what is the difference. You should know what is the difference. And yes, even one more thing I would like to ask, what is the difference in between a neuron, nerve fiber and nerve? Do you know that? What is the difference in between a neuron, nerve fiber and nerve? जैसे देखो, when you have a single neuron, when you have group of neuron, you will say nerve fibers. Obviously they are having a sheath covering. When so many nerve fibers are together, they will form nerve. ठीक है? They will form nerve. You should know these differences. You should know these differences. So many neurons together will form nerve fiber, many nerve fiber together will form nerves. So you should know about the layers and sheath. Bache, the basics should be strong. But as of now, that's what you need to master. So any doubt? Any doubt? Any doubt? So next is the generation and conduction of the nerve impulse, and that is best explained with the help of ionic theory. And bache, this is in the case of non-myelinated nerve fibers, right? This is in the case of non-myelinated nerve fiber. ठीक है? It is in the case of non myelinated nerve fiber. What is it? What is it? It is the ionic theory given by Hawkins and Huxley. Hawkins and Huxley. ठीक है? ठीक है? So neurons are excitable cells. They will receive the stimulus, they will get excited, obviously they will conduct it, right? They will conduct it. So now we have to discuss few terms here. जैसे what is action potential, what is resting potential, right? All that things, okay? All that things. So now let's say this is the axon. What's that? This is the axon. You know that this is the exoplasm. Here also we will be having some ions. Here also we will be having some ions. This is the surrounding. This is that extracellular matrix. Here also we will be having some ions. Am I right? Am I right? This is what we have. Now let's say when a neuron is not at all in an excited state, it is in a relaxed state. It is not at all in an excited state, it is in a relaxed state. So when it is a relaxed, right, it will be the resting potential. It will be the resting potential. ठीक है बच्चे? Right, it is near about minus 70 MV. It is near about minus 70 MV. It is indicating that neuron is taking a rest. ठीक है बच्चे? The neuron is taking a rest. So it is the potential difference across the membrane at rest. ठीक है? It is the potential difference across the membrane at rest. So which word are you going to use for it? That the neuron is polarized. Neuron is polarized. So now I will use the words one by one. You understand this. When you talk about the neurons, we say that they are the excitable cells. This is the term that we have used. We say that they are the excitable cell. This is the term that we have used. What are these neurons? They are the excitable cells. They will get excited. Actually their membrane is having that special property that these ions can come in and can go. Now when it is in the relaxed state, when there is no conduction, right, obviously ionic differences are there. Ionic differences are there, right? And that potential difference across membrane is deciding its resting potential. Now when you talk about the ions, in the surrounding, sodium is more, potassium is less. ठीक है? Potassium is less. Sodium is more, potassium is less. And within the neuron, within this axon, sodium is less and potassium is more. And moreover, within this axon, negative, right? Negatively charged proteins are also present. Negatively charged, right, negatively charged organic compounds are also there. This is how you can write it. And when you talk about the extracellular matrix, this is, they are having chloride ions, but these chloride ions play no role in nerve impulse conduction. First of all, tell me this part clear. First of all, tell me this part clear. We have to go one by one. We have to go one by one. This is the neuron that we have. Obviously this is the layer, outer covering, that right. So I am saying sodium is less within, potassium is more outside. Potassium is more, sodium is less. So basically when they are not conducting any nerve impulse, so amount is equal, right? You can say that, right? That even just say here negatively charged organic compounds are more. Here chloride is there, but chloride is playing no role in this ionic movement. So this is what we have. Okay, this is the first thing. Now when you talk about this membrane, I told you now it is having special property. It is having different types of ion channels. Just say here you will also talk about sodium leaky channels, sodium and potassium leaky channels, right? You will see different ion channels here. Just say sodium leaky channels, potassium leaky channels are also there, right? You will also see voltage gated ion channels. First category is of leaky channels, then it is the voltage gated ion channels. Voltage gated ion channels that will open and close when there is a change in the voltage. So this is the sodium, right? Voltage gated ion channel, sodium voltage gated ion channels are also there, potassium voltage gated ion channels are also there, and other than that we have sodium potassium ATPase pump. But when the word is pump, okay, when the word is pump, definitely pump, so it will be, movement will be against the concentration gradient, because it is the pump, okay, against the concentration gradient, and ATP is used. It is against the concentration gradient and ATP is used. Are you getting my point? It is against the concentration gradient and ATP is used. This part clear? This part clear? So I am talking about this membrane. I am saying when it is not conducting anything, it is having the resting potential. It is in the polarized state, that is minus 70 MB. So the potential difference across the membrane is minus 70 MB. Just say, let's say here more positives are outside, more positives are outside. So let's say here you have 100 positive, 30. So difference will be minus 70 like this, like this. See, okay, like this. It is the condition. I am saying there will be many ion channels here. Just say sodium leaky channels are there, potassium leaky channels are there, voltage gated ion channels are there, and sodium potassium ATPase pump is there. Why? These pumps are there to maintain this resting potential. These ion channels are here to maintain this resting potential. So tell me up to this part all clear? Tell me up to this part all clear? If everything is clear up to this part, then I will definitely speed up. Then I will definitely speed up. Hanji, quick. Sure. ठीक है. So now when you talk about this neuron, ठीक है, जैसे imagine there is a cell, ठीक है. So it is having the leaky channels. So here what do you see? In the case of neuron, it is having more leaky channels for potassium. It is having more leaky channels for potassium than sodium. Means leaky channels are for both, but it is more for potassium than for sodium. Now let me tell you what is going on. When you talk about these leaky channels, the transport is a passive transport. Let me explain how. Dekho, you all know this line that when a neuron is at resting state, right? When neuron is at resting state, when it is doing what? When it is at resting state, it is more permeable. Do you remember this? It is more permeable. It is more permeable for potassium ion. है ना? It is more permeable for potassium ion. Now let me tell you the story. What is happening? Imagine this is a neuron. I told you within potassium is more, comparatively sodium is less. You cannot say that they are absent. No, potassium is more, sodium is less. Surrounding में when you have extracellular matrix, here also sodium is more, potassium is less. Now what is happening? Because we have more leaky channels for potassium, so the movement is passive. It is along the concentration gradient. It is along the concentration gradient without the expenditure of energy. So obviously your potassium will move from inside to outside, right? When it is at rest, potassium moves from inside to outside, yes or no? Even leaky channels are there, even sodium will also enter within cell. I am not saying that sodium will not enter, but comparatively it is less. Imagine it like this, कि outside Lakshmi more sodium, inside less sodium, more potassium. So potassium is moving from inside to outside and it is moving at a faster rate. Even sodium is entering from outside to inside, but comparatively at a lesser rate, because we have less leaky channels. So tell me, this first condition clear? So basically more and more positive ions are moving in the surrounding, and moreover here you have negatively charged proteins which cannot cross even membrane. So that is why inside there is more negative, outside there is less negative. Right? Inside there is more negative, outside there is less negative. I am explaining you the condition of resting potential. Resting potential means membrane is polarized, means outside positive, inside negative. This is the meaning of polarized membrane. Clear? Outside positive, inside negative. Outside positive, inside negative, polarized. So how that will be maintained? The first point is the leaky channels. Now the second point, sodium potassium ATPase. Now for sodium potassium ATPase there is a trick. The trick is Nokia Kintu. The trick is Nokia Kintu. What's the trick? The trick is Nokia Kintu. What is the trick? The trick is Nokia Kintu. Everyone repeat it. Nokia Kintu. What is the meaning of Nokia? N stands for sodium, O stands for out, K stands for potassium, I stands for in, A stands for ATP. So when it is the sodium potassium ATPase pump, sodium will go out, potassium will come in. Sodium will go out and potassium will come in. And how many potassium will come in? Kintu means came in, potassium in, and how many potassium will in? Only 2. So 3 sodium will go out by using energy and 2 potassium will come in. This is the sodium potassium ATPase pump. So imagine already outside we have, right, already inside we have less sodium, but still we are throwing them out. 3 sodium will go out, 2 potassium will go in. So obviously there is a gap. So that is why in resting potential inside is negative, outside is positive. That is how it is maintained. Okay, that is how it is maintained. This part clear? Right now I am telling you about the conditions. Right now I am telling you about the conditions that why this resting potential is like this. Now let's say there comes a point where we have received the stimulus. You have received the stimulus at point A. So when you receive the stimulus, suddenly what will happen? There will be the influx of sodium. Sodium will start moving in. Sodium will start moving in. So when sodium movement will be in, the voltage change will be there, because more and more positive is coming in, voltage change will be there. So because of that, what will happen? Voltage gated ion channel, voltage gated sodium channel, they will open. Voltage gated sodium channel, they will open. Why? Because sodium is coming in. So how the polarity will change when there is the influx of sodium? This is MCQ. So when there is the influx of sodium, more and more your sodium will come in, right? So inside will become positive, and at that point outside will become positive. And how? Because even voltage gated sodium channels, they will open. So how that voltage gated sodium channels will open? When positive will come in, the voltage will change. Voltage will change. These ion channels will open, more and more sodium will start coming in and less sodium is going out. Okay, so inside at this point will become positive, outside will become negative. So this state is the depolarization. This state is the depolarization. So now the action potential is generated. So action potential mein what will be there? You can see it will be somewhere in between plus 20 MB to plus 50 MB. Okay? So here inside will become positive, outside will become negative. So that is how you can remember. Just when we take rest, we don't do anything. We feel negative from inside. We feel negative from inside and we feel positive, right? We feel negative from inside. So when we do something internally, we start feeling positive. So that's how I remember this. So when there is action potential, inside is positive, outside is negative. So it is just like the current ka circuit. So at this point, this is the scenario. At this point, this is the scenario. So you know that how will it move? It will move from plus to minus. It will move from plus to minus. It will also move from plus to minus, right? So that is how it will be. So now at B, what is going to happen? B will become positive inside, outside it will become negative. And this membrane bache, this membrane, it will immediately reverse polarity. No doubt when there is action potential, there is the influx of sodium, membrane polarity will change. Now it is more permeable for sodium. Voltage gated ion channels will also open. More and more sodium will be inside, but this membrane polarity will reverse in seconds. Just let's say you are having your best friend, right? For some time your best friend got distracted and your best friend is like, I am not your best friend. But immediately that best friend realized, no, I cannot leave my best friend, I have to come back. It's like that. So immediately this polarity will get reversed, right? So at this point again the negative and positive will occur. It will again polarized, right? So it will again polarized, right? Repolarization will occur at this point. So this is how the circuit of current will move. This is how the circuit of current will form. And bache, obviously when more and more sodium is coming in, outside there is negative, inside there is positive. So there comes a time when leaky channels, you know what will happen, even the potassium gated ion channels will open. So they will start throwing out more and more sodium outside, more and more potassium outside. So this is how it is. So I have just given you the idea of it. Now come back to NCERT, and trust me, now each and every line of NCERT will be clear to you. Okay, so they are saying different types of ion channels, are present on neural membrane. Do you understand this line? Do you understand this line, that yes, different types of ion channels are present on the neural membrane? Do you understand this line, that yes, different types of ion channels are present on the neural membrane? So these ion channels are selectively permeable to different ion. So when a neuron is not conducting, means when it is in the resting state, the axonal membrane is comparatively more permeable to potassium and nearly impermeable to sodium. This is something very important, please highlight, because questions are going to come from this part only. Right, questions are going to come from this part only, that axonal membrane is comparatively more permeable to the potassium ions and nearly impermeable to the sodium ions. Impermeable to the sodium ions. Please highlight this. Please highlight this. Please highlight this. So similarly, membrane is impermeable to negatively charged protein, means proteins from inside, they will not be able to move outside. So that is why exoplasm is having more negative charge, right? So it is having high concentration of sodium, it is having low concentration of high concentration of potassium, low concentration of sodium. So is that clear? Is that clear? So if you have any doubt up to this part, do let me know. Next, bache. Okay, so these ionic gradient across the resting membrane, they are also maintained by active transport. So here I have given you the trick of Nokia. So Nokia means sodium potassium ATPase pump will also maintain this resting potential. So as a result, outer surface will have positive charge and inner surface will have negative charge. Understood? Understood? I'll come back to this diagram. Understood? Right. So this is membrane is positive. So this is the resting potential. Now come to the next part. Again, okay, so when a stimulus is applied at site A, right, the membrane at the site A will become freely permeable to sodium. So the question is, when there is the stimulus, what will happen? What kind of change will be in the membrane polarity? It will be more permeable for the sodium. Understood? It will be more permeable for the sodium. Okay? So when there is a stimulus, there is influx, not the outflux. This is the influx of sodium, right? And immediately the reversal of polarity will be there. So outer side will be negative, inside will be positive. So polarity at site A will reverse and then again it will be depolarized. Okay, so when there is the action potential, depolarization will occur. When there is the action potential, depolarization will occur, right? So electrical difference across the membrane at site A is action potential. So this is the nerve impulse, bache, right? So this is, it will move from one place to another. Let me show you how. So see this. So wherever there is a stimulus, there is the influx of sodium. So it will result in inside positive, outside negative. So see the circuit of current. Now after this, B will become inside positive, outside negative, and this A will again reverse the polarity. Then you will call it as repolarization, means again polarization will occur. Again polarization will occur. Is that clear? Again polarization will occur. Is that clear? Is that clear? So at sites immediately ahead, the axon membrane has positive charge. So the current will move from A to B, then B to A. Right, bache. So sequence will be repeated along the length of the axon, and consequently impulse is conducted. So rise in the stimulus induced permeability of the sodium is extremely short-lived. I told you na, this permeability is very short-lived, right? So it is quickly followed by again by the same thing. So within fraction of seconds, K positive diffuse outside and restore the resting potential. So that is how it will again come back to the same level. That's the point. So remember, it's not like that, bache, ki if this nerve impulse is at this point and new nerve impulse can generate. No, it works on all or none principle, right? So first of all, when it will receive the stimulus, it will pass it, then the next wave of stimulus will start. Okay? Next wave of stimulus will start. Okay? So now what will happen? You know about the neurons, right? You know about the neurons. Just let's say this is the one neuron ending. right? This is the one neuron ending. This is the next neuron. These are the dendrites of next neuron. So what will you do? What will you do? So in between there is what? In between there is a synaptic cleft. In between there is a synaptic cleft. ठीक है बच्चे? So this is a presynaptic neuron. Presynaptic neuron. This one is the postsynaptic neuron. Right, so this together, these three are making the synapse. So it is the synapse. So when the impulse will reach here, bache, what will happen? Ion channels open here. Calcium ion channels will open here. Calcium will start moving in. So because of that, these neurotransmitter, they will release these synaptic vesicles, they will release the neurotransmitter here, and that neurotransmitter will bind to the receptors here, and again the ionic movement will start, and the nerve impulse will pass from dendrites of this postsynaptic neurons to dendrites to cell body, and right, it will carry impulse from dendrites to cell body and cell body to axon. So can I say that axon is taking away the nerve impulse from cell body and dendrites is bringing it towards the cell body? Can I say so? Of course I can. Of course I can. So that is the point. That is the point. That is also PYQ. Okay? So see, synapse is formed by the membrane of a presynaptic neuron and a postsynaptic neuron. So there is a gap called synaptic cleft. So you know that we have electrical synapse and chemical synapse. So chemical synapse mein these neurotransmitters will be released which are inhibitory and excitatory. Excitatory means which will initiate the response. Inhibitory means which will stop that. So inhibitory mein glycine neurotransmitter is the example. So you will tell me another example in the comment section. Right, you are going to tell me the another examples in the comment section. Is that clear? Is that clear? Is that clear? Sure, sure. So at electrical synapse, the membrane of pre and post synaptic neurons, they are in close proximity. जैसे ये देखो, these two, they are very close to each other, right? So electric current will directly from one neuron to another. So transmission of impulse across chemical synapse is very similar to impulse conduction along a single axon, bache. So it will immediate quickly, right? So it is always faster than the chemical synapse, but it is very rare in our system, right? It is very rare in our system. So when it is the chemical synapse, you know that neurotransmitter will be released, bache. So this synaptic cleft is a fluid filled space, so neurotransmitter will be released there, right bache, right? So axon terminal contain vesicle filled with these neurotransmitter. So when action potential arrives at its center, the ionic changes will be there, neurotransmitter will fuse, synaptic vesicle will fuse, and the neurotransmitter will be released here in the synaptic cleft. So now it will bind to the receptor, again ionic channel changes will be there, and obviously nerve impulse will be conducted by postsynaptic neuron. Is that clear? Is that clear? Yes or no? Is that clear? Right? Right. Okay, so calcium ion channels will open here. Done? Any doubt? Yes, any doubt? So before starting this portion, come back to this part. Come back to this part. This part of nerve impulse conduction. Okay, so I'll ask you a few questions, you have to answer my question. So when your neuron is polarized, when polarization is there, when what is there? When polarization is there. So tell me, polarization mein, is sodium potassium pump is active or not? I'll write down the pumps, you have to mention it. Right. Sodium potassium pump, passive diffusion, of course, through passive diffusion is through leaky channels. Then sodium gated channels and potassium gated channels. So you tell me which channels are active when there is the polarization. That's what you need to tell me. This is the gist of the topic. This is the gist of the topic. Tell me. Tell me. What will happen when there is the polarization? What will happen when there is the polarization? Guys, be quick. Be quick. Nikita, very good. So, sodium potassium pump is active, passive diffusion, but they are not. What happened when there is the depolarization? What happened when there is a depolarization? So if it is the depolarization, this pump will not be active, these two will be active, this. And repolarization mein kya hoga? What will be there in the repolarization? This will be closed, but these three will be active. That's what you need to remember, and it will be fine, right? It will be done then. ठीक है जी? Okay. Okay, okay, okay. Is that okay? Is that okay? Sure. Chalo. So next part is the brain, central nervous system. So the very first thing we should know about brain is, देखो, skull is there. Skull is going to protect it. So when you people talk about the brain, in between you talk about the meninges, the coverings. ठीक है? You talk about the meninges. ये देखो. So human brain is well protected by skull. Inside the skull, brain is covered by cranial. Cranial माने brain, meninges, meninges. So for meninges, there is a trick, that is dab. From outside to inside, there is a trick, that is dab. What is the trick, बच्चे? There is a trick, that is dab. Yes, what is the trick? Tell me quickly. What is the trick? Tell me, what is the trick? It is dab. So outside to inside. So D stands for dura mater. D stands for dura mater. A stands for arachnoid. P stands for pia mater. So it is dap only from outside to inside. Okay? Do not change the sequence. Done? Done, bache? So, right. So dura mater, which is very thin. Middle layer is your arachnoid. Inner layer, which is in contact with the brain tissue, it is the pia mater. So these three meninges are also covering your spinal cord. They are also covering your spinal cord. Now come to the brain part. So I'll give you the trick here. Right, I'll give you the trick here. What are we talking? Brain. Brain is also known as encephalon. Encephalon. So it is having three parts: forebrain, midbrain, and hindbrain. My pen is like कि now I am not going to write. See, and then comes the hindbrain. ठीक है? Then comes the hindbrain. So forebrain, midbrain, and hindbrain is there. Done. So bache, the trick is focus on the trick, focus on the trick. Come back, guys. Are you ready for the trick? Hanji, are you ready for the trick? Quickly tell me, are you ready for the trick? Sure. Okay, so in the forebrain there will be cash on delivery, and in that cash on delivery the dian is delivered. Okay, in the forebrain there is the cash on delivery, and in the cash on delivery the dian is delivered. Okay, so cash on delivery, C O D. C means cerebrum. Cerebrum. It's not cerebellum, it's cerebrum. So it is also known as cerebral. It is also known as cerebral. Yes, hemispheres. Cerebrum it is. Okay, cerebrum. Cerebral hemisphere it is. Okay, then comes the O, olfactory epithelium. Then comes the O, olfactory epithelium. Right, so cash on delivery. So D means diencephalon. D means diencephalon. D means diencephalon. And what will be delivered? The dian. The dian means your diencephalon is having three parts. Your diencephalon is having three parts. So T stands from thalamus, H stands for hypothalamus, E stands for epithalamus. So in the paper the question can come like this: which are the parts of the forebrain? So in the forebrain there is the cash on delivery, and what was delivered? The dian. Cash on delivery, C for cerebrum, the largest part, then olfactory epithelium is there, then diencephalon. So the diencephalon means THE, so thalamus, hypothalamus, epithalamus. Understood? Now when it is the hindbrain is like I need to read PCM, right? I am done with biology, I need to read physics, chemistry and maths. Okay, I am done with biology, I need to read physics, chemistry and maths. So P stands for pons viroli, C stands for cerebellum, the little brain. It is cerebellum, little brain, and M stands for medulla oblongata. Okay, this trick is fine. Now when you talk about the midbrain, so here it will be C2CQ, here it will be C2CQ, here it will be C2CQ. So what is C2? C2 means crura cerebri. Crura cerebri. So crura cerebri means cruel uncle. It is also known as cerebral peduncle. It is also known as cerebral peduncle. So what's the trick for midbrain? It is C2CQ. C2 mane crura cerebri. It is also known as cerebral peduncle. Right. CQ mane corpora quadrigemina. It means corpora quadrigemina. Corpora quadrigemina. So these are the two parts here in the midbrain. These are the two parts here in the midbrain. Got it? Got it? So this is about the parts of the brain with the tricks, right? So this question can come and they can even ask you that the naming. So see this hypothalamus, thalamus, this is your cerebellum, this is the arbor vitae, tree of life. Okay, so this is the part of hindbrain, spinal cord, and see this word, it is also important, MCQ. So these two cerebral hemisphere, they are linked with the help of this tract. This tract of nerve fiber is corpus callosum. This tract of nerve fiber is corpus callosum. This tract of nerve fiber is corpus callosum. So guys, how's the energy? How's the energy? Low, ma'am. है ना? How's the energy? Low, ma'am. How's the energy? Low, ma'am. है ना? Done? So for the brain, this is my advice. Please do not do anything other than, right? Please do not go for anything other than NCERT. Just solve the PYQs and do it. Jaise forebrain mein, right? You know that cerebral hemispheres are there. So this is important, that corpus callosum is going to connect these two cerebral hemisphere. Very important it is. Okay? So in the brain, outer portion is the cerebral cortex. Cerebral cortex, and obviously it is your gray matter. It is your gray matter. Okay, and in this gray, and this inner one is what? It is the white matter. So when you talk about this gray matter, it is going to have what? What will be there in the gray matter? Anyone in the class? What will be there in the gray matter? So gray matter means nerve cells, the cytons and non-medulated nerve fibers are there. Cyton and non-medulated nerve fiber. And when it is the white matter, it will be having the myelinated nerve fiber. That's why it is whitish. Myelinated nerve fiber. So one question, corpus callosum, very important. Very important. So cerebral cortex is thrown into prominent folds. It is having the gray matter. Why? Because it is having the cell bodies, I told you, which is giving it the color. So cerebral cortex mainly have motor areas, sensory areas and large regions, right? So these regions are association area. They are responsible for intersensory association, for memory and communication. This question can come. So fibers of the tract. I told you, you should know the meaning of tract, nerves. So CNT, Cartoon Network, T. So tract is basically the collection of nerve fibers. It is in the CNS. So fibers of tract are covered with myelin here. They will form the inner part, the white matter, I told you. So cerebrum wraps around a structure called thalamus, bache. So another important question here is the hypothalamus. So it is the thermoregulator. It is the one which will regulate the temperature. Any doubt? It is the one which will regulate the temperature. Okay? So it is having number of centers in hypothalamus. You know that it will control body temperature, urge for eating and drinking, right? It is having neurosecretory cells which will secrete hormones. Any doubt? Any doubt? And cerebral hemisphere, it is also having amygdala, hippocampus. So they form a complex structure called limbic lobe or limbic system. So along with hypothalamus, they will regulate sexual behavior, emotional reactions. So this is a repeated MCQ. So don't leave this paragraph. Don't leave this paragraph at all. Okay? Don't leave these paragraphs at all. Okay? So midbrain mein, you know that. You already know that. So it is in between the thalamus of forebrain and pons of hindbrain. So it is having a canal called cerebral aqueduct. It is also known as iter. Cerebral aqueduct. Okay, so it passes through the midbrain. So dorsal portion, right? I told you about corpora quadrigemina. Quad word is basically 4. Okay? It is basically 4. So any doubt? Any doubt? Any doubt? Sure? Done? Done. So cerebral aqueduct clear? Done, bache? So it is also known as iter. It is also known as aqueduct of Sylvius. It is also known as aqueduct of Sylvius. Okay. Done. So hindbrain mein pons, cerebrum and medulla. So mostly they ask the question that which one, where is the respiratory center, which is regulating this and that. That's what you need to remember. Okay. So pons consist of fiber tracts bache that interconnect different regions of brain. So cerebellum is also having convolution, means folding. Right, so that they can have more surface area for more neurons. So medulla is connected to spinal cord. So medulla, you know that it will control respiration, reflexes and gastric secretion. Okay? So brainstem mein you are having, this is another important MCQ, please mark it. You are having midbrain, pons and medulla oblongata. Clear bache? So brainstem forms a connection between brain and spinal cord. Understood? Brains and spinal cord. So bache, I don't think that reflex action and reflex arc I need to explain you. You know that they are under the control of spinal cord. Just if someone is going to, if you will touch something hot, immediately you will pull your hand backward, you know that, right? These are our reflexes. Some are conditioned, some are unconditioned. So this is what you can remember. So obviously in such cases, afferent pathway will carry it to the information to the spinal cord, and here in spinal cord, you know that dorsal root ganglia is there, right? Dorsal side is having dorsal root ganglia. So processed information in between interneurons, then motor neurons will carry the impulse. Okay, bache, that is what you need to remember. And in spinal cord, inside there is the gray matter, this H shape, and outside white matter. Okay, ji? Outside white matter. So reflex section, reflex arcs, something very easy, and I think this is how you can manage it. Okay, this is how you can manage it. So now let's talk about the urine formation and countercurrent mechanism part. What say people? Let's discuss about that, because that is also something that we didn't discuss yet. So can we start? Can we start that portion? Hanji, can we start that portion? So can I ask you one thing? Bache, breathing and exchange of gases I have already discussed many times. So dekho, first of all, I know you must be thinking that class will go till 10. How are we going to complete it? First thing, animal kingdom, biomolecules, breathing and exchange of gases is already discussed. Secondly, we are having diagram based session. In this chapter, in body fluid and circulation, maximum diagrams are there. So in diagram based session, it is not like this ki I will give you the image and I will guess it. Of course not. We will discuss that diagrams. So we can cover that portion there if you are interested. Only if you are interested. If you are not, then I will not take it. And if we will get the slot, we can schedule the part 2. What say? But that I don't want, because dekho, what should we do? Let's right now still we have 1 hour. Still we have 1 hour. Let's complete this part first now. Then we'll think of something. Hai na? Otherwise we'll take the part 2. Why are you getting worried? Why are you getting worried? Dekho, this is not something that we should, just read it like this. Shh. No. Human physiology is going to give you majority of questions. So human physiology, plant physiology is something that you should focus. Okay, it is something that you should focus. Arey, biomolecule is already there on this channel. It is already there on this channel. Then why are you getting worried? Arey yaar, break nahi. Right, that break will break our flow. Okay, break will break our flow. We can just chit chat for some time. We can just chit chat for some time. Okay, so anything else? Chalo, may I ask you, okay, I want to ask some questions. So answer one question. The first question is, which sugar is known as the invert sugar? Yes, which sugar is known as the invert sugar? Which sugar is known as the invert sugar? It is a rapid fire, so you have to be very quick. Which sugar is known as the invert sugar? Seriously, which sugar is the invert sugar and you are saying insulin? Are you kidding me? Which sugar is the invert sugar? Sucrose? ठीक है. Okay, what is inulin? What is inulin? I'll just ask 5 questions. You can just judge your preparation. What is inulin? Anyone, what is inulin? Bache, inulin, I-N-U-L-I-N, it is a polysaccharide. It is a polysaccharide and it is a polymer of fructose. It's a homopolysaccharide. It's a polymer of fructose. It is a polymer of fructose. My third question is that which bond is present in polysaccharides? Which bond is present in polysaccharide? It's a very simple question. It is a very simple question. Which bond is present in polysaccharides? Bond. Arey yaar, polysaccharide it is. Why? How can you say peptide? Peptides are there in proteins. It is glycosidic bond. It is glycosidic bond. Okay. Which amino acids are sulfur containing amino acids? Which amino acids are sulfur containing amino acids? Which amino acids are sulfur containing amino acids? It is CMS. Very good, Guna. So it is cysteine methionine. It is cysteine methionine. Okay. Next question is which which fats are unsaturated fat? Name the unsaturated fatty acids. Name the unsaturated fatty acids. Yes, name the unsaturated fatty acids. Unsaturated fatty acids. Ola is the trick. Oleic acid, linoleic, linolenic, arachidonic. Oleic, linolenic, arachidonic. So guys, see, stop crying and see breathing and exchange of gases for 3 hours, and in 3 hours everything is discussed. Okay, matlab I will schedule part 2, but I'm just giving you an option here. Okay, I'm just giving you an option here. Then okay, then biomolecules is here. We have biomolecules. Ye dekho, we have biomolecules. So why are you people worried here? Everything is discussed there. Okay, chalo. So now let's talk about the excretory products and their elimination. So what is the difference in excretion and defecation? What is the difference in excretion and defecation? What is the difference in excretion and defecation? Hanji, what is the difference in excretion and defecation? Anyone? Removal of nitrogenous waste is excretion. I have seen that only few students are the sincere one, the rest all are chit chatting here. देखो, when it is the excretion, you are right that it is the removal of the nitrogenous waste. ठीक है, it is the removal of the nitrogenous waste, and in simple way you can also say that it is the removal of the metabolic waste. Right? What is that? It is the removal of metabolic waste. Right? It is the removal of metabolic waste. This is what we know. Right? This is what we know. Then when it comes to the defecation, it is basically what? It is the removal of undigested food. So if it is the removal of undigested food, means it was never the part of our metabolism. Okay, it was never the part of our metabolism. So that is why we are saying it is the removal of the undigested food. Now animals, they can accumulate ammonia. So if they release ammonia, they are ammonotalic. If it is the urea, they are ureotalic, and if it is the uric acid, they are uricotalic. ठीक है जी, they are uricotalic. So that's what you need to keep in your mind. So removal of ammonia from body is ammonotalism, removal of urea is ureotalism, removal of uric acid is uricotalism. So the kind of question that can come here is, which one is the most toxic nitrogenous waste? It is the ammonia, which is highly toxic, right? And बच्चे, ammonia, it will, in the form of ammonium ions, it will diffuse out from body, right? It is not getting removed with the help of kidney. No, with the help of ammonium ions, it will be removed from our body. What? The ammonia. Okay, the ammonia. So it is highly toxic. A for ammonia, A for aquatic. Aquatic organisms can afford it. Why, bache? Why? So see this, these substances has to be removed totally or partially, right? So you can see that ammonia, urea and uric acid, they are the nitrogenous waste that will be excreted. So ammonia is the most toxic, and it requires large amount of water for its elimination. And uric acid, being the least toxic, it can be removed with the minimum of water. So maximum water is required for ammonia. After that urea, then uric acid. So uric acid, you know na, birds and insects. Do you know that birds and insects, they release that waste in the form of pellets, right? In the form of some pellets, in the form of a kind of paste also. What's that? That is uric acid. So for the removal of uric acid from body, very less, right? From the removal of uric acid from body, very less amount of water is required. Okay? Very less amount of water is required. Is that clear? Right? So this is what you need to remember. So when it is the ammonia, so examples are important, people. Examples are important. So many bony fishes, even aquatic amphibian, even aquatic insects, they are ammonotalic in nature. Many bony fishes, aquatic amphibians, aquatic insects, they are ammonotalic in nature. Examples are important. So it is highly toxic. It will not be removed with the help of kidneys. So ammonium ions will diffuse out from this body. Take care, bache. So it is readily soluble. It is excreted by diffusion across the body surface or through the gill surface as ammonium ion. This is important. Don't underestimate this line. Okay? So kidneys, they do not play the role. So terrestrial adaptation mein, because we have to conserve more water, we cannot just use that much of water for the removal of waste. So bache, lesser toxic nitrogenous waste like urea and uric acid, they form. So mammals and many terrestrial amphibian and marine fishes, they excrete urea, they are called ureotalic. So ammonia produced by metabolism is converted to urea. So before starting it, let me tell you, in our body na bache, we are having the ornithine cycle. It is also known as urea cycle. It is also known as citrullin-arginine cycle. So carbon dioxide and ammonia, it will be converted to urea. And where will it be done? It will be done in the liver. Because ammonia is highly toxic. Even the plants cannot store them. Even the plants cannot accumulate it in their body. right? Even the plants cannot accumulate it in their body. ठीक है बच्चे, ठीक है बच्चे. So it will be converted like this. So CO2 plus NH3, they will form urea, right? So it will be done in the liver. Okay, it will be done in the liver. So for the removal of urea, of course kidneys are playing the role, but you cannot say that ki it is going to use less water than ammonia, but least water usage is done by the uric acid. Done bache. So it is not like that in our body the entire urea will be thrown out. No, some will be conserved to maintain the osmolarity, means to maintain the solute concentration. It's a very important line. Is that clear? It is a very important line, right bache? So reptiles, birds, land snails and insect, they excrete uric acid, okay, in the form of pellet or paste. The minimum loss of water is there. So any doubt? ठीक है. So there are different type of excretory structures, you know that. Just in some cases so it will directly be released by a diffusion, but sometimes structures are there. So when it comes to the structure, we will start, you know that kidneys are there in vertebrates, okay, but other than that we have the protonephridia or flame cells. Do you remember this? Protonephridia or flame cells, they are also known as solenocytes. So where are you going to see this? In the case of platyhelminths, okay, in the case of platyhelminths, and some rotifers and annelida and cephalochordates like Amphioxus, they will have protonephridia, right? They will also have protonephridia. Clear bache? Clear? So protonephridia is present in these cases. Examples are important. Protonephridia is present in these cases. Examples are important. Next, right? So nephridia are the tubular excretory structure of earthworm. Earthworm, you know na, 3 types of nephridia are there, right? So it will remove the nitrogenous waste. It will also maintain the fluid and ionic balance. Understood? Then Malpighian tubules, you know that Malpighian tubules are in the cockroach. So this question can be asked because cockroach is also the part of your syllabus. Okay, bache. So antennal glands or green glands are there in the crustaceans like prawn. So examples are important. So make sure you make the notes. Okay. So now let's come to this part, human kidney. Chalo, what do you know about the human kidney? Tell me quickly. What do you know about the human kidney? Quick. Then let's move to the important topic. What do you know about the human kidney? Quick बच्चे. Quick. Human kidney. देखो ना, that is the structure. So first of all, kidneys are retroperitoneal. Okay. First of all, what are the kidneys? They are retroperitoneal. What are kidneys? They are retroperitoneal. What are kidneys? Yes, people. What are kidneys? They are retroperitoneal. So look at the structure like this. 1, 2. That's the diagram, and you know that it is very easy, isn't it? It is very easy. है ना, kidneys. So I just said they are retroperitoneal. They are present towards this side. What is peritoneum, bache? It is a serous membrane that is covering our viscera. Okay, what is peritoneum? Peritoneum is the serous membrane that is covering the viscera. Okay, bache? That is covering the body organs. Obviously, it is meant for protection. Just say, let's say, imagine this is how the peritoneum is covering our viscera. So kidneys are present somewhere here. Okay, kidneys are present somewhere here. Okay, so basically, can I say that ventral side of the kidney is facing the peritoneum? Can I say that ventral side of the kidney is facing the peritoneum? Yes or no? Can I say that it is the ventral side of the kidney that is facing the peritoneum? Of course I can say so. Of course I can say so. Or you can say that the kidneys are behind peritoneum. That's why we use the word retroperitoneal. That is why we use the word retroperitoneum. So now when you look at its structure, it is also divided into cortex and medulla. it is also divided into cortex and medulla, and basically the nephrons are going to make the kidney. ठीक है, basically nephrons are going to make the kidney. So it is a reddish bean shaped structure. ठीक है, bean shaped organ, right, with a tough fibrous connective tissue covering. ठीक है, so it is a bean shaped structure which is having tough fibrous connective tissue covering. ठीक है जी, connective tissue covering. Are you getting it? So obviously fat layer is covering it, renal capsule, fat layer is covering it, and it is also having the connective tissue, that is renal fascia. Do you know that renal fascia? It is the outermost one. So this fascia will attach the kidneys to this cavity, right? It is going to attach it to this cavity, right, to that abdominal cavity. Are you getting it? Are you getting it? So adipose layer is there, then white fibrous connective tissue, renal fascia, this is present here in the kidneys. ठीक है, ठीक है बच्चे. Okay, okay. So it is in between the last thoracic and third lumbar vertebrae. It is in between the last thoracic and third lumbar vertebrae. Okay. So floating ribs are going to protect it. Done बच्चे, floating ribs are going to protect it. ठीक है बच्चे. Okay, so kidneys. So now in the kidneys, outer one is the cortex, inner one is the medulla, and this is what you are going to see in medulla. What's that? Can you tell me? What's that? Can you tell me? Can you tell me? This is the outer cortex, and here you have the inner medulla. You have the inner medulla. देखो, ultimately in this medulla, and see, the blood vessels will enter. It's something like this. Wait. Done? Done? It is something like this. So can you just tell me about its structure? Yes, bache. Can you just tell me about its structure? हाँ जी. Can you just tell me about the structure? देखो, it's very simple. Let's say this is the kidney. Here you will see a concavity. This is important. It is a PYQ. ठीक है? What will you see here, people? You will see a concavity here. What will you see here? A concavity. That concavity is hilum. That concavity is hilum. So this is the point from where the blood vessel will enter or blood vessels will leave. So you know that, right? You know that aorta, it will further divide. So renal artery will carry the oxygenated blood. Renal vein will bring back the deoxygenated blood. So it is the hilum, the concavity from where the blood vessel will enter and leave, and even this renal pelvis will also enter. This point clear? So this concavity is nothing but the hilum. Now when you look at this medulla, you will see some pyramids there. These are the medullary pyramids, right? These are the medullary pyramids. Can you see that? And they are also known as the renal pyramids. They are the medullary pyramids, and they are also known as the renal pyramids, right? They are the medullary pyramids. They are also known as the renal pyramids. Now if you will look at this part, this is the minor calyx, बच्चे, right? Minor calyx. जैसे let me draw this. Calyx is singular, calyces are plural. जैसे for an example, this one and this one. ठीक है? This is the structure. So this one will be minor calyx. Minor calyx. Calyx is singular. It is further joining, it is forming major calyx, and finally that calyces, they are opening here in the renal pelvis, right? They are opening here in the renal pelvis. Clear बच्चे? They are opening here in the renal pelvis. Done? Done, done, done. They are opening here in the renal pelvis. ठीक है? And then it is the ureter. This is what we have. You know it very well. Done बच्चे? So now from the cortex, some part of cortex is also extended in between these medullary pyramids. जैसे humans में these medullary pyramids are somewhere in between the 10 to 12 range. So a part of cortex is also extended in between these medullary pyramids, right? The part of cortex is also extended in between these medullary pyramids. Can you see that? Can you see that? And you call it as column of Bertini. It is not column of Bellini, it is column of Bertini, right? So when the part of cortex is extended in medulla, it is known as column of Bertini. What is that? It is known as column of Bertini. It is again a previous year question. ठीक है बच्चे, it is again a previous year question. ठीक है बच्चे, ठीक है बच्चे. So now ultimately we know that if you talk about each kidney, right? So this diagram clear, this column of Bertini clear, these renal pyramids clear, right? They are having the papillae here. Ultimately neurons are present here. Ultimately neurons are present here. So each kidney contains 1 million neurons. So both the kidneys will have 2 million neurons. Each kidney contains 1 million neurons. So obviously both the kidneys are going to have 2 million neurons. So now it is the neuron which is the basic structural and functional unit of kidney. But yeah, before that let's talk about the diagram, right? So bache, you know that this is a tube like structure, ureter, and you know it very well. It is also lined with the transitional epithelium. Even this urinary bladder is also lined with the transitional epithelium. And which muscles you will see here? Detrusor muscle, right, which will help in the spilling of substance outside. So detrusor muscle will be there in this urinary bladder. Can you see that? Can you see that? Yes. So before discussing the structure of this, Hanji, before discussing the structure of this, okay, firstly let's discuss the structure and then go ahead. So this part clear, this part clear. So ureter will passes from pelvis into the ureter, urine will passes from pelvis into the ureter, and finally they will open here, and here you will see the oblique openings. ठीक है बच्चे, ठीक है. So urinary bladder, its epithelium, I told you about it. Okay, so basically in the right, so urinary bladder mein, here you will have the sphincter. So there will be the outer sphincter muscle and there will be the inner sphincter muscle. So outer one is your voluntary, you can control it with your own will, but inner one is your involuntary. So circular muscles are going to line this. Done bache? Done. So inter is involuntary, outer sphincter is voluntary. Is that clear? Is that clear? So urinary bladder is having this, urinary bladder is having this, and then comes the urethra. Okay, so urethra will lead to the urethra leads to the end of the penis in males, right bache? You know that in the case of males it is going to be same, the urinogenital opening is same. So this part clear. So do focus on this thing that human kidney is 10 to 12 centimeter in length, 5 to 7 centimeter in width, 2 to 3 centimeter in thickness, and average weight is 120 to 170 gram. Average weight is 120 to 170 gram. Understood? The average weight is 120 to 170 gram. Understood? Understood? Right? Understood? So this part, all these points are clear. Now what you have to discuss, the structure of nephron. Okay, so structure of nephron is very important, and this diagram. So I think this diagram is clear. Dekho, each kidney is having 1 million complex tubular structure called nephrons, right? So it is having two parts, glomerulus and the renal tubule. So let's discuss the structure of nephron, my favorite diagram. Do you like the structure of nephron? See, if you want to understand the urine formation, even the counter current mechanism, you should know about the, yes, you should know about the structure of nephron. Done. And done bache? right? Something like this. Obviously here also the blood network is there, here also the blood vessel and network is there, but as of now I'm just showing you this. Dekho, when you look at this nephron, this cup shaped structure, it is the woman's capsule. It is the woman's capsule. It is the woman's capsule. And then you will talk about these tubules. Okay, what are you going to discuss here? You will be discussing the woman's capsule, right? It is the structural and functional unit of kidney, and bache, it is having woman's capsule, and then it is having these tubules, है ना? Then it is having these tubules. Is that clear? Is that clear? Yes or no? Then it is having these tubules. So this cup shaped structure, this cup shaped structure is the woman's capsule. Now next part of it, this part, it is your PCT. What is the meaning of PCT? PCT mane proximal. Proximal means nearby. Proximal convoluted tubule. It is your proximal convoluted tubule. Right, it is your proximal convoluted tubule. Now this U-shaped structure is your LOH, that is your loop of Henle. What's that, bache? That is your loop of Henle. Now in this loop of Henle, देखो, जैसे we will discuss their position also. जैसे let's say blood is coming, blood is getting filtered. That is how the things will move. So this limb is the descending limb of loop of Henle. So when you talk about okay, so when you talk about the structure, it is the descending limb of loop of Henle. It is a U-shaped structure. It is the descending limb of loop of Henle. This part is the ascending limb. Okay, this part is the ascending limb of loop of Henle. Is that clear? This part is the ascending limb of loop of Henle. Now, when you talk about this is DCT, distal convoluted tubule. This is DCT. This is distal convoluted tubule. And now here it is known as collecting duct. It is known as collecting duct. So DCTs, they will join with collecting tubules, then the collecting ducts. Tell me this part clear? Woman's capsule, PCT, loop of Henle having descending limb, ascending limb, DCT, and it is the collecting tubule. Now look at the blood vessels which are surrounding it. Dekho, it's very simple. You try to manage it. You try to write it. You please write it. You will understand it in a better way. You know that the heart, right? From the heart, the oxygenated blood will be carried by your dorsal aorta. Oxygenated blood will be carried by dorsal aorta. Now because I am talking about the kidneys, so kidney ke case mein renal artery will enter in the kidney. Renal artery will enter in the kidney, people. Renal artery will enter in the kidney. Now this artery will divide. It will divide into afferent arterioles. It will divide into afferent arterioles. Then there is the glomerulus. Right, then there is the efferent arteriole. Then there is what? There is the efferent arteriole. Are you getting my point? Are you getting my point? Then this efferent arteriole will form peritubular capillary network. You write down, please. Peritubular capillary network. Okay, peritubular capillary network. Then obviously there will be the venules, there will be the veins, there will be the inferior vena cava, and finally the there will be the renal veins. Okay, there will be the inferior vena cava, and there will be the heart. So when you look at this, it's very simple. Dekho, you know that this side is having the oxygenated blood. This side is having the oxygenated blood. है ना? Dorsal aorta will carry that oxygenated blood. Renal artery will further divide. देखो, in the kidneys there are 1 million nephrons. ठीक है? In the kidneys there are 1 million nephrons. So if there are 1 million nephrons, so obviously, right? If 1 million nephrons are there, so we have to break this renal artery and we have to make the arteriole so that each and every nephron can get the blood supply. So afferent arteriole, glomerulus, efferent peritubular capillary network. So now let's try to understand it in this diagram. Okay, let's try to understand it in this in this diagram. So you can see this is the afferent arteriole, This is the afferent arteriole. So bache, afferent arteriole will carry oxygenated blood. So you have to focus here, people. You have to focus here. This is wider. This is wider. Done bache. Then it is going to form this tuft of capillaries. We call it as glomerulus. We call it as glomerulus. Then this one which is leaving this woman's capsule, it is the efferent arteriole. E mane exit, it will be narrow. Okay? Afferent arteriole wider, glomerulus will be there. Then efferent arteriole will be there. Wazeem, sir, I am ignoring you. Okay, I am ignoring you. Wazeem sir is on fire. Seriously, I am taking session from last 11 hours and Wazeem sir is on fire. ठीक है भाई, आ जाओ. Please teach biology, and definitely no one is going to clear the NEET examination then. I can bet. Please, I love you more. और कुछ? मतलब you are finally here to disturb us, है ना? You are not liking it. Oh my God. Right? How can she teach? How can she teach without any disturbance? Acha, by the way, Eid Mubarak. I was busy na, so I was not able to call you. Eid Mubarak. Yavar sir, you too. To everyone basically. Why are you so cute? I don't know, yaar. I'm born cute. My genes, they are very good. Genetics. These are not Wazeem sir. ठीक है यार, कोई बात नहीं, ठीक है, सही है. Chalo, now come back. So afferent arteriole, it is wider. Glomerulus, tuft of capillaries. Efferent, narrower. So why this diameter difference is there? Because we want to maintain the blood pressure here, which is required for filtration. Now let's go ahead. This is the peritubular capillary network which is covering this PCT. Now when you look at this U-shaped structure, can you tell me what's that? It is actually that peritubular capillary network, right? You can also call it as vasa recta. This entire thing is what? It is the peritubular capillary network. We are calling it as vasa recta. Wait, let me show you. This is basically that vasa recta. Are you getting my point? So what is vasa recta? Dekho, please understand the diagram, because if you will understand diagram, that will be easy for you. Okay? The urine formation topic, the glomerular filtration will be easy for you. So look at this structure, people. Look at this structure. This U-shaped structure is what? It is vasa recta. One thing that you need to remember. See, Wazeem sir, I'll talk to you later. Right now I'm very busy. Look at this structure, please. This is something very important, students. This is the right now I'll specify something. Later on I will explain it. Okay, later on I will explain it. Look at this. Okay, look at this and please try to understand. Okay, why? So this one is your loop of Henle, right? This one is your descending limb of loop of Henle. This one is your ascending limb of loop of Henle. It is descending. Fluid is descending. Clear bache? And here it is ascending. Now it is covered with this U-shaped vasa recta. It is covered with U-shaped vasa recta, right bache? So this one is the ascending limb of vasa recta and this one is the descending limb. So what am I trying to tell you here? Then that adjacent to descending limb of loop of Henle, you will be having the ascending limb of vasa recta. You will be having the ascending limb of vasa recta. Clear बच्चे? Clear बच्चे? So they both are in opposite flow. This is the counter current flow. This is the counter current flow. ठीक है? Because both are in the opposite direction. So what you need to emphasize, people? What you need to emphasize, people? Right? You need to emphasize one thing, that descending limb of loop of Henle is having the ascending limb of vasa recta, and ascending limb of loop of Henle is having the descending limb of vasa recta. ठीक है? So the flow here in opposite direction. If you will see a descending, ascending, flow is in opposite direction. If you compare it with it, then also the flow is in opposite direction. So that is something you need to keep in your mind. Understood? That is something you need to keep in your mind. Understood? Understood? ठीक है बच्चे? ठीक है? And now when we look at that diagram again, obviously this is distal convoluted tubule and collecting duct. So diagram part clear? Diagram part clear? Sure? Sure, sure, sure. So glomerulus is nothing. It's a tuft of capillaries which is formed by afferent arteriole. So afferent arteriole is wider. ठीक है? It is blood from glomerulus is carried away by efferent arteriole. E for exit, it is narrower. ठीक है? So women's capsule is there which is having glomerulus बच्चे. So glomerulus plus women's capsule together we will call it as Malpighian capsule or renal capsule. Together it is known as renal capsule or Malpighian capsule. Done. It is together known as renal capsule or then highly convoluted primary proximal convoluted tubule is there. Right bache? Right bache? And then you know that you have hairpin like structures, the Henle's loop, then DCT, and then like this. So Malpighian capsule, PCT, DCT, they all are in cortical region. Now this is the part that we need to discuss. So the structure of nephron clear. So the structure of nephron clear. Now बच्चे there are two types of nephron. ठीक है? There are two types of nephron. जैसे let's say this is the kidney. Here you have cortex. Here you have medulla. Here you have cortex, here you have medulla. So when you talk about this structure, देखो, this PCT and all, they are present here. It's like this. That is how your nephrons are present. Do you know that? That is how your nephrons are present. Do you know that? That is how your nephrons are present. Means, what is the meaning here? That your women's capsule, PCT, DCT, and a part of collecting duct is in the cortex, and this loop of Henle and this part of collecting duct will be in the medulla. Is that clear? It will be there in the medulla. This part clear? So this type of nephron is the cortical nephron. Which nephron is cortical nephron? Bache, the one which are having very short loop of Henle. Okay, which type of nephrons are the cortical nephrons? The one which are having very short, right, very short loop of Henle. Which type of nephrons are the cortical nephrons? Right, the one having short type of loop of Henle. It is making 85% of total nephrons. Right, they are making 85% of total nephrons. What are they doing, people? They are making 85% of total nephron. Right, bache? So loop of Henle is right here. Loop of Henle is mostly confined to. It is mostly confined to cortex. It is mostly confined to cortex. Right, loop of Henle is mostly confined to cortex, and very small part of it, right, and a very small part of it runs in medulla. Very small part of it runs in medulla. Is that clear? Very small part of it runs in medulla. Is that clear? Is that clear? So here, peritubular capillary network. Okay, peritubular capillary network is present, but vasa recta is absent or highly reduced. ठीक है? But vasa recta is absent or highly reduced. Understood? It is absent or highly reduced. Understood? Understood? Clear, bache? Sure? Sure, sure, sure. Tell me quickly. Vasa recta is absent or highly reduced. Now we have another type of nephrons also. We have another type of nephrons also. These are known as juxtamedular nephrons. What type of nephrons are they? They are known as juxtamedular nephrons. So in their case, bache, their loop of Henle is deeply embedded in medulla. It is very long. Okay, it is very long and deeply embedded in medulla. Can you see that? Can you see that? It is deeply embedded in medulla. And obviously, what will you see here? What will you see here? You will see the capillary network, right? Vasa recta is there. Vasa recta is highly developed, right? Vasa recta is highly developed. So it is, right? Vasa recta is highly developed. So you know that what will it do? It is basically helping in. It is just making 15% of total nephrons. It is just making 15% of total nephrons. So they are the juxtamedullary nephrons. Okay, they are the juxtamedullary nephrons. And how will they help people? How will they help? They are the juxtamedullary nephrons, so they will help in countercurrent mechanism, means they will help in the water absorption when there is the water scarcity. So it will help in, it will basically help in the urine. It will help in the making the urine concentrated. Done, bache? Done, bache? Done. Any doubt? Hanji, any doubt? If you have any doubt, then tell me. Otherwise, right, I'll answer that doubt. Otherwise, let's go. Okay? Otherwise, let's go. These two types of nephrons clear. Their position clear. The blood flow clear. Hanji, the blood flow clear. Because the topic that we need to start is mechanism of urine formation. Okay? The next topic that we need to discuss is mechanism of urine formation, because mainly it is the Hanji, so it is mechanism of urine formation having 3 steps. How many steps are there? It is having 3 steps. First step is the glomerular filtration, also known as ultrafiltration. Second is your tubular secretion, and then tubular reabsorption. Tubular reabsorption and then tubular secretion. ठीक है, so glomerular filtration also known as ultrafiltration. ठीक है, it is also known as glomerular filtration and it is also known as ultrafiltration. ठीक है, it is glomerular filtration also known as ultrafiltration. देखो, it's very simple. Firstly, understand this part. This is the women's capsule. Here you are having the glomerulus. Now when you talk about the inner layer of this women's capsule, right? When you talk about the inner layer of women's capsule, you know it is having special epithelium. Outer one is having the normal one, but it is having the special epithelium. Do you know that? It is having the special epithelium. Do you know that? Hanji, anyone? Do you know that? Yes, so basically inner layer of women's capsule, it is having the podocytes. What type of cells are present here? It is having the podocyte. Poda mane feet, means these are the kind of cells which are having feet like appendages, and they are arranged in such a manner that slit pores are present. What am I saying? I'm talking about this portion here. I'm saying that we have the inner layer of women's capsule, and this inner layer of women's capsule is having the podocytes. It is having the podocyte. They are arranged in such an intricate manner. They are leaving some minute spaces, and we are calling it as slit pores. Okay? These podocytes, they are arranged in intricate manner, right? They are arranged in intricate manner, right? So that they leave slit pores. Okay, so that they leave slit pores in between, right? Slit pores in between. Now, when you people talk about this glomerulus, they are also having endothelial cells, and these endothelial cells are also having the spaces in between. These endothelial cells are also having the spaces in between. Do you know that? Do you know that? Yes or no? Do you know that? So it is the endothelium of the glomerular blood vessel. So they are having these pores in between, right? right? They are also having these pores in between. So it is the endothelium. It is the endothelium. Now students, you know that when it is the epithelium, it always lies on basement membrane. So there are three layers that we discuss here. One is the inner layer of Bowman's capsule having podocytes, endothelial of glomerulus, and basement membrane present in two. ठीक है, basement membrane present in between. So it is making our filtration membrane. ठीक है, it is making our filtration membrane. Are you getting it? It is making our filtration membrane. This point clear? This point clear? These three layers are present. So obviously blood is entering here. Here the diameter is less. So obviously there is a pressure by which blood will be filtered here. There is a pressure by which blood will be filtered here. So this filtration of blood is quite non-selective. ठीक है, this filtration of blood is quite non-selective. You cannot say that this filtration is selective. नहीं, pressure is maintained there and filtration is there. जैसे let's say today you have decided you are going to clean the room. You don't know how to start. You don't know what to do. ठीक है, you don't know how to start, you don't know what to do, and you are like कि चलो ठीक है, let's put everything out. ठीक है, let's put everything out. जैसे let's say in your room you have the bed, you have this, you have that. चलो bed you are not able to put, but table, chair, right, sofa, books, whatever it is, you are putting it outside. Now then you are like ठीक है, next thing is कि you are like चलो ठीक है, it was quite non-selective, right? All the things are outside. So what should I do? Let's start picking up what is required. So you will pick up some stuff, then you will check it. You will be like, okay, this one is required, this one is not. This is the general way. Same is the case here. Same is the case here. So because these three membranes are there, podocytes are there, endothelial cells are also having the gaps here, right? They are also having the gaps here. Some minute pores are also here, right? Some spaces are also here. So here what is going to happen? Due to this pressure na, the filtration will occur. Due to this pressure na, filtration will occur. What will be there, bache? What will be there? Due to this pressure na, filtration will occur. Are you getting it? Are you getting it? Right, it will be there in the, yes people, it will be, so filtration will be there. So it is known as ultrafiltration. It is also known as ultrafiltration, and the filtrate that form here is the ultrafiltrate, right? It is the filtrate that will form here is the ultrafiltrate. Is that clear? The filtrate that will form here is the ultrafiltrate. Is that clear? Is that clear? Right? So here what are you going to get? You are going to get the ultrafiltrate. ठीक है बच्चे, so it's quite non-selective. We need to absorb many things. So from these blood capillaries, only your plasma proteins. You know that from these blood capillaries, only your plasma proteins, because they are having larger size, they are not able to cross it. So only they will stay. Rest everything. मतलब if from plasma you minus the plasma proteins, remaining everything is there in this lumen. Remaining everything is there in this lumen. So it is the ultrafiltrate. So definitely we need to reabsorb the things as well. So here we talk about certain pressures. Do you know about that pressures? That pressures are not given in NCERT, but they are important, right? They are important. So this ultrafiltrate or glomerular filtrate is protein free plasma. How can you say that? It is just protein free plasma. Some proteins are not able to come, rest everything is there. Even in this ultrafiltrate you are having the glucose. So definitely we need to, the kidney needs to reabsorb that, right? The kidney needs to reabsorb that. So there are certain pressures which are going to play the role. So let's talk about that pressure. So mainly we talk about three pressures. So one is glomerular blood pressure. One is glomerular blood pressure. One is what? It is glomerular blood pressure. It is GBP. What's that? It is the glomerular blood pressure, GBP. Obviously, because of this diameter difference here, the pressure will be created, right? This is the glomerular blood pressure. It is the blood pressure in the glomerular capillaries. Okay? It is the blood pressure in the glomerular capillaries. What's that? It is the blood pressure in the glomerular capillaries. So students, tell me, do I need to mention that? कि this blood pressure is due to the difference in efferent and afferent arteriole? I really don't think so. I really don't think so. Do we need to mention it that it is due to the difference in this efferent and afferent arteriole? So it is somewhere in between 60 to 75 mmHg, somewhere in between 65 to 75 mmHg. So now obviously some filtrate will reach here. This filtrate will also oppose the filtration. Already this filtrate is there. So they were like, yeah, why are you sending other things? They will also oppose. This is also another kind of pressure. Plus the plasma protein present here, they will try to keep the plasma with them only. They will try not to send that liquid here. So that is also a kind of pressure. But these two are the opposite pressures. So another pressure is colloid osmotic pressure, right? Right. It is the next is colloidal. Next is colloid osmotic pressure. So what is this colloid osmotic pressure? It is the pressure, right? It is your COP, colloid osmotic pressure. It is the pressure which is due to. It is the pressure in glomerular capillaries. It is also the pressure in glomerular capillaries, okay, which is due to the plasma proteins, present in it, yes bache, which is due to the plasma proteins present in it. So obviously it resists filtration. It is not favoring it, it resists filtration, it stops filtration, right? So it is somewhere in a range of 30 to 35 mmHg. It is 30 to 35 mmHg. Okay, next is this is blood, you can also call it as blood colloidal osmotic pressure, BCOP. Next is the capsular hydrostatic pressure. Okay? Next is the capsular hydrostatic pressure. So as I said, some filtrate is present here. That filtrate will also resist. Right, that filtrate will also resist the filtration. That filtrate will be like, I don't want anything here. So it is the osmotic pressure created in the blood of glomerular. It is the pressure caused by the filtrate that reaches into the woman's capsule. Okay? It is the pressure caused by filtration. It is the pressure caused by filtrate present in the lumen of woman's capsule. So it is somewhere in between 10 to 18 mmHg. 10 to 18 mmHg. Are you getting my point? Are you getting my point? So obviously, dekho, you for your NEET examination, we have to check your marks in physics, in chemistry, in biology. We will add it, subtract it, and this is how we are going to check. Same is the case here. So one pressure is favoring filtration, two pressures are opposing filtration. So let's count the net effective filtration pressure. Right, net effective filtration pressure ki actually what is the net effective filtration pressure, that actually what is the pressure that is for the filtration, right? Net effective filtration pressure. Okay, net effective filtration pressure. So let's talk about it. So capsular, let's say, so we have to take this and we have to subtract these two pressures. Okay, we have to take this and we have to subtract these two pressures. ठीक है बच्चे, ठीक है. So it will be, let's say if it is 75 plus 32 minus 18, and it will be near about, okay, sorry, my bad, it is plus and here it is minus. So this is the pressure which is favoring, so we have to subtract it from this pressure which is opposing the filtration. So it will be yes bache, it will be, now so it's easy, obviously 25 mmHg. It is 25 mmHg. So this is the net effective filtration pressure. It is the net effective filtration pressure. Okay? It is the net effective filtration pressure. So let's focus on the NCERT here. So So Malpighian capsules, this part is done and dusted. The diagram clear, vasa recta clear. Okay, my screen is like, nah yaar, I don't want to work. So glomerular filtration. So when you talk about the glomerular filtration, so on an average 1100 to 1200 ml of blood is filtered by the kidney per minute. Okay, so per minute, how much blood is filtered by the kidney? You are talking about all the nephrons, 1100 to 1200. And you know that when you talk about the cardiac output, if you remember, cardiac output is near about 5030 ml, yeah, approximately 5 liter, right? It is approximately 5 liter. Yes or no? Cardiac output is 5 liter, na? That in 1 minute ventricles can pump out 5 liter of blood. So that is why it is written. Okay, that is why it is written. Yes, bache, that is why it is written. Just you can even consider it like this, 5030 ml. So 1/5 of it. Something like that, na? So basically they are saying 1100 to 1200 of blood is filtered by the kidneys per minute, which is basically roughly the 1/5 of the blood pumped out by each ventricle of heart. Okay? So glomerular capillary blood pressure, it causes filtration of blood through 3 layers. So you know that layer. Endothelial of glomerular blood vessel, epithelium, the basement membrane. Okay, bache? So epithelial layer is having podocytes, and they will form filtration thread. So blood will be filtered so finely that all the constituent of plasma except the proteins, they will pass into the lumen. So this part clear. This is as such NCERT. This part clear. So amount of filtrate formed by the kidney per minute. Okay, it is the amount of filtrate formed by the kidney per minute. Amount of filtrate formed by the kidney per minute is GFR, glomerular filtration rate. Amount of filtrate filtered by the kidney per minute is GFR. So GFR is 125 ml per minute and it is 180 liter per day, but the urine output is near about 1.5 liters. So basically 99% of the filtrate will be reabsorbed. Okay? So kidneys, they have the mechanism for regulation of this filtration rate, right? So this mechanism we will discuss later. Let's move to the next step of tubular reabsorption. Okay? So the filtrate formed is 180 liter per day, but you know that urine is 1.5 liters. So near about 99% of the filtrate will be reabsorbed. This is reabsorption. Okay? So tubular epithelial cell in different segments of nephron, you know that actively or passively it will reabsorb. So when it is proximal convoluted tubule, maximum reabsorption will be there. Your glucose, right, actively, your amino acid, sodium, they all will be reabsorbed actively. So this is what you need to know. Actively matlab by the usage of ATP they will be reabsorbed. Okay? Nitrogenous waste, chloride ions, they will be reabsorbed passively. Okay? So during urine formation, right, tubular cells, they also secrete H positive and K positive and ammonia into the filtrate. So H positive, K positive and ammonia, it will be released into the filtrate. Done, bache? Done. So rest, okay, so rest bache, I'll schedule another class, or in the diagram based session we will discuss it, right? So whatever slot I will get, if I will get slot for scheduling it, right, so I'll schedule the lectures for 5 hours. So we will complete the entire portion, and otherwise bache, we will solve it in diagram based session. But yeah, today I'm happy that you people didn't cry that please end the class, right? So I hope you guys have enjoyed the session. So one thing that I'm expecting from you is that after the class, do revise it from the NCERT, and trust me, we have covered everything, right? We have covered everything. Okay, tomorrow bache, already we are having a lecture scheduled, na? Question practice ka. So let's practice the question. With that I'll explain. And yes, if you like the lecture, so I want you people to comment in the comment section. Okay, I'll be waiting for your comments. Okay, so keep revising, just keep your mind calm. If you have just started few months back, do not think about the result. As of now, you should focus on the your only focus is to put the efforts in the right direction. Trust me, you will learn something good. Okay, right now you people should not expect your result. So hope you like the class. So I want to see the comments in the comment section. So we'll share the PDF in the Telegram group. So take care, tada, bye bye. Thank you people.